Man, I hate science.
Yeah, me too.
Science is so stupid, it doesn't make any sense.
I know, I'll never be good at science.
Have you ever been part of a conversation like this? Lots of people have. Learning and studying scientific concepts requires a special type of thinking that doesn't come easily to everyone. You may be surprised to know that even people who study science for a living often struggle with the complicated formulas and theories. Most people find science class fun when they're little, but in high school and college, scientific study gets way more difficult. The books are hard to read, the words are hard to pronounce, and even the pictures are hard to understand. Science can seem really unfriendly to a student, but there are ways to overcome the challenges and learn to study science effectively. Let's explore a few techniques that will help make science learning easier. One of the toughest challenges in science is understanding the long, strange vocabulary terms. Science textbooks are full of crazy words like plasmodesmata, thigmotropism, and chemiosmotic phosphorylation. It's really hard to learn science effectively when all these weird words are being thrown in your face. How did scientists come up with these words anyway? Why do they have to be so unfriendly? Most of the terms you see in science are combinations of word parts. This is especially true with technical terms. Scientists originally coined the terms by putting together word parts from the Greek and Latin languages. For example, the word thigmotropism comes from a combination of two Greek words, thixis, meaning touch, and tropos, meaning turning or direction. Thigmotropism is the word we use to describe the coiling growth patterns that some plants exhibit in response to touch. Think about vining plants like beans, grapevines, and the common morning glory. These plants can climb fences and trellises by coiling their shoots around any object that they touch. You might think it strange that a long, weird word like thigmotropism was ever invented, but imagine if you were a plant biologist trying to describe a morning glory. How would you talk about its coiling growth pattern? The morning glory climbs by using its ability to turn the direction of its vine growth in response to a touch stimulus. No, that takes too long. If you were a scientist, you would want to invent your own word to describe this concept. Then you could just say the morning glory climbs by using thigmotropism. That's exactly why scientists invented the word. Most scientific terms refer to Greek or Latin roots, but that doesn't mean you have to know Greek and Latin to understand science terminology. Many word parts are familiar to us in the English and other Romance languages. Take the word photosynthesis, for example. It comes from the Greek roots photo, syn, and thesis. Photo means light, as you probably already knew from other words like photography, photon, and photocopy. The word part syn means with or together. This might seem like a foreign definition to you, but think about familiar words like sympathy, synagogue, synchronize, system, and symbiotic. All these words use a form of the Greek root syn to describe a togetherness of something. So we've got photo and syn. What about the word part thesis? In Greek, this word means setting, putting, or placing. So photosynthesis means light together putting, or putting together with light. In other words, photosynthesis is the process by which plants put molecules together using the energy they get from sunlight. As you can see, understanding the meaning of science terms is easier when you break them down into smaller components. This might be a good time to remember the three main parts of a word: the root, the prefix, and the suffix. A word root is the primary unit of a word. It's often the longest, most central part of the word that carries the most significant meaning. Both prefixes and suffixes can be added to a word root in order to modify its meaning. Prefixes are word parts that appear before a root, like un, in, dis, re, and a. Suffixes are word parts that come after the root, like ed, ing, les, li, and ism. When prefixes and suffixes show up in science words, they work the same way as they do in regular English. For example, the suffix ism appears in the word thigmotropism. You don't need to know that ism is a suffix that indicates a noun. You already know plenty of similar isms like journalism, optimism, and tourism. These are all nouns, so thigmotropism must also be a noun, even if we're not yet sure what it means. Try to build connections between words that you already know and words you're just learning. You'll start to get a sense of what the word parts mean, and then you can apply them to other words. For example, the word sympedalous is used to describe a type of flower. Can you figure out what it means? Well, we know that flowers have petals, and we just learned that syn or sim means together. It shouldn't surprise you to learn that sympedalous describes a flower in which the petals are joined together. Lots of scientific terms are actually multi-word phrases like selective permeability and punctuated equilibrium. These terms require you to take one concept and apply it to another. It's not too tough as long as you remember that a phrase like selective permeability isn't any different than the phrase fluffy dog. Just like fluffy describes a dog, selective describes the permeability of something. For example, the permeability of a cell membrane. We can deduce from this phrase that apparently it is possible to have permeability that is not selective, just like it's possible to have a dog that is not fluffy. But here we're talking about a permeability that is selective. Cell membranes allow some substances to go through while blocking others. They are permeable, but they are selective in how permeable they are. So cell membranes have selective permeability. You see, it's not so hard to understand science phrases when you think about how the words relate to each other. As you're developing your science study skills, keep in mind that everyone has different learning styles. Some people are visual learners, meaning they tend to learn better when concepts are presented through images. On the other hand, verbal learners relate better to words and sentences. Try to pay attention to how you learn best and tweak your study methods to match your preferences. For example, I've noticed that I'm a visual learner. I like to use pictures, flowcharts, concept maps, and even colors and symbols to learn new things. I usually scan all the pictures in a chapter before I start reading any of the text. Sometimes I even make my own color-coded study guides. But if visual aids aren't really your thing, then you may be more of a verbal learner. Verbal learners often benefit from reading text aloud to themselves or writing about the subject. If you're a verbal learner, you could also try listening to a recording of someone talking about the subject or talk with a classmate about the subject. Most people do not subscribe completely to one learning style, but if you notice yourself leaning towards certain patterns, then use those patterns to your advantage. For example, when I study cellular processes, I find it helpful to keep reminding myself of how these processes fit into a larger scheme. This is called the holistic approach. Learning is holistic when new concepts are seen as being integrated and inseparable from one another. I like this approach for learning chemical and cellular pathways because it keeps me from getting lost in all the details. Using the holistic approach, I focus on relating chemical processes to the overall needs of a cell. Other times, I use a more analytic approach by breaking down concepts into subdivided parts. The analytic approach works best for ideas that need to be categorized into separate groups. The classification of organisms into kingdoms and phyla is a perfect way to learn analytically. Again, it doesn't matter exactly how you study. There's no one way to learn science or any other subject. What's most important is that you focus on the methods that work best for you. Learning and studying scientific concepts is challenging for many, but you can make it easier by following the tips you've learned here. Long, complicated scientific terms can be better understood by breaking down the components. Look for word roots, prefixes, and suffixes. Try to recognize any Greek or Latin word roots that you can and relate them to other words that you already know. Dissect phrases and take the time to understand how multiple words fit together. Beyond the vocabulary, pay attention to your favorite learning styles and use them to your advantage. Try to find out if you're a verbal or visual learner. Think about whether a holistic or analytic approach is best in every situation. Allow creativity into the process of learning. If you tailor your study methods to your own needs, then you won't have to stress about the challenges of studying science. When you first took science class in school, you probably learned the basic steps of a scientific investigation. You've likely heard of words like hypothesis, experiment, and observation. You may have even memorized a prescribed set of steps. The scientific method is a set of procedures that scientists follow in order to gain knowledge about the world. However, the steps involved in the scientific method vary widely among the different scientific disciplines. Chemists follow the method a bit differently than psychologists. Geologists and botanists have their own unique methods. So, is there really one scientific method that encompasses all of science? To find out, we'll need to learn more about the scientific process. There are 6 key steps that tend to characterize the scientific method. The first step is the question. This is the part where a scientist proposes the problem that he wants to solve. A well-conceived question usually leads to a hypothesis, a potential answer to the question at hand. Sometimes hypotheses look more like predictions. The scientist predicts what the outcome will be when he tests his hypothesis. The scientist's test is also called the experiment. Experiments are ordered investigations that are intended to prove or disprove a hypothesis. Important data comes from performing an experiment. The scientist has to make observations of the results that he gets from the experiment. An observation is a statement of knowledge gained through the senses or through the use of scientific equipment. Observations are crucial for collecting data. Once the results are in, the scientist must begin his analysis. Data analysis involves comparing the results of the experiment to the prediction posed by the hypothesis. Based on the observations he made, the scientist has to determine whether his hypothesis was correct. Then he sums up his findings with a conclusion. The conclusion of a scientific process is a statement of whether the original hypothesis was supported or refuted by the observations gathered. The scientific method usually employs all 6 of the steps I mentioned, but the steps don't always occur in the same order. Real scientists may go back and repeat steps many times before they come to any conclusions. It's actually better to use the word elements to describe the steps, since the first step, question, does not always come first. Sometimes, for example, it's an observation that came first and spawned the initial question. Likewise, observations that are made during an experiment can inspire more questions that scientists have to answer. The scientific method is much more fluid than you might think. Let me show you how the steps can feed back and branch out from one another with an example from my own experience. Last weekend, I had a minor ordeal with my internet connection at home. I had started up my laptop and I was frustrated to find that I couldn't get on the internet. I made the observation that my laptop wasn't receiving an internet connection. I asked myself a question: is something wrong with the internet itself, or was it just my laptop? One way to begin answering this question was to check the connection on the desktop computer. Quickly, I formed a hypothesis. If the internet isn't working on the desktop either, then the problem is beyond my laptop computer. The experiment I performed was to check the desktop's connection, and my resulting observation was that the internet didn't work there. So, by analyzing the evidence, I was able to form my first conclusion. Nothing is wrong with my laptop, and something is wrong with the internet connection. Now, this conclusion answered my first question, but it still didn't get my internet to work, so I had to pose another question. Where exactly was the problem occurring in the chain of internet devices? Was it the cord between the modem and the router? The cord between the router and my computer? Or was the problem in the router itself? I had to form another hypothesis. If both my internet cords are properly plugged in, then there must be a problem with the router. My experiment was to check both cords and the router. My observation was that both cords were plugged in and that the router was off. I analyzed the evidence, and my conclusion was that I couldn't connect to the internet because my router was off. Are you seeing a pattern here? Once I came up with one conclusion, it left me with another question that I needed to answer. From every question, I got a hypothesis. From every hypothesis, I got an experiment, and from every experiment, I got observations that led me to more conclusions. I don't need to tell you the rest of the story. Eventually, I figured out that my router was unplugged and solved my internet problem by plugging it back in. The important thing to see here is that the scientific method doesn't follow a straight line. It loops back on itself in countless ways. It branches out into new investigations. There's never just one way to answer a question. Keep in mind that the key elements of the scientific method are not the only things that keep science moving forward. It's not nearly as rigid as many textbooks describe. In addition to hypotheses, experiments, and analyses, science requires more subjective processes like creativity, experience, and intuition. In my problem with the internet, I needed to reference my basic knowledge of computerized technology. I knew from experience that unplugged cords were most likely the cause of the problem. But I also brainstormed for a minute or two about other possible causes. I have 2 curious cats and a husband who often changes the arrangement of our cords on the power strip. It's dusty under our desk and the router is getting old. Any of these factors could have affected the connection. I didn't end up pursuing those other possibilities, but I did use plenty of creativity while trying to solve my problem. If it had turned out that my problem was more complicated than an unplugged cord, then I would have had a handful of other ideas to try. Many scientists have used a combination of skills to develop their ideas. Take Charles Darwin, for example. He is credited with devising the theory of evolution by natural selection. It's a biological theory, but Darwin wasn't a biologist. He was a naturalist who studied not only plants and animals, but anything to do with the natural world. Darwin read up on geological principles written by Charles Lyell. He studied theories of economics and population by Thomas Malthus. He bred fancy pigeons and collected natural artifacts on his farm. And yes, he took an amazing voyage to study exotic creatures around the world. Darwin wouldn't have come up with his ideas about evolution without combining all of these influences. It took some creativity and 20 years of research to put all his ideas together. Darwin's work shows that the scientific method is really a fluid, integrated process. It's worth mentioning that scientific studies are kept in check by communities around the world. Most scientific journals employ a process of peer review, whereby scientists critique each other's work and decide whether it meets the standards of the scientific community. Scientists have to pool their resources and check in with one another before they can perform any significant work. But scientists also compete with one another to discover new things. They know that the prize goes to the scientist who gets things right and publishes first. Even Darwin had to compete with other naturalists like Alfred Wallace and Jean-Baptiste Lamarck. But he also worked closely with his friends, Joseph Hooker and Thomas Huxley, who also studied biology and helped him develop his theories. As you can see, the scientific method does not work in isolation. It functions within a community, both inside and outside the realm of science. So it turns out there really is no one way to follow the scientific method. There are key elements that should appear in any investigation, but science is done differently in every profession, and no one follows the exact same process. The scientific method describes the processes by which scientists gain knowledge about the world. It's characterized by 6 key elements: questions, hypotheses, experiments, observations, analyses, and conclusions. These elements are interrelated steps, so they don't always function in the same order. Other more subjective skills like creativity, experience, and intuition also have a place in the scientific method. Science is characterized by professional competition and develops through the collaboration of scientists in the worldwide community. Have you ever thought about what goes into a real scientific experiment? Most of us get to do science investigations when we're young. School experiments are easy and fun. But what about real scientists like chemists, physicists, and medical researchers? What kind of work did they have to do? How do they go about designing their experiments in the laboratory? Experiments, remember, are one of the key components of the scientific method, which is a set of procedures that scientists follow to gain knowledge about the world. Other components of the scientific method are questions, hypotheses, observations, analyses, and conclusions. While experiments are only one part of a scientific investigation, they end up being accountable to the other elements. In this lesson, we'll see what goes into making a valuable experiment and learn how scientists design useful investigations. A scientific experiment is an ordered investigation that attempts to prove or disprove a hypothesis. So its primary purpose is to test whether someone's prediction is correct. In designing experiments, scientists have to answer some pretty complicated questions, like, does my experiment answer the question I'm trying to solve? Does it adequately test my hypothesis? Can I make observations about the results of my experiment, and will I be able to analyze those results? Finally, if I run this test, will it allow me to come up with some kind of conclusion? Scientific experiments are different from other kinds of tests because they are required to fit in with the scientific method. Another important factor is peer review by the science community. A scientist's work isn't generally recognized unless he follows the standards set by other scientists around the world. A few basic rules apply to the design of a good experiment. Let's take a look at what a science experiment needs. Rule number 1. The experiment must show that a hypothesis is either supported or not supported. In science, we try to avoid using terms like right and wrong, and we don't say that hypotheses are proven or disproven until we're really sure about it. A single experiment is not enough to prove anything with 100% certainty. The second rule is that the results of an experiment must be measurable and objective. Scientists use standard units to measure different properties like length, time, volume, mass, and speed. Sometimes we need special equipment to observe things in a measurable way. For example, we can't see ultraviolet light or hear infrasonic sounds. We need special devices to detect and measure those properties for us. The third rule for scientific investigations is that the experiment must be repeatable by other scientists. Peer reviewers want to make sure that other scientists can run the same experiment and get similar results. This is one of the reasons we standardize our measuring tools and equipment. Scientists must be able to read anyone else's report, follow the steps exactly the same way, and compare their findings to the original test. In science, new ideas aren't taken seriously until many scientists have tested them many times. So it's important that scientists share their techniques and confirm each other's findings. So how do scientific ideas become part of the community knowledge base? If everyone's always double-checking each other's work, how do hypotheses become theories? How do theories become scientific law? Well, first of all, keep in mind that theory in the world of science is not the same thing as a theory in everyday language. I might have a theory that my friend Jackie is going to ask her classmate Jimmy on a date, but that's not the same as a scientific theory. In science, a theory is a statement that is generally accepted as a summary for a hypothesis or a group of hypotheses. You can also call a theory an accepted hypothesis. When one hypothesis has been tested by many different scientists, and most of them have come to the same basic conclusion, then we can start calling the hypothesis a theory. There isn't any grand master of science who makes the final decree about a theory. It's more like a general consensus, and a theory can still be disproven if further research reveals enough evidence to refute it. A law is different from a theory in that it is viewed as a universal fact. A scientific law is a general statement about a group of observations which has no exceptions to the rule. Most laws can be stated as mathematical equations, like Boyle's law and Pascal's law. Laws in biology are statements about how living things work. For example, Mendel's laws. To explain the results of his experiments with peas, Gregor Mendel developed the law of segregation and the law of independent assortment. No genetics experiment has ever disproven Mendel's laws, and so his statements are still viewed as laws today. But even Gregor Mendel had to design a valid experiment in order to receive credit for the work that he did. The use of a control is one element that really makes an experiment scientific. When scientists are trying to test one factor, they have to make sure there aren't any variables going on that could mess up their results. For example, let's say you wanted to see whether sunflowers or daisies grow faster from seed. You'd plant a sunflower seed in one pot, a daisy seed in another, and then put the pots in a window and water them every day. But what if you put the sunflower pot in a sunny window and the daisy pot in a shady window? Your findings would be skewed, right? You wouldn't know for sure whether sunflowers always grew faster than daisies, or if it was just your sunflower growing faster because it got more sun. In science, a control is a means of ensuring that only one factor is being tested at a time. In order to make yours a controlled experiment, you would need to place both flower pots in the same window. You'd also give each plant the same amount of water, make sure the seeds are planted in the same type of soil, and plant both seeds at the same time. The more you controlled the variable factors in your experiment, the more confident you'd be that the results would accurately address your experimental question. While we're on the subject, let's check over this flower experiment to make sure it follows the other rules. Remember, our peers in the scientific community are scrutinizing our every move. So first of all, is our experiment designed so that it either supports or refutes our hypothesis? Well, we need a hypothesis. Let's say our hypothesis is sunflowers will grow faster than daisies when grown in similar conditions. Our experiment is designed to test and compare the two growth speeds, so we've got that rule covered. Next, do we have a means of generating measurable, objective results? Our results are related to plant height over time, so we'll need to measure the plant's heights in millimeters and the time in days and hours. We'll probably make a chart that shows how much each plant grew over increments of time. Finally, can other scientists repeat our experiment in the same way we designed it? Well, obviously anyone can grow plants in their window, but as scientists we need to specify every detail about our experiment. How much soil are we putting in the pots? What will the average temperature be? How many hours of light will each plant receive? Exactly how much water will we give to the plants? What kind of potting soil will we use? How often will we measure the heights of the plants? How long will we continue our experiment? I know this seems like a lot of questions, but these are the rigorous requirements that scientists face in their work. If they don't defend their experiments with detailed information, then their work isn't recognized in peer review. As you can see, scientific experimentation is no walk in the park. In order to receive credit for their work, scientists have to design and implement experiments with great complexity and precision. In fact, some scientists publish hundreds of pages of work for every study they perform. It may seem like overkill, but the challenges of the scientific community help ensure that only valid, reliable science gets fully recognized and used. It takes a lot of work to design a good experiment. To fit in with the scientific method, experiments have to be relevant to the questions, hypotheses, observations, analyses, and conclusions of the investigation. Experiments are designed so that they support or refute a hypothesis and give results in terms of measurable, objective data. An experiment must be repeatable by other scientists so that it holds up in peer review. When a hypothesis becomes generally accepted by the science community, it becomes a theory. Scientific principles that are viewed as fact become laws. In designing their experiments, scientists must incorporate controls to ensure that only one variable is tested at a time. Experimental design and implementation is tough, but these challenges help to keep science moving forward. The scientific method is a set of procedures that scientists use to learn about the world. Scientists generally follow this method, but they also compete and collaborate on their work in order to reach a consensus within the scientific community. Sometimes a scientist's work can take years to complete, but he still has to communicate his ideas to others. How do scientists summarize their findings so that other scientists can understand it in a matter of minutes? The answer is in the tables, graphs, and charts that illustrate their data. In this lesson, we'll look at the basic types of charts and learn how to read them effectively. Let's say that you were a scientist who wanted to investigate how fast a sunflower grows compared with a daisy. You'd plant a sunflower seed and a daisy seed, grow them both with plenty of sunshine and water, and measure the height of each plant every day. To keep all your measurements organized, you'd likely use a table. Most scientists use tables as a way of arranging information into vertical columns and horizontal rows. Tables are useful for data that describes two different factors, or variables, because it reads in two directions. In your plant growth experiment, you'd collect data about two different things, time and the height of the plants. Let's say you'd record time measurements in the first column and the measurements for plant height in the second column. To be clear, you'd specify which units you're using for each variable. This is what a basic table looks like for most scientific experiments. The two variables that you're recording are different in that one variable is dependent upon the other. Since the height of the plant changes due to the passage of time, we say that height is dependent on time. So in this case, height is called the dependent variable. A dependent variable is the factor being measured in an experiment which changes in response to the independent variable. The factor that's not dependent on anything is time. That is, nothing's going to change how time elapses during the experiment. Therefore, we call time the independent variable. It is the factor that is considered to be constant during an experiment, which affects the dependent variable. In tables, the independent variable is usually listed in the first column, and the dependent variable is listed to the right. But remember, you'll need to add a third column to collect your data about the daisy. Every day you'll make two measurements, one of the height of the sunflower plant and one of the height of the daisy plant. So you should be filling up one row every day in your experiment. By the time the experiment's done, your table should be full of useful data. This table is a great way to keep your data organized, but it's not so easy to draw any conclusions by taking a quick glance. A better way to look at this information would be to transform it into a line graph. A line graph depicts the relationship between the dependent and independent variables. Each variable is plotted along one of the two axes on the graph. The x-axis is the horizontal axis, which usually plots the independent variable. Since your independent variable is time, then you'd plot time in days along the x-axis. The other axis is called the y-axis. This is the vertical axis that usually plots the dependent variable. So your dependent variable is plant height. You're actually going to have two lines on the graph, one to show the growth of the sunflower plant and one to show the growth of the daisy. Let's make the sunflower line orange and the daisy line blue. You'll need to add a key to the bottom to show which line describes the growth of which plant. As you're transferring your data, you'll need to be sure to plot all the numbers correctly. The first measurement for the sunflower says that it was 1/2 centimeter tall on the first day, so you'd find the first day on the x-axis and draw a dot that lines up with 0.5 centimeters on the y-axis. You'd continue the same way for all of your data in the second column. Then, to plot the growth of the daisy, you'd use the data from the third column and draw the line in blue. Creating a graph is pretty simple, but interpreting it can be tricky. Looking at our graph of plant growth rates, what kind of conclusions can we make? Well, we can easily see that the sunflower grew faster than the daisy on average. It reached a greater height in the first 9 days, and it sprouted a full day ahead of the daisy. By comparing the slope of the lines at separate intervals, we can also see that the sunflower had the highest peak growth rate. In other words, the highest growth speed in the sunflower was greater than that of the daisy. Alright, enough with the easy stuff. Let's take a look at a less familiar line graph. Can you read and interpret a graph like this? It looks like it's relating the percentage of basal metabolic rate to the time that has passed after eating a meal. They've put time on the x-axis as the independent variable, just like you did in your plant growth chart. The dependent variable here is the percentage of basal metabolic rate. Do you know what basal metabolic rate is? Well, guess what? You don't have to. To interpret this chart, you just need to see that proteins, carbohydrates, and lipids account for different percentages that fluctuate over several hours. It looks like carbohydrates account for the highest percentage in the first 45 minutes. But after that, proteins win out and take the highest percentage. Proteins peak at about 30%, and lipids stay fairly constant as the lowest contributor. You see how easy it can be to read a line graph? I don't even have to understand the terminology. I only need to use the labels and the x and y axes to interpret the relationship between these two variables. Line graphs aren't the only way that scientists represent their data. Different types of charts may be better suited for the type of information a scientist wants to show. All charts are visual representations of data that use symbols to indicate information. In a line graph, the data is represented in lines, but data can also be represented as bars in a bar chart or wedges in a pie chart. Let's take a closer look at those two charts. In a bar chart, information is represented in rectangular bars that are proportional in length to the data. Here's a typical bar chart depicting numerical values for several geographic regions. Without even knowing what the values represent, we can tell from the relative heights of the bars that the United States has the most, Japan has the least, and South Africa and Europe are somewhere in the middle. We typically use a bar chart when we want to compare the amounts of something between unrelated groups. In a pie chart, information is represented in sections of a circle that are proportional in width to the data. In this pie chart, we see 2 very large slices of pie, a handful of medium slices, and many small slices. Pie charts are used when we want to show how different groups make up the percentages of something. So unlike bar charts, pie charts have a defined limit of 100%, which is the entirety of the circle. If one slice took up half the circle, then we'd know that it represented 50%. This particular pie chart represents 100% of mammal species. Each slice represents a different family of mammals. From the key, we can determine that the largest slice represents the family Rodentia. So that means rodents make up the largest family of mammal species. Line graphs, bar charts, and pie charts are all good for representing numerical data, but other charts can be used to illustrate relationships between concepts. Take a flowchart, for example. A flowchart is a diagram that shows how concepts or steps in a process are related to one another. This flowchart is showing us a negative feedback loop for glucocorticoids. Do you know what glucocorticoids are? Neither do I, but according to the arrows, they probably come from the adrenal cortex, which is somehow acted upon by something called ACTH from the pituitary gland. Again, I don't need to know the terminology. Flowcharts are designed to oversimplify difficult concepts. This makes them a great tool for summarizing, memorizing, and studying science. Here's another flowchart, which is really a food web. It shows how different organisms are related to each other based on who eats who. From this diagram, we can see that the bald eagle likes to eat sea ducks and large fish, since there are arrows pointing from these organisms to the eagle. The sea ducks, in turn, eat bivalves and benthic invertebrates. But bivalves are not only eaten by sea ducks, they are also eaten by tundra swans and herbivorous ducks. I could go on, but I don't need to. This food web tells me everything I need to know about who eats who in this environment. As a student learning science, you will end up studying more complicated flowcharts like this one, depicting the citric acid cycle. Don't get intimidated by intricate charts like this. They work just the same way as the flowcharts we saw before. You've got labels and symbols describing each step in the process. You've got arrows showing how one step proceeds toward the next. Take your time with complex charts like this and use the legends and keys you're given. Just remember that the purpose of any graph or chart is to make detailed information easier to understand. The information that scientists accumulate in their studies is often too complicated to be summarized in words. To communicate their findings to the rest of the community, scientists use tables, graphs, and other charts. Tables organize data into rows and columns, which can be transformed into line graphs. Bar charts compare data between unrelated groups, while pie charts compare data as relative percentages. For non-numerical information, scientists organize concepts into flowcharts, which can show relationships between concepts or the order of steps in a process. Tables, charts, and graphs are designed to summarize information and make it easier to understand scientific ideas.
All things are composed of matter. You, me, and everything around us is made up of matter. Matter is a physical substance that takes up space and has mass. An atom is the basic unit of matter. But what does it look like? What is it made of? Atoms are composed of 3 subatomic particles: protons, neutrons, and electrons. We can categorize protons, neutrons, and electrons using 2 basic traits: charge and mass. Charge is the electrical property of the particle, whether it's positive, negative, or neutral. Mass is just a measure of how much matter the particle has. Let's talk about the protons, neutrons, and electrons in terms of these 2 characteristics. We'll start with the protons. Protons are positive in charge and have a mass of 1 atomic mass unit. At the opposite end of the spectrum, you have the electron with a negative charge. The mass of an electron isn't 0, but it's very, very small, about 0.05% of a proton's mass. It's close enough to 0 for our purposes that we'll consider it to have essentially negligible mass. And finally, we have the neutron, which is neutral in charge and also has a mass of 1 atomic mass unit. But how are these particles arranged in atoms? The first thing to know is that each atom has an atomic nucleus, which is a collection of protons and neutrons at the center of the atom. This cluster of protons and neutrons is very small, very dense, and very positively charged. That just leaves the electrons, which exist in the electron cloud, or the space in which electrons move within an atom. Electrons move around the nucleus of the atom, kind of like how planets in our solar system orbit the sun. We'll talk more about the electron cloud later. We know how atoms are structured, but how do we know whether we're looking at an oxygen atom, a nitrogen atom, or a carbon atom? All we need to do is find the atomic number, the number of protons in the atom's nucleus. This number determines the atom's identity. For example, carbon's atomic number is 6, so we know that any atom with 6 protons in its nucleus is a carbon atom. An element is a substance that is composed of a single type of atom, such as oxygen, aluminum, and gold. These are all elements because each is only made up of one type of atom: oxygen atoms, aluminum atoms, and gold atoms. There are over 100 known elements, each with different properties and characteristics. Rather than memorize everything about these different elements, scientists have developed an organized and convenient reference chart of the elements called the periodic table. Each element is listed with its full name, symbol, atomic number, and atomic mass. The way these elements are organized into columns and rows tells us a lot about the individual characteristics of each element, too. That way, scientists can figure out different properties of these elements based simply on their location in the periodic table. Matter is any physical substance that takes up space and has mass, and an atom is the basic unit of matter. Atoms are made up of 3 smaller parts called subatomic particles, each of which has a characteristic mass and charge. A proton has a mass of 1 and a positive charge. A neutron is a subatomic particle with a mass of 1 and a neutral charge. Both protons and neutrons are found in the atomic nucleus, which is in the center of the atom. An electron is a subatomic particle with a negligible mass and a negative charge. Electrons surround the atomic nucleus in the electron cloud. The atomic number, or number of protons, gives the atom its identity. An element is a pure substance that consists of only one type of atom. For reference, all the elements are organized into a convenient chart called the periodic table.
We learned already that electrons are negatively charged subatomic particles. And we also talked about how the electrons are found in clouds around the nucleus. You've probably seen lots of symbols for atoms, something like this, where the electrons are represented as having specific orbits. What actually is the real case is that the electron cloud is a prediction of the area in which the electrons can be found. So if we have our nucleus here in the middle, and if we think about this circle here as being a three-dimensional sphere around the nucleus, we can say that at any given time, if I was to take a snapshot of this atom, that the electron would be found somewhere in this sphere or this cloud around the nucleus. But I couldn't be sure exactly where it is. It might be here, it might be here. And if I was to take different photos at different times, the electron would be in an unpredictable, non-random place somewhere in that cloud. So the electron cloud is just giving us a prediction of the area in which the electrons can be found. There's also energy associated with the electron clouds. So the electron clouds, they're also known as electron shells, and they're also known as energy levels. Let's just make a representation in 2D of an atom. And if we have our nucleus here, I can draw one energy level like this, and I can put an electron here. Now, think about subatomic particles in terms of their charge, right? Now, the electron has a negative charge, and the protons in the nucleus have a positive charge. So negative and positive things attract each other, right? If I had two powerful magnets here, and one end has a positive charge here, and one end has a negative charge, they're going to want to move together. Now, the fact that it's orbiting out here outside of the nucleus, that it's found out here away from the positive charge, means that it's moved away. It's moved farther away than that charge would probably prefer to be. If I was to take this, this negative magnet now and move it across the table, so now my negative magnet is over here and my positive magnet is over here. If these are really, really powerful magnets, I'm going to have to exert some force to be able to hold this negative magnet away from the positive one. Otherwise, it's going to rapidly move towards that positive magnet. So what I've created here now is something called potential energy. By moving this magnet away, I've created a scenario where the magnet wants to move. It wants to release this potential energy as kinetic energy, or it wants to move towards the positive magnet. So now, when I have my nucleus and I think about it in terms of these energy levels, we think about it in terms of energy levels because it took energy to have this electron out here at this orbital. So there's some sort of energy associated with this energy level. And if I have multiple energy levels, I'm going to have even more energy associated with this second energy level. So as I move out in energy levels away from the nucleus, the electrons that occupy these energy levels have more and more energy because they've been pulled farther and farther away from this nucleus. So if I was to talk about energy levels in terms of, say, the first energy level or the second energy level or the third energy level, there is more energy associated with energy levels as they increase in number. So energy is increasing as we increase the number of the energy levels. We can also talk about the energy levels in terms of the number of electrons that can occupy those levels. And I'll draw my first energy level. And it's referred to as 1s. So 1 for the number of the energy level. And the letter that's associated with the number is basically just defining the shape of the electron cloud. So it's not going to be important for our purposes to actually know what all the shapes are, but just be aware that that's what this is describing. So each kind of an orbital has a different shape. An s orbital has a specific kind of a shape. And in a moment, we'll talk about a p orbital, which has yet another kind of shape. Each orbital surrounding the nucleus can be occupied by 2 electrons. So let's draw our nucleus again, and I'll draw orbital 1. I can have an electron here, and I can have an electron here. Right? So this would be a full orbital. Right? And if I then go out to the second energy level, the second energy level is actually defined by 2s and then 3 kinds of 2p's for a total of 8 electrons. And just remember that this is a representation of an atom. But this is just for us to understand how many electrons we're talking about when we want to talk about atoms and how they interact with other atoms. And it's important to be able to represent the electrons because the electrons are going to define the reactivity of an atom with other atoms. And we talk about the electrons in the outermost shell. So in this case, let's say I have 1 electron shell and I have 1 electron in this orbital. This electron level is called the valence electron level. And the electrons in this orbital are the ones that are going to interact with other atoms. Now, atoms want to have full electron shells. So we talked about 1s as being able to hold 2 electrons. So this atom with only 1 electron in this valence level is unstable. And it wants to have a full valence level. Take another example here. If there's 2 electron levels here, if I had 1 electron out here in the second level, this valence level has 1 electron, right? So if I added another electron, that's not going to help me because at the second energy level, I want to have 8 electrons. So I would need to add 7 other electrons to fill this valence level. But I could also just remove this electron and end up with 2 electrons in this 1s level here. And that then would be more stable because this second electron level has been emptied. If we take a look again at the periodic table, we can now see how the organization of the periodic table is going to help us predict the reactivity of various kinds of atoms. So generally speaking, in biology, we're going to talk about the first, second, and the 13th through 18th columns here. And what you can see here is that the first, second, and then these other columns 3, 4, 5, 6, 7, and 8 are sometimes referred to as groups 1 to 8, signifying atoms that have 1 through 8 valence electrons. And all of the atoms are aspiring to be like the atoms in this 8th column because all of these atoms have 8 valence electrons and are most stable. And these atoms are referred to as noble gases. So we've learned a lot about electrons in this lesson. We've learned that the electron shell defines a space around the nucleus where the electrons are most likely to be found. We've also learned that this can be referred to as an energy level. And we've also learned that the electrons in the outermost energy level are referred to as valence electrons. And finally, we've learned a little bit about the organization of the atoms in the elements in the periodic table and how the atoms without a complete valence shell aspire to be more like the noble gases, which have a full valence level.
Look around you. Everything is made out of chemicals. The computer screen you're reading this on, the floor underneath you, your body, and even the air you breathe. All of these things contain atoms found in the periodic table of elements. These include hydrogen atoms, oxygen atoms, iron atoms, carbon atoms, and many more. But those atoms are not on their own, sadly moping about in space. No, instead they like to be friendly with each other and are connected together in all kinds of weird and wonderful combinations. These combinations are called chemicals, and the atoms in those chemicals are connected together by chemical bonds. A chemical bond is an attraction between two or more atoms and is what forms a chemical. This is an electrostatic attraction, an attraction between positive and negative charges. In each atom, there are positively charged protons in the nucleus and negatively charged electrons orbiting around the outside, and it's the interaction of these charges that causes atoms to stick together with chemical bonds. There are several types of chemical bonds, but the two most important and most common types are ionic bonds and covalent bonds. An ionic bond is an attraction between two atoms due to one transferring an electron, or several electrons, to the other. The electrons of an atom are arranged into shells, with 2 electrons in the first shell, 8 in the second, and 8 in the third. And atoms like to have full shells. If you have an atom that has just 1 electron in its outer shell, it wants desperately to lose that 1 electron so that it has a full outer shell. If you have an atom that has 7 electrons on its outer shell, it wants desperately to gain 1 electron. Both atoms feel incomplete. Put these two atoms together, and you get fireworks. One wants an electron, the other wants to get rid of an electron, so they transfer an electron between them. This makes the two atoms super happy, and just like in human relationships, when two atoms fit together so well, it's hard to tear them apart. The atom that gained a negative electron now has a negative charge, and the atom that lost a negative electron now has a positive charge. Since opposites attract, the two atoms stick together, and lo and behold, an ionic bond is formed. Ionic bonds tend to form between a metal and a nonmetal. This is because metals have a smaller number of electrons on their outer shell, and nonmetals have a large number of electrons. The shells of nonmetals are almost full, so metals can give their extra electrons away to them. A covalent bond is an attraction between two atoms due to the sharing of electrons between them. These kinds of bonds form when two atoms have mostly full outer shells. If these two atoms both have 7 out of 8 electrons in their outer shells, they can each share an electron. By doing this, each atom gains the 1 extra electron it needs to have a full outer shell of 8 electrons. Covalent bonds hold atoms together because the electrons sit in between the two atoms. The attraction between the negative electrons and the positive nuclei of the atoms is stronger than the repulsion between the nuclei of each atom. This is because the distance between the two nuclei is twice as large as the distance between the electrons and each nucleus. Since covalent bonds form between atoms with mostly full outer shells, they tend to form between two nonmetal atoms. There are two other types of bonds to talk about: polar covalent bonds and metallic bonds. Polar covalent bonds are covalent bonds in which the shared electrons are more attracted to one atom than the other and move closer to that atom. This makes one atom in the bond negatively charged and the other atom positively charged, which can hold the material together even more strongly. And, last of all, metallic bonds form between metal atoms because electrons are free to move inside of metals. Metals contain positive nuclei surrounded by a sea of free electrons. This sea of electrons holds the materials together in an unusually strong way, and is why metals can be so tough and hard to melt. A chemical bond is an attraction between two or more atoms that forms a chemical. Chemical bonds form because of attractions between charges, positive charges from protons in the nucleus and negative charges from electrons orbiting around the outside. There are 4 types of bonds we discussed in this lesson: ionic bonds, covalent bonds, polar covalent bonds, and metallic bonds. Ionic bonds form when a metal gives at least 1 electron to a nonmetal. This happens because both atoms want to have full outer shells. The metal wants to get rid of its extra electron, and the nonmetal wants to gain an electron to fill up its shell. When this happens, the metal becomes positively charged, and the nonmetal becomes negatively charged. These opposite charges make them bond, or stick, together. Covalent bonds form when two nonmetal atoms share electrons in order to fill up their outer shells. When this happens, they stick together because the electrons are attracted to the positive nuclei of both atoms. Polar covalent bonds are the same as covalent bonds, except that one atom attracts the electrons more strongly than the other. This causes an even greater attraction in the material, making it stronger. Finally, metallic bonds are bonds that form between metal atoms, and are due to the sea of free electrons that flows through the metal. This holds all the atoms together, making metal strong and hard to melt.
Let's say that you and a friend went away to a tropical island to go scuba diving. You spent your last day sitting on the beach and enjoying the sunshine, but your friend loved scuba diving so much that she went scuba diving on her very last day that the two of you had on the island. Then you got on a plane to go back home. Shortly after the plane took off, the plane had to land again because your friend was in a lot of pain. What happened? It turns out that our bodies are largely made of water. Over 60% of our bodies are made of water, in fact. So our blood, tissues, organs, and cells are essentially solutions or mixtures of solvent and solute, where solvent, the substance that dissolves other substances in it, is water. And there are many different solutes or substances that are dissolved in solvent. In the cases of our bodies, these are sugars, salts, proteins, and dissolved gases. Now, what affected your friend in this case is the solubility of nitrogen in water. Solubility is the maximum amount of a solute that will dissolve in a particular solvent at a specific temperature and pressure. Nitrogen gas is much more soluble in water at the pressure you would find underneath the ocean, which is much more than our atmosphere of air provides, than at sea level atmospheric pressure. And nitrogen gas is even more soluble in water at the pressures one would find in the ocean than at cabin pressure. Cabin pressure in airplanes is typically pressurized to about 5,000 feet or 1,500 meters above sea level. So that's even less pressure than at sea level. So if the pressure changes slowly over time, like divers normally do when they come up from a dive, the nitrogen dissolved in the blood will undergo diffusion over time, and everything will be totally fine. Now, diffusion in this case means the spread of molecules from areas of high concentration to areas of low concentration. So in this case, as your friend comes up from her scuba dive, the concentration of nitrogen molecules that are dissolved in her body is higher than that in the surrounding ocean and in the surrounding air. So over time, this nitrogen will leave her body to move toward areas that have lower concentration. So something similar to this is, let's say you put a little bit of salt in water and you just let it sit there. If you come back later, you'll find that the salt crystals are no longer there and the water tastes salty. This is because water dissolves salt into solution, and as the water dissolves the salt, the area immediately surrounding the salt crystals becomes very salty. And this causes the spread of sodium and chloride ions away from the salt that's dissolving, so that the solution can become more homogeneous. Now, provided your friend did everything right, she would end up with an unsaturated solution of nitrogen in her blood, just like we all have, just from breathing, because nitrogen makes up a significant portion of the air we breathe. An unsaturated solution is a solution that has less than the maximum amount of solute. Now, in the case of your friend, she probably followed the rules of scuba diving and came up slowly, but her blood was still saturated with nitrogen when you guys got on that airplane. Now, a saturated solution is a solution where the solvent has dissolved all of a particular solute that it can. So an example of this is, let's say you're making salt water. You can pour salt in and put salt in and keep putting salt in, but at some point, no matter how much you stir, that salt will not dissolve, and that is because you have a saturated solution of sodium chloride in water. So in the case of your friend, she has a saturated solution of dissolved nitrogen in her blood and in her tissues. As soon as you took off, you went to a higher altitude, about 5,000 feet or 1,500 meters at cabin pressure. All of a sudden, the solution had more dissolved nitrogen than it should have, and it became supersaturated. Now, supersaturated solution is one where there's more dissolved solute than there normally would be at an equilibrium for a saturated solution. Now, supersaturated solutions, as you might imagine, are really unstable. The solute will precipitate out at the slightest provocation. In this case, you went to a higher altitude, so the nitrogen started coming out of your friend's tissues way too fast and started to form bubbles in her veins. Now, this is incredibly painful and is actually called the bends because people bend over from pain. So, fortunately, you had a really good cabin crew and pilot, and they were able to land the plane, and your friend was able to go to the hospital. Now, your friend isn't feeling great, so they want to hydrate your friend, so they give your friend a solution of saline, or sodium chloride in water. Now, let's think about this for a second. You're concerned about your friend, and you want to make sure that it's the right kind of solution. Now, if we take a step back, we can think about our cells. Now, our cells have what are called semi-permeable membranes. Semi-permeable membranes are thin slices of material that contain tiny holes and can let solvent molecules, like water, through. And the process of solvent molecules like water moving across a semi-permeable membrane is called osmosis. Now, as we learned from diffusion, if there's high solute concentration in one place and low solute concentration in another place that some molecules can get to, the tendency is for these things to be equalized. So if you have a whole lot of solute in one place, for example inside a cell, and a lesser concentration of solute outside the cell, your solvent will start to flow into the cell to try to equalize the concentration of solute in the two solutions. And the amount of external pressure that is needed to equalize these two solutions is called osmotic pressure. Now, in this case, this makes us think, okay, we want our friend to stay pretty much the same. We want her to get well hydrated without having any problems where a whole lot of water flows into her cells or a lot of water flows out of her cells due to osmotic pressure. So we want a solution of sodium chloride that is isotonic or has the same concentration of solute as the solution in her body, which is typically her blood, so that she can become hydrated with no issues. Giving our friend an isotonic sodium chloride solution is much better than giving her one that's hypotonic, or a solution that has a lower concentration of solute than a given solution, such as the blood, because this would cause the influx of water into the body's tissues and potentially could cause some cell damage or cause cells to explode because there's just too much water going in. It's also better than giving your friend a solution that's hypertonic, a solution that has a higher concentration of solute than a particular solution. The difference in concentration of solute there could cause water to flow out of the cells, leaving them all shriveled up. Ideally, we want a solution that has the exact same concentration so that water can flow in and out without causing any disruptions. So to recap what we've learned today, we've learned that solutions consist of solute dissolved in solvent, that solubility is the amount of a substance that can be dissolved in a particular solvent at a particular temperature and pressure. We've learned that solutions can be saturated, that is, they can have the maximum possible amount of solute dissolved in their solvent, or they can be unsaturated, which is where they have less than the maximum possible amount of solute dissolved in them. We've also learned that solutions can be supersaturated, which is where they have more than the maximum amount that would be dissolved at saturation under normal conditions, but they're very unstable. We've learned that diffusion is the passive movement of substances from areas of high concentration to areas of low concentration. Osmosis is the diffusion of solvent across a semi-permeable membrane, such as our cell membranes. And we also learned that we should not fly within 24 hours of going scuba diving.
Water is the most important natural resource in the world. Without it, we humans wouldn't exist. Not only can we not go more than a few days without water, but life on Earth most likely would never have evolved at all. And then there are all the more trivial uses: cooking, bathing, washing clothes, brushing teeth, flushing toilets, and watering plants. Humans use a lot of water. Water is a compound containing molecules, each of which is made up of 2 atoms of hydrogen and 1 atom of oxygen bonded together. Across a large proportion of the Earth, it's in liquid form, which is convenient because humans need to drink it. Though at the poles it is a solid called ice, it can also evaporate to form water vapor. Water vapor then rises to create clouds, which eventually cause rain. On Earth, water literally falls from the skies. This little fact is something we're so used to that it may seem trivial, but it's a pretty big deal. Water is vital to human biology and indeed all biology on Earth. We currently believe that the presence of liquid water is an important requirement for the development of life, to the point that it's the main thing we're searching for in our attempts to find life elsewhere in the solar system. But why exactly is water important to human biology? There are 5 main functions of water in the human body: cell life, transport, chemical reactions, temperature regulation, and waste excretion. Cell life is based on the fact that cells have water inside them. Water is used to transport nutrients to cells and within cells, intracellular. If cells don't get nutrients including vitamins, minerals, and sugars, they die. As mentioned, water is needed for transport. It's how oxygen and food make it through our bodies and how blood flows through the veins and arteries to take those nutrients to where they're needed. Blood is, after all, 92% water. In fact, 60% of your entire body is water. Chemical reactions also require water. Chemically, water is an important solvent and a major part of a lot of chemical reactions that happen inside the cells of our bodies. You can think of the human body as a gigantic chemistry lab. Chemical reactions are how our food is turned into energy, how our waste is handled, how blood is oxygenated, and thousands of other processes. Water is also useful to control your body temperature. That's because water has a high heat capacity, meaning it takes a lot of energy to change its temperature. This makes releasing warm water the perfect way to cool off. That's why you sweat. When the water evaporates, it takes its stored heat with it. It also makes it harder for the environment to cause your core body temperature to change. Last of all, water is important for the excretion of waste. Those chemical reactions that happen in the body often produce waste products and toxins that need to be removed. We remove them through urine, which is mostly water. If your urine had the consistency of honey, it's fair to say you'd spend a lot more time in the bathroom. Water is a compound made of molecules containing 2 hydrogen atoms and 1 oxygen atom. That might not sound very impressive, but water is necessary for life on Earth. Without water, we wouldn't have evolved. In fact, humans cannot go more than a few days without it. Water is vital for human biology because it fulfills 5 vital functions: cell life, water carries nutrients to, from, and within cells; transport, the blood is mostly water, around 92%. Chemical reactions, water is a solvent and needed for the body to operate. Temperature regulation, sweat is mostly water because it has a high heat capacity. And waste excretion, urine is also mostly water. Because of this, water is the most important natural resource in the world.
What do you think of when you hear the word organic? Do you think of the baby carrots that you saw in the grocery store the other day? Or do you think of something entirely different? When we talk about organic foods, we're saying that they were grown using only natural fertilizers. Now, what does this have to do with chemistry? Well, it turns out that for a long time, chemists have been studying the molecules that make up living things. And for a long time, they called this organic chemistry. To them, organic chemistry was the study of molecules that came from living things and from formerly living things. These molecules included sugars, such as glucose, proteins and the amino acids that make them up, such as glycine, fats, and also hydrocarbons, such as methane, which you know is natural gas, and octane, which is a component of gasoline. Think high-octane gasoline, and you've got octane, which are molecules that came from things that used to be living. At some point, scientists decided that they were going to start making these types of molecules synthetically, so the old definition no longer fit. Organic chemistry could not just be the study of molecules that came from living things, but it needed to be a little bit broader. So the scientists went and looked at the different kinds of molecules that were found in living things and found that what they have in common is that they all contain carbon. This leads us to our new definition of organic chemistry, which is that organic chemistry is the study of molecules that contain carbon. Now, if all of these molecules were like methane and octane and contained only carbon and hydrogen, well, life wouldn't be very exciting. In fact, life probably wouldn't exist, since these molecules mostly sit around interacting with other like molecules unless oxygen and fire come along, in which case they burn and turn into carbon dioxide and water. Since we're not running around as flaming people, this means there must be a little more to the story, and it turns out this is true. While organic chemistry is the study of molecules that contain carbon, they also contain other atoms as well. So organic molecules can contain carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. And all of these atoms come together to make what we call functional groups. Functional groups are groups of atoms that we often find together that have a specific behavior. They're also a really good way to help you recognize different classes of molecules, as different types of molecules contain different functional groups. Now, one way that I like to think of this is it's sort of like you're going to go on an OkCupid date or a Match.com date. You've never met this person before. So you say, well, I need to remember what they look like. So you go and you look at their profile and you say, okay, this person whose name is Chemistry is awesome has glasses and brown hair, and they're about 5'8". So all of this information gives you some idea of what this person looks like and how you will recognize them. And that is exactly what functional groups do for us when we meet new biological molecules. So as we meet new biological molecules, functional groups can help us to recognize them because each of the different types of biological molecule has a characteristic set of functional groups. So we're going to go and meet the functional groups now. The first functional group we're going to meet is the alcohol or hydroxyl group. Now, looking at the name hydroxyl, you might think, gee, this thing probably contains hydrogen and oxygen. And if that's what you're thinking right now, it turns out that you're right. A hydroxyl group is an oxygen atom that is single bonded to a hydrogen and is also single bonded to a carbon-containing group. A common alcohol that you probably know about is ethanol, which can be used as a cleaner and is the alcohol that is found in alcoholic beverages. So a general picture of an alcohol is that it is some group that contains carbon attached to an oxygen that is single bonded to a hydrogen. And a lot of times, chemists use the shorthand R to talk about something that contains carbon. In some cases, it can be carbon or hydrogen. But in the case of an alcohol, it's always a carbon, because if it was not a carbon-containing group, then this would be water. The next functional group that I'd like to introduce you to is the amine. The simplest amine is called ammonia. It is often found in bathroom cleaners. It has a very simple chemical structure, which is a nitrogen atom that is bonded to 3 hydrogen atoms. It's quite small and quite volatile, and that's why it has a very pungent smell. More generally, amines are a nitrogen atom that is single bonded to 3 different groups that contain either hydrogen or carbon. So in the case of amines, an R group can be either hydrogen or carbon, and it doesn't matter. Amines are found in amino acids and are very important in biological systems because they're basic and they can accept protons from more acidic substances. The next functional group that I'd like to introduce you to is the ether, which is found in sugars. The ether group consists of an oxygen that is single bonded to 2 different carbon atoms. An example that you might be familiar with is diethyl ether, which was used as an anesthetic for a very long time. So if you've ever seen a movie where someone's going in for surgery and they put a cloth over their face, that contained diethyl ether and was meant to knock them out. A more general version of an ether would be just an oxygen atom with any 2 carbon-containing groups on either side of it. The next functional group is the carbonyl. A carbonyl is a carbon that is double bonded to an oxygen. A carbonyl-containing molecule that you're already familiar with is acetone, which is found in nail polish remover. Carbonyls are found fairly often in biological systems. A carbonyl-containing functional group is the carboxylic acid. Now, when you look at the word carboxylic, you probably see carb, as in carbonyl, and oxyl, as in hydroxyl. And it turns out this is the structure of the carboxylic acid. It consists of a carbon that is double bonded to an oxygen and single bonded to an oxygen that is single bonded to a hydrogen, which we know is a hydroxyl group. So it is a carbon that is both a carbonyl and bonded to a hydroxyl group. And these are found in amino acids and in fatty acids in our bodies. One that you might be familiar with, acetic acid, which is the major component of vinegar, and it's what gives it that lovely pungent smell. The last functional group that I'd like to introduce you to today is the ester. Esters consist of a carbon atom double bonded to an oxygen atom that is single bonded to an oxygen atom that is attached to a carbon atom. So it's sort of like you have a carbonyl where the carbon is also part of an ether. And we call this an ester. Esters are important in biological systems because they are what holds fats together, bonding fatty acids to glycerol to form mono, di, and triglycerides. In addition, esters are one of the things that make fruits smell really nice. We've reached the end of our lesson, and I hope that you've enjoyed learning a little bit about organic chemistry and also meeting some of the different functional groups that we'll be encountering as we meet new molecules. In this lesson, we're going to be talking about monomers and polymers. You may not know it, but polymers are something that you interact with every single day. You can find polymers in the plastic milk jug you may have drunk out of this morning, or in the fleece jacket you're wearing. You can find them in trees outside your window, and you can also find them inside your own body in the form of proteins. Now, what I mean when I say polymer is that a polymer is a long chain molecule that is made up of many smaller units, or is made from many smaller units. These smaller units are called monomer for single, and polymer means many, so a single unit can be combined with other single units to make a many-unit molecule, or polymer. In the case of the plastic milk jug we talked about, it's made of something called polyethylene. Polyethylene is made from a small molecule called ethylene. It contains 4 hydrogen atoms and 2 carbon atoms that are connected by a double bond. Now, what's really fascinating about this is it's amazing when you think about it, because ethylene is a gas at room temperature. So instead of just relying on ethylene molecules to get together and make something interesting for us, we take these molecules, cram them down into a small container, pressurize them, and then we add a catalyst to start the polymerization process. So the catalyst will cause a hydrogen atom to break loose, and this will go and interact with one of these ethylene molecules, forming a bond with one of the carbons. Carbon can only have 4 bonds, so the carbon-carbon double bond breaks, and then we'll go and find another ethylene molecule to form a bond with, and so on. So first it forms a dimer with another ethylene molecule, then the double bond between the carbons in that ethylene molecule break, and go and find another ethylene molecule to interact with, forming a trimer or 3-unit polymer, and so on until you end up with a very long polymer. Now, the reason that milk jugs stay together as well as they do is these molecules are incredibly long, so they can have really strong intermolecular forces between them. So if you consider something like Christmas lights, you put them together and then they come out as a big jumble, all intertwined with one another, and you can't pull them apart. That's what happens with these long chains of polymers. They all get wrapped around one another, and then they form very strong forces between the molecules that keep them from coming apart. Now, in this case, we showed a polymer that is made from a single monomer, but it's also possible to have polymers that are made from different monomers. One polymer that is made of different monomer units is nylon, which is a combination of hexamethylene diamine and adipic acid. In this case, the nitrogens in the hexamethylene diamine react with the carbonyl carbon of adipic acid, forming a byproduct of water and creating very long chains of alternating units of hexamethylene diamine and adipic acid. The same is true of Kevlar, which is a copolymer of 1,4-phenylene diamine and tetrathalloyl chloride. Those names aren't important, but what's important to recognize here is that polymers can be made of different types of monomers. The nitrogens in the phenylene diamine bond to the carbonyl carbon of the tetrathalloyl chloride, forming a byproduct of hydrochloric acid in the process. These molecules come together to form a very strong copolymer that is used in bulletproof vests. In addition to all of these man-made polymers, there are natural polymers as well. One example of a natural polymer is cellulose. Cellulose is a polymer that's made up of repeating monomers of glucose. And all of these glucose molecules, cellulose is one molecule that's responsible for helping hold plants upright. And finally, we have proteins. Proteins are another example of a natural polymer, and they have many possible monomer units because there are 20 different amino acids, which are the building blocks of proteins. They're formed by the reaction of a nitrogen on an amino acid with a carbonyl carbon of another amino acid, very similarly to the formation of nylon, forming peptide bonds. In this case, we can form a small polymer. The nitrogen on the alanine can bond to the carbonyl carbon of the glycine, leaving water as a byproduct. Similarly, serine can come along, and its nitrogen can bond with the carbonyl carbon on alanine with water as a byproduct, forming peptide bonds. Similar bonds can form among all of the amino acids, forming very large molecules that we call proteins. To recap, we've learned that polymers are long-chain molecules that are made from small single units that are called monomers. We've learned that plastics are a good example of polymers and can be made from single monomers or from different monomers, forming copolymers. Additionally, we've learned that there are natural polymers, such as cellulose, which is a glucose polymer found in plants, and proteins, which are made from the 20 amino acids.
In this lesson, we'll be talking about carbohydrates, which are also known as sugars. The word carbohydrate comes from the atom carbon and hydrate, or water, because the first carbohydrates that were discovered consisted of carbon, oxygen, and hydrogen atoms. To give you an idea of what these look like, well, first we'll introduce 4 sugars that you come across often in biology, and then we'll move on to discuss how they can come together to form larger polysaccharide or big sugar molecules. The first is glucose. Glucose is the sugar that serves as fuel for our bodies. Next to it is fructose, which is the sugar found in high fructose corn syrup. The next sugar is ribose, which plays an important role in holding together our genetic material. Next is deoxyribose, which is very similar to ribose, except that it lacks an oxygen or hydroxyl group on one of its carbons, hence deoxyribose. This is also an important sugar that helps hold our genetic material together. Now, if you look closely at these, what you'll see is that one thing all of them have in common is that they all contain an ether group. So they contain an oxygen that is single bonded to 2 different carbon atoms. They also contain several hydroxyl groups or OH groups. You'll also notice that in each of these molecules, there's one carbon atom that is single bonded to 2 different oxygen atoms, and this carbon is special. This helps us decide where we're going to start counting the carbon atoms on our sugar. The order of the carbon atoms is very important in sugars because this can tell us about how sugars are linked to one another when they form bonds. Using glucose as an example, I'll show you how to count the carbons. First, we locate the carbon that is bonded to 2 different oxygen atoms. Then we see how many carbons this carbon is attached to. If this carbon is at the end of a carbon chain and is only attached to one carbon, then this carbon becomes carbon number 1. If it isn't, then you move out a little bit further, and sometimes there's one other carbon atom there, and that can become carbon 1 in that case. Then we go around the ring. So moving on, we have carbon 1 with 2 oxygen atoms bonded to it. The carbon next to that is carbon 2. Carbon 3 is the next one to that. Carbon 4, the next one to that. Carbon 5, the next one to that. And since glucose has 6 carbons, carbon number 6 is the last carbon that we have for glucose. Now, sugars are super cool because not only can they exist on their own and have their own functionality, like glucose is a source of fuel for our bodies, they can also form bonds with other sugars and do other really cool things. Don't take it from me just because I'm a sugar chemist. Let's go and look at a nutrition label and look at the carbohydrate section and see how it breaks down. This label is for one large apple. It contains 130 calories, and by all means is pretty healthy since it has no calories from any kind of fat. If you look down the label though, you'll see that there are 34 grams of total carbohydrates, and this includes 5 grams of dietary fiber and 5 grams of sugar. So some of the sugars in this may be those monosaccharides or single sugars that we saw before, but some of these might be disaccharides, such as sucrose, also known as table sugar. Sucrose is a disaccharide formed by the linkage of glucose from carbon 1 to carbon 2 of a fructose molecule. This glycosidic linkage, or bond between 2 sugars, is what holds the disaccharide, or 2 monosaccharide sugars, together. So one really cool thing about glycosidic linkages, which are the bonds that hold sugars together, is that they're formed by dehydration, and that dehydration comes when two hydroxyl groups come together, leaving a carbon bonded to an oxygen bonded to another carbon, or ether, and water is a byproduct from that hydroxyl group and the other hydroxyl group. Now, carbohydrates can do things other than form disaccharides. They can form trisaccharides, or large sugars made from 3 different monosaccharide units, or they can form polysaccharides, which are sugars made from many sugar units. A glycosidic bond is a covalent bond that is formed when 2 carbon atoms belonging to 2 different monosaccharides are linked together. This type of bond is only present in various sugars and carbohydrates, and substances that contain these bonds are called glycosides. Glycosides are often categorized according to the elements which are involved in the chemical bond. While many glycosidic bonds simply involve carbon, oxygen, and hydrogen, some may also involve elements like nitrogen or sulfur. For example, the N-glycosidic bond has a carbon atom that is attached to 1 oxygen and 1 nitrogen. Glycosidic bonds are generally more stable than our sugars themselves because the ether group is very stable. Since glycosidic bonds are found in long chains of sugars, like the polysaccharides that involve 50 or more connected monosaccharides, they provide strength to these large sugars. Without glycosidic bonds, many of the structures that they create would be too fragile and would break apart. Since this type of bond is found in DNA, that could create some real trouble. The 2 major types of glycosidic bonds that form between monosaccharides are the 1,4-glycosidic bond and the 1,6-glycosidic bond. Each one of these types of bonds produces a water molecule and is therefore a condensation reaction. In the 1,4-glycosidic bond, the OH group attached to the first carbon of one monosaccharide reacts with the OH group attached to the 4th carbon of a second monosaccharide. In the 1,6-glycosidic bond, the OH group attached to the first carbon of one monosaccharide reacts with the OH group attached to the 6th carbon of another monosaccharide. The 1,4-glycosidic bond results in a linear chain, while the 1,6-glycosidic bond arrangement creates a carbohydrate that is branched. Now, we've already explained where a lot of sugar in fruit comes from, so why don't we look at some of those other things on the label? What we call dietary fiber is also known as cellulose, and it is an important structural material in plants. It's what helps blades of grass stand up straight, so you've seen it before. Now, one really interesting thing about cellulose is that it's a very long strand of glucoses all linked together, thousands and thousands of glucoses in one really long strand. Now, another interesting fact about cellulose is that what my grandmother referred to as roughage is something that cows really like to eat. This is really great because cows can get energy from this, while humans can't because they have little bacteria that live in their stomachs that help them to break down the cellulose and get energy from the glucose in it. Now, if we look again at our nutrition label, we see that we've already talked about sugars and we've talked about fiber or cellulose, but we haven't talked about that gap between the 34 grams of total carbohydrates in this apple and the 10 grams that we have accounted for. That's 24 grams of carbohydrates that has not been taken care of. It turns out that almost all of the rest of these carbohydrates are found in starch. What we think of as starch is actually 2 complex sugars, amylopectin and amylose, shown here as amylose. Starch is actually where we get a lot of our energy from. So if you think of an athlete who's doing a carbo load before a big race, usually you don't think of them sitting there eating pixie sticks and chocolate. You picture them sitting down to a big dinner full of pasta. And it turns out that that's because pasta also contains a lot of starch. And starch is really great because our bodies can digest it, unlike cellulose. But because these are really long sugars consisting of thousands of units, it takes our bodies a while to break them down. So you sort of get an extended release formula of sugar using starch. Now, there's a lot of starch and cellulose in plants because that's how they store their energy for future use. To store our carbohydrates, mammals use something called glycogen. It turns out that the chemical structure of glycogen is almost identical to that of amylopectin. It's a big long strand of glucose, and this sugar can be millions of sugars long, so maybe it's a little bit bigger than what you might find in plants, but it performs the same purpose, which is storing energy in the form of glucose for future use. I hope you've enjoyed meeting some different monosaccharides today, seeing how they can come together to form a disaccharide or 2-sugar unit, and also the polysaccharides that consist of many thousands or even millions of sugars that we can use for energy storage.
Lipids are biological molecules that are insoluble in water but are soluble in nonpolar solvents, meaning that they're nonpolar molecules. The lipids that we're most familiar with are probably dietary fats. So if you get a little hungry and you decide you want to eat some macaroni and cheese and you look at the nutrition label, what you'll find is that it contains 12 grams of fat per serving. It also contains 31 grams of carbohydrates and 5 grams of protein. So you have 12 grams from fat that give you 110 calories, and 36 grams of carbohydrates and protein combined, giving you 140 calories in each serving. So the fat gives you 9.2 calories per gram, while the carbohydrates and proteins give you only 3.9 calories per gram, which means that the fats contain over 2 times as much energy per gram. So this tells us that lipids and fats are good for storing energy. But what do they look like? Lipids fall into 2 categories. One is based on glycerol, and the other are steroids. First, we'll talk about the glycerol kind. Most dietary and storage fats are triglycerides. This means that they are made of glycerol combined with 3 carboxylic acids, which we call fatty acids. These form esters via dehydration. Now, to give you an idea of what this means, glycerol is a 3-carbon alcohol that contains 3 different hydroxyl or OH groups on each of the carbon atoms. You may also recall the carboxylic acids are molecules that contain a carbon atom double bonded to an oxygen atom, the carboxyl group, and that carbon atom is also bonded to a hydroxyl group, forming the carboxylic acid group. In the case of triglycerides, this glycerol combines with the 3 fatty acids or carboxylic acids to form esters. Now, esters are a functional group that consist of a carbon atom that is double bonded to an oxygen atom, and that same carbon atom is single bonded to an oxygen atom bonded to another carbon atom. And these are formed via dehydration or loss of water. Now, to get to know our triglycerides a little bit better, we can look back at the nutrition label. On the nutrition label, there are 3 types of fats. 2 of these are listed on the label, saturated fats and trans fats, and the rest are unsaturated fats that are not trans. Saturated and unsaturated fats are the kinds that we find in nature, while trans fats are synthetic. Saturated fats are triglycerides that have no double bonds in their carboxylic acid chains. Saturated fats are found in things like butter. Because their fatty acid chains are long and flexible, they can intertwine with one another. And because they're nonpolar, they're attracted to each other and not to polar things like water. So this is part of why butter is a solid at room temperature, is because of the molecular interactions between these different fatty acid chains. Unsaturated fats are triglycerides that have double bonds in their carboxylic acid chains. These are found in things like olive oil and can be monounsaturated, which means they contain 1 double bond, or they can be polyunsaturated, meaning they contain many double bonds. In unsaturated fats, the carbons on either side of the double bond are on the same side of the double bond. These double bonds kink and bend the chains of carbons, making it much harder for them to interact closely. It's sort of like if saturated fat chains are spaghetti and can wrap around one another very easily and stick together, these are like trying to fit together puzzle pieces that don't have the same edges, so you can't get them terribly close to each other. This is why substances in which there are many unsaturated fats, such as olive oil, tend to be liquids. Trans fats, the synthetic kind, are triglycerides that have trans double bonds in their carboxylic acid chains. What trans means is that the carbons on either side of the double bond are on opposite sides of the double bond. Trans fats are the byproduct of hydrogenating polyunsaturated fats. The goal of scientists was to make saturated fats, and these happened as a side product. The trans double bonds found in these fats make the carbon chain very rigid and straight, so you can get very close packing between these different carbon chains. It's sort of like if the saturated fats are like spaghetti and can wrap around each other to interact, there's still some room for movement. And trans fats are a little bit more like if you put a bunch of rods and stuck them together. There's not a whole lot of room for them to move. Because trans fats are able to pack so tightly together, they're able to become solid at higher temperatures. And this is part of why they're bad for us. If these trans fats are able to come together in our arteries, they may form blockages. Now to change topics a little bit, one of the things we use to get rid of fat on our dishes is soap. And soap works so well because it's made from things that look an awful lot like fatty acids on their own. They have a polar head group, much like the carboxylic acid, that is soluble in water, and long tails that can dissolve in nonpolar environments like fats. This way, the soap molecule can surround a blob of nonpolar lipid and help to pull it off of a dirty pan as water is washed over it, because the head group interacts with the polar water. We've talked about lipids as a storage mechanism, but they're also extremely important because they make up part of our cell membranes. The lipids that make up our cell membranes are phospholipids, and they're awfully similar to soap. They consist of glycerol attached to a phosphate group and to two fatty acids, or carboxylic acids. Basically, the phosphate group is very polar and can dissolve in water, while the carboxylic acid chains like to hang out with each other because they're nonpolar. What we end up with is a lipid bilayer, because the carboxylic acid chains like to hang out with each other, and both the inside and outside of the cell are polar aqueous or water-containing environments. Lipids can also serve as chemical messengers or hormones. These don't look much like the glycerol-based lipids we've seen so far, but they too are important nonpolar biological molecules. Most of them are steroids. Steroids get kind of a bad rep, but they're incredibly important. Sex hormones like estrogen and testosterone are steroids, but all that means is that they contain a particular system of carbon rings, as you can see here. To summarize, lipids are biological molecules that are insoluble in water but are soluble in nonpolar solvents. Lipids are incredibly good for storing energy and contain twice as much energy as the same weight of carbohydrates or protein, so they are the most energy-dense food. Most dietary and storage fats are triglycerides, which consist of a glycerol molecule combined with 3 carboxylic acids. These can be classified in 3 ways. Triglycerides can be saturated, which is when the fatty acid chains contain no double bonds in their carboxylic acid chains at all. They can also be unsaturated, which is when the triglycerides contain double bonds in their carboxylic acid chains. They can also be trans, which is where the carbons on either side of the double bond are on opposite sides of the double bond. We've learned that lipids make up part of the cell membrane in the form of phospholipids. We've also learned that lipids can serve as chemical messengers in the form of steroid hormones. Proteins are something that we've heard about because they're an important part of our diet. If we look at this nutrition label for macaroni and cheese, we can see that in addition to containing several grams of fat and carbohydrates, they also contain protein. Proteins are large biological molecules that are made of amino acids that are joined together by peptide bonds. To form proteins, many amino acids come together by dehydration to form peptide bonds, which is the bond between the carbonyl carbon of the carboxylic acid of one amino acid and the amino group nitrogen of another amino acid. Now, peptide bonds are incredibly strong. This helps proteins to do their jobs. An important role that proteins play in our bodies is that they provide structure. One example is the protein keratin. Keratin is a protein that is found in our hair and in the outer layer of our skin and in our nails. Keratin proteins come together and form filaments, which are long strands that are intertwined. If you think of a strand of rope that is made of many different smaller pieces of rope, it's made stronger by the fact that all of these different pieces are twined together, and that's exactly what happens in the case of keratin. Keratin is very hydrophobic and is insoluble in water. This hydrophobicity, or fear of water, is part of why keratin is so strong. The different strands of keratin protein are very attracted to one another, and less so to the outside environment, which helps them stay together. Another important structural protein is collagen. This is a main component of connective tissue, such as tendons and ligaments. And again, it's important for the structure that we find in our bodies. Another important role that proteins can play in biological systems is that they can act as enzymes. Enzymes are proteins that can make chemical reactions happen in our bodies. The way that they can make chemical reactions happen is that they decrease the activation barrier to making the reaction happen. So as an example of what I mean by an activation barrier, let's say your alarm goes off in the morning and you really don't want to get out of bed. It's really cold outside of your bed, which increases the activation barrier to you getting out of bed, or the amount of oomph it will take you to get out of bed. In the case of chemical reactions, we're talking about the amount of energy that it takes to make a certain reaction happen. So by decreasing the activation barrier, enzymes make reactions much more likely to occur. That would be similar to if somebody was making you coffee, or somebody put a nice warm rug down on the floor for you to step onto. Maybe that would decrease your resistance to getting out of bed. Now, enzymes can make chemical reactions happen, and these are really important in many places in our body. One example you might have heard of that sort of is outside of our bodies is that of lactate. Lactate pills contain an enzyme that splits apart lactose sugar. And this is really important because some people cannot digest lactose and it causes them indigestion. Adding the enzyme breaks apart the lactose sugar and helps these people's stomachs to feel better. Another important role that proteins play in our bodies is that they allow for movement. Two of the most important proteins that help us move are actin and myosins. Actin is a protein that is highly involved in cell motility and cell mobility. It's also important for our muscles to contract so that we can move or walk. Myosins, I say myosins because there are many different versions of this protein, are a motor protein, and they're also really important for making our muscles contract. Proteins can also transport things throughout our bodies. An example of this is hemoglobin. Hemoglobin is a protein that carries oxygen from our lungs to tissues throughout our body. And lipoproteins are proteins that can transport multiple fat molecules throughout our blood. Proteins are also able to act as messengers, taking messages from one part of our body to another. One example of this is insulin. Insulin is a protein that is able to lower our blood sugar. And when it's released from the pancreas by certain signals, it signals other cells to take glucose in and decrease our blood sugar. Similarly, human growth hormone is another protein that acts as a messenger. After it leaves the pituitary gland, it can travel around our bodies, bringing the message to grow or reproduce to our cells. Yet another important role that proteins can play is that they can protect us. Antibodies are large proteins that are able to recognize and attach to foreign substances within our bodies. So whether it's a cold virus that we've already had before and the antibody can recognize, or we're allergic to pollen and the antibody tries to attack that, antibodies are proteins that try to protect us. Finally, proteins are really important because they're able to regulate genes and other proteins. Sometimes they do this by cutting other proteins and activating them, or they can be proteins like transcription factors, which are able to bind certain pieces of DNA and decide whether they can be turned on or turned off. To summarize, proteins are large biological molecules that are made of amino acids and are joined together by peptide bonds. Peptide bonds are bonds between the carbonyl carbon of one amino acid and the amino nitrogen of another amino acid. Proteins are important because they can store energy, provide structure, act as enzymes to make reactions happen, can allow for movement, transport things, act as messengers, protect us from invaders, and regulate genes and other proteins.
The human body has thousands of different proteins, all of which are necessary for staying alive and healthy. Amino acids are the building blocks of proteins. There are 20 different amino acids in nature. All amino acids include 5 basic parts. 1. A central carbon atom. 2. A hydrogen atom. 3. An amino group consisting of a nitrogen atom and 2 hydrogen atoms. 4. A carboxyl group consisting of a carbon atom, 2 oxygen atoms, and 1 hydrogen atom. 5. An R group or side chain consisting of varying atoms. The R group side chain is what makes each amino acid unique. Each of the 20 amino acids has a different side chain structure. Side chains contain mainly hydrogen, carbon, and oxygen atoms. Some amino acids have sulfur or nitrogen atoms in their R groups. Each amino acid has a name, abbreviation, and unique side chain makeup. Some amino acids are considered essential amino acids, which are amino acids that our body cannot make on its own. We must get essential amino acids from the foods we eat. Non-essential amino acids are amino acids that our bodies can synthesize regardless of what we eat. To help you remember, you can think, it is essential that I eat food with essential aminos. In humans, there are 10 essential and 10 non-essential amino acids. However, arginine is kind of a special case. Adults can synthesize arginine, but infants cannot. So, arginine is an essential amino acid only for babies. Most proteins found in animals contain all essential amino acids, so it is easy to get them from your diet if you eat animal products. If you are a vegetarian, it is a little bit harder to get all the amino acids your body needs. This is because most plant proteins are missing a few essential amino acids. You will just have to eat a wide variety of different plants to make sure you get all essential amino acids. Amino acids are molecules used to build proteins. All amino acids have a central carbon atom surrounded by a hydrogen atom, a carboxyl group, COOH, an amino group, NH2, and an R group. It is the R group or side chain that differs between the 20 amino acids. Essential amino acids are those that your body cannot make. Your body can make non-essential amino acids. Think about all the food you ate today. It must come from somewhere, right? All of that food came from other organisms that used to be alive. We need to eat these other organisms because they provide energy for us to live. Because you need to eat other organisms to get energy, this makes you a heterotroph. Heterotrophs are also called other feeders, and because they need to consume energy to sustain themselves, they are also known as consumers. Some organisms are actually able to survive by making their own food. These organisms are called autotrophs. Autotrophs are also called self-feeders, and they are able to produce energy from sunlight and carbon dioxide, and are therefore known as producers. The only autotrophs that we know of are plants and some types of algae. This makes all other organisms heterotrophs. Not all plants are autotrophic. A few are actually heterotrophic. The European mistletoe is a parasitic plant, surviving off of a host plant. Other plants, such as pitcher plants, are carnivorous and feed on other organisms like insects. You are a heterotroph. Your dog, cat, bird, fish, etc. are all heterotrophs too, because you all depend on other organisms as an energy source. Other animal heterotrophs you are likely familiar with include deer, squirrels, rabbits, mice, and other animals you may see around your yard or a nearby park or forest. Bacteria are also heterotrophs as well as fungi. Fungi break down dead and decaying organisms, which makes them detritivores. Detritivores serve an important role in helping to recycle plant and animal material that is no longer living and return those nutrients to the ecosystem. It is important to note that not all heterotrophs rely on the same food sources for energy. Some heterotrophs eat only producers, like a gazelle eating grass. Some eat both producers and other consumers, like a bear eating game and berries. And some eat only other consumers, like a cougar eating a deer. Consumers that eat only producers are called herbivores. Those that eat only other consumers are called carnivores. And those that eat both producers and consumers are called omnivores. Any organism that can't make its own food is a heterotroph. Heterotrophs may eat food sources that are different from other heterotrophs, but as long as they are not able to produce their own food, they are other feeders. In contrast, autotrophs are able to produce their own food, and because of this, autotrophs often provide food to many heterotrophs. Most organisms on Earth are heterotrophs, even some plants, which are parasitic or carnivorous.
In a cell, nucleic acids are represented by two separate yet equally important forms: the DNA that stores information in the nucleus, and RNA that is used to translate that information into proteins. These are their stories.
Good day, ladies and gentlemen of the jury. My name is Miss Crimson, and I'm here to prove to you without a shadow of a doubt that my client, Colonel Custard, is not guilty of the most heinous crime of murdering poor Mr. Bones. The host of the International Cookbook Writers Convention was cruelly murdered with a lead pipe in a spiral staircase while on his way to deliver his award-winning cookbook to the kitchen. However, the defendant, Colonel Custard, was not responsible. The testimony of my expert witness will not only clear my client of all wrongdoing, but will also reveal the identity of the true killer of our poor departed Mr. Bones. Your Honor, I call to the stand Professor Pear, an expert in DNA structure and function. Professor, could you please explain to us what DNA is?
Why, yes, I'd be happy to tell you about DNA. DNA stands for deoxyribonucleic acid, and it's a fascinating molecule. It's one of two basic types of nucleic acids, the other being RNA, or ribonucleic acid. Nucleic acids are the molecules that cells use to store, transfer, and express genetic information.
That's very nice, Professor, but could you elaborate on the DNA molecule?
Yes, of course. It's the molecule which stores genetic information in an organism. It's essentially providing directions, like a recipe, if you will, for pretty much everything that makes us, well, us. If proteins are like the building blocks for structures, enzymes, and other cool things in a cell, DNA is like a recipe that tells a cell how to create those building blocks.
So you're saying that everyone has this DNA stuff inside his or her body?
Oh my, yes. DNA is an integral part of every organism, just like other types of organic molecules found in our body, like carbohydrates, lipids, and protein. But the structure of DNA is very distinct from those other types of organic molecules. Deoxyribonucleic acid gets its name from the fact that DNA possesses a sugar called deoxyribose. Now, that sounds like a fancy name, but really it's very easy to remember that it's a sugar like other sugars you may have encountered, such as sucrose and fructose. Just remember that all sugars end in OSE. A monomer of DNA is called a nucleotide.
Professor, can you please restate that definition in simpler terms?
Oh, I'm sorry. A monomer is just the most basic subunit of a complex molecule. That makes the nucleotide the most basic subunit of DNA, or more generally, of any nucleic acid. A nucleotide is composed of three things: a sugar, in the case of DNA, the deoxyribose I mentioned earlier, a phosphate group, and a nitrogenous base. As you can see in this diagram, the phosphate group and nitrogenous base are both attached to the sugar. When a nucleotide is part of a polynucleotide chain, it is often referred to as a base. Oh, did I forget to tell you what a polynucleotide is? A polynucleotide is simply a long chain of nucleotides. Poly means many, so polynucleotide literally means many nucleotides. Now you're probably wondering how these nucleotides are held together. Nucleotides in a polynucleotide molecule are held together by the bond between the phosphate group of one nucleotide and the sugar of a second nucleotide. A hydroxyl group, or OH, in the sugar and one of the oxygen atoms in the phosphate group form what's called a diester bond. For this reason, the bond between the phosphate group and the sugar in a polynucleotide molecule is called a phosphodiester bond. Linking nucleotides together forms a structure that looks like half of a ladder that was cut down the middle with an axe. The phosphate groups and sugars form the backbone of the ladder, while the nitrogenous bases provide the rungs. There are 4 different types of nitrogenous bases, and the order of those bases in a polynucleotide is what tells a cell how to make a structural protein, an enzyme, or any other number of important components of a healthy functioning organism.
Okay, Professor Pear, let me stop you right there. You've been throwing around a lot of fancy jargon. Before you go any further, let me see if I can summarize for the jury what you've told us so far. Nucleic acids are the molecules cells use to store, transfer, and express genetic or hereditary information. Nucleic acids can be categorized into DNA, or deoxyribonucleic acid, and RNA, or ribonucleic acid. DNA is the molecule which stores genetic information in an organism. A single monomer subunit of a nucleic acid is called a nucleotide. A DNA nucleotide is composed of a sugar called a deoxyribose, a phosphate group, and a nitrogenous base. A polynucleotide is a nucleic acid molecule consisting of a long chain of nucleotides. Nucleotides in a polynucleotide molecule are held together by a bond between the phosphate group of one nucleotide and the sugar of a second nucleotide, which is called a phosphodiester bond.
Previously on DNA and RNA.
The testimony of my expert witness will not only clear my client of all wrongdoing, but will also reveal the identity of the true killer of our poor departed Mr. Bones.
Nucleic acids are the molecules that cells use to store, transfer, and express genetic information. DNA stands for deoxyribonucleic acid. It's the molecule which stores genetic information in an organism. That makes the nucleotide the most basic subunit of DNA, or more generally, of any nucleic acid.
So, Professor, you told us that a DNA nucleotide consists of a phosphate group, a sugar, and a nitrogenous base. Can you tell us how nucleotide structure pertains to the case at hand?
Oh, yes. You see, you need to understand the chemistry behind DNA to fully appreciate the importance and function of the molecule. The phosphate group and sugar are the same in every nucleotide, but there are 4 different nitrogenous bases: guanine, adenine, thymine, and cytosine. They are often abbreviated by the first letter of each nitrogenous base: G, A, T, and C. They essentially function as a 4-letter alphabet. Or, if I may make an analogy to the case at hand, the information in DNA is like a recipe in one of our poor victims' cookbooks. Reading the DNA code ultimately tells a cell how to make proteins that it can use to perform various functions necessary for life. For instance, reading a specific sequence of DNA tells one cell how to make hemoglobin protein to carry oxygen molecules throughout the body. On the other hand, another cell might read a different recipe which tells it how to make insulin protein to control blood sugar levels. Oh, and reading or transcribing DNA is really an intriguing process.
Yes, Professor. I'm sure DNA transcription is very interesting, but let's stick to the basic characteristics of DNA that pertain to the trial at hand. You were telling us about the nitrogenous bases.
Oh, yes, you're quite right. The bases can be categorized into 2 different groups. The single-ring nitrogenous bases, thymine and cytosine, are called pyrimidines. And the double-ring bases, adenine and guanine, are called purines. Oh, yes, I guess you might wonder how I can remember that. But it's really quite simple. All gods are pure. Adenine and guanine are purines, and by process of elimination, that means cytosine and thymine have to be pyrimidines. See?
Yes, that's a very nice mnemonic aid. Adenine and guanine are purines, but we're getting off track. You were telling us why the chemical structure of nucleotides is important.
Oh, yes. The chemistry of nitrogenous bases is really the key to the function of DNA. It allows something called complementary base pairing. You see, cytosine can form 3 hydrogen bonds with guanine, and adenine can form 2 hydrogen bonds with thymine. Or, more simply, C bonds with G and A bonds with T. It is called complementary base pairing because each base can only bond with a specific base partner. The structures complement each other in a way like a lock and key. C will only bond with G, and A will only bond with T in DNA. Because of complementary base pairing, the hydrogen-bonded nitrogenous bases are often referred to as base pairs. Remember how I said that DNA polynucleotides look like half a ladder? Well, hydrogen bonding completes the ladder. Since the nitrogenous bases can hydrogen bond, one polynucleotide can bond with another polynucleotide, making the nitrogenous bases the rungs of the ladder. Each polynucleotide participating in this ladder is often referred to as a strand. Because the bases can only fit together in a specific orientation, a parallel orientation between the strands won't work. The strands must be anti-parallel or upside down relative to one another.
What do you mean, anti-parallel?
Well, remember that the backbone is made of phosphate groups and sugars. Therefore, each strand will always have a phosphate at one end and a sugar at the other end. Rather than having to refer to the phosphate or sugar end, scientists simply refer to the ends of the DNA by the closest carbon in the sugar ring. Since the carbons in the sugar are numbered 1 to 5, the sugar end of the strand is called the 3' end, and the phosphate end of the strand is called the 5' end. Remember that complementary base pairing works like a lock and key, so there's only one orientation in which hydrogen bonding will work. If you try to orient the two strands parallel to each other, the sugar end of the polynucleotides are both at one end, and the phosphate groups are at the other end. However, the nitrogenous bases can't hydrogen bond in this orientation. The key can't fit into the lock. For hydrogen bonding to work, the two DNA strands must run in opposite directions. The 3' end of one strand can hydrogen bond with the 5' end of the other strand. If we represent the strands as arrows, with the arrowhead at the 3' end of the strand, we can see that the strands in a DNA molecule are organized anti-parallel relative to each other.
Okay, let me stop you again, Professor, so I can summarize your testimony for the jury. There are 4 nitrogenous bases found in DNA that are called guanine, adenine, thymine, and cytosine. They are abbreviated by the first letter in their name, or G, A, T, and C. The bases can be divided into 2 categories. Thymine and cytosine are called pyrimidines. Adenine and guanine are called purines. Each nucleotide base can hydrogen bond with a specific partner base in a process known as complementary base pairing. Cytosine forms 3 hydrogen bonds with guanine. Adenine forms 2 hydrogen bonds with thymine. These hydrogen-bonded nitrogenous bases are often referred to as base pairs. Because of the alternating nature of the phosphate groups and sugars in the backbone of nucleic acids, a nucleic acid strand has directionality. The end of a nucleic acid where the phosphate group is located is called the 5' end. The end of the nucleic acid where the sugar is located is called the 3' end.
Finally, DNA strands are antiparallel, meaning that the strands in a DNA molecule are parallel but are oriented in opposite directions. Essentially, the 5' end of one strand pairs with the 3' end of the other strand.
Complementary base pairing describes the manner in which the nitrogenous bases of the DNA molecules align with each other. Complementary base pairings are also responsible for the double helix structure of DNA. If you imagine yourself looking at a closet with 4 pairs of shoes that are all mixed up, complementary base pairing would be the set of rules that you would use to know which shoes go together. In a similar way to which only the correct 2 shoes will form a working pair, you don't want 2 left shoes or a black and a brown one. Only some of the nitrogenous bases of DNA can interact to form a stable DNA molecule. DNA, or deoxyribonucleic acid, is an amazing molecule that stores all of the genetic information of an organism. You might have heard that DNA is a polymer. That is to say, a molecule made of the many smaller subunits called monomers. In the case of DNA, the monomers are called nucleotides, and the polymer is usually called a polynucleotide. Poly means many in Greek. There are 4 different kinds of nucleotides that make up DNA, each with one of 4 possible nitrogenous bases: adenine, A; cytosine, C; guanine, G; and thymine, T. You can think of polynucleotides as strings of words consisting of combinations of just 4 letters, A, C, G, and T. If you were to read a polynucleotide molecule, it would read something like AGTCGCCTAGGC, etc. When people first realized that DNA contained all of our genetic information, they became very interested in understanding how it was organized and structured. While it was known that the DNA molecule contained 2 polynucleotide molecules, no one knew how they were organized together. Some people believe that they interacted through the phosphate part of the nucleotides, while others thought that they interacted through the bases. You can think of the DNA molecule being a zipper, with each DNA strand represented by 1 strip of fabric. If you had never seen a zipper and someone gave you the 2 strips separated, how would you know if they normally attached to each other through the metal teeth, the bases in our case, or through the straight side of the fabric strip, the phosphates? In 1953, Watson and Crick showed that the DNA molecule consists of 2 polynucleotide molecules that stand face to face against each other, interacting at the level of the nitrogenous bases. If we go back to our zipper analogy, the 2 strands in DNA attach to each other through the nitrogenous bases in a similar manner to which the 2 strips of fabric in the zipper interlock to each other through the small metal pieces. While Watson and Crick worked on the problem of the structure of the DNA molecule, Erwin Chargaff, an Austrian biochemist who immigrated to the U.S. during the Nazi era, was studying the actual composition of DNA. It was known that DNA consisted of A, G, T, C. But what Chargaff found was that the proportion of each of these bases in DNA was not random. In fact, across different animal species, from sea urchin to salmon and so on, he found that the number of A's in the DNA was always equal to the number of T's in the DNA, and the number of G's was equal to the number of C's. This rule, %A equals %T and %G equals %C, is known as Chargaff's parity rule 1, and played a key role in Watson and Crick's understanding of the structure of DNA. While working on the structure of DNA, Watson and Crick not only figured out that the 2 polynucleotides in the DNA, i.e. the DNA strands, interacted through the bases, they also deduced, with the help of Chargaff's rules, that the bases were pretty picky about whom they interacted with. Nucleotides containing adenine would only interact with nucleotides containing thymine, while nucleotides containing cytosine would only interact with nucleotides containing guanine. This meant that only two kinds of base pairs were possible, GC or CG and AT or TA. So the two strands of DNA are said to complement each other. This is what we call complementary base pairing. Complementary base pairing explains Chargaff's findings. Since adenine always associates with thymine, the number of A's and T's in DNA must always be the same. You can remember which base associates with which by using the mnemonic GCAT. One of the cool consequences of complementary base pairing is that, if we know the nucleotides that are present in one DNA strand, we actually already know the nucleotides present in both strands. Try it. If one strand of DNA contains the following nucleotides, A, C, G, T, T, A, then what will the opposite strand have? Pause the video here and write down your answer. If you deduced TGCAAT, then you were right. Companies, hospitals, and doctors take advantage of this information whenever they want to study a person's DNA. They will sequence only one strand and from it deduce both. Just think of all the time and money this saves during testing and research. How is it that the bases interact in the DNA to form its characteristic double helix structure? Well, the bases are interacting through hydrogen bonds, which are a form of weak chemical interaction taking place between hydrogen and an atom of negative charge or polarity. Examining the base pairs in DNA, researchers figured out that there are 3 hydrogen bonds between C and G, and only 2 between A and T. This difference is due to the actual shape and composition of each of the nucleotides, but has consequences in the stability of the DNA. Because the interactions between C and G are stronger, a DNA molecule with a higher proportion of CG over AT will tend to be more stable than one of the opposite composition. In fact, researchers can look at a DNA sequence and predict how easy or hard it would be to separate the two strands using this property. If you continue to learn about genetics, you will see how complementary base pairing plays a key role in many important cellular processes such as the duplication of the DNA, the synthesis of RNA, knowing what amino acids to use to synthesize proteins, etc. DNA is an extraordinary molecule containing all of the genetic information necessary to make an organism. It consists of 2 strands or chains of nucleotides, which arrange themselves in space in such a manner that the nitrogenous bases A and C are always opposite to T and G respectively. This means that only 2 kinds of base pairs are possible, GC or CG and AT or TA. This specific arrangement of the nitrogenous bases is known as complementary base pairing and is a key principle of DNA organization.
More than 100 years ago, scientists knew that proteins came in complex shapes and sizes and were used in the cell for many purposes. They knew that there were many different amino acids that could be combined to make a variety of complex protein molecules. Thus, they assumed that the hereditary material, which they knew must also be varied and complex, must be a protein. Even when DNA, deoxyribonucleic acid, was discovered, many people believed that it was not important because it was only made up of 4 different nitrogenous bases: adenine, thymine, guanine, and cytosine. It seemed less complex than proteins that were mixtures of 20 amino acids, so its importance was largely unrecognized. Frederick Griffith identified a process called transformation in 1928. He was studying a type of bacteria that infects mammals, called Streptococcus pneumoniae. He had 2 strains of this bacteria, a smooth strain, S, that caused disease, and a rough strain, R, that was relatively harmless. The S strain has a capsule made of polysaccharides that protects it from the mammal host's immune system, but the R strain doesn't have this capsule and is attacked and killed. Griffith did several different experiments where he injected the bacteria into mice. The mice lived and were healthy when injected with just the live R strain or just the heat-killed S strain, and they died if given a live S strain, all of which was to be expected. But the mice also died when given a mix of heat-killed S strain and live R strain. But why? The live R strain was harmless, so something had to have happened to make it virulent. Upon examination, the dead mice had both live R and live S strain in their blood. Griffith concluded that the live R strain had somehow been transformed by the heat-killed S strain. In this transformation, information could be transferred from a dead cell to a live cell, in this case, from the heat-killed S strain into the R strain. It had to be some sort of hereditary material, but at this point, he didn't know what. Oswald Avery published a paper in 1944 that stated that the transformation factor was DNA. Avery and his colleagues isolated and removed various organic compounds from the S strain bacteria and tested the compounds that remained to see if they caused the transformation. Through the process of elimination, they showed that proteins and RNA did not cause transformation, but DNA did. This indicated that DNA was the hereditary material. This finding was not very popular, as little was known about DNA. Most scientists assumed that the genetic material was a protein, and were not convinced that the hereditary material would be the same in simple, single-celled organisms as it was in more complex organisms, like mammals. However, Alfred Hershey and Martha Chase demonstrated conclusively in 1952 that DNA is the genetic material using a bacteriophage, a virus that infects bacteria, that is essentially made of just protein and DNA. When the infected bacteria were separated from the remaining materials, only the DNA was detected, showing that the bacteria had incorporated DNA, but not the protein. James Watson and Francis Crick published a paper in 1953 titled Structure for Deoxyribose Nucleic Acid. Thus, they are credited with discovering the structure of DNA. They would go on to win the Nobel Prize in Medicine with physicist Maurice Wilkins in 1962. Watson and Crick devised a model of the structure of DNA based on the experimental data from several different sources and laboratories. It was known from the late 1800s that DNA, first called nuclein, existed as a molecule through the work of Friedrich Miescher. Phoebus Levine started the first investigation of the structure of DNA in the early 1900s, including identifying its nucleotides. In 1944, Erwin Chargaff had discovered that the bases adenine and thymine were always present in a 1-to-1 ratio, as were the bases guanine and cytosine. Rosalind Franklin had taken high-resolution X-ray images of DNA fibers that indicated a helical, corkscrew-like shape. The time was ripe for someone to put all this information together. It became somewhat of a race. Now, Rosalind Franklin had been working in a lab with Maurice Wilkins, and they did not get along. Wilkins took some of Franklin's unpublished data and images on the structure of DNA to Watson and Crick. Adding this new data to the other existing information known about DNA, and using cardboard cutouts of the bases, Watson realized that hydrogen bonds could form between A and T and between G and C. To reconcile Franklin's X-ray data and Watson's model of the relationship between the bases, Crick realized that the DNA strands had to be oriented anti-parallel to one another. This led the two of them to postulate the double helix structure of DNA. Rosalind Franklin's contribution to this discovery went largely unrecognized. She died in 1958, and in 1962, Watson, Crick, and Wilkins were awarded the Nobel Prize. If a helix is a cylindrical spiral, a double helix is basically just two cylindrical spirals. Think of DNA as a ladder with the long sides made of phosphate groups and sugars, and rungs made of nitrogenous bases held together by hydrogen bonds. Then, twist the ladder around an imaginary central axis, so the structure of the molecule looks a little like a spiral staircase. Just like the English alphabet has 26 letters, DNA has an alphabet with 4 letters, nucleotides, that can be used and reused to combine into many long words or strands. The order of the nucleotides determines which protein, if any, is produced from transcribing a sequence. Even though there are only 4 nucleotides, the chance of 2 non-identical twins having the same DNA is almost impossible. Most living things have more than one copy of each gene, one from the parent egg and one from the parent sperm, but only one copy is expressed at a time. The genes can be rearranged during fertilization, producing yet another variant in the offspring. By a series of experiments in the 1900s, DNA was shown to be the hereditary material for living organisms. This discovery encompassed the work of many researchers spanning multiple decades. Friedrich Miescher discovered the DNA molecule in the late 1800s. Phoebus Levine identified the nucleotides in DNA in the early 1900s. Friedrich Griffith discovered the transforming principle. In transformation, information is transferred from a dead cell to a live cell. Oswald Avery identified the transforming factor as DNA. Alfred Hershey and Martha Chase confirmed that the hereditary material was DNA by using bacteriophages. Rosalind Franklin used X-ray imaging to show the helical structure of DNA. Watson and Crick put it all together, and in 1953 proposed the double helical structure of DNA. DNA functions as the hereditary material in the nucleus of cells. Genes are made up of DNA. Its shape is a double helix, or 2 cylindrical spirals. Its long sides are made of phosphate groups and sugars, and its rungs are made of nitrogenous bases held together by hydrogen bonds.
Previously on DNA and RNA. Today we know that permanently changing the characteristics of an organism can be accomplished by changing its DNA content. James Watson and Francis Crick devised a model of the structure of DNA based on the evidence produced by several different laboratories at the time.
I contest that my client is innocent of the murder. In fact, based on the DNA evidence, we have reason to believe that Mr. Teal murdered Mr. Bones in the staircase with a lead pipe.
Professor, you said that you found DNA evidence at the scene of the crime. However, you said nothing about RNA evidence. Didn't you say that there are 2 types of nucleic acids? What about this ribonucleic acid, or RNA? It seems like you've conspicuously avoided talking about RNA altogether. Is it because the lack of RNA evidence directly links Colonel Custard to the crime?
That's an excellent question. I would be remiss to talk about nucleic acids and only talk about DNA. RNA is, in fact, the second of the 2 types of nucleic acids. However, there are a number of structural differences between the 2. First, let's address the name. Like DNA, RNA is a nucleic acid composed of a sugar, a phosphate group, and a nitrogenous base. One difference between DNA and RNA is the sugar. Whereas the sugar in DNA is deoxyribose, the sugar in RNA is ribose. Now, I won't dwell on the exact chemical difference between the 2 sugars, but ribose has 1 extra hydroxyl group compared to deoxyribose. Second, there are 4 different nitrogenous bases found in DNA and RNA. However, there is one difference. The bases found in DNA are guanine, cytosine, adenine, and thymine. The bases found in RNA are guanine, cytosine, adenine, and uracil. Uracil forms 2 hydrogen bonds with adenine and functions just like thymine does. It's simply used in RNA instead of thymine. Finally, unlike DNA, which is double-stranded, RNA is single-stranded.
Please answer the question, Professor. Knowing that RNA is structurally different than DNA is interesting, but not really relevant to this murder trial.
But it is. You see, I needed to explain the structure of RNA so you could better understand the function of RNA. DNA, RNA, and protein are functionally linked together in a concept known as the central dogma. Remember that DNA houses recipes to make different biological molecules. However, this information is not accessed directly from the DNA. Instead, a copy of the recipe is made in the form of RNA. This copy of the recipe can then be read to make a protein. The central dogma states that DNA makes RNA and RNA makes protein. At each step, a cell translates the information between the different molecular languages. That is, DNA language is transcribed into RNA language at the first step, and RNA language is translated into protein language at the second step. Three major types of RNA play a role during the journey from DNA to protein, although the functions of each type of RNA are different. One type of RNA is called messenger RNA, or simply mRNA. mRNA is created when the DNA recipe is copied in the first step of the central dogma. The information found in mRNA can be interpreted by using two other forms of RNA in the second step of the central dogma. mRNA is translated into protein at a cellular structure known as the ribosome. A second type of RNA helps form the structure of a ribosome. This type of RNA is called ribosomal RNA, or rRNA. Remember that DNA and RNA differ slightly at the nucleotide level. Therefore, the process of transcribing DNA into RNA not only changes the information from a double-stranded into a single-stranded molecule, but also changes all the thymine bases into uracil ones. Proteins are made of amino acids, so the formation of any protein requires assembly of a chain of amino acids. Transfer RNA, or tRNA, molecules ferry amino acids to the ribosome for this assembly. As you can see, there are many types of RNA performing all kinds of interesting jobs. In fact, why don't I tell you about why we believe RNA actually preceded DNA?
Professor, please get to the point. You've laid out very nicely that RNA plays a number of key roles in translating the information in DNA into protein. But you have yet to provide an adequate explanation to account for the absence of RNA evidence in your testimony.
Oh, right, I'm sorry. Sometimes I just get carried away talking about nucleic acids. One of the major roles of RNA in a cell is to make proteins, and proteins carry out many cellular functions in biology. However, it's inefficient for the cell to maintain a constant level of mRNA and protein.
Explain for the jury, please, Professor, what all this really means.
Think of the electrical devices in your room. Let's say that I keep my computer on 24 hours a day because I want to be able to do an online search whenever I feel like it. There's a cost to that practice. First, I'm going to have to pay for the electricity to power the computer. Second, let's also say I've decided to leave my monitor, printer, speakers, and a number of other devices plugged in as well. So many, in fact, that I'm using up all the electrical outlets in the room. Now, if I want to plug in a new electrical device, I will need to turn off one of my devices before I can plug in a new one. I may be able to surf the internet faster, but it may be at the cost of the setup time to use another device, like, say, a hairdryer. If I use a lot of other electrical devices besides the computer, I might not be saving myself that much time in the long run if I constantly have to turn off the computer to plug other things in. An alternative strategy would be to keep all of the electrical devices off, both to conserve energy and to minimize the startup time for using any one electrical device. The same conservation strategy applies to a cell. If RNA were a very stable molecule, it might tie up a lot of resources in a molecule that isn't being used. For instance, yeast consumes sugar for energy. Although there are many different types of sugar that a yeast may encounter in its environment, it makes sense to only express the RNA and protein to consume the available type of sugar, rather than waste energy maintaining every RNA and protein required to break down every type of sugar. For a variety of structural reasons, mRNA has a very short lifespan compared to DNA. There are many hydrogen bonds holding a DNA molecule together. While the bases in an RNA molecule can hydrogen bond with each other, usually far fewer bonds can form compared to a DNA molecule. Fewer hydrogen bonds means a less stable structure. Second, the extra hydroxyl group in the ribose sugar of RNA makes RNA more reactive than DNA. A reaction between that hydroxyl group and another molecule could destroy the RNA molecule. Finally, proteins that degrade RNA are found everywhere. If you consider all of these differences in stability between RNA and DNA, you can see why RNA is harder to isolate from a crime scene. However, even if we had isolated any RNA, consider the information we would gain from comparing mRNA samples to DNA samples from the same person. According to the central dogma, mRNA is merely a temporary copy of the corresponding piece of DNA. Simple sequence analysis would yield the same results, albeit with uracil instead of thymine.
Ladies and gentlemen of the jury, I ask you to find my client, Colonel Custard, innocent of murder. The prosecution's main argument against the DNA evidence in this trial was the absence of RNA evidence. I believe Professor Pear has satisfactorily demonstrated that while RNA, or ribonucleic acid, is a nucleic acid like DNA, it is both structurally and functionally distinct from DNA. Ribose is the sugar found in RNA instead of deoxyribose like DNA. Uracil is the nitrogenous base found in RNA that bonds with adenine instead of thymine that is found in DNA. Like thymine, uracil forms two hydrogen bonds with adenine. The central dogma tells us that DNA makes RNA and RNA makes protein. Three major types of RNA are mRNA, or messenger RNA, that serve as temporary copies of the information found in DNA, rRNA, or ribosomal RNA, that serve as structural components of protein-making structures known as ribosomes, and finally tRNA, or transfer RNA, that ferry amino acids to the ribosome to be assembled into proteins. My witness has told you that RNA is inherently unstable, and the central dogma states that mRNA is merely a copied version of DNA, so lack of RNA evidence is irrelevant. The absence of my client's DNA at the crime scene and the presence of Mr. Teals should exonerate my client. Listen to the evidence and find my client not guilty. I rest my case.
Transfer RNA, or tRNA, is a member of a nucleic acid family called ribonucleic acids. RNA molecules are comprised of nucleotides, which are small building blocks for both RNA and DNA. tRNA has a very specific purpose: to bring protein subunits, known as amino acids, to the ribosome, where proteins are constructed. One of the discoverers of DNA, Francis Crick, first suggested the existence of tRNA. At the time, scientists knew that genetic information was kept in the nucleus as DNA, and that DNA carried the instructions on how to make proteins. DNA doesn't leave the nucleus, though, so our cells make a copy of the DNA called messenger RNA, or mRNA. mRNA leaves the nucleus and is bound by ribosomes, the molecular machines that act as the factory that makes proteins. Scientists understood that while DNA and RNA have almost the same alphabet, proteins are very different. Francis Crick proposed that there must be a small molecule capable of translating mRNA into proteins. Other scientists proved his theory with the discovery of tRNA. The job of tRNA is to read the message of nucleic acids, or nucleotides, and translate it into proteins or amino acids. The process of making a protein from an mRNA template is called translation. How does tRNA read the mRNA? It reads the mRNA in 3 nucleotide sequences called codons. Each individual codon corresponds to an amino acid. There are 4 nucleotides in mRNA. If you do the math to figure out how many codons exist, you arrive at 64, or 4 cubed. There is one tRNA molecule for each and every codon. Interestingly, there are only 21 amino acids. This brings up the idea that our genetic code is redundant. That is, we have 64 codons, but only 21 amino acids. How do we resolve this? More than one codon can specify for an amino acid. This table shows all of the combinations of nucleic acids or codons, as well as which amino acid is specified by which codon. As you can see, not every amino acid has 4 codons. In fact, methionine only has 1. Notice, however, that each codon has only one corresponding amino acid. Thus, we say that the genetic code is redundant, but not ambiguous. For example, the codons GUU, GUC, GUA, and GUG all code for valine, redundancy, and none of them specify any other amino acid. No ambiguity. So we now know that the job of tRNA is to bring an amino acid to the ribosome. We also know that each codon has its own tRNA, and that each tRNA has its own amino acid attached to it. Further, we know that the job of tRNA is to transport amino acids to the ribosome for protein production. The tRNA doesn't become part of the protein, which suggests that tRNA can either be attached to an amino acid or free. We call this charged or uncharged. How does this work? Briefly, an uncharged amino acid goes to a pool of amino acids. Here it finds the one specific amino acid to which it can attach and binds it, or becomes charged. Then it carries the amino acid back to the ribosome, where the amino acid is transferred from the tRNA to the growing protein. At this point, it's important to remember that amino acids are added to the protein in a specific order. The order is contained in the code of mRNA inside the ribosome. How does the tRNA know if it's carrying the right amino acid? That is, the amino acid that needs to be added next according to the codon sequence of the mRNA. Easy. tRNA have anticodons as part of their sequence. An anticodon is the reverse sequence of a codon. In this, imagine you can see a black region at the bottom of a blue loop. This is the anticodon sequence. The anticodon sequence also tells the tRNA which amino acid it can attach to at the pool. The amino acid is attached to the yellow tip of the tRNA. Anticodon sequences work just like codon sequences. Thus, each amino acid has more than one anticodon that recognizes it, but each anticodon only recognizes one amino acid. In eukaryotic cells, tRNA are made by a special protein that reads the DNA code and makes an RNA copy, or pre-tRNA. This process is called transcription, and for making tRNA, it's done by RNA polymerase III. Pre-tRNA are processed once they leave the nucleus. First, some nucleotides are cut off, and then the remaining 73 to 94 nucleotide long string is folded into the shape you see in this image. Transfer RNA, or tRNA, is a specialized RNA molecule that brings amino acids to the ribosome for protein production. In eukaryotic cells, tRNA is made by a special protein that reads the DNA sequence and makes an RNA copy, or pre-tRNA. Pre-tRNA is processed once it leaves the nucleus. tRNA's role involves multiple steps. First, it reads messenger RNA, or mRNA, in 3 nucleotide sequences called codons. Using this information, the tRNA finds the one specific amino acid to which it can attach and binds it, carrying the amino acid back to the ribosome. The amino acid is then transferred from the tRNA to the growing protein, and tRNA's job is finally complete.
Making a snowman is always a big ball of fun. 3 snowballs full of fun to be exact. And each snowball starts out small, getting bigger as you roll it in white fluffiness. Let's imagine that you go outside in a fresh snowfall to build one of these guys, but you don't have the best suited yard for rolling snowballs. In fact, your backyard is one giant hill. This hill is fantastic for sledding, but not so much for making snowmen. Anyways, in order to make a giant snowball for the snowman's bottom, you'll need to actually push this snowball up over the hill and then down the other side. You do it because it's fun, but make no mistake, it takes a lot of energy to get started up this hill, and this is hard work. Right now, you share a quintessential problem with some chemical reactions within a cell. These chemical reactions might be a lot of fun when you're in a cell, but it takes a lot of energy to get them started. This is called the activation energy, or the energy required for a reaction to start. You can think of activation energy as that energy you needed to muster up in order to push that snowball up and over the hill in your backyard in order to maximize the size of your snowball and get it to the other side. What your cells have to help overcome a problem of high activation energy are called enzymes. Enzymes are proteins that lower the activation energy of a reaction. In doing this, enzymes increase the rate of a reaction, helping it to occur faster. However, enzymes are not consumed in a reaction, they simply help it to occur. They do not add snow to your snowball, they just make it easier to make a snowball. This makes enzymes reaction catalysts. The presence of enzymes is analogous to decreasing the size of your hill in your backyard. Enzymes make things easier for your cell and help chemical reactions occur. There are hundreds of different kinds of enzymes in your cells, which all participate in different types of reactions. Enzymes can break molecules apart, build or add molecules, and even rearrange them. In lowering the activation energy of a reaction, enzymes decrease the barrier to starting a reaction. It's important to note, however, that the change in energy remains the same between the start and end of a chemical reaction. For example, if a reaction released 200 kilojoules of energy without an enzyme, the same reaction would still release 200 kilojoules of energy with some enzymatic aid. The difference would only be a lower activation energy and a faster rate of reaction. In other words, if enzymes were helping you build a snowman, you'd be done quicker, but you'd have just as much fun. Most of us are fortunate enough to be able to enjoy a grilled cheese sandwich and hot chocolate when we take a break from building our snowman outdoors in the cold. This is because our cells contain an enzyme called lactase. It's interesting to note and to remember that many enzyme names end in the suffix ase, A-S-E. Lactase breaks down lactose disaccharide sugars into glucose and galactose. Lactose, of course, is the sugar found in dairy products. Some individuals are lactose intolerant and they lack this enzyme. Without lactase, lactose will break down on its own, but is an extremely slow process, maybe even taking days. Lactase speeds up the rate of this reaction, allowing us to digest lactose. So let's talk about how it's done. Lactase starts its reactions by binding to its substrate. A substrate is the substance or molecule on which an enzyme functions. So the substrate for lactase is lactose. Together with enzymes, substrates form an enzyme-substrate complex. Enzymes like lactase are block-like, globular proteins with pockets. These pockets contain the active site, which is the area of an enzyme where the substrate binds and the chemical reaction takes place. In the active site, amino acids of the enzyme protein will bind to the substrate. The substrate fits perfectly into the active site of an enzyme, meaning that enzymes are specific for their substrates and not any others. Lactase cannot break down any other disaccharide besides lactose. Sucrose, for example, simply does not fit into the active site. When binding to a substrate, enzymes may undergo an induced fit. This is the change in enzyme shape to accommodate and bind to a substrate. Although it's true all substrates are specific for their enzyme, you can imagine this is just like a little tweak to the enzyme-substrate complex to make the reaction occur. This change does not need to be drastic. It might be analogous to cradling a baby and then snuggling the baby into a position closer to your body for a better hold. In summary, enzymes are proteins that lower the activation energy of a chemical reaction. Enzymes are not consumed or produced in a reaction, but they are catalysts that allow the reaction to proceed at a faster rate. There are hundreds of enzymes in each cell that all have specific roles and work on specific substrates. Together, they form the enzyme-substrate complex. Substrates bind to a perfectly matched pocket in the enzyme known as the active site. Sometimes we like things just right. If it's too hot, we swelter under the sun, and if it's too cold, we shiver. If there's too much to eat, we hit a point where we can't swallow another bite. but when we're hungry, there never seems to be enough food in the house. It's not that we're nitpicky, right? It's just that we're human, and after all, we know what we like, and we know what conditions suit us best. We are not the only ones who value the perfect conditions. Plenty of animals and other organisms do too. And on a microscopic level, so do our cells. Even within our cells, enzymes, the proteins that catalyze chemical reactions, like things just right too. As a quick review, an enzyme works on a substrate, or substance or molecule on which an enzyme functions. An active site is where a substrate binds an enzyme in order to facilitate a reaction. Each enzyme works on a specific substrate in a specific condition. These conditions are optimal for the enzyme, just like how the perfect amount of sunshine in food keep us satisfied and happy. If you had 2 cookies in front of you, you could eat 2 cookies. And if you had 5 cookies in front of you, you could eat 5 cookies. However, maybe there are 100 cookies in front of you. Could you eat 100 cookies in one sitting? Probably not. And at some point, you'd become full or saturated, and you wouldn't be able to eat one more cookie at this time, no matter how many more are put in front of you. Enzymes, well, they don't work on cookies, but they work on substrates. Therefore, the substrate concentration in an enzyme's environment has a major effect on how much work can be done. There's only so much substrate an enzyme can work on, just like there's so many cookies that you could possibly eat. Let's say that one enzyme works on one substrate at a time. Let's also say in a cell there are 10 units of one type of enzyme, but only 4 substrates for the enzyme to work on. In this case, all the substrates will bind to an enzyme and the reaction rate will be low. If you add a few more substrates in, say like 20 at a time, then the reaction rate will increase until the point where all the enzymes are currently busy catalyzing reactions. The reaction rate will increase until all the enzymes available are working on substrates. This is shown on this graph by the highest point of the curve. At this point, the reaction rate will plateau and reach its maximum. Here, the enzyme has reached a point of saturation. This point of saturation is when more substrate will not increase the rate of reaction. The rate of reaction will not increase with a higher substrate concentration unless the amount of enzyme also increases. Just like it affects us, temperature also affects enzyme function. Enzymes work optimally at a specific temperature. Again, different enzymes might have different optimum temperatures. Colder weather slows much of life down, and cold temperatures will also slow the reaction rate of enzymes. When things heat up, molecules move faster, similar to how when we go outside or run around when the weather starts to warm up in the spring. An increase in temperature usually means more movement and a greater likelihood that molecules collide. Higher temperatures can increase the rate of reaction between an enzyme and its substrate, but only to a point. This temperature point of maximum function is called an enzyme's optimum temperature. As you can probably guess, the optimum temperature for enzymes in our body is actually our body's temperature. If the temperature gets too high, it can distort the enzyme, making it unable to function properly. In fact, if temperatures got too high, enzymes can become denatured or undergo a structural change in shape that inhibits function. If the change in shape affects the active site, then an enzyme may no longer recognize the substrate. With more enzymes becoming more non-functional, the rate of the reaction decreases with an increase in temperature. Enzymes are also affected by pH. Let's do a quick review of pH. Remember that pH measures how acidic or basic a solution is. Acidic solutions have a pH below 7 and contain more hydrogen ions. Vinegar and orange juice are examples of acidic solutions. Basic solutions have a pH higher than 7 and contain more hydroxide ions, such as bleach and baking soda. Neutral solutions have a pH of 7, having an equal concentration of hydrogen and hydroxide ions. The wrong pH can interfere with an enzyme's bonds and shape, possibly changing its active site. Here the enzyme can also be denatured. Each enzyme works at an optimum pH. The wrong concentration of hydrogen and hydroxide can denature an enzyme and change the rate of reaction. Different enzymes operate optimally at different pH. For example, your stomach is an acidic environment. You probably know that from an occasional heartburn. Pepsin is an enzyme secreted by cells in your stomach. It works in this acidic environment to help you digest your food. Pepsin works really well in your stomach, but it's inactive in other parts of your body. And that's a good thing, because you wouldn't want pepsin getting to digest the rest of you as it makes your way into the other parts of your digestive system. In summary, enzymes work optimally in specific conditions that maximize their reaction rate. Reaction rate will increase with higher substrate concentration until it reaches a point of saturation, where all available enzyme is working on substrate. Reaction rate is also optimum at a specific temperature and specific pH. At extreme temperatures and extreme pH, an enzyme might be denatured or undergo a structural change in shape that makes it unable to recognize its substrate or catalyze a reaction. Sometimes we get by with a little help from our friends. There are some tasks like going to the store for groceries that are perfectly easy to do by yourself. Some tasks like moving a large piece of furniture into your house and up a few flights of stairs, well, friends are good to have in situations like this, particularly strong ones who will lift anything for some free pizza. We all rely on other people at certain times in our lives, and so we shouldn't be surprised that the same thing can happen in a biological process. For example, enzymes benefit from a few close friends too. Remember that enzymes are proteins that catalyze chemical reactions. In other lessons, we've learned how they functioned. In this lesson, we'll learn about who functions with them, or rather, who helps them catalyze these reactions. Some enzymes need helpers or partners, and some don't. There are different types of enzyme helpers too, with different enzymes requiring different helpers or like different kinds of friends. The first type of enzyme partner is a group called cofactors, or molecules that increase the rate of reaction or are required for enzyme function. Cofactors are not proteins, but rather help proteins such as enzymes, although they can also help non-enzyme proteins as well. Examples of cofactors include metal ions, like iron and zinc. A specific type of cofactor, coenzymes, are organic molecules that bind to enzymes and help them function. The key here is that they're organic. Organic does not mean you'll find them in a special aisle in the grocery store. Rather, organic molecules are simply molecules that contain carbon. Don't let the name coenzymes fool you either. Coenzymes are not really enzymes. As the prefix co suggests, they work with enzymes. Many coenzymes are derived from vitamins. These molecules often sit at the active site of an enzyme and aid in recognizing, attracting, or repulsing a substrate or product. Remember that a substrate is the molecule upon which an enzyme catalyzes a reaction. Coenzymes can also shuttle chemical groups from one enzyme to another enzyme. Coenzymes bind loosely to enzymes, while another group of cofactors do not. Prosthetic groups are cofactors that bind tightly to proteins or enzymes, as if holding on for dear life, they are not easily removed. They can be organic or metal ions and are often attached to proteins by a covalent bond. The same cofactors can bind multiple different types of enzymes and may bind enzymes some loosely as a coenzyme and others tightly as a prosthetic group. Some cofactors may always tightly bind their enzymes. It's important to note, though, that these prosthetic groups can also bind to proteins other than enzymes. An enzyme that requires a helper group really isn't complete without it. Once attached, it's called a holoenzyme. A holoenzyme is an enzyme with any metal ions or coenzymes attached to it that is now ready to catalyze a reaction. There are examples of cofactors, coenzymes, and prosthetic groups in many biological processes. For example, cellular respiration occurs in all of your cells, which is a process that converts food into energy. The details of this process will be saved for other lessons, but you can imagine it as a long series of events that need to occur, like steps in a relay triathlon, except with way more than 3 events. Enzymes are the proteins that perform many of these steps in cellular respiration, passing the biological baton from the cold water swim to the bicycle race. We won't go over the individual events here, but let's take, for example, the steps that convert pyruvate into acetyl-CoA. Don't be mesmerized by the chemical names. What's important here is the example. This one step is carried out by a complex of 3 different enzymes and uses 5 different partners. These enzyme partners bind to the enzyme active site. Some help move chemical molecules from one step to another. 4 out of the 5 of these partner molecules are derived from different B vitamins, meaning they are organic compounds and therefore coenzymes. Some of these are bind tightly to the enzyme tricomplex, making them also prosthetic groups. You get B vitamins from foods such as eggs and milk and certain veggies. You can see why these vitamins are so important. They are used to drive these molecular helpers to carry out just one step of an essential cellular process that gives you energy throughout the day. Just think of that the next time you have an omelet. Your friends are helpers that move your furniture, but our bodies derive different types of helpers from the food you eat that allow specific enzymatic processes to take place. It's hard to imagine that doing something so simple as eating could have such a complicated effect. In this lesson, we've learned about some of the help an enzyme can get to perform its reactions. Cofactors is a term broadly given to molecules that increase the rate of reaction or are required for enzyme function. Metal ions are usually cofactors. Coenzymes are a specific type of helper or partner that are organic molecules required for enzyme function that bind loosely to an enzyme. They are often, though not always, derived from vitamins. Prosthetic groups are enzyme partner molecules that bind tightly to an enzyme. Enzymes facilitate important reactions in a cell. If an enzyme were a baker and he were to bake non-stop, the mounds of bread loaves and cupcakes would be fantastic until the point when there was no room in the kitchen left for baked goods. Then the living room would fill up, followed by the rest of the house. Then bagels would be pushing through the windows and spilling onto the grass. Enough is enough, right? There is such a thing as too much of a good thing. So let's say you stopped the baker from baking by taking away the oven. Then over time, the baked goods are eaten or sold. A holiday comes by and you realize another few hundred cookies would be a pretty good idea. Thank goodness it's quite easy to ask the baker to start up again. He's happy to oblige. After all, he is a baker, and this is what he loves to do. Enzymes don't usually have free reign of the cell and their substrates either. They're not allowed to run amok in a cell, baking up a storm. Instead, they are carefully regulated. This regulation can be positive or negative and serves to make sure an enzyme is working when it needs to and not working when it's no longer needed. An enzyme activator is a molecule that positively regulates an enzyme's activity. Many enzymes require activators to begin or continue a process, recognize a substrate, or reach their maximum reaction rate. On the flip side of enzyme activation is inactivation. This is achieved by an inhibitor, or molecule that binds to an enzyme and disrupts its activity. Activators are the green light of enzyme regulation, while inhibitors are the red light of enzyme regulation. Neither activators nor inhibitors are substrates. These regulators can be proteins or other molecules. Inhibition can be either competitive or non-competitive and is often reversible. Remember that in a normal enzymatic interaction, an enzyme will recognize and bind to a substrate in order to catalyze a reaction. It will then release the products. Competitive inhibition is the interruption of an enzyme's ability to bind to a substrate due to a different molecule binding to the active site. Competitive inhibitors bind with the active site. They may or may not catalyze a reaction. All competitive inhibitors prevent the substrate from binding the enzyme, literally competing or fighting for the use of the active site. In our enzyme as a baker example, this would be similar to a dinner chef competing with the baker for the kitchen. An example of competitive inhibition is used in medical treatments. Your cells contain an enzyme called alcohol dehydrogenase that converts alcohols into other chemicals. Methanol is a type of alcohol found in antifreeze, and not one that we normally ingest. Methanol competes with other alcohols for the active site of alcohol dehydrogenase. Therefore, if antifreeze or other methanol-containing substances are accidentally ingested, alcohol dehydrogenase will recognize and bind to this substrate. In this case, a reaction is catalyzed, but methanol is converted to a toxic chemical called formaldehyde, which can cause blindness. Competitive inhibition can be used to stop this from happening. Intravenous ethanol can be given to a patient, which will then compete with the methanol for the active site of alcohol dehydrogenase, thereby decreasing the amount of formaldehyde that is made. In a different type of inhibition, there is no competing for the kitchen. Instead, in non-competitive inhibition, a non-substrate molecule binds to an enzyme to disrupt enzyme activity, but does not bind the active site. In this example, a non-competitive inhibitor binds to a different area of the enzyme. This often causes the enzyme to change shape, and may make the active site unrecognizable to a substrate. If the substrate can no longer bind, the reaction cannot take place. To our baker, this might be like shutting off the oven so the bread can no longer be baked. A real-life example of non-competitive inhibition could happen right in your backyard. Hopefully you know to avoid picking and eating wild mushrooms. It's difficult to distinguish wild mushrooms that are safe to eat from the ones that are poisonous. What makes some of these fungi toxic? A molecule called alpha-amanitin can be found in a genus of mushrooms. Alpha-amanitin binds to the enzyme RNA polymerase II. RNA polymerase II performs an important process in your cell called transcription, which makes RNA from DNA. Alpha-amanitin does not bind the RNA polymerase II active site, but the location it binds to interrupts RNA polymerase II activity, making it slower. The amount of transcription decreases until mRNA is no longer made. Transcription is an essential process. Alpha-amanitin is a non-competitive inhibitor that prevents transcription and causes cell death. In summary, in today's lesson we learned about how enzymes are regulated. Enzymes can be regulated by an activator, which is a protein or molecule that positively regulates enzyme function, or an inhibitor, which is a protein or molecule that negatively regulates enzyme function. Two types of reversible inhibition include competitive inhibition, or the inhibition due to non-substrate binding to an enzyme's active site and blocking substrate binding, and non-competitive inhibition, or the inhibition due to a non-substrate binding to an enzyme at a location other than the active site, which blocks enzyme function. You're probably familiar with what happens to a bottle of Italian dressing when it sits out on the counter for too long. The oil floats in a layer on top with a watery layer underneath, and if you don't shake it up, you're likely to end up with an oily mess all over your salad. Oil is a hydrophobic substance. Hydro comes from the Greek word for water, and phobic is literally the fear of it. Although oil is not exactly scared of water, it does run away from it and huddles together so that the least amount of oil is touching water at any time. This gives you the two separate layers of oil and water that you see in Italian dressing. Anything in that watery layer of the salad dressing is hydrophilic. Hydrophilic means a love of water. It comes from the Greek hydro for water and philia, a love for it. Of course, hydrophobic and hydrophilic substances are not unique to your salad dressings. One of the most important examples of a substance that's both hydrophobic and hydrophilic interacting in nature is actually within all of our cells. Our bodies and all living things on Earth are made up of one or more cells, the basic fundamental unit of life. Our bodies are composed of trillions of cells. Some belong to our bodies, like skin cells and heart cells, and some don't, like the single-celled bacteria that are living in all of our guts. All cells, however, have a few things in common, and one of these things is the need to create a barrier between the outside world and the inside of the cell. Plant, animal, and bacterial cells alike do this by having a cell membrane that separates the intracellular environment from the extracellular environment outside the cell. Although some components of the cell membrane are different, one of the most uniformly important and conserved components of a cell membrane is an important molecule that has a dual personality. It's both hydrophobic and hydrophilic. Behold, a phospholipid. A phospholipid is named for its two main parts, a phosphate group and a lipid. It's usually drawn with the phosphate group as a circle, and this is referred to as the hydrophilic head. Due to its negative charge, this phosphate group is also polar. The phosphate group and the lipid are connected by a glycerol group. Most phospholipids have two lipid tails made of hydrocarbon fatty acids bound to the glycerol by an ester linkage. These hydrophobic tails are nonpolar. So what do you think would happen if we took a couple of these phospholipids and threw them into our bottle of Italian dressing? Birds of a feather flock together, and hydrophobic molecules like to stick together too. So do hydrophilic molecules. Therefore, the hydrophobic tails of a phospholipid would want to stay with the oil layer of the salad dressing, while the hydrophilic head of the phospholipid would be attracted to the water layer. It would look like the phospholipid was doing a handstand in the salad dressing. Cell membranes have a phospholipid bilayer surrounding the entire cell. Bi meaning there are two layers of these hydrophobic and hydrophilic phospholipids. Having two layers of phospholipids is an extremely significant characteristic of the cell membrane. How is a phospholipid bilayer organized in the cell membrane? Let's think about what happened when we dropped a few phospholipids into the bottle of salad dressing. What would happen if we organized a phospholipid bilayer inside this bottle? What would it look like? Well, in a bilayer, the hydrophilic heads of the phospholipids will want to be touching water at all times. So the first layer will form a ring with the heads facing water. This leaves the hydrophobic tails touching the inside of the circle. However, the intracellular environment of a cell is aqueous or water-based, so the tails aren't going to like this. How can we account for it? Well, that's why a second layer of phospholipids is so important. A second layer of phospholipids forms an inner ring, with the hydrophobic tails touching each other and the hydrophilic heads oriented towards the outside and inside of the membrane. This is a phospholipid bilayer, which allows for an aqueous environment both inside and outside the cell, but still creates a barrier between the cell and its surroundings. This barrier is both protective and selective against foreign objects, but it's not completely impermeable. In other lessons, we'll discuss methods of transporting molecules across this cell membrane. The arrangement of the phospholipid bilayer is essential to cell organization and creation of the cell membrane, which separates the intracellular environment from the extracellular environment. Although different organisms can have slightly different phospholipids or other cell membrane components, they share a common structure. The hydrophilic phosphate heads like water, so they touch the inside and the outside of the cell where the environments are aqueous. The fatty acid tails form a hydrophobic region in the middle, which is free of water. This cell membrane helps in structuring the cell and also controls which substances can cross it.
A cell is the basic fundamental unit of all life on Earth. It contains numerous important structures and components that function together, with all the parts interacting with each other. The cytoplasm is the intracellular space within the cell, containing all these structures and components. Cells are often likened to a tiny metropolis. Like any town, a cell needs to create boundaries and to protect itself. Not much inside it is static or stationary, including the cell membrane that surrounds it. All cells have a fluid but defining cell membrane that encircles the cytoplasm, like an old city would have a fluid protective moat. In this way, a cell membrane would provide a barrier to separate the cytoplasm and extracellular space. Things are brought in and shipped out, but it would be dangerous to let just anything pass through it without the right credentials. Therefore, we also describe a cell membrane as selectively permeable, or only allowing certain substances to pass through the cell membrane. A cell membrane, however, is made up of a few more sophisticated parts than a moat, and is constructed from entirely different materials. This cell membrane is primarily composed of a phospholipid bilayer, cholesterol, proteins, and carbohydrates. Though the parts are different, it's easier to think about how the membrane functions by, again, comparing it to a protective moat. The composition of the cell membrane is best described by the fluid mosaic model, where different parts of this membrane can float in a fluid-like space. Like it sounds, this model suggests that the cell membrane is dynamic or moving and asymmetrically scattered with different pieces, such as phospholipids, cholesterol, proteins, and carbohydrates, and that these different parts of the cell membrane float in this fluid-like space, moving laterally throughout the membrane. Let's start to build what this mosaic looks like around a cell. We'll start with one of the most popular components of the cell membrane, the phospholipids, which form the phospholipid bilayer. As a refresher, this is two layers of phospholipids that give us a membrane with hydrophilic heads oriented towards the outside and inside of the cell where there is an aqueous environment. The fatty acid hydrophobic tails of the phospholipids don't like to get their tails wet, so they face each other, creating a hydrophobic region inside the bilayer. A function of this layer then is to create a distinct hydrophobic region, which is essential in both keeping fluidity and moderating what can pass the selectively permeable membrane and enter our miniature city. In later lessons, we'll talk about who's allowed through and when. Now we can start adding molecules to the phospholipid bilayer that will turn this membrane into a beautiful piece of mosaic art. We'll start with cholesterol, which is found floating throughout the membrane. Remember that not only is there a lot of cholesterol in a double cheeseburger, there's also a good amount in our cells. Here it does a lot more good than clogging our arteries. Like phospholipids, cholesterol is also a hydrophobic molecule too, So where would it want to spend time? Because alike molecules typically hang out together, hydrophobic cholesterol is also located within the hydrophobic region of the cell membrane created by the phospholipid tails. Cholesterol resides within the hydrophobic space and helps maintain membrane fluidity. It both provides structure and keeps the membrane from freezing. In addition, cholesterol helps the membrane act as a protective barrier to unwanted molecules. Cell membrane fluidity refers to the viscosity or resistance to fluid flow of the phospholipid bilayer. In more common terms, viscosity is the thickness or stickiness of the fluid layer. This fluid is important for several reasons, which include transport of proteins and lipids through the bilayer, even distribution of cell membrane during cell division, maintaining cell shape and homeostasis. Both cholesterol and fatty acids influence the fluidity of the cell membrane. Nearly a third of the entire cell membrane is composed of cholesterol. This cholesterol is crucial for maintaining cell membrane fluidity during fluctuations in temperature. The cholesterol molecule contains a polar hydroxyl group which pulls phospholipids together with rising temperature. However, when the temperature is lowered, the phospholipids are then kept further apart. This enables the cell membrane to continue functioning at a wider range of temperatures. Cholesterol also prevents the diffusion of molecules that are water-soluble through the membrane. Fatty acids also affect the fluidity of the cell membrane. Fatty acids can be either saturated, having no double bonds, or unsaturated, double bonds are present. Saturated fatty acids have a straight configuration, increasing the rigidity and decreasing the fluidity of the cell membrane. On the other hand, unsaturated fatty acids have a more crooked or bent form, which causes them to increase cell membrane fluidity. Longer fatty acid chains also create more membrane rigidity. Let's keep adding to our magnificent mosaic. Proteins are another important part of the cell membrane. There are two types of membrane proteins, integral and peripheral. These proteins are so named for the space they occupy relative to the cell membrane. Integral proteins are membrane proteins that integrate themselves throughout the bilayer, where they can even cross both sides to touch the inside and outside of the cell. In order to have this special power, these proteins also have hydrophobic domains that span the hydrophobic regions and hydrophilic domains that touch the hydrophilic areas inside and outside of the cell. Peripheral proteins are membrane proteins on the periphery or outside of the phospholipid bilayer. What do all these different proteins do? Proteins perform a lot of different jobs in the cell membrane and come in all shapes and sizes, perfect for making a mosaic. They provide structure to the cell membrane, act as recognition sites for other proteins, participate in cell-cell communication, and help transport substances across the membrane. Now we have one last piece to add to our picture, carbohydrates. Not only are carbohydrates delicious in some fresh baked bread, they are an important part of the cell membrane too. Carbohydrate chains can be found on the outside of a cell membrane, attached to lipid or protein molecules. They are molecules attached to lipids or proteins that act as binding sites or help cells adhere to other cells. In summary, all of these pieces come together to build this beautiful mosaic cell membrane, which encloses the cytoplasm of the cell. Remember that the cytoplasm is the inside of the cell, and all its structures enclosed by a cell membrane. It is a dynamic structure best described by the fluid mosaic model, which states that cell membrane components float in a fluid-like space. The cell membrane is composed of a phospholipid bilayer as the background canvas for this mosaic, which is then embedded with cholesterol, proteins, and carbohydrates that float and move laterally within this bilayer like objects in water. These components aid in keeping a fluid-like structure to the cell membrane, maintaining a barrier between the cytoplasm and extracellular environment, recognizing cell-cell communication signals, and providing transport.
Cells have a pretty sophisticated cell membrane, which acts as a barrier to the outside world. We've described this membrane as selectively permeable, meaning not just anything can get through it. The key to this phrase is that the cell membrane is selective, but not impermeable. This is something like how you would keep your home. The walls of your house create a boundary and define the space, but there are still doors and there are windows through which you can let in your friends or some fresh air on a summer's day. So what crosses the cell membrane and why? Well, there are several types of ways to transport things across the cell membrane. When and how things travel depends largely on the concentration of solutes in your cells or the dissolved molecules. In this lesson, we'll discuss methods of transporting solutes across a concentration gradient. A concentration gradient is a gradual difference in solute concentration between two areas. In this case, it's the difference in solute concentration between the outside of the cell and the inside of the cell. Solutes here would move by diffusion, or movement from a higher concentration of solutes to a lower concentration of solutes in order to equalize solute concentration. This evens out the concentration on both sides. This is like what happens when you leave the windows of your house open while your neighbor's having a barbecue. The smell might diffuse from next door, where the smell is stronger in the air, into your house, where, unfortunately, there are no hamburgers on the grill. Diffusion occurs until the inside of your house smells like the outside. Diffusion across the cell membrane is a type of passive transport, or transport across the cell membrane that does not require energy. Remember that the cell membrane is a phospholipid bilayer. Although the inside and the outside of a cell are both water-based, there is a hydrophobic region in the middle, and this is an important barrier to anything large, charged, or hydrophilic. Molecules that are hydrophobic, just like the hydrophobic region, can pass through the cell membrane by simple diffusion. Therefore, simple diffusion is the unassisted passage of small, hydrophobic, nonpolar molecules from higher concentration to a lower concentration. Very small molecules can slip through the cell membrane too, even if they are hydrophilic, just like a few ants might crawl through a crack in the wall just because they're tiny. So how do large, charged, or hydrophilic molecules pass through the cell membrane if they can't simply just diffuse in? Well, think of how your friends come into your house. Under most circumstances, they'll use a door. A package delivered at your residence would come through your door too, but it would need someone to carry it in. A fly might come through an open window on its own, while a squirrel could come down your chimney. There are different types of passageways into the cell, just like there are different ways to get into your home, depending on who or what is trying to get through. So each method of passage through the cell membrane might be useful to different molecules. So, facilitated diffusion is passive transport that uses integral membrane proteins to help larger, charged, hydrophilic, and polar molecules across a concentration gradient. Remember that integral membrane proteins span the phospholipid bilayer, connecting the inside and the outside of the cell. There are two types of integral membrane proteins that help transport molecules like ions and polar molecules that can't diffuse on their own through the hydrophobic layer. The first are carrier proteins, which are proteins that bind a molecule to facilitate transport through a cell membrane. The second are channel proteins, which are proteins that create a passageway to transport molecules and ions through the cell membrane. This channel protein creates a pore through the hydrophobic region that allows polar molecules to just pass right through. The last type of passive transport involves water. No matter what your house is made of, water seems to find a way in during a big rainstorm. Water is a powerful substance. Even though your cell is surrounded by a hydrophobic region created by the phospholipid bilayer, water can still make it into the cell too. It can pass through your cell membrane in part because it's very small, but it also gets a little help from other molecules and membrane proteins too. Even with this help, however, this type of transport does not require energy. You might remember that we call this osmosis, or passive transport of water across a membrane. Water is an excellent way to even out solute concentration without having to move solutes themselves. In this cell, there are more solutes outside the cell and less solutes inside the cell. Therefore, you might remember that we'd call the inside of the cell a hypotonic solution, having less solutes, more water. Think of the O in hypotonic as a hippo swelling with water by the river, as a reminder that the hypotonic solution is whichever solution has more water in it. We'd refer to the outside of the cell as a hypertonic solution, hypertonic, having more solutes, less water. In osmosis, water will move to equalize solute concentration. Therefore, water will move from a hypotonic environment to a hypertonic environment. In this case, water would be transported out of the cell, causing the cell to shrink. This would continue to happen until the two sides were isotonic, or equal concentration of solutes. So what would happen if a cell was placed in a hypotonic solution with more water and less solutes outside the cell? A cell in this situation would have water move into the cell, causing the cell to swell. If it grows too much, it might burst like a balloon filled with too much air. In this lesson, we've learned several different methods of passive transport, or transport across the cell membrane that does not require energy. Passive transport moves across a concentration gradient, or a gradual difference in solute concentration between two areas. Simple diffusion is the diffusion of small, uncharged, or hydrophobic molecules from an area of high concentration to an area of low concentration across the cell membrane. Facilitated diffusion is diffusion using carrier or channel proteins in the cell membrane that assist in the movement of molecules across a concentration gradient. The third type of movement is known as osmosis, or the movement of water to equalize solute concentration. Have you ever been to the fair when it's really crowded? Well, maybe you wanted to go, but it's too crowded for you. So the lines are too long and the people are shoulder to shoulder carrying cotton candy and spilling popcorn all over the place. Perhaps you wait in the busy parking lot, observing the throngs of people and wondering if you shouldn't wait till it's not so busy. You're not a fan of the crowds, but you are a huge fan of the fair. So finally you decide, let's face it, it's a fair. A fair is always busy. There's only one thing to do in a situation like this. You muster up all your energy and you fight your way through the admission gate to get yourself a roll of tickets and some fried dough. Is a cell like the town fair? Well, there sure are a lot of things going on in the cell at the same time, so it might seem busy like a carnival. There's also many times when a cell needs to transport something in that goes against the concentration gradient. So let's think of the fair like a concentration gradient. Remember that a concentration gradient is the gradual difference in solute concentration between two areas, much like you standing outside the fair while there are thousands of people inside. There is a higher concentration of people inside the fair. So when a cell needs to transport something along a concentration gradient or from an area of high concentration to an area of low concentration, it does this by passive transport. This would be like people leaving the fair because it's crowded. That's easy. However, when a cell needs to transport something against a concentration gradient, or you decide to move from an area of low concentration of people to an area of high concentration of people, this is going to take some willpower, or in the case of the cell, some chemical energy. Active transport is transport against a concentration gradient that requires chemical energy. Active transport moves ions or molecules in a specific direction through the use of an integral membrane protein. Think of these like different gates at a fair. There are three types of membrane proteins that use energy to push these substances against the concentration gradient. A uniport is an integral membrane protein that moves an ion or molecule in one direction. Uni can remind you of the word one, such as one substance moving in one direction. This would be like an admission gate that people could only enter, or an exit gate through which people could only leave. An antiport is an integral membrane protein that moves one ion or molecule in one direction while moving a second substance in an opposite direction. The anti in antiport means against, and can remind you of two substances moving against each other, like an admission gate that only lets a few people enter if a few people exit at the same time. A symport is an integral membrane protein that moves two ions or molecules in the same direction. Sym in symport stands for the word same, as in two substances moving in the same direction. This would be like an admission gate that had two lines of people to enter the fair at the same time, in the same direction. Both antiports and symports are a type of coupled transport because they transport two different substances through the same integral membrane protein. Importantly, all three different types of integral membrane proteins use chemical energy to make this active transport happen. This allows a cell to maintain specific concentrations of substances different than in its surroundings. Let's talk about a specific example of active transport that is going on in all of your nerve cells right now. Your nerve cells require that a specific concentration gradient of sodium and potassium be maintained in order to properly conduct your signals. There is a higher concentration of sodium ions outside a cell than inside a cell. There is also a higher concentration of potassium ions inside the cell than outside the cell. The sodium-potassium pump ensures this gradient exists by constantly pumping sodium ions and potassium ions against the concentration gradient using active transport. First, the sodium-potassium pump in the cell membrane binds 3 sodium ions from inside the cell. Next, with the help of chemical energy, the sodium-potassium pump changes shape and exports these sodium ions outside the cell. This is against the concentration gradient of sodium. 2 potassium ions from outside of the cell then bind to the pump and are imported into the cell. Again, this is against the concentration gradient. Because 2 substances are transported in opposite directions, like the Fairgate that is both an entrance and an exit, the sodium-potassium pump is a type of antiport. In summary, we've learned that active transport is transport that requires chemical energy to move substances against a concentration gradient. There are 3 types of integral membrane proteins that aid in active transport. Uniports are proteins that move one substance in one direction. Antiports are proteins that move 2 substances in opposite directions. And symports are proteins that move 2 substances in the same direction.
Let's just imagine for a second that each one of your cells has an appetite. Perhaps one cell in particular has a chocolate addiction. Can you imagine what might happen if this cell saw a plate of brownies? What about a chocolate milkshake? Could a cell figure out a way to transport that across its cell membrane? Where there's a will, there's a way. If your cell is as much of a chocoholic as we're describing, it might be able to figure something out even though it doesn't have a mouth like you or me. We've talked about active and passive transport mechanisms used to move substances across the cell membrane. In this lesson, we'll focus on additional methods of transportation that allow some cells to eat or drink its surroundings. In addition, we'll discuss how a cell digests these items and what happens to the leftover food that can't be digested at all. In order to be able to chow down on substances in its environment, a cell will actually engulf its surroundings, folding its cell membrane around whatever it wants to ingest as if it's taking a giant bite. This process is called endocytosis, or the import of molecules and particles into the cell by folding the cell membrane inwards. The prefix endo means within. During endocytosis, vesicles or circular buds comprised of membrane are formed around the substance to be digested. These vesicles are then transported into the cytoplasm. There are 4 types of endocytosis, 3 of which are based on clathrin and 1 that is not. Clathrin is a component of the protein that coats vesicles as they form from the plasma membrane. The first of the 3 clathrin-based types of endocytosis is phagocytosis. Phagocytosis comes from the Greek word for devour. Phagocytosis is the process where large molecules or other cells are eaten by engulfing them. In phagocytosis, the cell membrane folds around the object to be eaten, creating a vesicle that can then be internalized. For example, a white blood cell in your immune system can eat a foreign bacterium whole in one bite. The second type of clathrin-based endocytosis is penocytosis, which comes from the Greek word for drink. It is the process where small dissolved substances and liquids are taken up by a cell. Penocytosis is similar to phagocytosis. Both of these processes allow cells to non-specifically take up substances from the surrounding by engulfing it with its membrane. However, in penocytosis, smaller vesicles are taking up liquid and dissolve solutes in that liquid, such as these small squares here, rather than large molecules. For example, the cells surrounding your blood capillaries perform penocytosis to drink your blood, while at the same time taking up any solutes dissolved in your blood. In the third type of clathrin-based endocytosis, the cell membrane also folds around a substrate to engulf and internalize it. However, in receptor-mediated endocytosis, this is a process that recognizes and targets a specific substrate through the help of integral membrane proteins on the cell membrane surface. Here, integral membrane proteins called receptor proteins recognize and bind to a specific target, such as these stars. In this example, these receptor proteins would not recognize and bind to non-star objects, like the squares. When a receptor protein binds to a target, it signals the cell membrane to enclose the target and internalize it inside a specialized vesicle. Phagocytosis and pinocytosis are non-specific methods of endocytosis. They would eat any kind of chocolate. Receptor-mediated endocytosis is specific, picky. It knows exactly what it wants. The term caveoli is Latin for little caves. Caveoli are pockets in the plasma membrane, which were first identified with an electron microscope in 1953. Caveoli do not have a clathrin-based protein lining them. Instead, they have caveolins, which are membrane proteins that play a role in various metabolic pathways and cellular signaling. In mammals, there are 3 types of caveolins, which are, you guessed it, caveolin 1, caveolin 2, and caveolin 3. Cardiac and skeletal muscle cells employ only the caveolin 3 type of protein, while smooth muscle cells employ all 3 types. Caveoli-based endocytosis is still the topic of research debate, and for some time it was not certain that this specific cell pathway did involve endocytosis. However, it has at least been shown that caveolin 1 is involved in endocytosis, creating internal compartments in the plasma membrane. Whether specific or nonspecific, all methods of endocytosis internalize a substrate into a vesicle, which then enters the cytoplasm. So what happens next? Think about what happens when you eat or drink something. It travels down to your stomach, where it is then digested by enzymes in the acidic environment of your stomach. What happens after endocytosis is not too different. The vesicles fuse with structures called lysosomes. A lysosome is a structure within the cell that contains digestive enzymes. The lysosome is acidic compared to the rest of the cell because integral membrane proteins in the membrane surrounding the lysosome pump protons into the space. The digestive enzymes within the lysosome work optimally at this lower pH. Both a vesicle and a lysosome are made up of a phospholipid bilayer, allowing these 2 structures to merge into one. Lysosomes are able to break down food and foreign material from these vesicles using their digestive enzymes. In addition, lysosomes are also able to break down molecules that originated from the cell, such as proteins or even whole cellular structures. Digested material from a lysosome can then be reused or recycled by the cell. The lysosome is both the garbage disposal and the recycling bin all in one. You might be wondering what happens to substances that can't be reused or recycled by the cell. Whether it was something that originated outside the cell or something that originated inside the cell, there are some things that a lysosome just can't digest. What happens to this material? It's not too much unlike what happens to our undigested material. The cell needs to get rid of it. To do this, the cell performs exocytosis, or the export of substances from the cell through a vesicle that fuses with the cell membrane. In this process, particles that cannot be digested by a lysosome are carried by a vesicle to the cell membrane. Because these vesicles are made of the same phospholipid bilayer as the cell membrane, it can merge with the cell membrane and release its contents outside of the cell. The vesicle then becomes part of the cell membrane. Exocytosis takes out the trash. But it's important to note that exocytosis doesn't just keep things tidy in the cell. Exocytosis can also be used to transport useful things to the cell membrane and extracellular space. Exocytosis is used to add new integral membrane proteins, phospholipids, and other membrane components to the cell membrane. Vesicles with new integral membrane proteins in their phospholipid bilayers can fuse with the cell membrane. As the 2 membranes become one, the new integral membrane proteins become part of the cell membrane. In addition, all the phospholipids and other vesicle membrane components also become part of the cell membrane. This is a great way to refresh the cell membrane with new parts. Exocytosis can also release useful products created by your cell into the extracellular space. For example, cells in your pancreas make digestive enzymes that are secreted outside your pancreatic cells by exocytosis. In summary, endocytosis is the import of molecules and particles into the cell by folding the cell membrane inwards to form vesicles. There are 4 kinds of endocytosis, 3 of which involve clathrin, a component of the proteins that coat vesicles, and 1 that does not. Phagocytosis is nonspecific cellular eating of large substances. Pinocytosis is nonspecific cellular drinking of liquids and dissolved substances. Receptor-mediated endocytosis is the specific endocytosis of substrates recognized by receptor proteins on the cell membrane. Caveoli are the little caves that comprise the non-clathrin-based form of endocytosis. Vesicles containing substances internalized by endocytosis can fuse with a lysosome, which is a structure that can digest substances originating from the outside or inside of the cell. Substances that can't be digested by the lysosome are released from the cell during exocytosis, which is the process that secretes material outside the cell through a vesicle that merges with the cell membrane. Remember that exocytosis can release undigested material or new material created by the cell, in addition to adding new membrane components to the cell membrane.
Your cells are fine-tuned and well-oiled machines. For the most part, they run smoothly like a brand new car on a freshly paved surface. Many cells have an extremely important structure that holds all the essential information vital to their ability to perform. Each of these cells comes with an owner's manual, instructions on how to run day-to-day operations and specialized functions. These instructions are your DNA, which is stored in its very own cellular glove compartment, the nucleus. The nucleus is a membrane-bound structure that contains the genetic material of a cell. The plural of nucleus is nuclei. Not all cells have nuclei, but many cells, such as those in plants, fungi, animals, and protists, contain these structures. The nucleus is not just a storage compartment for DNA. It's the site of some essential cellular processes. First, DNA can be duplicated in the nucleus. This process is called replication and creates an identical copy of DNA. Creating two identical copies of the owner's manual is the first step in cell division, where each new cell will get its own set of instructions. Second, the nucleus is the site of transcription. The central dogma of biology states that DNA is copied into RNA, which is then turned into protein. Transcription is the process of creating different types of RNA from DNA. Transcription would be like making copies of individual pages of the owner's manual that can then be passed out and read by the rest of the cell. There are three types of RNA that are formed in the nucleus. mRNA, or messenger RNA, is a code that can be read by the cytoplasmic structure called the ribosome during translation. Translation is a process that creates proteins from mRNA. A ribosome, which we will discuss in future lessons, is made up of proteins and a second type of RNA, rRNA, or ribosomal RNA. tRNA, or transfer RNA, is also used during the process of translation. All three of these RNA types, mRNA, rRNA, and tRNA, are created by the process of transcription within the nucleus. The nucleus is the structure responsible for containing DNA and providing a place for replication and transcription. These processes make sure that the instructions on how to make you who you are carried out. This is obviously a very important job. So to keep the cell functioning at top speeds, the nucleus is a well-organized structure. The nucleus is surrounded by a nuclear envelope, which is a double membrane comprised of an outer membrane and an inner membrane. This double membrane shields the nucleus and its contents from unwanted guests. Some proteins need to be in the nucleus to assist in processes such as replication and transcription. Many proteins, however, have no business being there. Your DNA, your very own patented genetic owner's manual, is a unique and prized possession. It needs to be protected. Only very small proteins can get into the nucleus through the nuclear pores, which are small openings in the nuclear envelope where the inner and outer membranes come together. Each pore is lined with proteins, creating a nuclear basket on the nuclear side of the membrane. There are thousands of these nuclear pores surrounding a nucleus. Large proteins are unable to pass through these nuclear pores without the right credentials. Only large proteins with a specialized amino acid sequence, known as the NLS, or nuclear localization signal, are allowed into the nucleus through the nuclear pore. If they have the right pass, these large proteins are escorted through the help of a specialized group of import proteins that carry NLS protein cargo into the nucleus. This action requires chemical energy, causing the pore to stretch to accommodate these bigger proteins. Why are only certain proteins allowed in the nucleus? Well, remember this is where essential DNA processes occur. Your DNA instructions are carefully folded up and stored in the nucleus in the form of chromatin, which is DNA packaged with the help of specialized proteins. You can imagine that you'd also want to keep your DNA owner's manual from getting torn or wrinkled. The nucleus protects and organizes chromatin also through the nuclear lamina, which attaches to the nuclear envelope and chromatin to help maintain the shape of the nucleus. The nucleolus is an area of the nucleus that is the starting site of ribosomal synthesis from rRNA and proteins. In summary, the nucleus is the structural site containing genetic material, as well as the site of replication, transcription, and ribosomal synthesis in some cells. The nucleus is surrounded by a nuclear envelope, or outer and inner membrane, and is impermeable to large proteins without a specialized amino acid sequence called an NLS. Small proteins, or proteins with an NLS attached to carrier proteins, can enter the nucleus through nuclear pores, or openings into the nucleus. The nuclear lamina provides structure to the nucleus by attaching to the nuclear envelope and chromatin. The nucleolus is an area of the nucleus that is the starting site of ribosomal synthesis from rRNA and proteins. We've reached a point in science where it's possible to find out all the letters in your DNA sequence, and that's pretty cool. But if you've ever looked at a DNA sequence, you might have noticed it isn't exactly English. We can find out all the A's, the G's, the T's, and the C's in a human genome, but it's a completely different language than the one you likely learned in grade school. And how can you tell which of these letters made your eyes blue and which ones gave you those big ears? Well, fortunately, your cells can read your genome language just fine, though it takes a few steps. Your DNA tells your cells what proteins to make. Remember that it does this through the central dogma, where your DNA is first transcribed into RNA. DNA is a huge manual of instructions, so transcription is like writing down one page of DNA instructions at a time. There are 3 types of RNA made during transcription: messenger RNA, or mRNA, transfer RNA, or tRNA, and ribosomal RNA, or rRNA. In the second step of the central dogma, mRNA is translated into protein during translation. You'll remember that proteins are built from several different amino acids in a specific order. So after that refresher, it's time to introduce you to the smarty pants of the cell who is able to speak this DNA language. This would be the ribosome, the cellular structure that performs translation or protein synthesis. Ribosomes are both language translators and efficient assembly workers. Putting together any sort of protein is like putting together a piece of furniture that has 3 boxes of more than 100 parts. And the instructions are in a foreign language, with no pictures. We've all tried to do it, and it's not pretty. But for ribosomes, this is a cinch. They read an mRNA sequence, translate it, and use tools to build it into a protein. This protein is then used by the cell. Specifically, ribosomes use tRNA as tools. This helps bring the correct amino acids to the building protein. Ribosomes are composed of rRNA and proteins. They exist free in the cytoplasm until they receive an mRNA message to translate. When they receive it, they waste no time in cracking the code. Ribosomes can be either free ribosomes in the cytoplasm of the cell, or they may find that the mRNA they are translating has a specific destination. In this case, some ribosomes will attach themselves to another cellular structure, and this is called the endoplasmic reticulum. Here, these are called the bound ribosomes. Proteins produced by bound ribosomes are usually destined for some kind of specific structure, or they're meant to exit the cell through exocytosis. There are also ribosomes within the mitochondria and chloroplasts of different types of cells, but we'll discuss that in other lessons. Although different organisms might have slightly different sized ribosomes, they are very similar in the structure, and they all perform translation. Specifically, translation is protein synthesis by creating a chain of amino acids linked together by polypeptide bonds. Ribosomes are composed of 2 different subunits, called the large subunit and the small subunit. Both subunits are composed of rRNA and protein. These subunits usually exist separately in the cytoplasm until the small subunit recognizes and binds to a specific sequence of an mRNA message. It then recruits the large subunit to form a complete ribosome. Ribosome structure allows the mRNA message to be translated by holding the mRNA and tRNA molecules in the right place at the right time. Each tRNA carries a specific amino acid. The ribosome is responsible for matching the correct tRNA to the mRNA sequence. This is done with the help of 3 tRNA binding sites on the large subunit. They are called the A, P, and E sites. They are used in that order, spelling ape. The A site stands for the amino acid site, where a specific tRNA binds to a specific mRNA sequence. The P site stands for polypeptide site, where a polypeptide bond is formed between a new amino acid and the growing amino acid chain. The E site, that's easy. It stands for the exit site, where the tRNA exits. We'll learn more about these steps in translation during the translation lesson, but for now you can appreciate that this structure is such an efficient translator, able to decipher our genetic instructions by having very good organizational skills. The ribosome is the cellular structure and location of translation, or protein synthesis. It is composed of rRNA and protein. Normally, ribosomes are in the cytoplasm as a small subunit that recognizes and binds to an mRNA transcript, and a large subunit, which is recruited to the small subunit to translate the mRNA into protein. During translation, a ribosome reads and translates the mRNA sequence, matching it with a specific amino acid using tRNA as a tool. This tRNA moves through the A, P, and E sites as it adds new amino acids to a growing protein. A translating ribosome can perform its function free in the cytoplasm or bound to the endoplasmic reticulum. Some ribosomes are also located in structures called mitochondria and chloroplasts. No matter where you live in the world or what you celebrate, you've likely heard of Santa Claus and his tiny elves. These little elves make toys and games, wrap them up, label them with the names of good little boys and girls, and send them off with Santa to be delivered for Christmas. Christmas seems to come every day for your cells, where tiny ribosomes make proteins all throughout the year. Once they're made, these proteins need to find a way to a good home, the right good home. And in your cell, this is where the endomembrane system comes in. The endomembrane system is a series of compartments that work together to package, label, and ship proteins and molecules. In your cells, the endomembrane system is made of both the endoplasmic reticulum and the Golgi apparatus. These compartments are folds of membranes that form tubes and sacs in your cells. There are two parts to the structure known as the endoplasmic reticulum. Both have a membranous tubular structure and are located near the nucleus of the cell. Notice that the membrane folds, providing plenty of surface space for work to be done, like multiple benches for Santa's elves. This also creates compartments without any open ends. The lumen is the inside compartment of the endoplasmic reticulum. The lumen is completely separated from the cytoplasm that surrounds it. You can tell apart the two types of endoplasmic reticulum in a cell image because one is studded with small ribosomes and one is not. The one without ribosomes is known as the smooth endoplasmic reticulum because without ribosomes it really looks like a membranous compartment of smooth tubes. This can also be referred to as the smooth ER or simply the SER. The smooth ER is the site of lipid and steroid synthesis. In the lumen of the smooth endoplasmic reticulum, proteins and small molecules can be chemically modified. The smooth endoplasmic reticulum is also the detoxifying space of the cell. The liver is the detox organ of your body. Therefore, your liver cells can have more smooth endoplasmic reticulum than your heart cells, for example, as it uses this cellular component to detoxify drugs and alcohol. The part of the endoplasmic reticulum that contains ribosomes is named the rough endoplasmic reticulum. In the rough ER, or simply RER, there are membranous compartments and sacs studded with ribosomes. Ribosomes attached to this membrane make it look like folds of bubble wrap, perfect for wrapping presents. As we have learned, ribosomes are the site of protein synthesis, otherwise known as translation. When a ribosome begins to translate an mRNA message into a protein, it might hit a specific sequence that tells the ribosome that this is the protein that needs to be synthesized on the rough ER. This is like telling the ribosome to build a protein in an area of the workshop that has the right tools, like putting a dollhouse together near the hammer, the nails, and the pink paint. As they are being synthesized, these proteins enter the lumen of the rough ER, and here proteins are chemically modified or folded inside the RER lumen. Proteins synthesized on the rough ER can be sent to specific cellular compartments or exported through exocytosis. Once a protein has been made in the rough ER, it needs to be packaged and shipped to its final destination, just like a present needs to be wrapped in a festive bow and sent on its way to the right waiting child. In the cell, this is done by the Golgi apparatus. the Golgi apparatus really is the shipping center of the cell. And like the ER, the Golgi is also a membranous compartment of flattened sacs, and is found near the rough ER. Its membrane folds also to create a lumen space. It packages, sorts, and sends off proteins from the rough ER. These proteins could be destined for another specific compartment inside the cell, such as the lysosome, or these proteins could be sent out of the cell via exocytosis. In addition, the Golgi is also involved in lipid transport and the creation of new lysosomes. Vesicles from the endoplasmic reticulum bud off and join the Golgi on its cyst side, which is near the rough ER and nucleus. The membranes from the vesicle and the Golgi fuse together, allowing the contents of the vesicle to enter the Golgi lumen and then work its way through the middle of the Golgi. The contents are sorted and modified before budding off the trans side to exit the Golgi with an address label that tells the cell where the product needs to go. In this lesson, we've learned about the endomembrane system, a membranous compartment consisting of the endoplasmic reticulum and the Golgi apparatus. This system works together to produce lipids and proteins and ship them to the correct destination. The smooth endoplasmic reticulum is the site of lipid and steroid synthesis, detoxification, and modification. The rough endoplasmic reticulum is the site of translation for proteins needing modification and transport. Proteins synthesized in the rough ER are sent to the Golgi apparatus, which is the site of protein sorting and shipping to specific cellular compartments or out of the cell through exocytosis. Together with your muscles, your nerves, and your skin, your skeleton gives your body that characteristic human shape. It allows you to stand on 2 feet, to leap, and to play around a flag football. Did you know that all of the cells in your body also have a skeleton all of their own? Many different types of cells have a complex called the cytoskeleton, or network of thin fibers, that can help these cells do a range of activities. Maybe a cell can't pass around a pig's skin, but it can do some equally cell-like impressive things with the help of its very own skeleton. A cytoskeleton can provide support and shape for a cell, like your skeleton supports and shapes your body. Your skeleton helps you stand, and it also helps you run. In a cell, a cytoskeleton can also anchor a cell in one place, or allow a cell to move. The bones in your arm can help you catch a football, independent from what's going on in your legs. The parts of your cell's cytoskeletons can also help structures move independently within your cells. A cytoskeleton can position cell structures in specific places within the cell, or it can move cell structures from one end of the cell to the other. A cell's cytoskeleton is comprised of 3 different types of fibers: microtubules, intermediate filaments, and microfilaments. Microtubules are the largest fibers of the cytoskeleton, with a 25 nanometer diameter. They are composed of 2 different proteins called alpha and beta tubulin. 1 dimer of tubulin contains 1 alpha subunit and 1 beta subunit. A microtubule is made up of many, many tubulin dimers. These dimers form long chains of alpha and beta tubulin. 13 chains of tubulin dimers make up 1 microtubule. The chains form a hollow tube, which is where microtubules get their name. They look like a paper towel roll. Each end of a microtubule is referred to as the plus end or the minus end, and each microtubule is dynamic, meaning it can grow and it can shrink by adding or subtracting dimers, usually to the plus end. Microtubules function to support the cell shape, but they also help cell division. Microtubules can be used like train tracks in the cell, along which vesicles and structures can be transported by riding across them. A 4th important function of microtubules is in cell movement. Some cells have flagella and cilia made of microtubules. Flagella are long snake-like whips that drive cell movement. Sperm cells are an example of cells that have flagella, which allows them to swim. And cilia are multiple short hair-like structures that beat to move liquid around a cell. Cilia are found in the cells that line your respiratory tract, where they rhythmically beat to help you move mucus when you're sick. Both flagella and cilia are made with microtubule structures surrounded by a plasma membrane. If you looked at a cross-section of flagella or cilia, you would see what's referred to as a 9+2 arrangement of microtubules. This is 9 pairs of microtubules in a circle with 2 single microtubules in the center. The The next components of a cytoskeleton are the intermediate filaments. These fibers are called intermediate because they are the mid-sized fibers of the cytoskeleton, measuring 8 to 12 nanometers in diameter. They can be made from any of several fibrous proteins twisted together like twine to make a rope. Intermediate filaments have a diverse role in structure and support within the cell. One specialized group of intermediate filaments has an important role in the nucleus, where they can make up the nuclear lamina. Remember that here they're essentially the cytoskeleton of the nucleus, specifically supporting a structure of your cells, like the bones supporting your hands. Here, intermediate filaments provide support to the nuclear lamina and aid in regulating nuclear processes. They also extend from the nuclear envelope, helping the nucleus keep its position within the cell. Microfilaments are the thinnest fibers of the cytoskeleton. They are 7 nanometers in diameter. These fibers are composed of actin protein. Monomers of actin combine to form long double helical chains. Microfilaments share a lot of similarities with microtubule function. They are also dynamic, and they can grow and shrink by adding and subtracting actin subunits. In addition, they also have a plus and a minus end. Microfilaments function to keep cell shape. They also play a crucial role in muscle cell contraction, essential to the ability of your skeleton and muscle systems to work together and throw and catch a football. Perhaps you could blame that bad throw on your microfilaments. Like microtubules, microfilaments can cause cell movement. However, they do this differently than microtubules. Microfilaments in amoeba, for example, form temporary extensions of the cell membrane that allow cells to push out projections and walk across a surface, one foot right in front of the other. In summary, we've learned that the cytoskeleton of your cells is a network of microtubules, intermediate filaments, and microfilaments that perform a wide range of functions. Microtubules are the thickest cytoskeleton filaments, hollow tubes formed from alpha and beta tubulin dimers. Microtubules provide cell shape and structure, aid in transport within the cell, help in cell division, and drive cell movement by flagella or cilia. Intermediate filaments are the mid-sized cytoskeleton fibers comprised of proteins twisted into rope-like structures. These fibers are important in stabilizing not only whole cell structure, but nuclear structure as well. Lastly, microfilaments are the thinnest fiber of the cytoskeleton, a double helical structure of actin subunits. Microfilaments are important to cellular shape, muscle contraction, and cell movement. Your mother always told you to eat your breakfast. Breakfast is the meal of champions. It's the right way to start your day when you wake up hungry, giving you the energy you need to tackle all your to-do lists. But while you're feeding yourself, did you know you're also feeding your cells? Your cells might not have the taste buds to appreciate the authentic Vermont maple syrup you put on your pancakes this morning, but they do appreciate the sugar. The cells in your body use the food you eat as a way to get the energy they need to get through the day. They do this through cellular respiration, a process that converts food into usable chemical energy in your cells. Does your cell eat pancakes? Not exactly, but once the food is broken down by your digestive system, molecules from the food are transported to your cells for use. Many cells, including those in animals, fungi, and plants, contain mitochondria. Mitochondria are membrane-bound structures where cellular respiration occurs. Mitochondria is the plural form of the word mitochondrion. In these mitochondria, molecules from your pancakes are used to create chemical energy that your cell can then use to perform different activities in its daily routine. For example, chemical energy allows active transport of molecules against a concentration gradient in the cell membrane. A cell can have 1 to thousands of mitochondria, depending on how much energy it needs. A cell in your biceps, for example, needs a lot of energy to pump iron and would have more mitochondria than a cell in your skin. You can fit a lot of mitochondria in one cell because they're small, about the size of a bacterium. Bacteria, by the way, do not have mitochondria for reasons that we'll talk about in other lessons. Mitochondria are composed of 2 membranes. Both are phospholipid bilayers, like the membrane around the cell. The outer membrane is a selectively permeable membrane that surrounds the mitochondria. This membrane comes complete with integral membrane proteins and pores for transporting molecules, just like the cell membrane. The inner membrane of the mitochondria folds inward to form these extra surfaces for cellular respiration. In this way, the inner membrane creates more space out of a small area. It's almost like having booths at a diner, giving customers multiple places to sit down for their daily breakfast special. Having 50 tables allows more people to dine in than if there were only 2 or 3 tables in the restaurant. Unlike the outer membrane, the inner membrane does not have pores or channels and is very impermeable. The cristae are the folds created by the inner membrane, or the restaurant booths themselves. The surface area of the cristae can be several times larger than the perimeter of the mitochondria. Cells that need more energy can also have more booths, or cristae, for those reactions. The cristae contain proteins and molecules used for making chemical energy for the cell. Finally, there's the matrix, which is the inside of the mitochondria created by the inner membrane. The matrix contains enzymes for cellular respiration, as well as its own ribosomes and DNA, needed to create some of the proteins important for this process. In summary, many cells have mitochondria, or structures that convert food sources into chemical energy through the process of cellular respiration. The outer membrane surrounds the mitochondria. It is a semi-permeable membrane similar to the cell membrane. The inner membrane is impermeable. Its function is to create more space to perform cellular respiration. The folds created by the inner membrane are known as the cristae, which contain proteins and molecules that participate in cellular respiration. The matrix of the mitochondria is the space surrounding the cristae, which also includes molecules that participate in cellular respiration, as well as ribosomes and mitochondrial DNA. Plants love to relax in the sun, and they don't really think about slathering on the sunscreen. That's because sunbathing serves an important purpose to plants beyond getting a tan. You see, plants aren't exactly able to order their favorite food by the poolside. Instead of consuming food, plants make it. And as any good chef would know, creating food can be hard work. These things require a lot of energy to do. And instead of ordering takeout or putting on their favorite apron to get busy in the kitchen, plant cells go through an efficient process to create food, photosynthesis, a process that converts energy from sunlight into food in plant cells. We'll get into the details of how photosynthesis works in future lessons. In this lesson, we'll discuss an important structure essential to this process. Each plant is made of many plant cells performing photosynthesis. Inside of each of these tiny plant cells are 1 to hundreds of small structures called chloroplasts. Chloroplast comes from the Greek word chloros, for green, and plastis, for the one that forms. That's because chloroplasts are what give plants a green color and help the plant cells form something delicious. Chloroplasts are the structural sites of photosynthesis, where light energy is converted into food. A cell that resides in a plant leaf, for example, might have hundreds of chloroplasts that capture light from its tanning session and use it to make the plant equivalent of a burger and fries. Chloroplasts exist in the cytoplasm of plant cells. They are flat, frisbee-shaped structures filled with thylakoids. Thylakoids are small, disc-like compartments composed of membranes that are the sites of sunlight-dependent photosynthesis. The thylakoids are surrounded by the stroma, or the inner liquid portion of the chloroplast. Both the stroma and the thylakoids contain important molecules for photosynthesis. Thylakoids are often stacked on top of each other. They look like a stack of flapjacks. Grana, or singular granum, are stacks of thylakoids within chloroplasts. The thylakoids contain numerous integral and peripheral membrane proteins, as well as chemicals important to photosynthesis. One of these important molecules is called chlorophyll, a pigment or compound that absorbs a specific wavelength of energy from sunlight to use in photosynthesis. This is really like the solar panels of the plant cell, harnessing light energy from the sun. Chlorophyll is also the pigment responsible for making chloroplasts, and therefore all plants on Earth, such lovely shades of green. Another interesting tidbit about chloroplasts: they contain their own DNA and ribosomes. The DNA is different than the DNA in the nucleus, and may code for proteins essential to photosynthesis. Having its own ribosomes also allows the chloroplast to perform translation of these proteins, independent of what goes on outside the chloroplast inside the cytoplasm of the cell. In summary, the chloroplast is an important plant cell structure and the site of photosynthesis, which converts energy from sunlight into food. The chloroplast contains chlorophyll within its thylakoids, which absorbs light energy and gives chloroplast its green color. Stacks of thylakoids are known as grana, which exist in the open spaces of the chloroplast, known as the stroma. Your friend asked you to watch her plants while she went away for the summer. You know plants need plenty of sunshine and water, so you put them on your windowsill and give them a daily drink. But maybe one day you get home late from work and you just completely forget to water the plants. Today turns into tomorrow, which turns into the next day, and before you know it, you walk by the plants and you notice they've started to wilt. Whoops! Well, even though the plants are wilting, you notice they still look like the same plant with roughly the same shape. There's still hope, and with a few extra days before your friend comes back, you still have some time to get back on track and water them daily. Thankfully, the plants perk right back up in no time, and you're still rewarded for your plant sitting with a box of chocolates. Lucky you. If you ever stop to wonder why plants don't completely collapse when you forget to water them, there's a good reason for that. Plant cells share a lot of components with the cells in your body, such as DNA in a nucleus, an endoplasmic reticulum, and ribosomes. However, plants have some original cell components that are different than what's in your cells. For one, plant cells have chloroplasts that help turn light energy into food. In today's lessons, we'll talk about two more structures important to plant cells. All plant cells have a plasma membrane just like an animal cell, which provides the same barrier and regulates transport. However, plant cells also have a specialized structure called the cell wall. The cell wall is a protective layer surrounding the cell on the outside of the plasma membrane. A cell wall can be up to 800 times thicker than the plasma membrane. It's composed largely of cellulose, a polysaccharide sugar that provides strength to the cell wall. If you ever notice how strong the bark of a tree is, that's because this bark is composed of dead cells with really tough cell walls. The cell wall also grows with the cell, getting bigger as the cell gets bigger. Although plants aren't the only organisms with a cell wall, this structure is a characteristic of all plants. The cell wall serves several purposes. Its pupae-like toughness provides great protection, strength, and shape to the cell, helping a plant cell to be both flexible and rigid. Think about a bouquet of roses. The stems are strong enough that you need a sharp knife to cut them, but flexible enough that they fit easily into a beautiful vase. In addition to a cell wall, plant cells also have this very large structure that can take up as much as 80% of the cell's volume. This is called a central vacuole, a large storage compartment in plant cells. What can this vacuole hold? Well, although other non-plant cells can also have vacuoles, plant cells have these characteristically gigantic vacuoles that are largely a storage place for water. While animal cells are around 70% water, plant cells can be 90% full of water, and they need a place to put it. However, in addition to water, a vacuole can also contain food and other nutrients, as well as waste products. The central vacuole can also contain digestive enzymes like those in animal cell lysosomes. A central vacuole can also help maintain a neutral pH in the cell by pumping hydrogen atoms, or protons, from the cytoplasm into the vacuole. What happens if you pump a lot of protons into the vacuole? This makes this plant cell component acidic, which is also similar to a lysosome. So how does the cell wall in the central vacuole relate to those plants you forgot to water? Remember that cells transport water across their cell membranes through this process of osmosis. When you forget to water the plant cells are then in a hypertonic environment, or an environment with less water around them and more solutes. This causes water to leave the plant cells. The consequence? A wilting plant. Notice, however, that even in a hypertonic environment, the cell wall helps maintain the shape of the plant cell. As soon as you started watering the plant regularly, the plants were again in an isotonic or equal water environment. As a result, the cell's central vacuole takes on more water, and the cell can fill the cell wall's shape. Now, what if you poured on a little too much water to try to make up for lost time? You may have seen a plant wilt when it isn't watered enough. But have you ever seen a plant explode with water? I doubt it, and that's because in a hypotonic environment with more water outside the cell, the central vacuole will swell with water, putting pressure on the cell walls. However, those cell walls will keep the cell rigid and strong. It'll prevent the cell from bursting. This pressure that is put on the cell walls is known as turgor pressure. In fact, most healthy plants are in a constant state of turgor pressure, and this is what gives plants their rigid shape. In summary, we've learned more about two important plant cell components today. The first is the cell wall, which is a protective layer surrounding the cell on the outside of the plasma membrane. The cell wall is composed of cellulose, or a polysaccharide sugar that provides strength to the cell wall. A second structure important to all plant cells is the central vacuole, which is a large storage compartment in plant cells. Both the cell wall and the central vacuole help plant cells regulate water movement, or osmosis, to keep plant cells strong enough to withstand a few days without water. Your body is composed of trillions of cells. Lots of different types of cells that make up different organs and other parts of your body. Your body is also where 10 times that number of bacteria call home sweet home. But don't be afraid, these bacteria do more good than harm to you, and besides, just in case you wanted to strike up a conversation with your tenants, you and your bacteria do have a few things in common. All cells share some common characteristics that make them living things. All organisms are composed of cells, the basic fundamental unit of life. They contain DNA as a heritable genetic material, and they can reproduce. They transcribe DNA into RNA and translate RNA into proteins on ribosomes. They can also regulate transport across the cell membrane and require chemical energy for some cellular processes. The number one biggest difference between the bacteria in your body and the cells making up your body are these tiny cellular components called organelles. You've actually learned a lot about organelles in other lessons without knowing it. Organelles are simply membrane-bound compartments within a cell, such as the nucleus, mitochondria, chloroplast, Golgi, and endoplasmic reticulum. You are a eukaryote. Your cells are eukaryotic. Eukaryotic cells contain membrane-bound organelles, including a nucleus. Eukaryotes can be single-celled or multicelled, such as you, me, plants, fungi, and insects. Bacteria are an example of prokaryotes. Prokaryotic cells do not contain a nucleus or any other membrane-bound organelle. Prokaryotes include two groups, bacteria and another group called archaea. Having organelles is a big deal for a cell. A bacteria cell gets along just fine without organelles, but bacteria are tiny. That's why we're able to have so many of them on our body without really noticing them. Our cells, though, they're still small to the naked eye, but they're huge in comparison to bacteria. Our eukaryotic cells are bigger in size, with much more DNA. More DNA means more transcription, and more transcription means more translation, and more translation means more proteins, and bigger cells create the need for organelles. You can think of it this way. If you only had 2 pairs of shoes and a few simple outfits, you could just hang up your outfits and put your shoes on the floor inside a small closet. Simple. However, let's say you have a shopping addiction and you have 7 different pairs of black pants, 10 pairs of shoes, and completely different shades of brown, and other colors, of course, and you hardly ever wear the same hat twice. You can imagine that you would need a walk-in closet, complete with shelving systems to organize everything, right? Well, organelles are an efficient way to organize everything that's going on in the cell, to compartmentalize cellular functions. That's exactly what a eukaryotic cell is doing, separating cellular processes and organizing its space. But don't be fooled by the simplicity of prokaryotes. Their smaller size and simplicity is an advantage to their lifestyle. If you have a lot of shoes and a walk-in closet, the nucleus would obviously be the shoe rack, a structure to hold and organize all this important material. Having a nucleus or not is the biggest difference between eukaryotes and prokaryotes. Remember that your eukaryotic cells have linear DNA. Prokaryotic cells have a smaller, circular DNA genome. With a smaller size and a smaller genome, prokaryotes don't really need a nucleus. Their DNA is housed in a nucleoid, which isn't really a structure at all. There are no membranes around it, so it's not a separated compartment. The nucleoid is simply the area where DNA exists. This is the defining difference between prokaryotic and eukaryotic cells. Again, don't be fooled by the lack of compartmentalization. Prokaryotic cells are less complex, but they can still perform the same processes similar to eukaryotic functions. They just do it differently. For example, DNA replication and transcription take place in this nucleoid area of the cytoplasm. While translation is still carried out on ribosomes, these are not membrane-bound organelles, and all ribosomes in a bacterium float free in the cytoplasm. There is no endoplasmic reticulum. Prokaryotic and eukaryotic ribosomes are both made of rRNA and proteins, but the subunits are going to be different sizes. In addition, a group of bacteria can perform photosynthesis like plants. Without a chloroplast organelle, a bacterium can perform photosynthesis in the folds of its cell membrane. These cells may have been the first early organisms to ever perform photosynthesis. Prokaryotes also have this way of keeping their structure and shape. Many of them have a cell wall, like plant cell walls. However, prokaryotic cell walls are made of a different specialized molecule. For example, bacterial cell walls can contain peptidoglycan, or a polymer of sugar and amino acids that give shape and structures to the bacterial cell wall. Prokaryotes also have this version of a cytoskeleton that contains some of the same proteins that are similar to the actin and filaments of eukaryotic cells and function in a similar, though simpler, way. Some prokaryotes also have flagella, as do some eukaryotic cells. The function is the same, a long appendage that drives movement. However, prokaryotic and eukaryotic flagella are structurally different. While we have learned that eukaryotic flagella are composed of the cytoskeletal proteins called microtubule, flagella in bacteria are made of the protein flagellin. Bacterial flagella are also driven by motor proteins that swing the flagella around clockwise or counterclockwise, while eukaryotic flagella whip back and forth, not around. Let's summarize the similarities and differences between prokaryotes and eukaryotes. Both organisms are composed of cells, the basic unit of life, with each cell surrounded by a cell membrane. The biggest difference between prokaryotes and eukaryotes is that eukaryotes have a nucleus. They also have other membrane structures called organelles. Organelles are structures such as the endoplasmic reticulum and the mitochondria. The DNA in prokaryotes exists in a space called the nucleoid, and it is circular DNA, while eukaryotes have linear DNA, and it's organized into a higher order structure. Both eukaryotes and prokaryotes have ribosomes. Ribosomes are not a membrane-bound organelle, but in both eukaryotes and prokaryotes, they are used for translation of RNA into proteins. Eukaryotes and prokaryotes can both perform transport, DNA replication, transcription, translation, and movement. It's important to note, however, that although they share these similar processes, many are carried out within organelles in eukaryotes or use different proteins. For example, flagella and the cytoskeleton may be structured differently in prokaryotes and eukaryotes, but they have the same function of movement and providing cell structure, respectively. Every year around the time the leaves start to turn brilliant colors of gold and red, you're reminded to grab a scarf, drink some hot apple cider, and get your flu shot. The winter season is ahead, and with it brings a greater chance of catching the flu virus. There are great things about the winter, but the flu is not one of them. We are all familiar with viruses as something that makes us ill. Countless viruses are all around us. They infect not only humans, but also other organisms, such as animals, plants, and bacteria. They don't all cause the flu, and most of them can't be treated or even vaccinated against. So what exactly is a virus? A virus is an acellular parasitic particle consisting of protein and nucleic acid. Acellular refers to the fact that viruses are not made of cells. They can't reproduce on their own. They can't transport substances in and out of any membranes. They can't metabolize any sort of food. What can viruses do? Viruses can't really do anything on their own except invade a host cell. On their own, they do not exhibit characteristics of living things. For all these reasons, unlike the hosts that they infect, viruses are not alive. There's huge diversity in viruses and what hosts that they can infect. A bacteriophage is a virus that infects bacteria. Bacteriophages are nicknamed phages for short. Though it's true that a bacteriophage can't really give a bacterium the flu, bacteriophages share some similar features with the types of viruses we routinely avoid. However, all viruses are also host-specific. Bacteriophages will infect a specific strain or species of bacteria, but will not infect any other organism. They will not, and cannot, infect you. This is one of the reasons that bacteriophages are such a popular area of current research. In the growing epidemic of antibiotic-resistant bacteria, bacteriophage could potentially be used as a new type of treatment for bacterial infections. These viruses can recognize and kill human bacterial infections without destroying our own cells. All viruses are surrounded by a capsid, or viral envelope made of proteins. This capsid can be made of different types of proteins, and it can come in all different shapes, increasing the variety of these viruses. All viruses target a specific strain or species of bacteria and won't infect other species. Proteins in a viral capsid will recognize specific proteins or carbohydrates on the surface of the bacterial host cell wall that it's able to infect. All viruses also contain nucleic acid within this capsid. This nucleic acid can be double-stranded or single-stranded DNA or RNA. Unlike most other viruses, bacteriophages have special tail fibers that aid in injecting the viral nucleic acid into the bacterial cell like a syringe. Because a virus can't replicate on its own, it requires a host. A bacteriophage recognizes a specific host and attaches to the bacterial cell wall. Proteins or carbohydrates on the outside of the bacteria serve as markers for bacteriophages to recognize and bind their targets. Next, using its tail fibers, it injects its nucleic acid into the bacterial host. At this point, the virus can enter one of two possible cycles. The first is the lytic cycle. In the lytic cycle, bacteriophage will replicate and cause the bacterial cell to burst, or lyse, to release newly assembled phage. After injecting its nucleic acid into a bacterium, a phage will direct degradation of host DNA. In this way, it can ensure that its own nucleic acid is the only set of instructions in the host cell. All of the cell's resources can then be directed towards making more virus, not towards making more bacteria. In the next step, the bacteriophage will completely take over the cell machinery to replicate its nucleic acid, transcribe viral RNA, and translate viral proteins. These new nucleic acid copies and viral proteins will be assembled into brand new bacteriophage within the cell. The last step of the lytic cycle is release of the viruses by lysing the bacterial cell wall, so that the new viruses can go on to infect more hosts. A bacteriophage may not be alive, but its success as a parasite has relied on some very lifelike qualities. When it attaches to the cell and injects its nucleic acid, some bacteriophages must make a decision. Should it replicate and lyse the cell, or stick around for a while? In the lytic cycle, the host cell obviously dies, and the viruses must find new hosts. This might be the best course of action if the bacterial host is under some kind of stress. If a bacterial host is thriving, however, a virus might decide to move in. And why not? Imagine you had a friend that was an excellent host, providing good food, company, entertainment, and a place to sleep. You might not want to leave either. If a bacterial host is growing and dividing, this gives a virus an opportunity to grow and divide within the cell too. Here the bacteriophage would attach to its host, inject its nucleic acid, and then enter the lysogenic cycle. In the lysogenic cycle, phage nucleic acid will integrate into the bacterial genome. The term genic in lysogenic can remind you that the phage genes are incorporated into the bacterial genes, a step that does not occur in the lytic cycle. The prophage is the phage DNA that is integrated into the bacterial DNA. Every time the bacterial cell replicates its DNA and divides into two new cells, it will also replicate the viral genetic information and pass this on to new cells. The virus has now infected two hosts without any additional effort. This will continue to happen through multiple generations until the bacterial host is no longer hospitable. At this point, some bacteriophage are triggered to enter the lytic cycle. In summary, we've learned that viruses are non-living parasitic particles made of nucleic acid surrounded by a capsid made of protein. Bacteriophages are viruses that infect bacteria. After recognizing and attaching to a specific host, bacteriophages may replicate by the lytic cycle or the lysogenic cycle. In the lytic cycle, bacteriophages degrade the bacterial DNA and use bacterial resources to reproduce, eventually lysing the cell to release newly assembled bacteriophages. In the lysogenic cycle, bacteriophage nucleic acid integrates into the bacterial genome. This prophage, or phage DNA inserted into bacterial DNA, is then replicated with the bacterial DNA and passed down to dividing cells. The bacteriophage will lie dormant in the lysogenic cycle unless it is triggered to enter the lytic cycle, possibly by sensing that the bacterial cell is stressed and no longer the best host.
Do you see a difference between this guy and this guy Ever wonder how someone has the energy to run a marathon? What about being able to push themselves when they're exhausted and can't breathe? It's all due to something called cellular respiration. This is the process that living organisms use to turn food into the energy they need for survival. Cellular respiration can be summarized by the equation. Organic compounds plus oxygen yields carbon dioxide plus water and energy. So what does all this mean? Some organisms eat plants. Some organisms eat animals. Some organisms even make their own food, like in the case of plants and some bacteria. However, what we all do with that food is the same. Food contains important chemicals called organic compounds. You may remember that carbohydrates, lipids, and proteins are all organic compounds. Nucleic acids are also organic compounds, but they don't come from food, so we won't worry about them right now. These chemical compounds have energy stored in their bonds that your body wants and needs. So your body needs to extract that energy and turn it into a usable form. That usable form is the molecule ATP, or adenosine triphosphate. ATP is the form of energy used by all living things. It's composed of a ribose sugar, an adenine base, and 3 phosphates. 1, 2, 3. The easiest way to think about ATP is like a rechargeable battery. When ATP is charged and energized, it has 3 phosphates. But in order for your body to use ATP energy, it must break the bond between the last 2 phosphates. Breaking that bond releases the energy your body needs for survival. However, this chemical reaction also changes ATP into ADP, adenosine diphosphate. ADP is like a rechargeable battery that has lost its charge. To recharge ADP, your body must convert food to energy through this process of cellular respiration. So what's the difference between this guy and this guy? There are 2 types of cellular respiration: aerobic cellular respiration and anaerobic cellular respiration. Maybe you made the connection that aero sounds like air and therefore must have something to do with oxygen. Aerobic cellular respiration is a process that requires oxygen to convert food into ATP. Or maybe you recognize the prefix "an." The prefix "an" means without, so anaerobic cellular respiration is without oxygen, or a process that does not require oxygen to convert food into ATP. Anaerobic cellular respiration versus aerobic cellular respiration is the big difference between this guy and this guy. This guy looks exhausted and out of breath. As a result, he's probably not getting enough oxygen, causing his body to use anaerobic cellular respiration to make energy. Unfortunately, anaerobic cellular respiration makes very little ATP, only adding to his exhaustion. Meanwhile, this guy looks like he's in the zone and probably has a breathing rhythm to make sure he gets enough oxygen for his body to perform aerobic cellular respiration. Aerobic cellular respiration will provide him with the ATP that he needs to keep going. So let's recap. All living things perform cellular respiration. Cellular respiration is the process of converting organic compounds, also known as food, into ATP, or energy. ATP, also known as adenosine triphosphate, is the molecule all living things use to store energy. ATP is like a rechargeable battery. When the battery loses its charge, in the form of a phosphate, it becomes ADP, or adenosine diphosphate. However, since ATP is rechargeable, cellular respiration can turn that uncharged ADP into a charged ATP. In humans, how much charged ATP gets created is a question of whether or not a person has enough oxygen to perform aerobic cellular respiration. When you have plenty of oxygen, your body performs aerobic cellular respiration and makes plenty of ATP. But when you don't have enough oxygen, your cells switch to anaerobic cellular respiration and make very little ATP.
We have learned previously that energy production is an extremely important cellular task. Failure to produce enough energy in the form of ATP can result in fatigue, among other things. It's the difference from being the runner whose body is still performing at a high level and the runner who's gassed. Let's explore how our cells try to keep our bodies from running out of energy by learning about the chemical reactions that contribute to making ATP. During cellular respiration, oxygen reacts with an organic compound to produce carbon dioxide, water, and energy. This seems like a pretty generalized formula for chemical reactions. So let's see if we can make it a little more specific and chemical-like. The sugar glucose is the main fuel source for cellular respiration. So let's replace organic compound with the chemical formula for glucose, which is C6H12O6. If you recall, we also know that ATP is the molecule that is the currency for energy in cells. Cellular respiration converts ADP into ATP, so let's add that to our cellular respiration equation as well. This is the basic formula that describes cellular respiration. We shall soon see how the cell uses a series of redox reactions to break down glucose to release energy. That energy is used to change ADP into ATP that can be used to power biological processes throughout the cell. Recall that a redox reaction is simply shorthand for an oxidation-reduction reaction. That means that during cellular respiration, some molecules in our cellular respiration chemical reaction will be oxidized and some will be reduced. What exactly does that mean? Well, remember our mnemonic aid, Leo the lion says, grr. A molecule that is oxidized loses electrons, and a molecule that is reduced gains electrons. Well, that's a super mnemonic reminder, but what does it mean in terms of cellular respiration? How is a redox reaction going to help make ATP? There's a lot of energy stored in the bonds between the carbon and hydrogen atoms in glucose. During cellular respiration, redox reactions basically transfer this bond energy in the form of electrons from glucose to molecules called electron carriers. So an electron carrier is basically a molecule that transports electrons during cellular respiration. By using electron carriers, energy harvested from glucose can be temporarily stored until the cell can convert the energy into ATP. Two molecules that serve this role are NAD and FAD. NAD stands for nicotinamide adenine dinucleotide. It is one of the major energy carriers during cellular respiration. Let's see how NAD is able to store energy for a cell during cellular respiration. Recall that we are going to store energy in NAD by adding electrons to it. That means that the NAD molecule can exist in either an oxidized or a reduced form. NAD+ is the oxidized form of NAD. When NAD+ reacts with two hydrogen atoms, two electrons can be added to the NAD+ molecule, resulting in a NADH molecule and a proton, or H+. This equation may seem a little confusing, but let's break it down further to see if it makes more sense. A hydrogen atom consists of a proton and an electron. So we can rewrite the two hydrogen atoms as 2H+ and 2E-. In the reduction reaction, NAD+ accepts the two electrons and one of the protons to form a neutral NADH molecule. That leaves a free proton as the second product of the reaction. FAD is a second electron carrier used by a cell during cellular respiration. It stands for flavin adenine dinucleotide. Like NAD, FAD can temporarily store energy during cellular respiration via a reduction reaction. When FAD reacts with two hydrogen atoms, it can form FADH2. Both NAD and FAD play a crucial role in cellular respiration to temporarily store energy as it's released from glucose. Transferring the electrons NAD and FAD are carrying during an oxidation reaction releases the stored energy that was harvested from glucose. NADH is oxidized back to NAD+, and FADH2 is oxidized back to FAD. This stored energy can then be used to generate ATP, the basic energy currency of a cell. As we learn more detail about the cellular respiration process, we will see specifically how and where these reactions take place within a cell. In summary, cellular respiration is the biological process in which organic compounds are converted into energy. A redox reaction is shorthand for oxidation-reduction reaction, and is a chemical reaction in which one molecule loses electrons while another molecule gains electrons. An electron carrier is a molecule that transports electrons during cellular respiration. NAD is an electron carrier used to temporarily store energy during cellular respiration. This energy is stored via the reduction reaction NAD+ plus 2H yields NADH plus H+. FAD is another electron carrier used to temporarily store energy during cellular respiration. This energy is stored via the reduction reaction FAD plus 2H yields FADH2. The energy stored in NADH plus H plus and FADH2 will be released later in cellular respiration to generate ATP, which can be used as an energy source throughout the cell.
When we're feeling tired or lethargic, sometimes all we need is some good food and some fresh air. A little outdoor picnic not only lifts our spirits, it also literally energizes us, as well as our cells. For a few lessons, we've done some dancing around cellular respiration, which is the process that converts food into chemical energy. All of our cells are constantly performing cellular respiration for us, and we can be reminded of this every time we sit down for a picnic and breathe in the open air. This air is chock full of oxygen. In this lesson and future lessons on cellular respiration, we'll walk through the steps that our cells take to use components in the food we consume and the air we breathe. But first, let's take a step back for a reminder on the components of cellular respiration. The fancy chemical formula for cellular respiration is C6H12O6 plus 6O2 plus 32 ADP yields 6H2O plus 6CO2 plus 32 ATP. C6H12O6, otherwise known as glucose, comes straight from the blueberry pie or whatever else is on your picnic plate. It represents the organic compounds that are derived from our food. O2, or oxygen, of course, is in the air that we breathe. You'll also remember that cellular respiration yields water and carbon dioxide. Carbon dioxide is removed from our bodies with every exhale. Now, let's take a second to review the final reactant in this equation, ADP, or adenosine diphosphate. An ADP molecule has two phosphate groups. You'll recall that this ADP is like an uncharged battery. The big goal of cellular respiration is to charge ADP to its fully energized form, otherwise known as the product ATP, or adenosine triphosphate. ATP is simply an ADP molecule with a third phosphate molecule. Take a good look at this chemical structure because this is what cellular respiration is all about. ATP is the chemical energy that cellular respiration strives to make. It's essentially the chemical currency of life used to pay for all the reactions that cost energy in the cell. It's the energy that we get from our little picnic outdoors. The last player in this chemical process we should remind ourselves of is not included in this equation. In our cells, we also have nicotinamide adenine dinucleotide, or NAD+, which is a coenzyme. NAD+ collects electrons and carries them to another location. When NAD+ picks up an electron, it becomes reduced, and is now represented as NADH. Now, creating ATP through cellular respiration is a long process. It occurs in 3 stages: glycolysis, the citric acid cycle, and the electron transport chain. In this lesson, we'll focus on what happens during glycolysis and how this feeds into the other stages. Glycolysis is the first stage in cellular respiration and happens in the cytoplasm. So to combine our players with the process, glycolysis is the first stage of cellular respiration and uses the following molecules: glucose, NAD+, ATP, and ADP. Glucose is a 6-carbon sugar. We can represent glucose as a 6-carbon sugar by using 6 blue circles, 1 blue circle for each carbon. In glycolysis, glucose is broken down using 10 enzymatic reactions to produce 2 3-carbon molecules of pyruvate. Essentially, glucose is split in half and rearranged a bit. In this process, we come away with some valuable products. Instead of going through the nitty-gritty of all the enzymes and chemical reactions involved, let's focus on some of the steps and how they get us to the next stage of cellular respiration. Importantly, you need to know that we use 2 ATP molecules to perform glycolysis. It may sound sort of funny that we need to spend chemical energy to make chemical energy, but think of it like investment of money that grows over time. You need to spend some chemical money to make some. Glucose is transported into the cytoplasm of our cells. Here, it's rearranged and receives 2 phosphate groups from 2 different molecules of ATP, which then become ADP. Here, the addition of phosphate groups is represented by these 2 red circles. This is the energy investment. This new 6-carbon molecule is then divided into 2 3-carbon sugars, each containing a phosphate group. These 2 molecules are identical, and what happens next happens to both molecules. One enzyme brings 1 NAD+ electron carrier and 1 phosphate group to each 3-carbon molecule. Each electron carrier picks up electrons, becoming reduced as NADH. The phosphate group is added to the molecule. In subsequent steps, 2 ADP molecules per 3-carbon molecule come by to pick up all the phosphate groups. This creates 4 ATP molecules total, or 4 units of chemical energy, and leaves us with 2 molecules of 3-carbon pyruvate. Now let's tally what happens during glycolysis, the first stage in cellular respiration. 1 glucose molecule is broken down into 2 molecules of pyruvate in the cytoplasm. In the process of creating 2 pyruvates, 2 molecules of ATP are used, but 4 are created. Therefore, our net chemical energy from glycolysis is 2 molecules of ATP. 2 NAD+ electron carriers collected electrons, producing 2 NADH+ proton molecules. The pyruvate will move on to the next part of this process, which is the citric acid cycle. The ATP, of course, is usable energy for the cell. The NADH+ proton molecules will be used later in cellular respiration during the electron transport chain to help our cells make even more ATP. At this point, we've taken the first step to using the food from our picnic and turning it into chemical energy. In the next 2 stages of this process, we'll see how we can use oxygen and electron carriers to create even more ATP to energize our cells. In previous lessons, we started to learn about cellular respiration, the process which turns food into chemical energy. We learned how this process begins to use the food we eat in the air we breathe. It's a 3-phase process, beginning with glycolysis, followed by the citric acid cycle, and finally the electron transport chain. In this lesson, we'll learn how the products of glycolysis feed into the citric acid cycle, and how the products of the citric acid cycle ultimately end up with the products of glycolysis in the electron transport chain. In glycolysis, you remember that we broke down sugary glucose molecules from the food at our picnic. 1 glucose molecule in glycolysis became 2 3-carbon sugars, called pyruvate. During this process, we also netted 2 ATP molecules, or units of chemical energy, as well as 2 NADH+ proton molecules, or electron carriers. Now, before we can get to the next stage of cellular respiration, the citric acid cycle, there's some prep work that needs to be done. Before you barbecue your steaks at the picnic, you probably want to thaw and season them first, to get them ready to go. So let's get those products of glycolysis ready for the grill. See, each pyruvate, which is produced in the cytoplasm, enters the mitochondria to be converted into acetyl coenzyme A. Remember that 2 pyruvates are created from glycolysis, meaning 2 acetyl coenzyme A molecules are produced. Acetyl coenzyme A is a 2-carbon molecule. Are you wondering where the 3rd carbon from pyruvate went? Well, it's found in a second product of the reaction as a carbon dioxide molecule, which eventually diffuses out of the cell and into your bloodstream. It's part of the same carbon dioxide that you exhale. This preparation for the citric acid cycle is called pyruvate oxidation because the pyruvate is oxidized or loses electrons to form NADH plus protons. 1 NADH plus proton is produced per pyruvate. This brings our total for this reaction to 2 acetyl coenzyme A's, 2 carbon dioxide molecules, and 2 NADH plus protons. This reaction links glycolysis to the citric acid cycle. With 2 acetyl coenzyme A inside the mitochondrial matrix, we are finally able to start the steps of the citric acid cycle, or second stage of cellular respiration. The citric acid cycle is also known as the Krebs cycle. No matter what you call it, you'll notice the name fits the bill. This stage of cellular respiration is a cyclical process of 8 different chemical reactions. While all the reactions that occur are important, in this lesson we're just going to focus on the reactions that are essential to create products that are important to the next phase of cellular respiration. To start, oxaloacetic acid, a 4-carbon molecule, combines with acetyl coenzyme A from pyruvate oxidation. The coenzyme A molecule separates, donating the acetyl group to oxaloacetic acid so that it becomes a 6-carbon molecule. This is called citric acid. Do you see where the citric acid cycle got its name? Citric acid is oxidized by the electron carrier NAD+. In turn, NAD+ is reduced to become NADH plus protons. This reaction also releases a carbon dioxide molecule and turns the 6-carbon citric acid molecule into a 5-carbon molecule. Another reaction produces the same result, creating a 4-carbon molecule, carbon dioxide, and NADH plus proton. Remember that this carbon dioxide is the same carbon dioxide that we exhale, just like the carbon dioxide made earlier during pyruvate oxidation. In a later step, this 4-carbon molecule undergoes a change that eventually ends up converting an ADP molecule to ATP. This is the only citric acid step that releases chemical energy. Through the last few steps, this 4-carbon molecule is further oxidized by 1 more NAD+ and 1 FAD, another type of electron carrier, to become NADH plus proton and FADH2. This 4-carbon molecule that results is familiar. We eventually end up back where we started. What goes around comes around, right? So this cycle recreates the 4-carbon oxaloacetic acid, which is ready to collect the second acetyl coenzyme A from pyruvate oxidation and re-enter the citric acid cycle to complete the reactions again. The wheel will keep turning twice for each glucose molecule that entered the cell from whatever you ate for dinner today. Okay, so now for every glucose from the food we enjoy, we've taken 2 rides around the citric acid cycle, the second stage of cellular respiration, using products from glycolysis. Let's see where we stand for the final stage of cellular respiration, the electron transport chain. 2 molecules of pyruvate were produced from glycolysis and converted into 2 molecules of acetyl coenzyme A, 2 carbon dioxide, and 2 NADH plus proton molecules through pyruvate oxidation, an intermediate step between glycolysis and the citric acid cycle. Each acetyl coenzyme A proceeded once through the citric acid cycle. Therefore, in total, it created 6 NADH plus proton molecules, 2 FADH2 molecules, 4 carbon dioxide molecules, and 2 ATP molecules. That's a lot of products. Perhaps it didn't create a lot of chemical energy, but our reduced electron carriers from this stage of cellular respiration have picked up lots of energetic electrons for the last step, the electron transport chain, to help our cells create plenty more energy from our food. So just stay tuned. Now, before you go to any big show, there's some preparation to be done. You can think of the steps of cellular respiration as the opening acts to the main event. We've been doing a dance of cellular respiration for a few lessons now, building up to the finale. Remember that cellular respiration is the process that converts food into chemical energy. In this spectacular show of how our cells perform this process, we opened up with glycolysis, followed by the citric acid cycle. Collectively, these two acts of cellular respiration get us ready for the main event, the electron transport chain. If you were an electron, this would easily be the most fun you've ever had, and we'll show you why. But first, let's recap. In cellular respiration, our cells use glucose and oxygen to produce water, carbon dioxide, and energy. And this is in the form of ATP. Remember that we get glucose from the food we eat, like from that delicious pie from that family picnic we attended when we were learning about cellular respiration. In glycolysis, the sugar glucose was broken down into 2 pyruvate or 3-carbon sugars. These pyruvate molecules are further modified through pyruvate oxidation before they enter the citric acid cycle. The products of both glycolysis and the citric acid cycle combined totaled 4 net ATP molecules and 6 carbon dioxide molecules. You'll notice this doesn't yet account for all the products of cellular respiration. We still have to make water, as well as another 28 molecules of ATP. You'll also notice we haven't yet used the oxygen we breathe for this process. However, if you remember, the steps of glycolysis and the citric acid cycle collectively oxidized carbon molecules, transferring electrons to electron carriers, and produced a whopping total of 10 NADH plus protons and 2 FADH2 molecules. Now these reduced electron carriers are poised to donate these electrons to the fireworks of the show, the electron transport chain. If these electrons were all actors in cellular respiration, this would be their time to shine. The electron transport chain is the third stage of cellular respiration. 4 protein complexes in the inner mitochondrial membrane form the electron transport chain. These complexes exist in a descending order of energy. Here, the electron carriers come along to drop off all their electron and proton cargo that they picked up during the glycolysis and citric acid cycle stages. NADH plus protons and FADH2 become oxidized, donating electrons to the first and second protein complex respectively. These complex proteins now become electron carriers themselves and are now reduced. They become oxidized as they pass these electrons down the electron transport chain. You can think of this like someone taking a slinky and dropping it onto the first step of a staircase. The slinky will continue to move down the steps just like an electron moves down energy levels until it hits the bottom. Now as the electrons are moving down the stairs, the protons donated from NADH plus protons and FADH2 are also put to good use. They are pumped to the other side of the inner mitochondrial membrane through the protein complexes 1, 3, and 4. They are moved by active transport from the mitochondrial matrix to the space between the inner membrane and outer membrane of the mitochondria. They do this by using the energy derived from the electrons that flow down the electron transport chain stairs. The last stair of the electron transport chain is oxygen. You're wondering when we're going to use that, right? Well, a single oxygen molecule accepts 2 electrons and 2 protons from the final protein complex. This produces a molecule of water. Why do all of us need oxygen? To complete cellular respiration. Pumping all these protons outside across the inner membrane of the mitochondria creates a high concentration of protons between the inner and outer mitochondrial membranes, and therefore a concentration gradient of protons. In the last step of the electron transport chain, an enzyme called ATP synthase is used. This is a channel protein in the inner mitochondrial membrane. The protons flow through this pump at max speed back into the mitochondrial matrix. This causes the part of an enzyme to spin in circles like a whirling dervish. This spinning motion provides the final dance and song number of cellular respiration. ATP synthase catalyzes the phosphorylation of ADP, creating the last 28 molecules of ATP. And how's that for a final act? Let's bring all this back to our formula for cellular respiration in order to summarize the reactants and products from the process as a whole. 1 glucose molecule was broken down in glycolysis to net 2 ATP molecules and electron carriers. Pyruvate oxidation and the citric acid cycle produced 2 more ATP molecules, more electron carriers, and 6 molecules of carbon dioxide. Finally, in the electron transport chain, The electron carriers were used to donate electrons and protons that turned oxygen molecules into water and created the remainder of the 32 ATP molecules, all from one glucose molecule. In this lesson, we learned how we took the products of glycolysis and the citric acid cycle and used them in the electron transport chain, the third stage of cellular respiration. Electron carriers are reduced during glycolysis and the citric acid cycle to NADH plus proton and FADH2. These carriers then donate electrons and protons to the electron carrier proteins of the electron transport chain. The final electron acceptor is oxygen. Together with oxygen, electrons and protons forms molecules of water. In addition, protons are actively pumped to the other side of the inner mitochondrial membrane, creating this proton gradient. These protons flow through ATP synthase, a channel protein that uses this power to phosphorylate ADP to make ATP. The electron transport chain, therefore, produces 28 ATP molecules as well as water. Your body's cells normally undergo aerobic cellular respiration, or a process which uses oxygen to convert food into energy. However, there are times when cells undergo anaerobic cellular respiration, or a process that does not use oxygen to convert food into energy. Your muscle cells, when deprived of air, will use anaerobic cellular respiration. Yeast, which is an organism used to make beer and wine, will follow a similar pathway when deprived of oxygen too. Before we delve into how this process works, let's first review cellular respiration as a whole. Remember that in aerobic cellular respiration, there are three stages: glycolysis, the citric acid cycle, and the electron transport chain. In glycolysis, a net of 2 molecules of ATP, or chemical energy, are produced. The citric acid cycle produces another 2 molecules, while the electron transport chain produces a whopping 28 molecules of ATP. Oxygen is used in aerobic cellular respiration as the final electron acceptor in the electron transport chain, which is part of why it's able to create so much ATP. But what happens when oxygen doesn't exist? Like when you're running so hard that you can't catch your breath during the game. Well, the electron transport chain can then no longer be used because there's a shortage of oxygen. Instead, in anaerobic cellular respiration, the only step of this process that occurs is glycolysis. This is unfortunate because ATP is like the chemical currency of the cell, and we lose the ability to pay for all the cellular processes when we run out of ATP. Glycolysis only provides 2 molecules of ATP. By skipping the final 2 steps of this process, we lose out on a lot of cash. However, sometimes some energy is better than no energy at all. Therefore, in anaerobic conditions, some organisms perform fermentation, which is a process that anaerobically generates ATP by performing glycolysis and one extra step. There are 2 types of fermentation we'll discuss in this lesson: lactic acid fermentation and alcoholic fermentation. Glycolysis occurs in the cytosol. Along with 2 ATP, it also produces 2 NADH plus proton electron carriers and 2 3-carbon pyruvate molecules. Normally, the pyruvate molecules would continue on to the citric acid cycle, while the electron carriers would continue on to the electron transport chain. Here, these electron carriers would drop off their electrons and become oxidized back to NAD+. Then, NAD+ molecules would be able to return to glycolysis to perform cellular respiration all over again. But when there is no electron transport chain to go to, what is a poor electron carrier to do with its electrons? Let's look specifically at how this happens in your muscle cells when you're playing such a good game of defense on the basketball court. During high activity, your blood is not shuttling oxygen quickly enough and you're out of breath. You still want to be able to move, so your body turns to anaerobic cellular respiration. In some animal cells and microorganisms, cells can perform lactic acid fermentation. In this process, pyruvate is reduced by NADH plus proton. In doing so, the electron carriers are then returned to their NAD plus state so that they may re-enter this glycolysis cycle. Meanwhile, pyruvate is converted to the 3-carbon molecule lactic acid, or lactate. Yeast fermentation is used to make wine and beer, as well as bake bread. This process relies on a similar principle as lactic acid fermentation. Under anaerobic conditions, these cells can't go through the citric acid cycle and electron transport chain. Therefore, they also need to oxidize their electron carriers after glycolysis. In alcoholic fermentation, pyruvate is converted to a 2-carbon molecule called acetaldehyde. The third carbon is released as carbon dioxide. This is a lovely side effect that adds bubbles to beer and makes bread rise. Meanwhile, acetaldehyde is reduced to ethanol or ethyl alcohol, while the electron carriers NADH plus proton are oxidized. This returns them to NAD+, which can then re-enter glycolysis. In this lesson, we learned how some cells and organisms perform anaerobic cellular respiration, or a process that converts food into energy without oxygen. Oxygen is normally the final electron acceptor in the electron transport chain. Without oxygen, these cells only undergo glycolysis and produce 2 ATP per glucose molecule. Next, they need to reduce pyruvate to regenerate NAD+ from NADH plus proton. Fermentation is the process that performs this job. In lactic acid fermentation, pyruvate is reduced to lactic acid. This occurs in cells such as our muscles when we're exercising. In alcoholic fermentation, pyruvate is reduced to alcohol and releases carbon dioxide. This type of fermentation is commonly used with yeast to make alcoholic beverages and cause bread to rise. If you've looked outside today or even around your house, you've likely seen a plant. Trees, potted plants, ferns, grass, weeds, flowers, maybe if you're lucky some palm trees. There is plenty of plant diversity on the planet that we often don't take enough to appreciate on a daily basis. It's also likely you haven't taken a minute today to thank a plant for something vital it's giving to you right this second. While the rest of us need to eat to get our energy, plants have the awesome ability to make their own food simply by soaking up some sunshine and taking a drink of water. And that is pretty amazing if you think about it. Plants, as well as some bacteria and protists, perform photosynthesis, or a process that converts energy from sunlight into food. No eating required to get the energy they need for the day, just a healthy dose of sunlight, carbon dioxide, and water. The chemical equation for photosynthesis is 6CO2, or carbon dioxide, plus 6H2O, or water, yields C6H12O6, which is glucose, and 6 plus O2, or oxygen. If you've already learned about cellular respiration, or the process that converts food into chemical energy, then this formula should look pretty familiar to you. In fact, photosynthesis is essentially the reverse of cellular respiration. Photosynthesis uses the carbon dioxide you breathe out, along with sunshine and water, to create sugars and expel oxygen. Because as we all know, you and I both appreciate using that sugar and oxygen to perform cellular respiration. It's what keeps us alive. And so here it is, folks. This is the circle of life. So, have you thanked a plant today for the fresh air you breathe in? In this lesson, we'll start to investigate one of the major players in this process that is contained within the plant chloroplasts, or organelles which are the site of photosynthesis. Chloroplasts reside in most leaf cells. Both chloroplasts and the leaves themselves are different shades of green due to a lovely pigment they contain. A pigment is a compound that absorbs a particular wavelength of visible light. Chlorophyll is a green pigment contained in thylakoid membranes of chloroplasts that is used in photosynthesis by plants. In bacteria that also perform photosynthesis, chlorophylls are in the plasma membrane. So you might be wondering, how does the pigment absorb light to fuel photosynthesis? First, we need to talk about light itself. The electromagnetic spectrum is the range of wavelengths from the radio waves of your car stereo to high-energy gamma rays. Chlorophyll and other pigments absorb specific visible wavelengths of the electromagnetic spectrum. The visible light is literally the colored wavelengths that are visible to your eyes. They range from red at a wavelength of 700 nanometers to blue at a wavelength of 400 nanometers. Shorter wavelengths have higher energy, such as blue, and longer wavelengths like red have lower energy. Chlorophyll mostly absorbs blue and red wavelengths from sunlight. Therefore, it reflects the middle part of this visible spectrum, the green wavelengths. This is why plants look green to us. The color of a pigment is the wavelength of light that is reflected or the wavelength that is not absorbed. Chlorophyll contains electrons that are normally at a low energy state or ground state, but when chlorophyll absorbs light energy, the electrons get really excited. Literally, they jump to a high energy level or excited state. Although we'll save the details of this process for other lessons, this superexcited electron is then transferred from the chlorophyll pigment to a primary electron acceptor. Hence, chlorophyll becomes oxidized. Photosynthesis can use both chlorophyll pigments and other accessory pigments that reflect colors other than green. These accessory pigments aren't exactly the jewelry type accessories of photosynthesis, but they are as helpful as the right pair of shoes to complete an outfit. These accessory pigments absorb other wavelengths of light and pass on energy to chlorophyll, expanding the range of wavelengths that can be used for photosynthesis. In summary, photosynthesis is the process that converts energy from sunlight into food in plants and some protists and bacteria. Photosynthesis is essentially the reverse of cellular respiration, where our bodies convert food into usable energy. Photosynthesis is driven by the use of a pigment, or molecule that absorbs a specific wavelength of energy. In this case, the primary pigment is chlorophyll, which is a pigment that absorbs blue and red wavelengths while reflecting green wavelengths. Chlorophyll exists within the thylakoid membranes of plant chloroplasts, and that's what makes plants green. In photosynthesis, sunlight energizes electrons in chlorophyll, moving them to a higher energy state. In turn, chlorophyll molecules become oxidized, and these electrons are used in the next steps of photosynthesis. When plants start to get a little hungry, they need to only turn their leaves to the sun to start photosynthesis, the process that turns energy from sunlight into food. In this lesson, we'll start to break down how this process takes place. Photosynthesis consists of two parts. The first part of this process consists of the light reactions, while the second is called the dark reactions. This might seem like two sides of something like Star Wars good versus evil, but we promise that everything is good when it comes to photosynthesis. It's just that the light-dependent reactions use sunlight and the dark reactions don't use sunlight. Before we begin on how the light reactions work, let's remind ourselves of the products and reactants for photosynthesis. In this process, remember that light energy, carbon dioxide, and water are used to produce glucose or sugar and oxygen. In this lesson, we'll learn how two of these reactants are used. Specifically, we'll learn how light energy is used to split water, a process known as photolysis. Inside a plant cell, inside a chloroplast, and within a thylakoid membrane, there are photosystems, which are the sites of the light-dependent photosynthesis reactions. There are two types of photosystems, photosystem 1 and photosystem 2. Photosystem 2 is actually used before photosystem 1, but they are numbered for the order they were discovered. Photosystems contain an antenna complex. The antenna complex contains the pigment chlorophyll A, and it's what makes them green. Photosystems also have accessory pigments of other colors. Pigments, you'll remember, absorb specific wavelengths of light. In photosystem 2, chlorophyll molecules are called P680 because they optimally absorb light at this wavelength, while in photosystem 1, they are P700. In these photosystems, pigments pass the energy absorbed from light to the reaction center. This is in the middle of the photosystem. The reaction center contains a special pair of chlorophyll molecules coupled with proteins. Here comes the exciting part. Literally, when the light energy hits the reaction center, electrons from the chlorophyll molecules are excited to a higher energy level. Imagine light energy jumping on a trampoline along a photosystem and then, boom, landing straight in the center, sending electrons up in the air, handed to a primary electron acceptor. Now, what goes up must come back down, and these energized electrons come back down in energy levels by moving through an electron transport chain. You might remember the electron transport chain from cellular respiration. The electron transport chain in photosynthesis is similar to this. Electron acceptor proteins reside within the thylakoid membrane. These proteins also become reduced as they accept electrons and oxidize as they pass electrons down this chain. The primary electron acceptor passes the electrons down to these membrane proteins as they decrease in energy level until they reach photosystem 1. Meanwhile, the electron transport chain of photosynthesis, just like cellular respiration, creates a proton concentration gradient that makes ATP, or chemical energy, as protons move through an ATP synthase. When the electrons leave the chlorophyll molecules, it leaves behind a hole. This electron hole is filled in by a water molecule that is oxidized or loses electrons as it essentially splits into two hydrogen atoms, or protons, and an oxygen atom. Two oxygen atoms combine to form one molecule of oxygen gas that gets released as a waste. Of course, this is waste to the plant, but it's gold to us, and we couldn't live without it. Meanwhile, the new electrons from the split of water molecule are next in line to be bounced up to the primary electron acceptor. The protons that are released help create the proton gradient in the thylakoid lumen, important to making ATP. When an electron from photosystem 2 hits the reaction center of photosystem 1, the energy captured from the pigments in this photosystem are used to bounce these electrons to a higher energy state, where they then fall down the second electron transport chain. Here the electrons ultimately land on the molecule NADP+, which is an electron carrier similar to the electron carrier NAD+, used in cellular respiration. NADP+ becomes reduced to NADPH. These reduced electron carriers can then continue on with these electrons onto the second stage, the dark reactions, otherwise known as the Calvin cycle. In this lesson, we learned about the light reactions of photosynthesis. These reactions use photolysis, or the use of light energy to split water molecules and produce oxygen. In these light-dependent reactions, light energy is absorbed by chlorophyll and other pigments and transferred to the reaction center of photosystem 2. Here, electrons are excited to a higher energy level and passed down an electron transport chain to photosystem 1. When these electrons land on the reaction center of photosystem 1, they are excited again by light energy and are passed on to a second electron transport chain. Here, NAD+ is the final electron acceptor, which becomes reduced to NADPH. Meanwhile, ATP is also formed. Therefore, these light reactions release oxygen as a waste product of photosynthesis, while ATP and NADPH are essential to the next step in photosynthesis, the dark reactions.
Photosynthesis is the process that converts energy from sunlight into food. Photosynthesis feeds the world, taking rays of sunlight and turning it into food on your plate with just a few steps in between. Remember that in photosynthesis, light energy, carbon dioxide, and water are used to produce glucose, or sugar, and oxygen. After shedding some sunlight on how photosynthesis begins with the light reactions, we're now ready to learn the second part of photosynthesis, called the dark reactions, otherwise known as the Calvin cycle. This step is also sometimes referred to as the Calvin-Benson cycle, as it gets its name from the scientists who discovered it. While sunlight is used during the light reactions to produce ATP and the electron carrier NADPH, the dark reactions use these products to complete photosynthesis without the use of light energy. The light reactions produce ATP and NADPH in the thylakoids of chloroplasts. These products are released into the stroma of the chloroplast. Here the Calvin cycle can take off, using carbon dioxide from the air to create sugars. This is the synthesis part of photosynthesis, where the plants will finish cooking up some food with the help of a little carbon dioxide. The key to creating sugars comes from carbon fixation, where inorganic carbon is fixed into organic carbon, such as sugars. In the Calvin cycle, carbon dioxide is fixed into an organic molecule through the help of an enzyme called rubisco. Here, the carbon dioxide that we exhale as waste is used by plants performing photosynthesis. Let's highlight some of the steps of the Calvin cycle. Rubisco joins 3 carbon dioxide molecules with 3 5-carbon molecules that each have 2 phosphates, called RUBP. 6 3-carbon molecules are created, each with 1 phosphate. 6 ATP molecules are then used, donating another phosphate to each molecule. Next, 6 NADPH electron carriers from the light reactions carry electrons to the Calvin cycle. They are oxidized to NADP+, donating electrons to the carbon molecules, which become reduced. This results in 6 superenergetic 3-carbon molecules, called glyceraldehyde 3-phosphate, or G3P, for a 3-carbon molecule with 1 phosphate group. One of these lucky molecules gets used to create glucose, or other carbon molecules, while the other 5 molecules are rearranged with the aid of 3 ATP to create 3 RUBP molecules that begin the cycle again. This is the regeneration of RUBP that allows the Calvin cycle to continue. The G3P molecule created that exits the cycle is the most important. 2 G3P could create glucose. This is the building block of creating everything else in the plant and what fuels every other organism that eats plants. Let's go back to that first stage of the Calvin cycle. In this stage, the 3 carbon dioxide molecules join the 3 5-carbon molecules, each having the 2 phosphates. But instead of just picking up the right amount of carbon dioxide and making it all into 5-carbon molecules, proceeding neatly with the Calvin cycle, sometimes the Rubisco enzyme acts like it has a mind of its own. So instead, it picks up oxygen, initiating a process called photorespiration. Photorespiration is a side reaction that sometimes occurs during the Calvin cycle. It undoes the good things that are usually done by photosynthesis because it makes carbon that has already been fixed into organic compounds turn back into the carbon dioxide from whence it came. This then decreases the amount of glucose that is synthesized. The trouble all begins with Rubisco, which is a fickle enzyme because it can bind with either carbon dioxide or molecular oxygen. But what determines which of these it will choose? The factors that determine who will be Rubisco's date for the evening are the relative concentrations of carbon dioxide to oxygen and the ambient temperature. When the leaf pores or stomata of a plant are open, oxygen and water vapor are diffused out to the atmosphere. Carbon dioxide, however, enters the plant. So when the stomata are open, there is much less photorespiration as the Rubisco binds with the carbon dioxide. However, when the stomata are closed, oxygen is able to build up inside the plant's leaf, and photorespiration is then more likely to occur. The fickle Rubisco also has a higher affinity for oxygen when ambient temperatures increase, so photorespiration is more likely to occur under hotter conditions. In this lesson, we learned how the dark reactions, or the Calvin cycle, uses carbon dioxide from the atmosphere, as well as ATP and NADPH from the light reactions to make sugar. Remember that the light reactions also used water and produced oxygen as waste. 3 CO2 molecules are used in carbon fixation by the enzyme Rubisco. 1 carbon dioxide is used at a time, resulting in steps that fix, rearrange, and energize carbon molecules. Eventually, 1 3-carbon molecule called G3P, or glyceraldehyde 3-phosphate, is released. G3P is the starting material for glucose and other sugars. Therefore, the products of the light reaction created with sunlight fuel the dark reactions to create food, completing photosynthesis and feeding the world. Sometimes, however, the Calvin cycle can take a different reaction if Rubisco binds with oxygen instead of carbon dioxide. This process is called photorespiration, and it undoes all the good done by photosynthesis because it turns fixed carbon back into carbon dioxide.
The model that Watson and Crick proposed in 1953 to describe the molecular structure of DNA was a landmark discovery, but at the time, many scientists weren't convinced that the model was right. Along with their structural model of DNA, Watson and Crick also proposed a model to describe how DNA is copied inside a cell. Many scientists thought their model of DNA production didn't make sense, and it led some people to doubt whether they were even right about the double helix. Let's learn more about the science behind DNA discoveries to find out how this problem was solved. Now, scientists had known for a very long time that organisms make copies of their DNA. Making extra copies of the instructions in DNA allows an organism to grow and reproduce. The scientific word for copy is replication. So when we talk about DNA making copies of itself, we call it DNA replication. Let's quickly review the things we've already learned about DNA. A DNA chain is composed of smaller components called nucleotides. Each nucleotide is composed of a sugar, a phosphate, and a nitrogenous base. The nucleotides are arranged in 2 strands that link together like rungs on a ladder, and the ladder is twisted into a shape we call a double helix. Watson and Crick first proposed this structural model, and further scientific study has shown that they were basically correct. So why were they challenged by the scientific community? Watson and Crick had proposed that in order to copy itself, DNA would have to open down the center, sort of like a zipper coming apart, so that a new DNA strand could be built on top of the exposed strands. Following the rules of complementary base pairing, adenine would pair with thymine and cytosine would pair with guanine. This idea was called a template model since one DNA strand serves as the template for a new one. Watson and Crick figured that this model would result in 2 new double strands of DNA, each one with 1 strand of parent or template DNA and 1 strand of daughter or newly synthesized DNA. They called this the semi-conservative model because half of the parent DNA was conserved in each new DNA molecule. Now, scientists looked at the double helix of DNA and wondered how in the world it could possibly open itself up without getting tangled or torn apart. So they thought of some other ideas about how DNA replication works. One hypothesis, called the dispersive model, suggested that DNA only copied itself for short chunks at a time, producing new strands that alternated parent and daughter DNA. Another idea, called the conservative model, argued that DNA didn't split open at all, but somehow kept the parent strands intact while creating an entirely new and separate copy. Nobody knew for sure how DNA replication really worked until two scientists named Matthew Meselson and Franklin Stahl devised an ingenious experiment in 1958. They realized that they could test all three models at once by keeping track of what happens to one parental DNA strand as it generates a series of copies. Each model predicts a different distribution of parent DNA following a round of DNA replication. If Meselson and Stahl were able to keep track of parent versus new DNA, they could either support or refute the predictions of the three different models. Meselson and Stahl decided the best way to tag the parent DNA would be to change one of the atoms in the parent DNA molecule. Remember that nitrogen is found in the nitrogenous bases of each nucleotide, so they decided to use an isotope of nitrogen to distinguish between parent and newly copied DNA. The isotope of nitrogen had an extra neutron in the nucleus, which made it heavier. You can see from any periodic table that most nitrogen atoms have an atomic weight of 14. We call these atoms N14. But an isotope with an extra neutron has a weight of 15, so we call it N15. The scientists decided to start with parent DNA molecules that only contained N15. If only N14 nucleotides were available during DNA replication, they would be able to tell which parts had come from the original double strand and which parts had been created during the replication process. In order to make DNA go through many rounds of replication, Meselson and Stahl harnessed the reproductive powers of the common bacteria E. coli. They made sure that the first batch of bacteria contained only N15 DNA. Then they put the bacteria into a medium that only contained N14 atoms. That way, whenever the bacteria reproduced, they would be forced to incorporate the N14 into their new DNA. The scientists sat back and let the bacteria go to work. With each new generation of bacteria, Meselson and Stahl took a sample so that they could see how the N15 DNA was being distributed in the daughter molecules. Now, you may be wondering, how could they tell the difference between N15 and N14 DNA? It's not like you can actually see an isotope of nitrogen. How did the scientists know how much N15 was inside each molecule? The answer is the atomic weight. Because N15 has one extra neutron, it's slightly heavier than N14 and therefore makes the DNA molecule more dense. We can separate DNA molecules based on the differences in their densities. To do this, we use a centrifuge, a device that spins a test tube at very high speeds. When a test tube is spun inside a centrifuge, all the contents are pushed toward the bottom. The substances that are heaviest sink further down the tube, and the lighter substances float. So if you apply a centrifugal force to a mixture of two types of DNA, the heavier N15 DNA sinks to a lower level than the N14 molecules. Every time Meselson and Stahl wanted to take a sample of the bacterial DNA, they had to chop up the tiny organisms and empty all the contents into a test tube. They mixed in a salt solution and then spun the test tube for many hours to make the substances separate out. Then they used special techniques to see how far the DNA molecules sank inside the tube. When they sampled their first group of bacteria, Meselson and Stahl saw a darkened band in the test tube, where the N15 DNA had sunk and gathered in one spot. But after they let the bacteria reproduce, they got much different results in their sample. The DNA still sank down in the tube, but not nearly as far as the first generation. It was a lighter form of DNA, meaning that it wasn't completely made with the N15 isotope. After one replication, all the DNA had been converted to a hybrid of N15 and N14 DNA. Right away, Meselson and Stahl knew that they could rule out one of the three models. The conservative model, which suggested that the original DNA molecule remains intact, had to be false. The conservative model predicted that the centrifugation experiment would produce two distinct bands. one band representing DNA with only N15 and one band representing DNA with only N14. Because they observed only one band with DNA of intermediate density, they knew that each and every one of the new DNA molecules contained a mixture of both nitrogen forms. But Messelson and Stahl still had to figure out whether DNA replication followed the dispersive or semi-conservative model. Since both models would result in a hybrid of parent and daughter DNA, the intermediate band was still consistent with both models. In order to figure out what was really going on, Messelson and Stahl had to let the bacteria keep replicating and study the DNA samples after every generation. Before we find out what actually happened, let's think about the remaining possibilities for this experiment. How would we know whether DNA replication is dispersive or semi-conservative? What would we expect to see if either of the possible models were correct? First, we'll assume the dispersive model correctly describes DNA replication. In dispersive replication, DNA is copied in short chunks, and the result is a molecule that alternates pieces of parent DNA with daughter DNA. After one replication, the new molecule would be 50% parent and 50% daughter DNA. After another replication, the result would be 25% of the original parent and 75% newly copied DNA. So in each successive generation, the amount of parent DNA would be cut in half. In the case of our experiment, this second generation would have 25% N15 and 75% N14 DNA. In the third generation, only 12.5% of the DNA would be N15. So we would always expect to see one continuous band of DNA in the test tube. This band would move slightly higher up the tube with each successive generation as the DNA molecules became progressively lighter and lighter. On the other hand, what data would we expect to see if the semi-conservative model is correct? In this model, each new DNA molecule would contain one full strand of parental DNA, linked down the center with one full strand of daughter DNA. After one replication, all the new DNA would be the same density. But after the second round of replication, two different kinds of DNA would emerge. Some that were a hybrid of N15 and N14 like the previous round, and some that were fully composed of N14 DNA. This is because the original parent strands, while split apart from each other in the beginning, are conserved and kept as continuous strands of N15 DNA. Those parent strands may partner up with new N14 nucleotides, but they will always be connected along the length of the chain. Therefore, while the amount of N14 will grow and grow over each generation, there will always be two DNA molecules that contain one strand each of the parent DNA. In our experiment, we would expect to see two separate bands emerge inside the test tubes, one with a growing population of N14 DNA and one with the initial N15 hybrids. As it turned out, Messelson and Stahl observed a splitting of bands that became more pronounced with each new generation. They only had to observe 4 rounds of replication before they knew for sure that the semi-conservative model was correct. This was a major breakthrough in the field of biology because so many scientists had been arguing over the issue of DNA replication. Messelson and Stahl were able to disprove 2 hypotheses and strongly support the third one just by performing a simple, ingenious experiment. In summary, DNA replication is the process of making copies of DNA. DNA replicates by semi-conservative replication, which means that one strand of the parent double helix is conserved in each new DNA molecule. Messelson and Stahl were the scientists who showed that DNA follows the semi-conservative model. They were able to disprove conservative replication, whereby all parent DNA is conserved in the original molecule, after only 1 round of DNA replication. After 4 more replications, they also disproved dispersive replication, which suggests that new DNA consists of alternating parent and daughter DNA. The work of Messelson and Stahl in the 1950s helped us to understand that DNA replication occurs by a semi-conservative model. But it doesn't explain all of the intricate molecular movements that are necessary in order to achieve such a complicated feat. Remember that many scientists didn't agree with the semi-conservative model at first. In fact, they didn't agree with Watson and Crick's DNA model altogether because it was so elaborate. They argued that even if a molecule like this did exist in our bodies, there'd be no way for it to make copies of itself. But once they discovered the truth about the semi-conservative model, they had even more tough questions to answer. The biggest question they had was, how could a twisted, convoluted molecule like DNA open itself up for semi-conservative replication? Remember that the semi-conservative model states that one parental strand of DNA is conserved in each of the new daughter DNA molecules. The parental strand, which is the original DNA strand, acts as a template for the daughter strand, or a strand of newly synthesized nucleotides. In order for that to happen, DNA must actually split down the middle so that all of the nitrogenous bases are exposed. Once the bases are left out in the open, then new nucleotides can be added on. Remember that there's a rule about which nucleotides pair with which. We call it the rule of complementary base pairing. So the bases adenine and thymine will always pair together, and cytosine and guanine will always pair together. That means that as the new nucleotides are being added on to form the daughter strand, they can only add on at the places where they match a complementary base. So still the question remains, how do we actually get in there to the center of the DNA double helix, unwind all the twisted strands, and expose those nucleotides in the first place? Well, it looks like we've got some bond breaking to do. Remember that the nitrogenous bases in DNA are linked in the center by hydrogen bonds. Hydrogen bonds are generally weaker than all the other bonds in the molecule, so this is a perfect spot to start separating the nucleic acid strands. But keep in mind that DNA is a very important molecule. It holds all of our genetic information, and it can't just split open at the drop of a hat. It needs a specialized helper to assist in orchestrating that movement. That helper is an enzyme that we call DNA helicase. DNA helicase is an enzyme that unwinds the DNA double helix by breaking the hydrogen bonds between the complementary bases. It's easy to remember the name because it has part of the word helix in it. You can imagine it sort of as a wedge that forces apart the two strands, just like the slider on a zipper wedges apart the two long rows of teeth. DNA helicase is responsible for taking apart our DNA double helix. Just like a zipper makes a Y shape with the two strands of teeth, the action of DNA helicase results in a Y-shaped fork in the molecule, which we call a replication fork. The enzyme attaches to the DNA at the origin of replication, which is the place where the process of DNA replication begins. As helicase moves down the length of the strand, the replication fork moves along in its wake. You can imagine that by the time it gets to the end of the strand, all the parent DNA has been split in two. Now there's only one more problem with this model of how DNA splits apart for replication. An actual DNA strand isn't just a few hundred nucleotides long. In the human genome, there are over 3 billion nucleotides to replicate in every single cell. In order to keep up with the needs of our bodies, we have to add about 50 nucleotides every second. We could never get that much replication done quickly enough by just starting at one end of the DNA strand and unzipping straight toward the other. So in reality, DNA replication starts at many different places throughout the parental strand. There are many helicase enzymes attaching at many different origins of replication. This creates a whole line of replication forks, which end up making bubbles where the parent strands are separated in isolated locations. These bubbles grow and grow as replication continues until all of them merge together and two entirely new complete DNA strands are made. Let's review what we've learned so far. DNA helicase is the enzyme that unwinds the DNA double helix by breaking the hydrogen bonds down the center of the strand. It begins at a site called the origin of replication, and it creates a replication fork by separating the two sides of the parental DNA. This unzipping of the molecules will expose the nitrogenous bases of the nucleic acid chain, so that new nucleotides can be added to create the daughter strand. By the time helicase has finished its job, both strands of the parent DNA will be totally separated, and each will be linked to a new strand of daughter DNA.
So far, we've been talking about DNA replication by using a zipper as an example It really is one of the closest things we have to help us understand how DNA replication works. We talked about how the DNA molecule splits apart during semi-conservative replication, and how the enzyme helicase acts just like a zipper slider to separate the DNA right down the middle. Helicase breaks down the hydrogen bonds just like the wedge in the slider breaks apart the teeth in a zipper. But we didn't get to talk about how the daughter strands are put together on top of the parent template. So in this lesson, we're going to continue with the steps of DNA replication, and we'll keep thinking of it as being similar to the form of a zipper. Imagine if your best friend gave you one side of a zipper tape, and they asked you to complete the zipper by attaching the other side. But instead of giving you an entire zipper tape, what if they only gave you a bunch of little teeth that weren't connected to each other? You would have lots of work to do. You'd have to slide those little metal teeth in between the grooves on the zipper tape, one at a time, until you completed the opposite half of the zipper. That's sort of how it is with DNA replication. Once the parent DNA has been split into a replication fork, a daughter strand has to be built onto the parent strand one nucleotide at a time. The hands that work to grab those little nucleotides and arrange them into their spaces are actually a special type of enzyme. The enzyme is called DNA polymerase. DNA polymerase sounds like a really long word, but if you break it down, it actually makes a lot of sense, and it's easy to remember. The suffix ase simply tells you that this molecule is an enzyme, or a protein that helps with chemical reactions. The word root polymer refers to any large molecule that is made of many smaller parts, or monomers. And you already know that DNA stands for deoxyribonucleic acid. So if you add up all of the parts of the word, then you can see that DNA polymerase is an enzyme that helps put together the small parts of the DNA molecule. Those small parts, of course, are the nucleotides. DNA polymerase is like a vehicle that buses in the individual DNA nucleotides. It builds the daughter strand of DNA according to the template set up by the parental strand. For every cytosine, it lays down a guanine. For every guanine, it lays down a cytosine. For every thymine, it lays down an adenine, and for every adenine, it lays down a thymine. Each daughter nucleotide is attached one at a time by DNA polymerase. Now, most enzymes, especially DNA polymerase, are a bit picky when it comes to doing their job. They want all the conditions to be just right, and they'll only help with a chemical reaction when they can have everything go their way. The job of DNA polymerase is to add daughter nucleotides onto the exposed bases of a parent strand, but since it's so picky, it won't just start adding nucleotides on its own. It has to attach them onto the end of a newly synthesized daughter strand. Now, how is that supposed to work? It's sort of like if I said, I'm not going to go grocery shopping until somebody buys me some groceries first. DNA polymerase basically refuses to do its job until part of its job is already done. Wouldn't it be nice if we had another helper to solve this conundrum? Well, fortunately we do, and it's yet another enzyme. It's called RNA primase. The job of RNA primase is to make or synthesize a primer for replication to start. First, it waits for DNA helicase to open a replication fork. Then it swings in behind helicase to lay down a primer. What's a primer? Well, a primer is a short polynucleotide segment that primes or prepares the way for DNA replication by helping DNA polymerase to get started in doing its job. The primer is made out of RNA, so it's called an RNA primer. Let's go through that again because it gets a little confusing here. The RNA primer is a short strand of RNA that initiates DNA replication. So the primer that initiates DNA replication isn't even made out of DNA. Don't get confused about the difference between the RNA primer and the RNA primase. Again, the enzyme that puts together the RNA primer is called RNA primase. Just remember that the one with the ACE is the one that's the enzyme. Just like DNA polymerase buses in the DNA nucleotides, RNA primase buses in the RNA nucleotides in order to build the RNA primer. It builds up a segment of RNA according to the template, just like before. For every cytosine, it lays down a guanine. For every guanine, it lays down a cytosine. For every thymine, it lays down an adenine. And for every adenine, it lays down a... No, it doesn't lay down a thymine. Here's one spot where things are a bit different. Remember, this is RNA we're talking about. There is no thymine in RNA. Instead, it lays down uracil. This segment of RNA might only get about 10 nucleotides long before DNA polymerase feels ready to start putting on DNA. So it finally gets itself over to the RNA primer and begins to attach the actual DNA nucleotides that will become the daughter strand. The RNA primer isn't needed anymore once DNA polymerase is doing its job, so it actually gets removed, and later, DNA polymerase will come back around and make sure that spot is filled in with DNA. So far, we've talked about the roles of DNA polymerase and RNA primase in DNA replication, but there is another enzyme that also plays an important role in this. That enzyme is called topoisomerase. When the DNA helicase unzips the double-strand zipper of DNA, the amount of tension on the unwound double strand is increased. This means that there is a tendency for the strand to simply recoil back to its state of lower energy. If that should happen, replication would be impossible. So topoisomerase comes to the rescue by relaxing the usual coiled state of the DNA molecule so that replication can proceed. This way, the DNA polymerase and RNA primase can get at the molecule to replicate it. There are two major types of topoisomerase, which are topoisomerase 1 and topoisomerase 2. Topoisomerase 1 cuts one strand of the DNA molecule to extend it out straight. Topoisomerase 2 cuts both strands of the DNA. Topoisomerase 1 does not need any ATP to do its work, but topoisomerase 2 does. In fact, it requires 2 ATP molecules for every one reaction. Like helicase, which breaks the hydrogen bonds between the two strands of DNA, topoisomerase is an enzyme. However, topoisomerase breaks the phosphodiester bonds between the actual DNA nucleotides themselves. This helps to straighten things out for the replication process. As you're learning about these enzymes, it's easy to get confused about which enzyme has which job to do. Scientific terminology can all begin to sound the same. So remember, there is usually a hint about the job they do inside of the enzyme's name. For example, DNA polymerase is responsible for making a polymer of DNA. An RNA primase is responsible for making a primer out of RNA. Topoisomerase is the enzyme that makes it all possible by reconfiguring the DNA from its coiled state into a shape that is accessible to the other enzymes. Now, I know I made DNA polymerase sound like a picky, lazy bum, but it's actually a pretty efficient enzyme. Remember that it has to synthesize that daughter strand of DNA by the rules of complementary base pairing. If it makes a mistake by mispairing a base, like attaching cytosine to adenine, for example, then harmful or even deadly mutations might occur. Fortunately, incorrect pairings only happen in about 1 in a billion nucleotides, and for the mispairings that do occur, DNA polymerase goes back for a proofreading step and fixes all the mistakes. It will even check for any damage that may have been caused by radiation, like ultraviolet light and X-rays. This kind of damage can be repaired by DNA polymerase and other enzymes. So really, DNA polymerase is a valuable enzyme. Believe it or not, there are even more things to understand about DNA replication before we can complete the picture. But for now, let's summarize what we've learned in this lesson. DNA polymerase is the enzyme responsible for adding the daughter nucleotides to the parent DNA strand. In order to help it get started in its process, an RNA primer is built upon the parent strand at the base of the replication fork. The enzyme responsible for building that RNA primer is RNA primase. Once replication is begun by DNA polymerase, the RNA primer is removed, and the daughter strand is eventually completed from one end to the other.
So far, in our discussions about DNA replication, we've talked about a handful of enzymes that help us by changing and moving parts of the DNA molecules. One of the most important enzymes here is DNA polymerase. This enzyme is the one that carries the individual nucleotides to the site of replication. DNA polymerase builds the daughter strand by matching new nucleotides to their complementary bases on the parent strand. When scientists first began studying how DNA polymerase works, they assumed that it always added nucleotides in a continuous fashion. That is, they thought the enzyme always followed right behind the replication fork, laying down nucleotides as soon as the parent strands were exposed. But in the 1960s, a molecular biologist named Reiji Okazaki challenged that view. He and his colleagues had begun to think that the action of DNA polymerase was not always continuous. Their ideas stemmed from a combination of discoveries they'd made in the lab and a thorough knowledge of the structure of DNA. Okazaki understood the DNA molecule, and he knew that DNA backbones run in opposite directions. Remember that the strands in a DNA molecule are oriented anti-parallel to one another. You can think of the two strands like arrows, with the arrowhead of one strand matching up with the tail of the other strand. Scientists name the ends of the DNA strands according to the carbons in the sugar ring. One end is called the 3' end, and the other is called the 5' end. So, on any complete molecule of DNA, one strand will run from 3' to 5', and the other will run from 5' to 3'. Okazaki and his colleagues knew something was wrong. Remember, enzymes can be picky about how they do their jobs. DNA polymerase builds a strand of DNA that runs 5' to 3'. So, for the parental strand of DNA that runs 3' to 5', replication occurs just like we always thought, with DNA polymerase working in an anti-parallel direction, continuously adding the nucleotides. But it looks like bad luck for the other strand, which runs in the 5' to 3' direction. If DNA polymerase can't replicate that way, then how does the other parent strand get replicated too? Before we go on, we should give a name to each of the two strands we're talking about. The first one is called the leading strand. This is the parent strand of DNA, which runs in the 3' to 5' direction toward the fork, and it's replicated continuously by DNA polymerase, because DNA polymerase builds a strand that runs anti-parallel to it in the 5' to 3' direction. The other strand is called the lagging strand. This is the parent strand that runs in the 5' to 3' direction toward the fork, and it's replicated discontinuously. Now, let's talk about the differences between continuous and discontinuous replication. When DNA helicase opens up the replication fork, the result is two parent strands that are exposed and waiting for new pairing strands to be built. The leading strand's free end is a 3' end, and the end that's nearest to the replication fork is the 5' end. So, DNA polymerase can simply start at the free end, working on the leading strand, which is running 3' to 5' continuously toward the replication fork. As DNA polymerase does its job, DNA helicase is still moving down the line, opening the replication fork more and more. So, the two enzymes work in sync with each other. The more DNA helicase splits open the fork, the more DNA polymerase keeps adding daughter nucleotides to the new strand. This is what it means for DNA replication to work in a continuous fashion, but this only occurs on the leading strand. Now, let's look at the lagging strand. When the replication fork is open, its 3' end lies at the base of the fork, and the 5' end lies at the opposite end. With this orientation, DNA polymerase has no problem moving into the base of the fork and replicating straight toward the 5' end. The problem arises when DNA helicase moves forward, exposing even more lagging strand in front of the point that DNA polymerase began doing its job. DNA polymerase can't go backward and fill in that spot. It only works in one direction. It builds in the 5' to 3' direction, so it only works on a parent strand that runs in the 3' to 5' direction. So at this point, it's basically run out of track. This is where Reiji Okazaki comes into the picture. He hypothesized that in this situation, DNA polymerase would quit its job once it ran out of space, and then swing back to the base of the fork. That is, the new base of the fork, now that it opened a little bit more, and then it would begin again from there, until it reached the point where it had begun the first time. In this fashion, DNA polymerase would be able to replicate the lagging strand of the DNA molecule, simply by making short lengths at a time. Okazaki and his colleagues worked with the bacteria E. coli to find out whether this hypothesis was correct. Eventually, they proved their theory of discontinuous replication, and the short lengths of DNA came to be known as Okazaki fragments. So, the Okazaki fragments are the short pieces of daughter DNA that are made on the lagging strand by DNA polymerase. Okazaki's hypothesis was challenged by the fact that nobody understood how the fragments joined together. Even though DNA polymerase is perfectly capable of making multiple fragments, it can't actually join the adjacent fragments together. But it turns out there's another enzyme here to help us out. This one's job is to tie or bind the Okazaki fragments to each other. The fancy scientific word for tie is ligate. The name for this enzyme comes from the word ligate, and like all other enzymes, ends with the suffix ase. The resulting name is DNA ligase. DNA ligase is the enzyme that binds adjacent Okazaki fragments on the lagging strand, resulting in a continuous daughter strand where DNA polymerase had worked in a discontinuous fashion. The dance of the Okazaki fragments and DNA ligase makes the process of DNA replication more complicated than we ever thought. But believe it or not, the details do all come together in the end. Later, we'll take a complete walkthrough of DNA replication. But for now, let's review what we've learned about the lagging strand of DNA. When replication begins, the two parent DNA strands are separated. One of these is called the leading strand, and it runs in the 3' to 5' direction, and is replicated continuously because DNA polymerase works anti-parallel, building in the 5' to 3' direction. The other strand is the lagging strand, and it runs in the 5' to 3' direction, and is replicated discontinuously in short sections. These sections are called Okazaki fragments. They are short lengths of DNA that are made by DNA polymerase working for short distances, so it can still work building in the 5' to 3' direction. The fragments are bound together by the enzyme DNA ligase in order to complete replication in the lagging strand of DNA.
The replication of DNA is a complex process that took scientists many years and lots of hard work to understand. Previously, we discussed the fine details of semi-conservative replication, how the DNA double helix is unwound, and how parent DNA is copied to produce daughter strands. We learned about a cast of helpful enzymes that make all the molecular movements possible. But understanding how all the pieces fit together can be a challenge. So in this lesson, we'll review all the parts of DNA replication and add some new bits of information that will help to complete the picture. Let's start by reminding ourselves of the basics of semi-conservative replication. We begin with the original DNA molecule, and both strands in that molecule are referred to as parent strands. The goal of DNA replication is to make a second DNA molecule, using the parent strands as a template to create two new daughter strands. The term semi-conservative refers to the fact that both parent strands are conserved or saved in each of the new molecules. So in semi-conservative replication, the parent strands split apart, but each remain whole while new daughter strands are added onto them. The end result is two DNA molecules, each containing one parent strand and one daughter strand. Now let's go over all the steps of DNA replication. Along the way, we'll check in with each essential enzyme and discuss how it helps in completing each step. Since the names and functions of the enzymes can get confusing, we'll make an enzyme chart before we begin. As we discuss the steps of DNA replication, we'll fill in our enzyme chart to keep all our information organized. The first step occurs when DNA helicase unwinds the double helix by breaking the hydrogen bonds between the parent strands of DNA. This splitting and unwinding process opens up the DNA molecule into a Y shape, which we call the replication fork. Already we have our first enzyme, so let's fill that in. DNA helicase is the enzyme that unwinds the DNA double helix. Before new daughter strands can be added on, the parent strands are first made ready by an RNA primer. The primer is built by the enzyme RNA primase. So let's go to the chart. RNA primase is the enzyme that builds an RNA primer on the parent strand to initiate DNA replication. Once the RNA primer is built, then the next enzyme, DNA polymerase, is free to do its job. DNA polymerase slides into the replication fork and positions itself behind the RNA primer. It begins to add DNA nucleotides onto each parent strand. DNA polymerase always works by starting at the 3' end of DNA and moving toward the 5' end. This means that on the leading strand it works continuously as it follows DNA helicase, which is constantly opening the fork more and more. But on the lagging strand, DNA polymerase works discontinuously, making Okazaki fragments in the opposite direction. So now that we understand what DNA polymerase does, let's go back to our chart. DNA polymerase is the enzyme that matches and lays down nucleotides to build the daughter DNA strand along each parent DNA strand. Now we're left with all these Okazaki fragments that are separate from each other. so they need to be joined together by the enzyme DNA ligase. DNA ligase binds the fragments end to end, forming a continuous daughter strand on top of the lagging parent strand. So that was our last enzyme. DNA ligase joins the adjacent Okazaki fragments on the lagging strand of DNA. Let's back up for just a minute. Have you noticed a couple of elements that don't quite seem to fit? If you have, then you're on the right track. Until now, we haven't had a chance to put the concept of the RNA primer together with the concept of Okazaki fragments. In earlier lessons, we talked about how the RNA primer must be present before DNA polymerase can begin to do its job. Later, when we talked about Okazaki fragments, we mentioned that DNA polymerase has to keep going back and restart its work at the beginning of every fragment. So you may be wondering about this. Doesn't there have to be a new RNA primer to begin every new Okazaki fragment? Well, the answer is yes. Every time DNA polymerase restarts its work, it needs an RNA primer. Of course, in the leading strand, only one primer is needed. This primer is built upon the 3' end, the free end, before replication begins moving toward the fork. But on the lagging strand, we need an RNA primer for each Okazaki fragment. Every time DNA polymerase finishes out its track and moves back for another go, it has to have a primer waiting there to help it get started again. So on the lagging strand, there are lots of RNA primers scattered all down the line, one for every Okazaki fragment that is to be made. At this point, you can probably see why the two strands are named as they are. The leading strand is always growing just as quickly as the replication fork opens up. It's always ahead of the other strand, so it leads the other in completing DNA replication. The lagging strand is always behind because of the discontinuous replication. It takes more time for it to complete its daughter strand because it has to wait for DNA polymerase to restart its work over and over again. And then it has to wait for DNA ligase to join the fragments, so it lags behind the leading strand. Now, all this talk about the leading and lagging strands may have you wondering something else. What about the replication bubbles? Remember, DNA replication begins at multiple places along the parent molecule. DNA helicase splits it open and creates replication bubbles that grow and merge together. Replication begins at the center of the bubble and moves in both directions, outward. But strangely, whenever we've talked about the leading and lagging strands, we've looked at it like everything just starts from one end. We've only been looking at half the bubble. So if DNA replication really does happen inside all these bubbles, then how do the leading and lagging strands fit in? The truth is, we've only been looking at half the bubble to make the leading and lagging strands easier to see. So now let's zoom out and look at the whole bubble. The leading and lagging strands actually join up with similar structures in the other half. For every replication bubble, there are 2 replication forks, one at either end. So at the top of the bubble, the leading strand from one replication fork joins the lagging strand from the other replication fork. And the same is true at the bottom of the bubble. So, as you can see, we end up with an intricate layout. On the upper left and the lower right, we have leading strands being replicated continuously, needing only one RNA primer each. On the upper right and lower left, we have lagging strands being replicated discontinuously with multiple RNA primers initiating all the Okazaki fragments. So now you've learned a couple of new things that tie together all the details of DNA replication. As complicated as it may seem, the end result is the same as we've always predicted by the model of semi-conservative replication. A new daughter strand is built upon each of the separated parent strands so that every parent strand is conserved in each of the new DNA molecules. One of the parent strands is the leading strand, which is replicated continuously. The other one is the lagging strand, and it's replicated by way of short Okazaki fragments. Each fragment requires that an RNA primer is built first upon the parent DNA strand. The RNA primers are made by RNA primase, and the Okazaki fragments are joined by DNA ligase. DNA polymerase is the enzyme that carries in the daughter nucleotides, and DNA helicase is the one that unwinds the double helix to open the replication fork. While every replication fork consists of a leading and lagging strand, each fork is actually joined to another fork in the form of a replication bubble. Once all of the bubbles grow and merge together, the process of DNA replication is finally complete. DNA. It's got to be the pinnacle of biology, right? Everyone's always talking about it. How powerful it is, how incredible it is, how scientists won Nobel Prizes for studying it. It has this amazing double helical structure, and it determines the characteristics that make you who you are. DNA decides the color of your eyes, the tone of your skin, the likelihood of developing certain diseases, and even some aspects of your personality. DNA is passed down from one generation to the next, and with it, all the traits that we see in people, plants, and animals. DNA is amazing. By studying and working with DNA, we can clone sheep, improve our crops, determine the culprit in a crime, and maybe even bring back the dinosaurs. All of these things are true about DNA, but we don't tend to hear much about how all the wonderful things actually come from a tiny DNA molecule. We know that there's DNA, and we know about some of the possible outcomes, but we don't know what really happens between DNA and the final product. So in this lesson, we're going to lay out the basics of how DNA becomes something that we can see in real life. Like other processes that happen inside our cells, it's a complicated set of interrelated steps. Scientists even invented a term to describe the basic flow of genetic information from DNA to our heritable traits. It's called the central dogma. It's a simple general story that provides a framework for understanding all of the more intricate things that happen within. But before I go more into the central dogma, I'm going to tell you another kind of story. Once upon a time, I made a trip to visit my mom. While I was there, she made me one of my favorite things for breakfast, French toast. Now obviously I can get French toast anywhere, but there's something special about my mom's recipe. I really missed her French toast, so I wondered if I could learn to make it on my own. I asked Mom to give me the recipe, and she got out her giant cookbook. She opened it to the right page and handed me a pen and a recipe card so I could copy the recipe down. Once I got home, I took out the card and made my own French toast. I wasn't using Mom's giant cookbook, but it didn't matter. I still had the recipe, copied word for word, and I made my breakfast taste just like Mom's. The end. The story I just told you is actually similar to the story of how DNA becomes what we see as living creatures like animals, plants, and people. Just like my mom's cookbook, DNA is a set of instructions for how to make something. That something, in the case of my story, was the French toast, but in the case of DNA, it's a protein. Protein is the product, the biological molecule that DNA provides instructions for. Now, both of my stories have an important first part. Remember how I had to copy down my mom's recipe in order to take it back to my house? That was because my mom couldn't give me her giant cookbook. My home, the place where I actually made the French toast, was too far away for me to read her cookbook, so I needed a copy to take with me. In the same way, the place where DNA lives is really far away from the place where proteins are actually made. DNA lives inside the nucleus of a cell, and proteins are made outside the nucleus in the cytoplasm. So just like my recipe had to be copied, the instructions in DNA have to be copied into some other form that can travel from the nucleus to the site where proteins are made. That other form is RNA. Remember that RNA is another kind of nucleic acid. It's similar to DNA, but instead of being a double-stranded molecule, it's just a single strand of nucleotides. You can imagine it as one half of a ladder. RNA plays the role of my recipe card by carrying the genetic code outside of the cell's nucleus. Once in the cytoplasm, the instructions for making proteins are read by the cell's protein-building molecules. All the directions are carried out until the protein is made, just like how I carried out the directions on my recipe card. So whether we're making French toast or proteins, the point in this second part is that we have to carry out the instructions in order to build the final product. So now let's go back to the term we used earlier, the central dogma. The central dogma is a framework for understanding the flow of genetic information. It states that DNA makes RNA, and RNA makes protein. Again, the process is way more complicated than this, but when we talk about the steps that occurred during any part of the sequence, we say that it's included in the central dogma. Let's polish up some of the terms we use here. When we say that DNA makes RNA, we're really saying that the information in DNA is being transformed or transcribed into RNA. It's just like how I transcribed my mom's cookbook recipe onto my little recipe card. I physically wrote out a copy that I could then carry outside her house. Transcription is the transfer of genetic information from the form of DNA to the form of RNA. Transcription occurs inside the cell's nucleus, and then RNA leaves to go do the next step. The next step is called translation. It's the process of using the code in RNA to put together the protein itself. In my French toast story, this would be the actual making of the French toast in my kitchen. The RNA code is like my recipe card, and the protein is like my French toast. Remember that proteins are large, complex molecules, or polymers, and they're made of smaller subunits, or monomers. The small units that make up a protein are the amino acids. Amino acids are like the ingredients in my French toast recipe. Just like I have to put my ingredients together to make my dish, the amino acids are put together to make up a protein. Recall that a chain of amino acids is called a polypeptide, and the chain eventually folds into the shape of a protein. So translation is the decoding of genetic information in RNA to form a chain of amino acids, which will become a protein. It's easy to get mixed up about the difference between transcription and translation. So here's one idea you can try. Transcription contains the word root scribe, and a scribe is a person who writes copies of important documents. So transcription relates to the copying of DNA into the form of RNA. On the other hand, translation is a word that describes the transfer of information from one language to another. You can think of RNA and amino acids as being different biological languages. So translation is when we switch from the language of RNA to the language of amino acids. The process of putting together the amino acids to build a protein molecule is called protein synthesis. To remember the term, just keep in mind that synthesis means putting together, just like an electronic synthesizer puts together different types of sounds. So protein synthesis refers to the making of proteins, but we have to be careful not to confuse protein synthesis with the process of translation. When we talk about protein synthesis, we're including all of the cellular activities that are involved in making proteins. One of those activities is translation, but lots of other things happen that have nothing to do with the central dogma. Remember, the central dogma only refers to the pathway from DNA to RNA to protein. We can look at the central dogma as being separate from, but still related to, the process of protein synthesis. The translation part of the central dogma is included in protein synthesis, but the transcription part is not. Likewise, there are many other processes that contribute to protein synthesis, but they're not directly related to the story of the central dogma. Protein synthesis is the cellular process of building proteins. Translation is just the decoding of RNA to make a chain of amino acids that will eventually become the protein. It's easy to lose sight of the fact that the purpose of DNA is just to make proteins. Proteins are concrete, physical things that make up our hair, nails, muscle, and skin. But we sometimes talk about DNA as though it can influence non-physical things like human behavior or personality. While it is true that DNA plays a role in these abstract areas, the process of how that happens is way beyond anything that we need to discuss here. So just keep in mind that when we're talking about the central dogma, we're saying that DNA is simply the instructions for building proteins. You can see evidence of your own proteins by looking into a mirror. You can see the color of your eyes and the shades of your skin and hair. You can see the shapes of your chin, your cheeks, and your nose, which reflect the bone and muscle structure you have underneath. You may have been told that you got your features from your mother or your father. Some people say it's in your genes. It's true that many of the traits you have are inherited from your parents' DNA, but DNA is not the same thing as a gene. Genes are only sections of a DNA molecule. They only tell the instructions for how to make one of your proteins. An entire DNA strand is split up into genes, each gene consisting of a specific sequence of nucleotides. If the DNA code is like a giant cookbook, then genes are the individual recipes found within its pages. For every recipe, there is one dish. For every gene, there is one protein. So for every protein that contributes to your overall appearance, there's one gene that provides the code for that protein. A gene is a distinct unit of hereditary information made of a specific nucleotide sequence in DNA. So that was the basic story of how DNA becomes protein. I'm sure you don't need to hear my French toast story again, But let's review the steps involved in the story of the central dogma. The central dogma is a framework to describe the flow of genetic information from DNA to RNA to protein. The process of transferring genetic information from DNA to RNA is called transcription. Then the RNA code is used as the instructions for building a chain of amino acids, and that process is called translation. When amino acids are joined together to make a protein molecule, it's called protein synthesis. Each protein has its own set of instructions, which are encoded in sections of DNA called genes. So the overall story of the central dogma is this. Inside the nucleus of a cell, the genes in DNA are transcribed into RNA. Once RNA leaves the nucleus, it's translated in the cytoplasm, and the process of protein synthesis begins. The end result is a fully formed protein, just like the end result of my recipe is a plate full of yummy French toast.
Everyone knows that DNA contains the instructions for living things, but if you've ever been confused about how DNA actually turns into a living creature, then you're not alone. Most people, even scientists, get overwhelmed at the details involved in all the steps of the central dogma. The central dogma describes the entire flow of genetic information from DNA to RNA to the final product, a protein. To simplify it all in my head, I think of it like I'm following a recipe. DNA is like the master cookbook, and RNA is like the card that I copy a recipe onto. I imagine that I have to make a recipe card because my mom owns the cookbook and I can't take it with me. The cookbook is similar to DNA, which lives inside the cell's nucleus and can't be taken out. So DNA has to be copied into RNA, which can be taken outside the nucleus. Why do we need to take the genetic information out of the nucleus? Because we're going to use it to make a protein, which we can only do in the cytoplasm. The cytoplasm contains all the machinery or the equipment that is used to make the proteins. It's just like how my kitchen contains all the equipment for cooking my recipe. If I'm going to make my food inside my kitchen, then I need my recipe card. If we're going to make proteins in the cytoplasm, then we need the genetic recipe. That recipe comes in the form of RNA. In this lesson, we're only going to be talking about transcription. That means we won't be getting through the entire central dogma. We'll only get to the point where we end up with RNA. In order to investigate the steps involved, we'll have to take a look at some close-up images that show how all of the molecules are arranged. Sometimes in biology, when you're learning the tiny details, it's easy to lose sight of the big picture. You may begin to wonder exactly where we are in the context of a cell. But remember that transcription starts with DNA, and DNA lives in the nucleus. So throughout this entire lesson, keep in mind that all of it happens inside the nucleus of a cell. Transcription is the copying of genetic information from the form of DNA to the form of RNA. Remember that RNA is a single-stranded molecule. It doesn't have a complementary strand like DNA does. It's only half a ladder, or a single strand of nucleotides. In order to make the RNA strand, we only need one of the original DNA strands. We'll talk later about how to know which DNA strand to use, but for now, we just need to know that one will be called the sense strand and the other will be called the antisense strand. The antisense strand is the strand of DNA which serves as the template during the process of transcription. The RNA will become a complement of the antisense strand. This means that with a minor exception we'll address later, it will essentially be a copy of the sense strand. The RNA strand has to be built one nucleotide at a time. Does that sound familiar? It should, because a similar process happens in DNA replication. And just like in DNA replication, we need the help of an enzyme to bus in all the nucleotides. You may recall that DNA polymerase was the enzyme that constructed the DNA. Well, in this case, we have RNA polymerase that constructs the RNA. RNA polymerase is the enzyme that assembles the individual nucleotides to create the RNA strand based on the DNA template. The RNA that is built here can actually be one of three different kinds. You may recall that the three different RNA types are mRNA, rRNA, and tRNA. The type that we're concerned with here is mRNA. The M stands for messenger, because this type of RNA serves as a messenger to carry genetic information from the nucleus to the cytoplasm. mRNA contains the codes for making a sequence of amino acids. As you know, a chain of amino acids will eventually become a protein when we continue on with protein synthesis. Remember that for every protein, there is one gene that provides the code for making it. Genes, of course, exist inside the DNA, but are transcribed into mRNA. So messenger RNA is the type of RNA that codes for amino acid sequences. Let's take a look at a hypothetical gene that we could find within the original DNA. We'll say it's a gene that codes for a familiar protein named keratin. So here's the keratin gene, just sitting here in the middle of the DNA molecule with other genes to its left and other genes to its right. The keratin gene has a starting point and an ending point. It's just like a recipe that flows in a linear sequence. It has to be read in the correct way, or else the amino acid sequences won't come out right. So to make sure that transcription goes in the right direction, there is an extra chunk of DNA that marks where transcription should begin. This piece of DNA is called a promoter. It's sort of like a big start here sign that tells the RNA polymerase to begin transcription at that point. The promoter itself is not actually part of the keratin gene, and it doesn't code for any amino acids. It's simply a signal for transcription to begin. Another chunk of DNA sits at the end of the keratin gene. It's sort of like the finish line, and it's called the terminator. Now, don't be afraid of the terminator. It's only a stop signal. Termination means to stop or end a process. So the promoter is the nucleotide sequence in front of the gene that signals the beginning of transcription, and the terminator is the nucleotide sequence at the end of the gene that signals the end of transcription. RNA polymerase recognizes the promoter and binds to it on the DNA molecule. This is the official beginning of transcription, and we call this phase initiation. That's not hard to remember. Initiation just means the beginning of something. If I was copying a recipe from my mom's giant cookbook, then initiation would be the moment where I sit down at the table with my pen and recipe card. Here, initiation is the first phase of transcription, during which RNA polymerase attaches to the promoter and begins to build mRNA. The promoter for our keratin gene only exists on one strand of the DNA, which is how we know which strand is the antisense strand. For the next gene, the promoter might be on the same strand, or it could be on the opposite strand. It doesn't really matter. Every gene has its promoter on one or the other strand, and it varies from gene to gene. Once initiation is done, then RNA polymerase moves down the length of the gene and transcribes it into a strand of mRNA. The enzyme works by matching up the complementary nucleotide bases. Do you remember what the names of those bases are? In DNA, the bases are adenine, guanine, cytosine, and thymine. However, in RNA, the nucleotide bases are adenine, guanine, cytosine, and uracil. In general, the RNA strand is built just like in DNA replication. For every cytosine, RNA polymerase lays down a guanine RNA nucleotide. For every guanine, it lays down a cytosine. For every thymine, it lays down an adenine. But whenever it encounters an adenine on the antisense strand, it lays down a uracil instead of a thymine because it's RNA. The result is a length of mRNA built on top of a DNA strand. It's a pretty intricate dance here, because the DNA molecule has to split open to allow transcription to occur, and it has to come back together as soon as that part is transcribed. So DNA splits open, gets transcribed, and zips back together all within a very short distance. There's no room for extra molecules in there. So what happens to the mRNA that's still growing longer? Well, it just peels away from the DNA template and hangs off to the side. You can think of it like a person knitting a scarf. As the knitter creates more and more length of fabric, the front of the scarf trails off to the side of the person's lap. Anyway, this whole phase of transcription is called elongation, because the mRNA strand elongates as transcription continues down the length of the gene. So elongation is the middle phase of transcription, during which mRNA grows longer with each additional nucleotide. Now we're at the end of the gene, and it's time to finish up. Can you guess the name of the last phase here? I'll give you a hint. It has something to do with the RNA polymerase finally reaching the terminator at the end of the gene. That's right, it's called termination. RNA polymerase sees the signal that it's time to stop transcription. It stops adding RNA nucleotides and comes off from the DNA gene. It also drops the mRNA that it was building because it's finally done. Then it floats off to do more transcription somewhere else. So termination is the final phase of transcription during which the RNA polymerase reaches the terminator and detaches from the gene and the mRNA. All three phases of transcription occur inside a cell's nucleus. Remember, everything we've discussed in this lesson is related to transcription, which is only the first part of the central dogma. The purpose of transcription is to provide a copy of genetic information that can travel outside the nucleus in preparation for translation. We start with the genes in the original DNA, and we end with the genes in the mRNA. Just to be sure that we've got all the steps here, let's walk through transcription from the top. Transcription begins when RNA polymerase attaches to a promoter within DNA. This is called initiation. The DNA molecule splits open and allows RNA polymerase to add RNA nucleotides onto the DNA template. The DNA strand that serves as the template is called the antisense strand. Elongation is the phase in which the RNA molecule grows longer as transcription continues down the length of the gene. Termination occurs when RNA polymerase reaches the terminator and detaches from both strands. The RNA that's produced at the end of transcription can be one of three different types. The type that we're concerned with here, which encodes the sequence for amino acids, is messenger or mRNA. Once the mRNA is complete, then it can leave the nucleus and continue on to the cytoplasm, where it helps with protein synthesis.
Have you ever wished you had an identical twin? Sometimes I'm jealous of people who have twins. I think it would be interesting to see how another me would turn out. I mean, identical twins don't turn out completely identical. Usually, they end up at slightly different heights, with slightly different facial features and different personalities. Some identical twins are easy to tell apart, even though their genes are exactly the same. But that seems odd, doesn't it? If identical twins have the same exact genes, then why are there noticeable differences at all? Are genes really the only things that define our physical features? Or do our genes get tweaked a little by the rest of our cellular functions? Let's go back to what a gene really is. It's a section of DNA that codes for or specifies a particular protein. The gene is first transcribed into mRNA, then translated into a polypeptide chain. That polypeptide is a component of the proteins that make up your body, including your height, facial features, and everything else. When the codes hidden inside our genes come out to light as physical traits, we call it gene expression. Gene expression is just what it sounds like. It's the act of genes expressing themselves. How do people express themselves? They show off their emotions, right? They put out actions and words that result from the thoughts and feelings they have inside. When genes express themselves, they put out protein molecules that result from the genetic codes they have inside. Genes express themselves by turning the DNA code into a protein by way of transcription and translation. It's basically another way of talking about the central dogma. Gene expression describes how the genetic makeup of an organism is shown as an organism's physical traits. It is the process by which information flows from genes to proteins. During the life of a living thing, it's important to have control over how much of a gene is expressed at any given time. For example, take the gene for keratin. Keratin is the protein that makes up our skin, hair, and nails. We generally need to grow these things at a continuous pace because our skin, hair, and nails get worn down over time. But if we make too much keratin, we could end up with way too much hair, really long nails, or really thick, tough skin. Life would be a pain if this happened to us. So it's important that we regulate the expression of the keratin gene. Regulation of gene expression describes a variety of mechanisms by which our cells control the amount of protein that's produced by our genes. Gene regulation happens differently depending on whether the organism is a prokaryote or a eukaryote. Do you remember the difference between these two? Let's quickly remember. Eukaryotes are organisms, like plants, animals, fungi, and protists, that all have cells with nuclei and membrane-bound organelles inside. Most eukaryotes are multicellular. A prokaryote is a single-celled organism, like bacteria, that doesn't have a nucleus or membrane-bound organelles inside. Since a eukaryotic cell has a nucleus and a prokaryotic cell doesn't, the regulation of transcription is different between the two. In a eukaryote, the mRNA that's transcribed in the nucleus must pass through the nuclear envelope to be translated in the cytoplasm. Before it can leave, it has to be processed. Some parts are added to the strand, and some are taken out. There's more to it than that, but we'll save eukaryotic RNA processing for another time. In a prokaryote, there's no nuclear envelope, so the mRNA can begin translation right there in the cytoplasm. Transcription and translation overlap with each other, so the production of proteins actually begins before the mRNA strand is complete. Unlike eukaryotes, prokaryotes have more than one gene on an mRNA strand. So, with the overlap of processes, all the genes on the mRNA end up getting translated together. Usually, an organism doesn't want to translate different proteins at the same time, because different proteins are involved in different cellular activities. So, in a prokaryote, genes that are related to each other are found side by side on the actual DNA. Clusters of related genes are called operons. When an entire operon is translated, a whole team of proteins is produced. Every protein on the team contributes to the same cellular function. The study of operons was the first way that we learned about the regulation of gene expression. In 1961, two French biologists studied the bacteria E. coli to learn how operons worked. Remember that E. coli is an important bacteria that lives in your intestine. It helps you digest certain foods you eat, like the lactose sugar found in milk and dairy products. E. coli has 3 genes that code for the lactose-digesting enzymes. Without the enzymes, you wouldn't be able to digest the sugar lactose. And without the 3 genes, you wouldn't be able to make the enzymes. The set of 3 genes is an example of an operon. Scientists call this one the lac operon because it controls the production of lactose-digesting enzymes. Now, we're going to look inside your intestine for a minute. Let's say that every morning when you wake up, you always drink a full glass of milk, but for the rest of the day, you don't have any more dairy products. Do you think the lac operon makes the lactose-digesting enzymes at a constant rate all day long? Of course not. The lac operon makes lots of enzymes in the morning when you first drink all that milk, but for the rest of the day, it doesn't need to make more enzymes because you're not having any more dairy. The lac operon regulates the expression of its genes depending on how much the enzymes are needed in different situations. The mechanics of gene regulation in the lac operon are pretty complex, but the short story is that the operon is turned on and off based on the amount of lactose in the bacteria's environment. And by environment, I just mean the inside of your intestine. That's all the environment an E. coli bacteria has. When you drink your milk, the lactose ends up in your intestine and surrounds the E. coli bacteria. The presence of lactose turns on the lac operon, just like a light switch. This is called induction. Transcriptional induction is an increase in gene expression due to the presence of an inducer. An inducer is a molecule that begins gene expression. In the case of E. coli and the lactose-digesting enzymes, the inducer is the sugar lactose. If an inducer turns an operon on, then what turns an operon off? Well, there's another molecule called a repressor. It's a protein that regulates gene expression by blocking gene transcription. Once you've finished digesting your morning dose of milk, the lac operon turns off so that the bacteria don't waste energy making enzymes you don't need. This is called repression. Transcriptional repression is the blocking of gene expression in response to a repressor. As you can see, our genes are not completely in charge of defining our physical characteristics. Instead, they're more like basic instructions for different possibilities of protein products. Other cellular functions get to help decide how our genes are expressed by controlling how much and how often our proteins are produced. It makes sense now that two identical twins can turn out looking so different because they don't always experience the same environments during their lifetimes. Their genes are expressed in different ways due to the processes of gene regulation. A segment of DNA that codes for a specific protein is called a gene. Genes are expressed when they are transcribed into mRNA and translated into protein. Gene expression is carefully regulated by all organisms so that the correct amount of each protein is made. Eukaryotic organisms regulate their gene expression differently than prokaryotes. While eukaryotic RNA is processed in the nucleus, prokaryotic RNA is arranged in clusters of related genes called operons. By studying the lac operon found in E. coli bacteria, biologists learned about gene regulation and the processes of repression and induction. Repression is a decrease in gene expression. Induction is an increase in gene expression due to the presence of an inducer. While our genes provide all the instructions for the proteins we make, our individual traits are influenced by the regulation of gene expression.
Gene expression describes how the genes in our DNA are turned into our physical traits, like proteins and enzymes. All living things can control the expression of their genes to some extent. Scientists first understood gene regulation when they studied E. coli, a type of bacteria that lives in our large intestine. This bacteria makes enzymes that help us digest our food, like the lactose in milk. E. coli, remember, is a prokaryote, an organism whose cells lack a nucleus. Prokaryotic DNA is clustered into groups of genes called operons. When scientists first studied the lac operon found in E. coli, they observed gene regulation through transcriptional repression and induction. We've talked about repression and induction before. Repression describes a decrease in gene transcription, whereas induction is an increase in gene transcription. The lac operon of E. coli is induced in the presence of lactose, and it's repressed when lactose is gone. Sometimes we describe induction and repression as turning on and turning off the operon. We can think of the operon as being controlled by a switch. Lactose turns it on, and the absence of lactose turns it off. But what is the true mechanism for transcriptional control? We know there's not really a switchboard down there microscopically managing everything that happens in the bacterial DNA. So what's the real story? How does repression and induction actually work? And what about lactose? It's only a sugar. Can it really have that much power over the expression of bacterial genes? This lesson will walk you through the main points of an operon, describing how each of the segments work. We'll take a closer look at the lac operon and how it responds to repressors and inducers. Then we'll revisit repression and induction for a thorough understanding of how an operon controls transcription in the prokaryotic cell. Alright, so here's the lac operon from E. coli. Like all operons, it's a cluster of related genes that code for similar things. We call the individual genes structural genes. A structural gene is any gene that codes for a structural protein or an enzyme. Remember, enzymes are just one kind of protein. The structural genes in the lac operon code for 3 different enzymes. The first enzyme is called beta-galactosidase. The second one is galactose permease, and the third is called thiogalactoside transacetylase. Whew! Let's just call them Betty, Gail, and Theo. All 3 of them are in charge of breaking down lactose in the intestine. Because Betty, Gail, and Theo are usually needed at the same time, then their genes are all transcribed at the same time. The Betty gene, the Gail gene, and the Theo gene are all side by side in the lac operon. Now let's look at what we have just in front of the structural genes. There's a short DNA segment here, and it's called the operator. This is the part of the operon that acts as a switch for transcription. It's just like an operator that works a switchboard for a telephone company. The operator controls whether or not transcription will occur. It does this by providing a binding site for the repressor, which blocks RNA polymerase from attaching to the promoter. Wait a second. RNA polymerase? Promoter? Haven't we seen those terms before? Recall that genetic transcription begins when RNA polymerase locates the promoter RNA polymerase starts building the mRNA strand right after the promoter. In a prokaryotic cell, transcription continues through the entire length of the operon. If we want all the structural genes to be transcribed and translated, then we need RNA polymerase to first attach to the promoter. The promoter lives just in front of the operator, and the operator sits just in front of the structural genes. All of these together make up the operon. Is there anything else? Oh yes, sitting just upstream from the operon is a regulatory gene that codes for the repressor. Remember, a repressor is a protein that regulates gene expression by blocking gene transcription. The repressor is a type of regulatory protein, so it makes sense that it's made by a regulatory gene. I think that all these strange new terms will begin to make sense when we see some examples. Let's go back to repression and induction. Earlier, when we talked about the lac operon, we said that repression is the blocking of gene expression in response to a repressor. But how does a repressor actually block transcription? To understand repressors, you have to know what they look like. Here's a repressor for the lac operon. It's a funny-shaped protein with a hole in its side and a lumpy arm sticking out in front. It also has little feet designed for sticking to DNA. When the lac repressor is active, it sticks its feet to the operator, and its lumpy arm ends up hanging over the promoter next door. Now the promoter's blocked. So what does that mean? It means that poor old RNA polymerase can't come in and attach to the promoter. If it can't attach to the promoter, then it can't begin building the mRNA. No transcription means no translation. So Betty, Gail, and Theo will never be produced. We won't have any enzymes to help break down our lactose. The lac repressor has succeeded in blocking transcription of our lac operon. Now that we know more about repressors, let's go ahead and expand our definition. We can say that a repressor is a protein that turns off transcription by binding to the operator and blocking the attachment of RNA polymerase to the promoter. Does it really matter to us that we can't produce our enzymes now? I mean, how much do we care about Betty, Gail, and Theo? They're just enzymes that break down the sugar lactose. Do we really need them hanging around? Well, if we plan on having dairy anytime soon, then yes. Remember, we owe it to the lac operon of the E. coli in our intestine to help us digest the sugar lactose. So if any lactose gets into our system, then we'd better have a way to get that repressor off the operon. Fortunately, we do have a way, and our way is the lactose sugar itself. When lactose molecules come down the pipe, they end up binding directly to the lac repressor. Remember that hole in the repressor's side? The lactose sugar fits perfectly in there. When lactose binds to the lac repressor, it ends up changing the shape of the protein. The little feet come unstuck from the operator, and now the repressor is in its inactive state. It can't do anything as long as the lactose is bound to it. So it floats off and frees up the operon to start transcription again. RNA polymerase swoops in to bind to the promoter, transcription continues all down the operon, and before you know it, Betty, Gail, and Theo are back in business, which is great because we have plenty of more lactose molecules that need to be broken down. This reversal of repression is what we call induction. We've mentioned before that induction is an increase in gene expression due to the presence of an inducer. We now know that lactose itself is the inducer. An inducer binds to a repressor and makes it fall off the operator. But if you think about it, lactose didn't really induce transcription. It only stopped transcription from being blocked. Lactose made transcription possible by making its blockage impossible. It induced transcription by repressing its repression. I know, it seems odd, but it makes a lot of sense if you consider how the body works. Whenever we consume something, we need enzymes to break it down. Whenever we're not consuming that thing, then we don't need the enzymes. Lactose ensures its own digestion by inducing the transcription of lac operon's enzymes. When lactose isn't there anymore, then the repressor becomes active again. The active repressor stops transcription, so we don't waste energy making enzymes we don't need. It turns out that controlling transcription really isn't as simple as flipping a switch. Transcription in the prokaryotic cell is regulated by the complex interactions of DNA, enzymes, and regulatory proteins. But when you step back from the mechanics of it all, you can still think of repression and induction like turning an operon off and on. The operon is effectively the center of transcriptional control. In addition to its main structural genes, the operon houses an operator and a promoter. In front of the promoter lies a regulatory gene that produces repressor proteins. When a repressor is in its active state, it binds to the operator. This blocks the promoter lying just upstream and keeps RNA polymerase from attaching to it. The full effect is a blockage of transcription, or transcriptional repression. But once an inducer shows up, it makes the repressor fall off the operator, allowing RNA polymerase to bind to the promoter. Transcription begins here and continues down the operon until all of the structural genes are transcribed. In the case of the lac operon, the structural genes code for 3 different enzymes that are responsible for lactose breakdown. Once the 3 genes are transcribed and translated, the enzymes can go to work. Lactose, therefore, is the inducer for the lac operon. All living things have methods for regulating their genes by controlling when and how their genes are expressed. When scientists first learned about gene regulation, they studied the bacterium E. coli and one of its gene clusters, the lac operon. Scientists used the lac operon to study gene regulation. Their research revealed plenty of information about the regulation of operons, but it didn't tell them much about the regulation of human genes. After all, we humans don't have operons. Our genes are scattered throughout the genome over multiple chromosomes. Also, our cells contain a nucleus, meaning that we have to perform transcription and translation separately. Transcription has to happen in the nucleus, but translation must occur in the cytoplasm. So, unlike prokaryotes, eukaryotes undergo an RNA processing step right after transcription and before translation. RNA processing is a challenge that's unique to eukaryotes because only eukaryotic cells have a nucleus. In this lesson, we'll walk through the most important aspects of RNA processing. Eukaryotic genes are split up into parts that are expressed as RNA products and parts that are not. The parts that are expressed are called exons. They appear in short sequences scattered throughout the gene. Exons are the parts of a eukaryotic gene that are expressed through transcription and translation. You can think of them like the meat of the gene. They're typically considered the actual coding regions that specify our proteins. In between all the exons lie the other segments called introns. Introns are the intervening sequences that are removed from a gene before the RNA product is made. Introns and exons alternate with each other along the length of a gene. Introns are usually considered non-coding regions because they don't seem to code for any enzymes or structural proteins. However, scientists are still learning about the importance of introns. One thing we do know is that introns are removed from an RNA sequence right after transcription. They exist in DNA, they're transcribed into RNA, but then they're taken out before the RNA has a chance to make any products. So exons hold the important coding regions of our genes, and introns aren't much more than extra fluff. How do we get hold of only the good stuff? How do we separate the wheat from the chaff? The RNA strand is processed so that its introns are removed and the exons are pushed together to make a continuous, shorter strand. This process is called RNA splicing. It produces a final draft of the mRNA before translation gets underway. RNA splicing is the removal of introns and joining of exons in eukaryotic mRNA. It also occurs in tRNA and rRNA. Splicing is accomplished with the help of spliceosomes, which remove introns from the genes in RNA. Spliceosomes are composed of a mixture of protein and small RNA molecules. They locate the ends of the introns, cut them away from the exons, and join the ends of adjacent exons together. Once the entire gene is devoid of its introns, the process of RNA splicing is complete. What is the point of RNA splicing? Why do we have introns in the first place if we're only going to remove them later? When scientists first discovered introns, they didn't think there was any reason for having them in our genes. They originally called the introns junk DNA, but further research revealed that introns play an important role in gene expression. They separate the coding regions of the gene and allow different combinations of exons to be spliced together. In other words, a single gene is capable of coding for more than one protein. This principle is called alternative splicing. Take a look at this sample eukaryotic gene. It has 4 different segments that can be spliced in alternative combinations. One combination counts the green segment as an intron and removes it from the mRNA. The resulting protein is translated only from the red, yellow, and blue gene segments. A different combination counts the yellow segment as a removable intron and the green segment as a translatable exon. Each combination yields a different protein, both of which were translated from the same gene. Scientists are just beginning to understand the possibilities presented by alternative splicing. Therefore, we are still learning about the role of introns in our genes. Once all the introns are spliced from the gene, then it's time for the mRNA to move out of the nucleus. This will allow it to begin translation in the cytoplasm. To leave the nucleus, the mRNA must pass through the double lipid bilayer of the nuclear envelope. You may recall that a lipid bilayer is like a dense forest riddled with protein complexes. The mRNA strand needs protection to pass through this double membrane. It also needs some form of identification so that ribosomes will recognize it on the other side. The mRNA protects and identifies itself by adding extra nucleotides to both ends of the strand. Recall that an RNA strand has a 5' end and a 3' end. During RNA processing, a specialized guanine nucleotide is added to the 5' end. This is called the 5' cap. To the 3' end is added a long chain of adenine nucleotides, which is called the poly A tail. The 5' cap and the poly A tail are extra modifications to the mRNA strand. Even though they're made of nucleotides, they don't actually code for proteins. They simply act like a uniform worn by a referee in a football game, by protecting the mRNA from attack by cellular enzymes and identifying it as an important element in the game of gene expression. Capping and tailing occurs inside the nucleus right after transcription and RNA splicing. Once the mRNA moves out into the cytoplasm, ribosomes use the cap and tail to recognize it, bind to it, and begin translation. RNA capping and tailing are special adaptations found only in eukaryotes. This is because only eukaryotic cells require mRNA to travel through the nuclear membrane. To see how all these processes fit together, let's take a sample gene from the top. Here's our old friend, the keratin gene, just sitting here in the middle of a DNA strand. Right now it's in the nucleus and transcription is about to occur. Transcription of the keratin gene produces a complementary mRNA, complete with all the introns and exons found in the original gene. This form of the mRNA is called precursor mRNA or pre-mRNA. This is the form of mRNA before any RNA processing occurs. Pre-mRNA contains more nucleotides than are needed to code for a protein. So with the help of spliceosomes, the introns are removed and the exons are joined together. Next, a 5' cap and a poly A tail are added to the ends. Now we call the mRNA mature mRNA. It's the final, shorter version of mRNA that's ready for translation in the cytoplasm. Mature mRNA travels through the nuclear envelope, binds with ribosomes, and begins to translate its code into proteins. After the mRNA leaves the nucleus, additional regulatory mechanisms take place. These mechanisms may occur during translation, after translation, and even following protein synthesis. As you can see, RNA processing is just one way that gene expression is regulated in a eukaryotic cell. But the only mechanisms we need to understand here are the splicing, capping, and tailing that comprise RNA processing. The regulation of gene expression differs between prokaryotes and eukaryotes. Eukaryotic cells possess a nucleus bound by a membrane, and their genes consist of alternating introns and exons. Therefore, the immature pre-mRNA must be processed before it leaves the nucleus. RNA splicing is the removal of non-coding introns and the joining of exons by spliceosomes. The presence of introns is thought to assist gene regulation by way of complex methods like alternative splicing. After splicing, a 5' cap and a poly A tail are added, and this final draft is called mature mRNA. Splicing, capping, and tailing comprise RNA processing in the nucleus. Once the mature mRNA moves into the cytoplasm, it begins translation and undergoes other forms of gene regulation. The central dogma describes the flow of genetic information from DNA to RNA to protein. The first step that converts DNA to RNA is transcription, and the second step that converts RNA to protein is translation. The mRNA strands that are formed in transcription contain the genetic code for making the sequence of amino acids that will eventually become a protein. But what exactly is the genetic code. We already know that DNA is split up into sections called genes. Genes are like recipes for making proteins. Every gene provides the instructions for making a different type of protein. If a protein is the final product and genes are the recipes, then amino acids are like the ingredients. You can't begin making a protein molecule until you have all the amino acids that are required in that chain. A chain of amino acids is also called a polypeptide. To make the polypeptide correctly, we must put all the amino acids in the correct order. Again, it's like following a recipe. If you don't follow the instructions in the correct sequence, then the product won't come out right. The genetic code that gives us the correct order of ingredients is found in the mRNA that results from transcription. mRNA is a single strand of nucleotide bases, an ordered combination of adenine, guanine, cytosine, and uracil. To make things easier in this lesson, we'll call the bases by their letters, A, G, C, and U. How could we use these letters to code for the amino acids, the ingredients in our protein recipe? Well, there are 4 nucleotide bases. Could it be that each base codes for a different amino acid? Let's take a look at how many amino acids we'll need. Wow, that's a lot of amino acids. In fact, there are 20 different amino acids that we might need to access in order to make a protein. Think of this chart like the pantry in your kitchen. Just like you need a full pantry of ingredients to start a recipe, we'll need access to every amino acid before we know we can begin a polypeptide chain. We'll need 20 amino acids, but we only have 4 different nucleotide bases. I guess we can't use the individual letters to code for all the amino acids. What if we used combinations of letters? After all, our own English language has only 26 letters, and look at all the different words and sentences we can make. The system of Morse code has even less to work with. It only has 2 choices, short and long units. Morse code uses combinations of short and long tones or lights to code for all the letters of the alphabet. So with the 4 bases we have on the mRNA strand, why not use combinations to code for all 20 amino acids? Well, it turns out that's exactly what happens in translation. When the mRNA code is read by the proteins that build the amino acid chain, it's not read as a sequence of individual letters. It's read in chunks of 3 letters or 3 nitrogenous bases at a time. Each set of 3 bases is a special code that indicates a different amino acid. The 3 base sets are called codons. A codon is a series of 3 bases in mRNA that specifies a particular amino acid. Codons can also indicate a stop or termination signal, but we'll get to that in a minute. For now, just think of codons as the basic unit for the genetic code. If we use codons instead of the individual bases as our units for the genetic code, then how many choices do we actually have? Well, there are 4 bases in sets of 3, so we can get 64 different codons. That's plenty of different ways to code for our 20 amino acids. In fact, it's more than enough. Let's take a look at an RNA codon chart. It's sort of like a dictionary for the genetic code. As you can see, every codon is assigned a different amino acid. But because there are less amino acids than codons, then most amino acids are indicated by more than 1 codon. The amino acid serine is indicated by the 2 codons AGU and AGC. The amino acid alanine gets 4 different codons, and the amino acid leucine gets 6 codons. So there are 6 different ways that the mRNA code can indicate that leucine be incorporated into a polypeptide chain. Let's see what else we can find on this codon chart. You may have noticed that there are 3 codons which don't specify an amino acid. These codons are UAA, UAG, and UGA. Instead of coding for amino acids, these codons are actually termination signals that are found at the end of a gene. We call them stop codons because they signal an end to the process of gene translation. Whenever a stop codon is read by the proteins building the amino acid chain, then they know to stop adding amino acids and quit making the polypeptide. If there are 3 different stop codons, then what about start codons? Well, there is only one start codon. It's the sequence AUG. AUG is the start codon for every gene, and it signals the beginning of genetic translation. It also codes for the amino acid methionine, So that means that during the making of a polypeptide, methionine will always be the first amino acid in the chain. So whenever you see the codon AUG in a strand of mRNA nucleotide bases, you should view it as the start of an amino acid chain. You may find that it's important to memorize the start codon AUG. While most science students are allowed to use a chart for the other codons, they're typically expected to know the start codon without one. So as a trick, you can remember that AUG is also the abbreviation for August, the month when the school year often begins. To remember that it codes for methionine, just think about your assigned summer reading and that all your requirements have to be met before August. Other than that, you'll usually get a chart to help you translate the other codons. Translation is the process of turning the genetic code in mRNA into an amino acid chain in preparation to make proteins. It begins with mRNA, which is a coded sequence of nucleotide bases that we call by the four letters A, G, C, and U. mRNA is read by groups of 3 nucleotide bases called codons. There are 64 different codons, and each codes for a specific amino acid or a stop signal. The stop codons signal translation to stop. The start codon AUG codes for methionine and signals translation to begin. While the RNA codon chart can be used to identify the amino acids for each codon, it's often helpful to memorize the start codon AUG. Translation is the process of converting the genetic information in the mRNA strand to the form of a protein. The basic unit of this genetic information is a codon. Earlier, we described a codon as a sequence of 3 nucleotide bases that specifies a particular amino acid. Codon recognition describes the process of matching codons to the correct amino acids. Codons are read down the length of the mRNA strand and translated into an appropriate polypeptide chain. But when we talk about reading the codons, what does that actually mean? Who is reading the sequence of mRNA codons? How are the codons being interpreted as instructions for amino acids? And who is responsible for bringing those amino acids together in the correct order specified by the genetic code? In this lesson, we're going to talk about how codons are recognized with the help of a new type of RNA. We'll study the molecular mechanics involved, and we'll practice using these molecules to make our own polypeptides. Let's begin by reminding ourselves what the word translation describes. Translation of mRNA to protein is similar to translation of one language to another. We're converting the language of mRNA, a sequence of nitrogenous bases, to the language of a protein, a sequence of amino acids. Translation between languages always requires the work of an interpreter, an agent responsible for recognizing both languages and drawing the connections between the two. For example, let's say that I took a summer vacation in France and that I didn't know how to speak any French. But if I brought along my cousin who knows both French and English, then I would have an interpreter to help me communicate. If I wanted to buy some cheese from a grocer, I could just tell my cousin, I want to buy some cheese. My cousin would use her translational powers to convert my English sentence into a French one and say, Je veux acheter du fromage. In the case of genetic translation, we have a molecule that acts as an interpreter between codons and amino acids. It's a special type of RNA called transfer RNA, or tRNA for short. Transfer RNA is the type of RNA that interprets the mRNA code during translation. When a cell is ready to have its genetic code translated into polypeptides, it first has to be prepared with all the essential amino acids. Remember, there are 20 different amino acids involved in making our proteins. Proteins can only be made correctly when the amino acids are assembled in the right order. The job of tRNA is to match up the amino acids with the correct codons in the mRNA strand. We can look at our codon chart to get an idea of what tRNA's job is like. Let's see here. If we were tRNA and we read the codon UUU, then we would know to grab a phenylalanine. If we read the codon AGC, then we would know to grab a serine. Transfer RNA reads a gene's codons from start to finish and matches the amino acids in the correct order. That sounds simple enough, but how does tRNA go about matching the codons to the correct amino acids? How does it know when it's found a codon and an amino acid that are supposed to go together? Like all RNAs, tRNA is a single strand of nucleic nucleotide bases. But a tRNA is short, only about 80 nucleotides long, and it folds up on itself so that some parts are actually double-stranded. It twists into a funny shape that sort of looks like a four-leaf clover, or a T shape. That's convenient because it helps to remember that tRNA is shaped like a T. At one end of the T is the attachment site for a certain amino acid. At the other end is a set of 3 nucleotides that match the codon that specifies the same amino acid. Note that these nucleotides on the tRNA do not make up a codon. They are, in fact, the exact opposite, an anticodon. The anticodon is a perfect complementary match to a codon. So if the codon for serine is AGC, then the anticodon for serine is UCG. The tRNA that contains the anticodon UCG will also be the tRNA that attaches to a serine amino acid. Okay, maybe I should back up for just a second. How did I get the anticodon for the codon AGC? Well, it's really simple. It's just like how we matched the complementary bases when we made copies of DNA in DNA replication. We also match complementary bases when we transcribe DNA to mRNA. In translation, we're using 2 different kinds of RNA, mRNA and tRNA. So we're only using the letters A, G, C, and U. G and C always pair together, and A and U always pair together. Just remember that G and C are both consonants, and A and U are both vowels. So let's look at the codons AGC on the mRNA strand. The complement for A is going to be U on the anticodon. The complement for G is going to be C, and the complement for C is going to be G. So if we put it all together, we get UCG as the anticodon for the codon AGC. We already knew about codon recognition before we got into all the stuff about tRNA and anticodons, but now we've begun to see the mechanics that are involved at the molecular level. So let's go ahead and expand our definition of codon recognition. It's the ability of tRNA to match a codon to the appropriate anticodon and therefore the correct amino acid. tRNA recognizes the codon by using its own complementary anticodon. It's just like a lock and key. You know you have the right key when it fits into the lock. The attachment site for the amino acid is also sitting on the tRNA. These bind together in a similar way. Since both the amino acid and the mRNA codon bind to the tRNA, then they can be brought together as an appropriate match. Just to be sure we've got everything clear, let's try an example of genetic translation. We've done it before, but this time we'll bring tRNA and the anticodons into the picture. Then we'll really get an understanding of how codon recognition works. We'll begin with the following mRNA strand: A, U, G, U, A, U, C, A, C, U, A, A. We'll draw a line in between each set of 3 bases. That's what scientists do. So we get a total of 4 codons: AUG, UAU, CAC, and UAA. The AUG codon we know is the start codon, and it codes for methionine. So first we need a tRNA that matches with methionine on one end and contains the correct anticodon at the other end. The anticodon for AUG is UAC. Here's a tRNA with the anticodon UAC, and it's bringing in a methionine attached to its other end. Codon recognition happens when tRNA pairs with the mRNA inside the ribosome. And now we've gotten started with methionine. The next codon is UAU, for which the amino acid is tyrosine. The anticodon that pairs with UAU is AUA. So here's a tRNA with the anticodon AUA carrying in the amino acid tyrosine. It pairs with mRNA at the ribosome, and now tyrosine is added next to methionine. The next codon is CAC, for which the amino acid is histidine. The anticodon for CAC is GUG. Here comes a tRNA with the anticodon GUG attached to the amino acid histidine. The tRNA pairs with mRNA at the ribosome, and now histidine is added to the end. Our last codon is UAA, which is one of the 3 stop codons. So we don't need any more tRNAs or amino acids. We're done. We stop making the polypeptide and we finish out with a chain of 3 amino acids. methionine, tyrosine, and histidine. Translation is the second step in the central dogma, which involves converting the nucleotide base code in mRNA into a chain of amino acids. Translation occurs in the cytoplasm of a cell, where the amino acids and tRNA molecules are also found. During translation, tRNA molecules first match up with the amino acids that fit their attachment sites. Then the tRNAs carry their amino acids toward the mRNA strand. They pair onto the mRNA by way of an anticodon on the opposite side of the molecule. Each anticodon on tRNA matches up with a codon on the mRNA. In this way, amino acids are assembled in the correct order dictated by the mRNA code. The ability of tRNA to match codons with appropriate amino acids is codon recognition. Good codon recognition is essential to the correct assembly of polypeptides, which in turn leads to the production of the right proteins.
Translation is the second step in the central dogma that describes how the genetic code is converted into amino acids. We've talked about how the mRNA codes are recognized by tRNA and how the amino acids are linked together by peptide bonds. A chain of amino acids is also called a polypeptide. Polypeptides are assembled inside the ribosomes, which are the tiny organelles on the rough ER of a cell. Now that we're learning more about the mechanics of translation, we're going to have to start putting the pieces together. We already understand the role of the ribosome and the amino acids in the process of translation, but how does polypeptide assembly actually occur? There are 3 important steps to the process of translation. There's a beginning step called initiation, a middle step called elongation, and a final step called termination. These 3 words may sound familiar to you. The same terms are used in transcription to describe the steps involved in making the mRNA strand. But here in translation, we're making a polypeptide strand. In either case, we're making a long molecule out of a chain of smaller subunits. So whether we're referring to transcription or translation, the 3 terms accurately describe the mechanics of the process. Let's walk through each step one at a time. We'll start with initiation. During initiation, the mRNA, the tRNA, and the first amino acid all come together within the ribosome. The mRNA strand remains continuous, but the true initiation point is the start codon, AUG. Remember that the start codon is the set of 3 nucleotides that begins the coded sequence of a gene. Remember also that the start codon specifies the amino acid methionine. So methionine is the name of the amino acid that is brought into the ribosome first. And how did methionine get itself to the ribosome? By attaching to the tRNA that contains the right anticodon. The anticodon for AUG is UAC. We know that because of the rules of complementary base pairing. The tRNA with the anticodon UAC will automatically match to the codon AUG, bringing the methionine along for the ride. So there you have it. mRNA is attached to tRNA, and tRNA is attached to methionine. That's initiation. The next step makes up the bulk of translation. It's called elongation, and it's the addition of amino acids by the formation of peptide bonds. Elongation is just what it sounds like. The chain of amino acids grows longer and longer as more amino acids are added on. This will eventually create the polypeptide. Now that we've begun with the start codon, the mRNA shifts a little through the ribosome so that the next codon is up for grabs. Let's say the next codon is UAU, so now we need a tRNA that has the matching anticodon, AUA. Well, look, here's a tRNA with the right anticodon, and it's brought along a tyrosine. Tyrosine is an amino acid that is specified by the codon UAU. The tRNA attaches to the mRNA in the ribosome and lines up tyrosine right next to the waiting methionine. A peptide bond forms between the 2 amino acids. Then the first tRNA leaves everyone else behind and floats off to find more work to do. Poor methionine. Now it's just drifting about like a lonely kite in the wind. That tRNA left methionine hanging by only one anchor, its peptide bond with tyrosine. The tyrosine is still attached to its own tRNA, which in turn is clinging to mRNA inside the ribosome. Already we can see the beginnings of a polypeptide elongating outward. Should we walk through that process one more time? Let's keep everything just as we have it here and move on to add our third amino acid. mRNA shifts over again, and now the third codon is ready for a match. What's the codon? CAC. Here comes a tRNA with the matching anticodon GUG. It's also brought us a histidine since CAC codes for histidine. The tRNA's anticodon matches up with the mRNA's codon, putting the histidine in perfect position for making a peptide bond with tyrosine. Now we have methionine, tyrosine, and histidine all connected. We won't be needing tyrosine's tRNA anymore, so that tRNA detaches and floats away, just like the first one did in the beginning. Now we have an even longer kite. Methionine and tyrosine are drifting around with only their peptide bonds to hold them down to the ribosome. But the histidine is still connected to its own tRNA, and it'll stay that way until it has the next amino acid to latch onto. You can see how this chain of amino acids would grow longer as each new codon is translated. The addition process and peptide bond formation continues over and over again until the chain is about 100 amino acids long. The chain finally ends when a stop codon moves into the ribosome. This is the final step of translation called termination. Termination begins with the arrival of one of the three stop codons, UAA, UAG, or UGA. When any one of these enters the ribosome, the last amino acid cuts off its anchor to the last tRNA. The tRNA and ribosome are no longer needed. The gene has been successfully translated, and now we have a completed polypeptide. The polypeptide is finally free to drift happily all over the cytoplasm, untethered by covalent bonds. This final step, termination, is the end of polypeptide synthesis, signaled by a stop codon entering the ribosome. So, does that mean we finally built a protein? Well, no, but we have built a polypeptide. It often takes more than one polypeptide to build a protein. Remember that protein synthesis is a separate process from translation, so just because you've made a polypeptide doesn't mean you've made a protein. When we talk about the process of translation of genetic material from DNA to RNA to a complete protein, it's important to remember the role played by the ribosomes themselves in the phases of initiation, elongation, and termination. Ribosomes are made up of two subunits, one large and one small. There are 4 sites located on the ribosome. One of these sites is for the mRNA, and 3 on the large ribosome subunit are for the tRNA. These 3 tRNA sites are called the P, A, and E sites. During initiation, the initiator tRNA is found on the binding site of the ribosome that is called the P site. The second ribosome binding site, called the A site, is at this point open. However, when a new tRNA molecule recognizes the next codon directions on the mRNA sequence, that new tRNA attaches itself to this open A site. Then the amino acid of the tRNA in the P site and the amino acid of the tRNA in the A site become connected by a peptide bond. As the ribosome continues to move along the mRNA molecule, elongation of the chain occurs. The tRNA in the P site is released, and the tRNA in the A site is relocated over to the P site. The A site is again vacant at this point and ready for another tRNA to come along with new codon instructions from the mRNA. The process continues like this until a long protein chain is formed. Finally, in the termination phase, the ribosome has reached the termination codon on the mRNA. The E site on the ribosome is the exit site, and at this point, after losing its amino acid, the last tRNA is released by the ribosome. Then the new protein chain undergoes whatever modifications are needed for it to function as intended. Now that we've worked through all the details of translation, we can give it a more specific definition. We can say that translation is the synthesis of a polypeptide using the genetic code found in mRNA. It still fits into our earliest outline of the central dogma. Translation is the mechanism by which the information in RNA is transformed into a protein. In this lesson, we learned the 3 steps of translation. Let's go back and make sure we've got all of them nice and clear. Initiation of translation occurs when mRNA, tRNA, and an amino acid meet up inside the ribosome. Once translation has begun, it continues down the line as mRNA shifts along through the ribosome. Each new codon matches with a new tRNA anticodon, bringing in a new amino acid to lengthen the chain. During elongation, amino acids are continually added to the line, forming a long chain bound together by peptide bonds. Once a stop codon reaches the ribosome, translation stops or terminates. At termination, the polypeptide is freed from the ribosome and tRNAs stop bringing the amino acids in. All of the components come apart from one another and translation is done. The result is a brand new free-floating polypeptide.
Have you ever thought that you could be a mutant? Have you ever wished you were? Maybe if you were a mutant, you could sprout wings and fly like a bird, or turn invisible, or shapeshift into different animals. Maybe if you got stung by a radioactive bee, or if you were raised in a wastewater treatment plant, then you could develop amazing superpowers and run around fighting crime all day. Alright, so we know that's not going to happen to any of us. Radioactive insects and industrial wastewater would probably do more harm to us than good. But there are mutant organisms out there, and they're the result of genetic mutations. Mutations can occur in any organism, from people and other animals to plants, bacteria, fungi, and protists. A mutation is any change in the nucleotide sequence of an organism's DNA. Some mutations are more drastic than others. For example, chromosome mutations involve changes to large sections of DNA or even entire chromosomes. We'll save those mutations for the chapter on genetics. Here, we're going to focus on smaller mutations called point mutations. Point mutations are changes in the genetic sequence that occur at a specific point along the DNA strand. There are lots of different ways a point mutation can come about. They're caused by random mistakes that occur while a DNA sequence is being built. Point mutations don't usually happen to more than one nucleotide. They generally involve a single nucleotide being added, subtracted, or replaced with a different kind. We can split up point mutations into two basic types: base substitutions and insertions and deletions. We'll look at base substitutions first. Base substitutions are just what they sound like. They're point mutations in which one nitrogenous base is substituted by a different base. Take this DNA sequence, for example, GATTACA. Let's say there's a mistake in the DNA strand which causes the first thymine, or T, to be switched with a guanine, or G. Now the nucleotide sequence is slightly different. It reads GAGTACA. A single base substitution has occurred. Base substitutions are further divided into two types: transitions and transversions. Transitions are base substitutions that swap a purine for another purine, or a pyrimidine for another pyrimidine. You may have forgotten the difference between a purine and a pyrimidine, so let me refresh your memory. A purine is a double-ring nitrogenous base, like adenine and guanine. A pyrimidine is a single-ring base, like thymine and cytosine. You can remember the difference by using the phrase, "all gods are pure." So A and G are purines, and C and T are pyrimidines. Let's get back to transitions and transversions. A transition is when a purine is swapped for a purine, or a pyrimidine for a pyrimidine. If a transition happened to our original DNA sequence, it could be that the G is swapped for an A. That would be a purine to purine substitution. We would also call it a transition if one of the T's was swapped for a C, or the C to a T. Now, if you switch a purine with a pyrimidine, or vice versa, then we call it a transversion. There are more possibilities for transversions than there are for transitions. If we take the nitrogenous base adenine, which is a purine, then it can make a transversion by being replaced with a C, or by being replaced with a T. While the A has two ways of making transversions, it only has one way of making a transition. The two words sound really similar, don't they? Just remember that in a transversion, the nitrogenous base switches to a different version, from purine to pyrimidine, or vice versa. And to remember transitions, just imagine this graphic and that all the transitions sit at the same level with each other. Alright, so we've covered transitions and transversions. Both are types of base substitutions. Base substitutions, remember, are just one kind of point mutation. The other kinds are insertions and deletions. Rather than just replacing a nucleotide base, insertions and deletions involve adding or subtracting a pair of nucleotide bases. An insertion is a point mutation in which one or more base pairs is added to a DNA sequence. Let's see what an insertion would look like. We'll pick a random site in the sequence, right here between the two thymines. This will be the site of insertion. An insertion mutation would add a base right in between the T's, making the entire sequence longer by one base. Now the DNA reads a slightly different sequence, and it's longer than before. On the other hand, a deletion would be just the opposite. A deletion is a point mutation in which one or more base pairs is removed from a DNA sequence. If a deletion mutation happened to our strand, then we would lose one base, and the DNA strand would end up one base shorter. We haven't yet discussed how the point mutations in DNA can affect the function of a protein. So before we get into all that, let's make sure we understand the basics. All mutations are changes in the nucleotide sequence of DNA, which occur by way of random mistakes. While some mutations involve large sections of DNA, point mutations are changes in the genetic sequence that occur at a specific point along the DNA strand. Some point mutations are base substitutions in which a single nitrogenous base is replaced by a different one. When a purine is replaced by another purine, or a pyrimidine by another pyrimidine, we call it a transition. When a purine is replaced by a pyrimidine or vice versa, we call it a transversion. Point mutations also include insertions and deletions. An insertion is when base pairs are added to a DNA sequence, and a deletion is when base pairs are removed from a DNA sequence. What do you think of when you hear the word mutation? Do you think of a vile three-headed monster from ancient mythology? Do you think of fantastic animals with magical powers? Or do more realistic images come to your mind? It may be entertaining to think about all the wild mutations out there, but the truth is most mutations are not that extreme. In biology, a mutation is any change in the nucleotide sequence of DNA. Some mutations affect large sections of DNA, and others only affect certain points along the DNA strand. These small, specific mutations are called point mutations. We've talked before about the different kinds of point mutations. Some are base substitutions in which a single nitrogenous base is replaced by a different one. Other point mutations are insertions and deletions. We know all about the changes that occur in point mutations, but so what? They're just tiny alterations to a DNA strand. How can that have a significant effect on a living creature? Always keep in mind the original idea set up by the central dogma. The central dogma says that DNA makes RNA and RNA makes protein. By this point, you probably know that the code in DNA determines the code in mRNA, and the code in mRNA determines the sequence of amino acids. The amino acid chains are the basis of protein structure, so if something goes wrong with the DNA, then the mRNA will also be wrong. If the mRNA is wrong, then the amino acid chain will be wrong, and that will cause a problem in the protein. Let's take a look at how different mutations can affect a hypothetical organism. We'll call our organism the pink-winged pony. It's a pony, and it has big pink wings, and it can fly. The feathers on its wings are made of the protein keratin, but it's a special kind of keratin that gives the pony its magical flying ability. We'll call it magic keratin. Magic keratin is a protein made of a single polypeptide, a single chain of 4 different amino acids. The first amino acid is methionine, the second is proline, the third one is serine, and the last one is valine. They just happen to be in alphabetical order. These 4 amino acids must be built correctly by genetic translation. Otherwise, the pony's magic keratin won't be properly synthesized. The pink-winged pony may even lose its ability to fly. We now know which amino acids we'll need, but how do we get them? They have to be translated from the mRNA code. The genetic code for magic keratin is AUG, CCA, UCA, GUU, UGA. The UGA is a stop codon. This code is the mRNA version of the magic keratin gene. But the mRNA came originally from DNA. To find the original DNA code, we have to use complementary base pairing. Make sure you put in the letter T instead of U, since we're going from RNA to DNA. So the original DNA code would have been TAC, GGT, AGT, CAA, ACT. This is the original gene for magic keratin. Now we've got our DNA strand, and now we can try out our own point mutations. We're going to test a series of base substitutions. We'll first take the second G here and replace it with a T. This is a new DNA code, so it will yield a slightly different mRNA code. From our codon chart, we know that AUG codes for methionine, CAA codes for glutamine, UCA codes for serine, and GUU codes for valine. And again, UGA is a stop codon. Well, it's close to our magic keratin polypeptide, but where proline was supposed to be the second amino acid, we got glutamine instead. So our protein is slightly off, and so are the feathers in our pink-winged pony. Oh no, our pony has changed. He can still fly, but his wings are all spotted. He may have trouble attracting a mate. This is an example of a missense mutation. A missense mutation is a point mutation that changes a codon to indicate a different amino acid. This usually changes the polypeptide and therefore can change the function of the overall protein. We call it a missense mutation because it causes the protein to be incorrectly translated from the original gene. Our pony incorrectly received spotted wings when he was supposed to get wings of plain pink. Let's start over and try a different base substitution. We'll switch out this G for a letter C. The resulting mRNA will be changed by one letter. So what will be the resulting polypeptide? AUG codes for methionine, CCA codes for proline, and UGA codes for... Wait, that's a stop codon. So we don't even get to keep building. We have to stop short, and now the magic keratin won't be produced at all. You know what that means? Our pink-winged pony will have to go featherless. This type of mutation is called a nonsense mutation. It's a point mutation that turns one codon into a stop codon. Nonsense mutations always result in the early termination of a polypeptide. In other words, the chain stops short and the protein is never completed. Our pony doesn't get any feathers and he'll never be able to fly. Now we'll start over with a third kind of base substitution. Let's switch out the last A for a T. Just looking at the setup, can you guess which of the 4 amino acids will be affected by this change? That's right, the fourth one. So we get the mRNA strand that codes for methionine, proline, serine, and valine. Hold on a second, the amino acid didn't change. Why did we still get a valine at the end? Oh, even though our codon changed, it still specified a valine. See, both GUU and GUA code for valine. A point mutation that has no overall effect on a protein's function is called a silent mutation. Sometimes a silent mutation results in the same exact sequence of amino acids. That's what happened here. Other times in a silent mutation, there's an amino acid change, but it doesn't affect the overall function of the protein. In the case of our pony, the magic keratin wouldn't be changed at all by this type of mutation. He would still have plain pink wings, and he'd still be able to fly. Using the pink-winged pony was a fun way to learn about the consequences of point mutations, but in real life, point mutations are often not so fun. They can cause health disorders because they end up changing the way important proteins work. Sickle cell anemia is one disease that's caused by a missense mutation. The mutation occurs in the hemoglobin gene. Hemoglobin is a protein that helps us carry the oxygen in our bloodstream. It also affects the shape of our red blood cells, which need to be round in order to move smoothly through our blood vessels. If someone has the missense mutation in their hemoglobin gene, their red blood cells will be sickle-shaped and won't travel well in the vessels. As a result, this person will have trouble circulating oxygen throughout their body. They'll become anemic, which is why they're diagnosed with sickle cell anemia. One disorder that's caused by a nonsense mutation is Duchenne muscular dystrophy, also called DMD. Patients with DMD suffer from severe muscular weakness that gets worse over time. They have a nonsense mutation in the dystrophin gene, which codes for the protein dystrophin. Dystrophin is a structural protein that helps make up our muscle tissue. When someone has a nonsense mutation in the dystrophin gene, then their muscles can't develop correctly. Patients with muscular dystrophy have trouble with walking and other important motor skills. Point mutations can cause serious changes to an organism if they change the way a protein works. A mutation in DNA alters the mRNA, which in turn can alter the amino acid chain. A base substitution may have 3 different effects on an organism's protein. It can cause a missense mutation, which switches one amino acid in the chain for another. It can cause a nonsense mutation, which results in a shorter chain because of an early stop codon. And a base substitution can also cause a silent mutation, in which the protein's function doesn't change at all. Base substitutions are responsible for disorders like sickle cell anemia, which is caused by a missense mutation, and Duchenne muscular dystrophy, which is caused by a nonsense mutation. Have you ever wondered how mutations arise in living things? You've probably heard stories about mutated beings emerging from toxic waste sites or secret laboratories. While these urban legends usually turn mutations into fantasy, it is true that mutations often come from external factors. The environment we live in has a real impact on whether we experience genetic mutations. The quality of water we drink and the air we breathe can actually affect the integrity of our DNA. Our bodies are designed to correct any mistakes, but dangers from the environment can increase our chances of ending up with a mutation. An environmental agent that causes a mutation is called a mutagen. Remember that all mutations are changes in the nucleotide sequence of DNA. While chromosome mutations involve large portions of DNA, point mutations are smaller and typically affect one or two bases. Point mutations include base substitutions and insertions and deletions. While base substitutions only switch one base for another, insertions and deletions change the length of a DNA code. But it's rarely the original DNA strand that undergoes a mutation. Most of the time, mutations occur while a new DNA molecule is being formed through DNA replication. Recall the work of the enzyme DNA polymerase. During replication, this enzyme's job is to build the daughter DNA strand along the parent strand. Every once in a while, DNA polymerase makes a mistake in matching the right nucleotide to the parent strand. It puts the wrong nucleotide down and produces a daughter strand that is slightly incorrect. Normally, this only happens in about 1 in a billion nucleotides, but there are certain things that can make it more likely that DNA polymerase will make a mistake. One of these is radiation from ultraviolet, or UV, rays. When UV radiation hits the cells in your body, it can change the way DNA polymerase works and increase the probability of a replication error. Ultraviolet radiation is an example of a mutagen. Things that cause mutagens are not always found outdoors. Mutagens are any chemical or physical agents that cause a mutation in an organism's DNA. We said before that mutagens come from the environment. But remember, the environment for an organism is nothing more than the chemical and physical factors surrounding it. A bacterium's environment might only be the inside of your intestine. A lab mouse's environment might only be its cage, its food, and its water. Mutagens can be found in any part of the environment. The word mutagen comes from the word part gen, which is found in lots of scientific terms. Gen means the origin or creation of something. Just like an allergen is anything that causes an allergy, a mutagen is anything that causes a mutation. UV radiation is just one of many mutagens that exist in our environment. Other mutagens include X-rays, extreme heat, or chemicals that react with DNA molecules. These agents can change the way molecules bond and react with one another, which increases the likelihood that a mistake will be made in the nucleotide sequence. An error in the DNA can cause problems for that cell. Skin cells are especially vulnerable to UV radiation because, obviously, they have the most direct exposure to the UV light that comes from the sun. This is why we try to protect our skin from the sun's potentially harmful rays. Though a point mutation may seem insignificant at the molecular level, we know that a single insertion, deletion, or base substitution can drastically harm an organism. Fortunately for us, DNA polymerase always double-checks its work to make sure all the nucleotides have been replicated correctly. When it finds an error in the DNA sequence, it quickly stops and fixes it before replication. That way, the mistake won't get bigger with every new generation of DNA. But nobody's perfect, and some mistakes do get through. It's all about probability. The best thing we can do to avoid mutations is to decrease our exposure to mutagens that are found in our environment. As we've seen in previous lessons, mutations can cause a variety of disorders. Human diseases caused by point mutations include cystic fibrosis, color blindness, Tay-Sachs disease, sickle cell anemia, and cancer. We usually think of point mutations as being harmful to living things. They often make organisms more vulnerable, less healthy, or otherwise less likely to survive and reproduce in their respective environments. On the other hand, there are plenty of cases of advantageous mutations. Don't get the false impression that all mutations are bad. Sometimes accidents are a good thing. In fact, mutations can take credit for many of the adaptations we see in organisms today. In any population, there are individuals that turn out significantly different from the rest. Mutations arise randomly in all species of organisms. If a mutation doesn't cause significant harm, then it may be passed down through successive generations. Over time, the mutation may become more and more common until it becomes the new normal. In fact, this is one way that species branch out and evolve new varieties of organisms. In the long run, all of us are the result of some kind of mutation, so we can thank mutations for the diversity we see in plants and animals today. Keep in mind, however, that simple mutations can't give an organism some amazing new structure or power. A person couldn't instantly sprout wings or fins because he got a mutation. All those legends about mutant monsters and lab-created superheroes are just fantasy. If you really want to gain superhuman powers, you're better off just hitting the gym. Point mutations are small changes in the DNA sequence that occur through mistakes during DNA replication. DNA polymerase normally matches the daughter nucleotides to the parent strand and corrects any mistakes through a proofreading step. Mutations are more likely to form when an organism is exposed to a mutagen. Mutagens are chemical or physical agents that cause mutations. While mutations often cause harmful disorders, they also increase the genetic diversity of populations. Advantageous mutations are passed on through generations and are ultimately responsible for the variety of living things. We already know that point mutations can cause changes to an organism. A mutation in DNA alters the mRNA code, which in turn can change what kind of protein is produced. Earlier, we saw the effects of base substitutions. We used the example of the pink-winged pony to describe these effects. A missense mutation makes a slight change to a protein, a nonsense mutation blocks a protein's production, and a silent mutation does not affect the protein at all. These three different effects are all caused by base substitutions. They all result from the switching of one base for another. If we look back at our chart on point mutations, we see that missense, nonsense, and silent mutations all happen because of base substitutions. But base substitutions are only one kind of point mutation. We haven't yet discussed the effects of insertions and deletions. Insertions occur when one or more base pairs are added to the sequence, and deletions occur when base pairs are removed from the DNA sequence. Insertions and deletions actually change the length of the DNA strand because they add or subtract one base pair from the code. That's a big deal compared to base substitutions. Insertions and deletions can have drastic effects on the DNA code and the final protein. Let's dig deeper to learn more about the consequences of these disruptive mutation types. We'll first revisit the magic-keratin gene that we used in our previous examples. It's an imaginary gene that codes for the magical keratin protein found in the feathers of the pink-winged pony. Here's the original DNA strand. The code is first transcribed into mRNA, and then it's translated into a chain of amino acids. The chain we normally get is methionine, proline, serine, and valine. That sequence makes up our magic-keratin protein. Now, what do you think would happen if we added one base to the strand of DNA? Would it change the length of the mRNA strand? Would it change the length of our amino acid chain? If so, then how much? Would we get one extra amino acid? 2 more? 3 more? What sort of effect would this have on the final product? Let's think back to our codons for a minute. Whenever we translate an mRNA code, we read the letters in groups of 3. We look at the long strand of bases, draw a few lines, and then home in on one codon. We keep our focus on that codon while we check the RNA codon chart to find the matching amino acid. In that moment, we sort of have tunnel vision for the codon in question. That tunnel vision is actually called a reading frame. It's a way of dividing the mRNA into codons and focusing on one codon at a time. The reading frame is something that we as humans use, but the concept is still the same for the molecules that are in charge of genetic translation. Inside our cells, ribosomes and tRNAs are working hard to match amino acids to every codon. They have their own sense of the reading frame and they build polypeptides accordingly. So what would happen if we added a base? The DNA code would grow by 1 letter, and so would the mRNA code, giving us how many codons? Oh, weird, look what happened to our reading frames. They all got shifted over by 1. We still have 5 codons, but now there's 1 extra base. And the codons are all very different from the ones we had before. Our reading frames have shifted on the mRNA strand. This all happened because of a single base insertion. The effect that we get is called a frame shift. A frame shift mutation is a mutation caused by an insertion or deletion, which causes a shift in the translational reading frame. Frame shift mutations have a more dramatic effect on the polypeptide than missense or nonsense mutations. Instead of just changing one amino acid, frame shifts cause a change in all the amino acids in the rest of the gene. Let's take our magic keratin gene for the pink-winged pony and see what results from a frame shift mutation. Here's the original DNA strand. We'll make an insertion of an adenine base right here between the G's. Here's the resulting mRNA strand, and here's the resulting polypeptide, methionine, leucine, isoleucine, serine, and another leucine. Whoa, that's nothing at all like our magic keratin protein. I don't know what that is, but it's definitely not something that will give normal feathers to our pink-winged pony. If this kind of mutation happened, then the feather proteins would turn out very strange. The magic keratin would be incorrectly synthesized due to a serious defect in the amino acid chain. It'd be a safe bet that this pony couldn't fly at all. So we've tried an insertion. What about a deletion? Let's say we deleted the second G from the original DNA strand. Our resulting mRNA would give us the following polypeptide: methionine, histidine, glutamine, and phenylalanine. Once again, it's nothing like our magic keratin polypeptide. If this deletion happened to our pony, we'd get something just as bad as what we got from the insertion. By now, you can see that frame shifts have a much more dramatic effect than single base substitutions. While base substitutions alter just one amino acid, insertions and deletions can alter giant portions of proteins. The effects of frame shifts are more dramatic when they occur in earlier parts of the gene. For example, if you had a gene made of 23 codons and the frame shift happened at codon 21, then only a small part of the protein would be changed. But if the frame shift happened at codon number 3, then pretty much all of the protein would be drastically different. We should mention here that not all insertions and deletions cause a frame shift mutation. If the segment being inserted or deleted is 3 nucleotides long, then the reading frame still lines up properly with all of the codons. If an insertion or deletion consists of nucleotides numbering a factor of 3, then a frame shift will not occur. It's also important to note that insertions and deletions can cause nonsense mutations. Even though we talked about nonsense mutations arising from base substitutions, we can also get them in cases where an insertion or deletion yields one of the 3 stop codons. Now let's look at how frameshift mutations can affect human health. You may have heard of a disorder called Tay-Sachs disease. Tay-Sachs is often caused by a frameshift mutation in a gene which codes for an important enzyme. Enzymes, remember, are also proteins. The enzyme affected in Tay-Sachs disease is called hexosaminidase A. Its job is to keep harmful chemicals from building up in our nerve cells. With a mutation on the hexosaminidase gene, the enzyme doesn't get built correctly and harmful chemicals accumulate in the nerves. People with Tay-Sachs may suffer from muscle weakness, seizures, deafness, blindness, or dementia. Tay-Sachs is a serious genetic disease that shows just how damaging a frameshift mutation can be. Point mutations are small changes that happen to the DNA sequence. They're divided into 2 groups, base substitutions and insertions and deletions. Base substitutions cause relatively minor mutations like missense, nonsense, and silent mutations. But insertions and deletions cause a change in the length of a gene, which causes a shift in the codon reading frame. A frameshift mutation occurs when a protein is drastically altered because of an insertion or a deletion. Tay-Sachs disease is a human disorder caused by a frameshift mutation. In general, the effects of frameshifts are much larger than those of base substitutions.
A mutation is a permanent change in a DNA sequence. DNA is the unit of heredity of all organisms, so this means that mutations can often be passed on to offspring. Mutations don't have to be bad. Some are beneficial, while others are neutral and have no effect. There are many different categories of mutations, but in this lesson, we will focus on point mutations, or those resulting from a change in one or a few nucleotides at a single location in a DNA sequence. Nucleotides are the repeating units of a DNA sequence. There are 4 nucleotides, each with a different nitrogenous base. Thymine, T. Adenine, A. Guanine, G. Cytosine, C. Genes are sequences of DNA that code for proteins, and the order of these bases determines the order of amino acids in a protein. Not all mutations are in these coding sequences, but if they are, then the result can be that different or non-functioning proteins will be made. There are two types of point mutations: base substitutions and frame shift mutations. Base substitution occurs when one base is switched out with another base. In this example, the C was replaced by an A, changing the second codon sequence. Frame shift mutation occurs when one base is added or removed. There are two types of frame shift mutations. An insertion is a mutation in which one or few nitrogenous bases are added to a DNA sequence. This results in a sequence that is a few bases longer than it should be. In this example, a T was added after the first A, shifting all the other bases to the right and affecting all the codon sequences. A deletion is a mutation in which one or a few bases are removed, resulting in a shorter than normal sequence. Here you can see that the first G was removed, creating a sequence that is too short and that affects both codons. Insertions and deletions are called frame shift mutations because they do not just affect one codon, a 3-base sequence that codes for one amino acid, like in base substitutions. Instead, frame shift mutations affect all the codons that occur after the point mutation. This changes how a DNA sequence is read or the reading frame. Many diseases are caused by point mutations. Let's look at a few of them. Base substitution, sickle cell anemia. One base substitution in a certain location in the gene for hemoglobin causes red blood cells to become abnormally shaped and clog blood vessels. If someone has 2 copies of this mutation, people get one from their moms and one from their dads. Then they will have the disease sickle cell anemia. However, if someone only has 1 copy of this mutation, he or she does not have the disease and actually benefits by being protected against malaria. Insertion, Tay-Sachs disease. Tay-Sachs disease can occur when a short nucleotide sequence is inserted into the hexosaminidase gene. This gene makes a protein important in removing specific toxins from the nervous system. If a mutation occurs in this gene, then nerve cells will start to die, causing symptoms of Tay-Sachs disease, which leads to death in infants and toddlers. Deletion, Duchenne muscular dystrophy. A deletion of a certain nitrogenous base in the dystrophin gene causes the disease Duchenne muscular dystrophy. This disease is sex-linked and tends to be much more common in males than females. It causes weakened muscles and early death just by one little base being removed. A point mutation is a change in one or a few nitrogenous bases at one location along a DNA sequence. Base substitutions are point mutations where one base is swapped for another. Insertions are point mutations where one or a few bases are added at one location in a DNA sequence. Deletions are point mutations where one or a few bases are removed from one location of a DNA sequence. Insertions and deletions are also called frame shift mutations.
The genome is the name that we give to the DNA content of the cell. Now, if we're thinking about DNA as a recipe for life, organized into a library, what happens in a library? You have a lot of organization. You have the information organized into discrete packages, books, right? And the DNA is organized into discrete structures, and those structures are known as chromosomes. So if we think about the chromosomes as, say, cookbooks, since we're working with this library analogy, each cookbook has recipes in it, right? Now, you might think that the biological information is organized by process or cell or something like that. In terms of a cookbook, we might think about that in terms of having a book for a specific type of food, say, Chinese and Italian, Japanese, things like that, right? So actually, it's more complicated than that. The cookbooks are more like general cookbooks. So each cookbook has an assortment of recipes. So I might have Chinese, Italian, and Japanese all in the same cookbook. And another cookbook might also have Chinese, but also have Korean and, say, Thai. All right? So say I want to have a three-course meal, I might have to access 3 different cookbooks if these cookbooks are all general cookbooks. By the same token, if I am trying to, say, make a mitochondria, I might have to access 3 different chromosomes. Now, it's important to note that this is a super oversimplification. Obviously, it's going to take many more than 3 chromosomes and 3 products of those chromosomes to be able to make something as complicated as mitochondria. But just for the sake of argument, let's just use that example. So as you can see here, it's really important then for the cell to be able to prevent loss of any information. So if I was to lose one of these chromosomes, I'm not going to be able to make this mitochondria. As I was saying before, the chromosome is coding for more information than just one process or one structure. So by the same token, now maybe I'm not able to make, I don't know, red blood cells or something like that. So the loss of even one chromosome is usually considered a lethal event. So the cell employs many mechanisms then to prevent this loss from happening. So most multicellular organisms like humans possess more than one chromosome. So humans possess 46 chromosomes in most of their cells. The exception to that rule is gametes. Now, gametes are going to be either egg cells that are produced by females or sperm cells that are produced by males. Now, the gametes only possess half as many chromosomes as most of the rest of our cells do. So they possess 23 chromosomes. Now, the number of chromosomes that a gamete possesses is referred to as the haploid number. So in the case of humans, the haploid number is 23. Now, haploid is often abbreviated N. What happens now is because we have only 23 chromosomes in gametes, when you have your egg and your sperm, when those 2 cells fuse, we can reproduce the normal chromosome count of 46. So it takes 46 chromosomes to cook up a human, so to speak. So we have 23 chromosomes in the egg, and we have 23 chromosomes in the sperm. Now, there are 23 unique chromosomes in each of these cells. So what that's saying is that both mom and dad are contributing some of their DNA. On chromosome 4, mom might be coding for, say, freckles, but dad might be coding for a completely different trait. He might be coding for no freckles. So these copies of chromosome 4 are similar yet different versions of the same information. Now, if we think about that in terms of the recipe book, think about this as being 2 different versions of the same cookbook. And the 2 different versions of the chromosomes are referred to as homologous chromosomes, also known as homologs. So you might ask, why have multiple sets of chromosomes? Essentially what I've said is we have 2 complete sets of chromosomes. If I was a chef, why bother to have 2 complete sets of recipe books? You know, if I already have Martin Yan's cookbook, why have the exact same cookbook that he published a year later? And essentially, if we are saying that a haploid set of chromosomes, so if this is, say, the egg set, and this is the sperm set, we have 23 chromosomes here in each one. And if these are 2 complete sets, really all I need is one of these sets. Really all I need is one set of 23 chromosomes to be able to have all of the information that I need to be able to code for a human. Having two complete sets of chromosomes here, we refer to this as being diploid. So organisms that have basically two haploid sets of chromosomes are called diploids. And chromosomes with a diploid complement have twice as many chromosomes as gametes, or 46 chromosomes. And we also abbreviate this as 2n, which is equal to 46. You can remember diploidy by just remembering some root words. So we talked about haploid, and this is approximately but not totally equal to one chromosome complement. So slight oversimplification. So if we just take this root ploid and we add di in front of it, di is a root that usually refers to 2. So what we're really saying is we have two sets of chromosomes. There are many reasons why scientists believe that diploidy evolved. One of them is an idea of backup information. If I have my set of cookbooks and say one of the pages gets ripped, if I have one of the old additions to the cookbook, I could actually go and photocopy that and replace the information that's been lost. And no recipes are more important than the recipes that are found on the chromosomes, because pretty much if any information is lost, that really, as we talked about before, is pretty much equivalent with death. So having a backup chromosome means that if I have some chromosome damage here, if I have a backup chromosome, I can use that to make some repairs and repair this DNA and have, again, two complete sets of chromosome information. So this is just one reason why diploidy is important. There are many more, and we'll explore some of those at a different time. We've learned today that the genome is the name that we give to the DNA content of the cell. And DNA is organized into chromosomes. Think of it, if the information in the genome are like recipes, the recipes are organized into cookbooks, aka the chromosomes, and those chromosomes are stored in the nucleus or the library. Chromosomes are basically just distinct collections of DNA in the genome. Now, we have different versions of each chromosome, and these are called homologous chromosomes or homologs. Both mom and dad are contributing one homologous chromosome to the new cell, and we can refer to the chromosome complement of these gametes, or the egg and sperm mom and dad are donating, as having a haploid complement of chromosomes. And this is abbreviated N. Humans have 23 chromosomes in their gametes, or N equals 23. Now, when those two gametes fuse, you end up reproducing the number of chromosomes that are found in most of our cells, and this is called a diploid complement. In other words, you have two haploid sets of chromosomes, and this is abbreviated 2n. So 2n equals 46. And hopefully this has helped you understand how DNA is organized into chromosomes and how chromosomes make up the genome. I think I'm going to be untangling these Christmas tree lights until New Year's. Well, at least I don't have it as bad as the cell does. Can you believe the cell has 3 billion bases of DNA, and it spends most of its time as tangled up as this? So, as you recall, DNA is the storehouse of information in the cell. And DNA is organized into chromosomes. And all of the DNA in the cell is referred to as the genome. So DNA is a linear molecule. It's kind of like a string. It's kind of like our light strings that we're talking about. The difference here, though, between our tangled Christmas tree lights and our DNA is that we have 3 billion bases. So that's a lot of string that's all got to be wadded up inside of the nucleus. So we've got to figure out a way to efficiently pack that DNA into the nucleus, one, so that it just fits inside the nucleus, and two, so that we can actually access the information that DNA has to offer. We have proteins that help us out with this job, and they're called histones. Histones are proteins that are going to help package the DNA more efficiently. And what's going to happen is, if I have my histones here, we're basically just going to take our linear DNA molecule and we're going to roll it around the histones. So now by rolling it around these histones, the DNA has necessarily become shorter, right? If I had this long piece of DNA, and if I'm rolling it up around balls, it's going to create a shorter length of linear molecule now that we have to fit inside the nucleus. Scientists, we like to name things and everything, so here we have the histone in the middle, the histone core, and this structure of histone and DNA, this little ball structure here, we refer to that as the nucleosome. All right? Now, all of this DNA and protein organization, all of the DNA rolled around the histone proteins, that's going to be called chromatin. Now, the nucleosomes are the basic subunit of the chromatin. What you can see, if you were to look under a high-powered microscope, is you'd see what looks like a beaded structure, right? So if I'm looking through the microscope and I have my cell and my nucleus, I'm going to see a lot of kind of stringy-looking things in here, and each of those stringy-looking things are going to look like a beaded structure. And it gets this beaded structure from the repeating segments of nucleosomes and linker DNA. So I said that it's going to be important not only to package the DNA into the cell, which organizing it into chromatin has helped us do, but I also said it's going to be important to be able to actually find information in the DNA. Right? So we said that DNA is a storehouse of information, and DNA is basically providing information to create different things that the cell is going to need. So if the cell needs microtubules, DNA is going to code for that. If the cell needs to make mitochondria, DNA is going to code for that. So that the DNA is going to code for things that the cell needs to be able to make all of the structures that it needs to be a cell. It's also going to code for things to tell that cell what to do. So telling that cell if it's going to be a red blood cell or if it's going to be a muscle cell. So it's providing all of the information the cell needs to be able to provide function not only for itself but also for you as an organism. The information that's stored in chromosomes is referred to as a gene. So a gene is a distinct unit of DNA that can code for RNA, and in turn that RNA can make protein. And then the proteins can provide the functionality that the cell needs. You might think of chromosomes then as just being jam-packed with genes, one after another. Right? That would be the most efficient way to store all of the information, is to have as many genes as possible packed into each chromosome. It turns out that's not actually the case. What actually happens is chromosomes are organized with intervening non-coding DNA. Between most genes is DNA that's not producing any sort of protein. Historically, scientists actually referred to this as junk DNA because they couldn't imagine that this DNA served any purpose if it wasn't producing any proteins. We actually now know that that's wrong, and actually non-coding DNA serves a number of purposes, both structurally and functionally. So we've learned today about how DNA is organized into chromosomes, and then how that DNA is further organized within the chromosomes. Histones, which are proteins that help package the DNA, play a central role in that process. As the chromosomal DNA is wrapped around those histones, the DNA is organized into something that is called chromatin. And each subunit of chromatin, which is the little ball of DNA wrapped around the histone proteins, is referred to as a nucleosome. We've also talked about the functional organization of the chromosome. DNA is made up of genes. A gene is basically a segment of DNA which can produce a protein. All right, it's almost time for us retail employees to be decorating for Christmas, but gosh darn it, I got that biology midterm hanging over my head. I wonder if this Christmas tree decorating job can help me study for the cell cycle. Okay, let's see what Christmas tree decorating can tell us about the cell cycle. All right, say we have some Christmas tree displays that we have to decorate. Now, unfortunately, we only have one box of ornaments, but we want to decorate both of these trees identically. Luckily, though, we are in the future, and we're going to use a replicator device to make copies of the ornaments. And by using this replicator device, we'll be able to decorate both of the trees. This idea about making copies of things is a central idea in the cell cycle. So the cell cycle is basically all of the events that can occur during the lifetime of a cell. The cell, it can be thought of in either one of two states. It can be not dividing or dividing. Pretty simple, right? So let's layer on top of that a few terms. So whenever a cell isn't dividing, that's referred to as interphase. And then cell division is just cell division. Now, there are three different phases that interphase can be divided into. You can have what's called gap 1, which is also abbreviated as G1. You can have what's called S phase, and that S just stands for synthesis. All right, and we'll talk a little bit about what's getting synthesized in a minute. And then finally, interphase also involves gap 2, or G2. Now, by the same token, cell division can also be further subdivided. You can have M phase, and during M phase, mitosis occurs. And mitosis is then followed by cytokinesis. So let's look a little bit more closely at these cell cycle phases, and let's start out with G1. G1 is kind of the everyday life of the cell. If we're thinking about it in terms of our Christmas tree decorating, you know, normally a store employee during the normal part of the year is doing various normal things like stocking the shelves, selling items, that sort of thing. And the same thing can be said for G1 phase. Normal things are happening, cell growth is happening, protein synthesis is happening. But towards the end of G1, what's going to be happening is the cell is going to be preparing for S phase. Preparations need to be made, just like as Christmas time is approaching, the store employee is going to have to start preparing for the decorating job that's going to occur. So we need to decorate those two trees, and we're going to use our replicator to make copies of the ornaments for each of the two trees. By the same token, the cell wants to make copies of its chromosomes in a process that we've learned before is called replication. This part of the cell cycle is called S phase because the cell is going to synthesize new chromosomes. So as a store clerk trying to make duplicate displays, we want both of the displays at each end of the store to look the same. And unfortunately, since we're only starting with one box of ornaments, we have to use this replicator to be able to accomplish that. Now, we are talking about interphase as a non-dividing part of the cell cycle, but basically what we can think of is we can think of interphase, at least in one sense, as being preparations for cell division. Now, if we have our one cell as a multicellular organism and we need to make two cells, we want those two cells to be duplicate copies of each other. As part of this process, we need to be able to make duplicate copies of our chromosomes. That's basically the point of replication, and S phase, this event in the cell cycle, is to make duplicate copies of the chromosomes. And as the cell is making copies of the chromosomes, it's synthesizing what are called chromatids. So a chromatid is basically just one of the two copies of the chromosome. So we have one copy of the chromosome here, then once replication occurs, we have two copies, and now we refer to these individual copies as chromatids. So we have two copies of the chromosomes, i.e. two chromatids. All right, so now that the cell has made copies of its chromosomes, it's almost ready for cell division. Just like as we've made copies of the ornaments, we're almost ready to decorate the tree. And let's say our trees are pretty tall. These are like those super big kind of trees that are out in front of a big department store. And we're going to need a ladder to be able to put some of those ornaments towards the top of the tree on, especially say like the star that's going to go right on top of the Christmas tree. So, we're going to have to trudge on down to the basement to be able to get the ladder to be able to finish the Christmas tree setup. Now, by the same token, the cell is going to have to spend some time making some final preparations for actually dividing its chromosomes, and these preparations occur during G2 phase. So lots of protein synthesis and more cell growth occur during this phase. And for instance, proteins that are needed for cell division, like say microtubules, are made. And we'll talk more about how something like a microtubule is important to cell division a little bit later. Now that we've gone through all of the steps of interphase, we're pretty much ready for cell division now. As you recall, there are two different steps in cell division. We have M phase, and during M phase mitosis occurs, and then we have cytokinesis. In terms of our Christmas tree decorating, once we've made duplicate copies of the ornaments, we'll be able to decorate those two trees. Because we have the two different trees, we want to make sure that one kind of each ornament ends up on each tree. We wouldn't want two stars on this tree and no star on this tree, for instance. So as we're replicating it, we can actually make it easier for ourselves and put our ornaments in two different piles, and that'll ensure that each tree is going to get the same ornaments that can be used in the decoration process. It's a little bit more complicated for the cell when you think about cell division. So as the cell is making new copies of the chromosomes, it can't go and divide those chromosomes at the same time. It wants to wait and do it all at once. Just like, say, if I was making copies of these ornaments, if the trees were on the opposite sides of a really, really big store, I wouldn't want to walk over and decorate the tree with each ornament as it gets copied. I would want to wait until all of the ornaments have been copied and then decorate the tree. The cell is going to have to go through a more complicated process to divide its chromosomes. We'll learn more details about how this complicated process of mitosis occurs later. However, cytokinesis is actually a lot simpler and more similar to our Christmas tree decorating. So cytokinesis is basically the division of everything else except the DNA nuclear content of the cell. So basically you can think about this as dividing up the cytoplasm of the cell. What's going to happen is we're going to just kind of arbitrarily divide the cellular contents into two new cells. And again, we'll talk about this in more detail at a later time. So hopefully you've learned a little bit about the life of a cell during this lesson. We've learned that the cell cycle consists of all the events that occur during the life of a cell. Cell division is a process in which a mother cell divides and gives rise to two new cells, whereas interphase is the non-dividing state during a cell's life. We can further divide interphase into three smaller steps known as G1, during which preparations for S phase occur. S phase, in which the chromosomes of the cell are replicated. And G2 phase, during which preparations are made for M phase. Now, the copies of the chromosomes that are made during S phase are referred to as chromatids. Cell division can be divided into two steps as well, and those are known as M phase, during which mitosis occurs. And remember that mitosis is the process in which the chromosomes are divided in preparation for cytokinesis. And cytokinesis is the process in which the cytoplasmic contents of the cell are divided. Can you believe our job can basically be summarized by the cell cycle? Is it me, or do these Christmas tree lights kind of look like chromatin to you?
Why is mitosis important? Cell division is an important part in the life of any multicellular organism. Although we all started from a single cell, cell division enabled us to grow in size to the person we are today. And cell division prevents us from running out of things like blood or skin cells. As noted when we studied the cell cycle, one of the most important goals of cell division is to make sure that each new daughter cell receives a full complement of genomic DNA. Since DNA is the blueprint for building a cell, any error in DNA could result in a defective cell. A single DNA error could potentially lead to disease or even death. The need to copy and move 3 billion bases of DNA around the cell leaves many opportunities for problems to occur. To ensure careful handling of the cellular blueprint, equal division of the cell's nuclear DNA is performed separately from all of the rest of cytoplasmic contents. The process of separating the nuclear DNA into identical complements for the new daughter cells is called mitosis. How does all the DNA fit inside the nucleus? If you were to put all of the DNA molecules in a single human cell end to end, you could end up with a molecule approximately 2 meters or 7 feet in length. That's a lot of DNA to store in a space that's around 6/1,000,000th of a meter, or roughly 2 10,000ths of an inch. How could we possibly fit that much DNA in such a small space? Well, we've already learned that DNA is wound around proteins called histones to form chromatin. This is the first step in organizing and compacting the DNA. DNA is further packaged by winding the chromatin into a more compact structure. How does all that DNA move during mitosis? The goal of cell division is to produce 2 copies of the original cell. Everything in the cell must be copied, including the DNA. Despite the packaging strategies the cell employs to fit the genome into the nucleus, the chromosomes are essentially still strings. They're just much shorter strings. During cell division, the chromosomes must be capable of moving into the 2 new daughter cells. But the cell is like a water balloon, so the cell is going to need to move these strings around in a water environment. Let's consider the following race. One participant will move 46-yard-long pieces of string to a precise location in a wading pool without lifting the strings out of the water. The second participant will move 46 pens to a precise location in a second wading pool, again without lifting the pens out of the water. Do you think you could move the pens faster than I could move the strings? Assuming I'm not harboring some comic book-esque superhuman speed, I think you'd probably beat me. The pens are small and compact compared to the strings, so it's easier to move them through the water. The cell must deal with the same issue to move chromosomes through the cytoplasm during cell division. Therefore, to make the chromosomes easier to maneuver during mitosis, the cell further compacts the DNA in a process known as chromosome condensation. We know that DNA is wrapped around proteins called histones in order to fit inside the nucleus of a cell, but histones do a lot more than simply serve as sites for wrapping and shortening DNA. They can also change the very shape and architecture of chromatin itself, affecting the transcription of genes. DNA and histone proteins work together via special chemical signals that occur on them, along with the action of enzymes. These signals direct the histones to either open or close a chromosomal region, i.e. to unwind or to become more tightly wound. The subject of histone modification is a fairly recent topic of research, and thus far about 9 different types of histone modification are known. 3 of the more important or well-known types include histone acetylation, histone methylation, and histone phosphorylation. Histone acetylation, This histone modification leads to an open chromatin structure, which means the gene expression is increased. It involves the addition of an acetyl functional group to the histone. This enhances DNA replication and also aids in repairing damaged genetic material. Histone methylation. Histone methylation involves the addition of methyl groups to the histone proteins. Although histone methylation has been thought to generally decrease gene expression, it can either increase or decrease this, depending on where the methyl groups are added to the protein. Histone phosphorylation. Histone phosphorylation involves the addition of a phosphate group to the histone protein chain. This action empowers the cell to quickly respond to various environmental stimuli. When scientists examine the number and appearance of chromosomes in an organism, they typically observe chromosomes in the middle of cell division. Compare how difficult it would be as a scientist to distinguish individual chromosomes in this mass of chromatin versus condensed chromosomes during cell division. The condensed chromosomes are much easier to visualize, right? An organized picture of the condensed chromosomes is called a karyogram. Note that the chromosomes have been treated with a dye which stains each one with a slightly different pattern. In addition, most chromosomes can also be distinguished by length. A karyogram can be used to study the karyotype, which is the number and appearance of chromosomes in an organism. You can clearly see that this individual has 20 pairs of homologous chromosomes. Note that the chromosomes in the 23rd pair are not identical because this individual is male. Mitosis is the process of separating the nuclear DNA into identical complements for the new daughter cells. Chromosome condensation packages chromosomal DNA more compactly in preparation for mitosis. A karyogram is an organized picture of all the mitotic chromosomes which can be used by scientists to study the karyotype. The karyotype is the number and appearance of the chromosomes of an organism.
As we've seen so far, chromosomes are pretty much the key to life. The information that's stored in a chromosome provides recipes to produce vital cellular components. The importance of this information is underscored by the fact that the cell dedicates an entire process known as mitosis to accurately separating or segregating the chromosomes into daughter cells. Now, before we learn about mitosis, let's talk some more about chromosomes. Have you been able to keep track of all this chromosome terminology we've been throwing around? Yeah, okay, me neither. So let's see if we can write everything down and sort of clarify everything in our minds before we move on. So let's see. Basically, a chromosome is a single molecule of DNA which represents a specific subset of the information in the genome. And I think we said we were going to try to remember this as each chromosome being a different kind of cookbook, right? Now, we also said that each gene is located on a specific chromosome. And we said that made sense because if we think about each gene as a recipe, each recipe is only found in a specific cookbook. Now, a diploid organism, like, say, humans, has 2 copies of each chromosome. And we call those copies homologous chromosomes or homologs. And we can think of them as kind of like backup cookbooks that might vary slightly because we have actually different editions of the same cookbook. All right, so in preparation for cell division, a cell has to make a copy of its DNA in a process known as replication. So replication yields 2 copies of every chromosome, and these are called chromatids. So it's important to note that replication doesn't produce more chromosomes. Rather, it just produces 2 copies of a given chromosome. So following replication, there are 2 chromatids associated with each chromosome. And we often refer to these chromatids as sister chromatids because they're just basically copies of the same chromosome. So really we can just think of these sister chromatids as basically just being exact copies of the same cookbook. All right. So now that we've got all that straight, let's go ahead and introduce some new terminology. Okay, so we keep seeing this representation of a chromosome, which is all fine and well, but why are we depicting it this way? It turns out a chromosome can be divided into basically 3 different parts: the centromere, arm, and the telomere. Now, notice that the chromosome is often depicted in an X-shaped structure with a constriction in the middle. This constriction point is called the centromere. And the centromere is a physical location along the chromosome where sister chromatids are held together throughout most of cell division. And as you can see in most of the diagrams that we've used thus far, the centromere is often depicted as a circle. Also, note that the location of the centromere can vary. The centromere may be located in the middle of the chromosome, or it could be located closer to one of the ends. That makes the location of the centromere one of the traits that scientists can examine when trying to distinguish the karyotype of an organism. As we'll see later, the centromere also plays a central role in cell division. So the centromere effectively divides the linear DNA molecule into two parts. Now, each segment above and below the centromere is called a chromosome arm. Now, note that the length of the chromosome arm above and below the centromere varies from chromosome to chromosome. So this is also a physical characteristic which can be identified in karyotyping. The tips of the chromosomes are known as telomeres. Now, the ends of DNA are highly susceptible to degradation. And to combat this obvious problem, a special composition and arrangement of DNA at the ends of the chromosomes limits the ability of chromosomes to react with other molecules. Now, this special sequence of DNA is known as a telomere. In summary, a chromosome is a single molecule of DNA, which represents a specific subset of the information in the genome. A diploid organism has two copies of each chromosome, which are called homologous chromosomes or homologs. A chromatid refers to the copies of chromosomes produced during DNA replication. During mitosis, a cell could possess at most twice as many chromatids as chromosomes in its genome. A sister chromatid specifically refers to one of the two copies of a single chromosome. During mitosis, at any given time, there can be at most two sister chromatids per chromosome. The centromere is a physical location along the chromosome where sister chromatids are held together throughout most of cell division. The chromosomal arm is the portion of a chromosome found on either side of the centromere. And the telomere is the portion of the chromosome tip which protects the chromosomal DNA from degradation. So we know that if we're going to accurately segregate or separate the chromosomes during mitosis, we're going to need to condense the chromosomes because a small compact object is going to be easier to move than a long, floppy, kind of stringy one. But now we need a way to move those objects around in the cell. Alignment of the chromosomes is an important step in mitosis, and it's accomplished by microtubules, which, as we learned earlier, is a component of the cytoskeleton. Now, microtubules organize into what is known as the mitotic spindle. But before we can understand how the mitotic spindle works, we need to know a little bit more about the components that make it up. Microtubules are the major component of the mitotic spindle. As far as mitosis is concerned, you can think of them as molecular rope. During mitosis, they're used to position the chromosomes at a specific position in the cell. Now, if we attach microtubules to opposite sides of each chromosome, we can basically increase or decrease the tension on each side to position the chromosomes in the middle of the cell. Let's see how the cell accomplishes this. So first off, if we're going to be using ropes to tether the chromosomes in the middle of the cell, we should probably anchor at least one end of the rope to something that's not going to move. And the centrosome can fulfill this function. The centrosome is an organelle that serves as a microtubule organizing center during cell division. It's duplicated during S phase, and the two copies move to opposite sides of the cell. Now, once the mitotic spindle has been assembled, the centrosomes are also referred to as spindle poles. The centrosomes initiate the assembly of several types of microtubules, but let's just consider two of them. The first type of microtubule is short, and it basically radiates out from the center of the centrosomes in a star-shaped pattern. So we call them astral microtubules. Now, astral microtubules anchor the centrosomes in the cell, and in the process, they position the mitotic spindle apparatus. You can kind of think of them like tent spikes that are going to basically just stabilize the entire spindle apparatus. The second type of microtubule is called a kinetochore microtubule. Obviously, to understand this microtubule type, we'll need to figure out what a kinetochore is first. The kinetochore is a protein structure which assembles in the centromere region of each chromosome during mitosis. A kinetochore assembles on opposite sides of each chromosome, or essentially on the outermost side of each of the sister chromatids. Probably not surprisingly, the kinetochore microtubules attach to the kinetochore. Okay, so now that we've established all of the players in the mitotic spindle, we can talk about how all these things are going to help the chromosomes move around in the cell. The astral and kinetochore microtubules are two of the principal components of the mitotic spindle. And the mitotic spindle apparatus provides the means of positioning the chromosomes during mitosis. So why is chromosome movement so important to mitosis? The goal of mitosis is to move the chromosomal copies, or chromatids, to opposite sides of the cell. This ensures that each daughter cell gets a full set of chromosomes. Now, there's so much information stored on a chromosome that loss of even a single chromosome is a catastrophic event that can lead to disease for the organism or death for the cell. It'd be most efficient if we divvied up the chromatids all at the same time. By positioning them into the middle of the cell in the same orientation would make that possible. The only question is, how are we going to get them there? Well, recall that we said that each chromosome is bound by microtubules on either side, and that the microtubules are sort of like molecular rope, which can exert tension on each side of the chromosome. So what that means is that by increasing tension on, say, the right side of the chromosome, while we simultaneously decrease tension on the left side, we could move the chromosome toward the right. But how are we going to control the tension? Well, the microtubules already nailed down at the centrosome side, so the kinetochore proteins are pretty much in the driver's seat. Let's think of the situation as a guy with a winch sitting on either side of the chromosome. The kinetochore on the right can decrease the length of the microtubule on its side, while the kinetochore on the left simultaneously increases the microtubule on its side. In this way, the chromosomes can move back and forth till they reach the center of the cell. Oh, one more thing before we go. Has there been something nagging you about this mitotic spindle? Like, why the heck it's called a spindle? Well, if you look at the shape of the kinetochore or spindle microtubules, they actually form the shape of a spindle. You know, like the ones that are used in the textile industry to form wool or other fibers into thread. In summary, the mitotic spindle is a structure composed of microtubules, which segregates chromosomes into daughter cells during mitosis. A microtubule is a rope-like component of the cytoskeleton. The centrosome is an organelle that serves as a microtubule organizing center during cell division. The spindle pole is the region of the cell where the centrosome is located and toward which the chromosomes will move. An astral microtubule is a short microtubule which emanates from the centrosome and serves to accurately position the mitotic spindle in the cell. A kinetochore microtubule is a microtubule which attaches to the kinetochore and positions the chromosomes during mitosis. Here's one more way to remember the mitotic spindle. Condensation makes DNA ready. Microtubules hold it steady. A microtubule is a fancy rope. To move the DNA's the hope. From spindle poles they do extend to align each chromosome in the end. So we've been talking about a lot of different strategies, how to make it easier for a cell to segregate or separate its chromosomes into two daughter cells during mitosis. So we know before mitosis, the chromosomes condense into compact structures that are going to be easier to maneuver. The chromosome movement is orchestrated by microtubules, which organize into a structure known as the mitotic spindle apparatus. And this complex structure is held together by the centrosomes at the spindle poles and the kinetochores at the chromosomes. Now that we have a pretty good idea of how these tricks work individually, let's see how they all fit together in mitosis. Now, although mitosis, like cell division, is a continuous process, it's really just easier for us to understand if we divide the process into several steps. Now, mitosis is typically divided into 5 phases, known as prophase, prometaphase, metaphase, anaphase, and telophase. Let's see what happens to the chromosomes of a simple 2-chromosome organism in each phase of mitosis. If this is a diploid organism, recall that means that the organism has 2 copies of each chromosome, known as homologs, giving the organism a total of 4 chromosomes in each cell. Actually, before we start talking about mitosis, let's briefly review the relevant steps that occurred during interphase. Now, during interphase, DNA replication occurs. And after DNA replication is complete, each chromosome is going to be composed of 2 sister chromatids. However, during interphase, the chromosomes are still loosely packed and uncondensed. The centrosomes, which will serve as microtubule organizing centers during mitosis, also duplicate during interphase. Prophase is the first step in the mitotic process. The prefix pro in Greek means before. During prophase, the chromosomes condense. The centrosomes begin to form spindle and move into position on opposite sides of the cell. Sister chromatids are held together by a protein called cohesin at the centromere. Prometaphase is the second step in mitosis. Prometaphase literally means before the middle stage in Greek. During prometaphase, the nuclear membrane breaks down. Chromosomes are beginning to attach to the mitotic spindle. Remember the guy with the winch at each of the kinetochores? Well, they allow the chromosomes to adjust the length of the kinetochore fibers. By adjusting the length of kinetochore fibers, the chromosomes begin to orient in the middle of the cell at the metaphase plate. The metaphase plate is an imaginary line in the middle of the cell that is halfway between the spindle poles. It is also sometimes referred to as the equatorial plate. You can think of the balanced tension that's holding the chromosomes at the metaphase plate kind of like 2 equally strong guys that are in a tug of war. Now, since the pull on each side is relatively the same, the flag in the middle isn't really going to move. And this is basically what the case is with the chromosomes. Since the tension on each side of the chromosome is essentially the same, the chromosomes are held in place at the metaphase plate. Metaphase is the third step in mitosis. Metaphase means middle stage in Greek. By metaphase, all of the chromosomes are aligned on the metaphase plate. Since chromosomes are maximally condensed during this phase, scientists often study the karyotype of metaphase chromosomes. Recall that we were thinking about metaphase chromosomes as being held in place by equal tension exerted by the kinetochore microtubules. The only thing preventing each chromatid from shooting towards its respective spindle pole is the protein cohesin, which is holding the chromatid centromeres together. Anaphase is the fourth step in mitosis. Now, once all of the chromosomes are at the metaphase plate, we're ready to begin segregating the chromosomes. A protein called separase cleaves the cohesin protein that's been holding the centromeres together. You can think of separase as kind of like a pair of molecular scissors that cleaves the cohesin protein. And with the physical connection between the chromatids gone, the chromatids are free to move towards each pole. Separation of the sister chromatids marks the beginning of anaphase. Anaphase ends when the chromosomes reach the spindle poles. This marks the beginning of telophase, the fifth and final step in mitosis. The prefix telo means end in Greek. During telophase, the spindle breaks down. The nuclear membrane reforms around the chromosomes, establishing 2 nuclei. Chromosomes begin to uncoil back to the level of packaging found in chromatin during interphase. The end of telophase also marks the end of mitosis. To summarize, during interphase, DNA replication occurs and the centrosomes duplicate. Prophase is the first step in mitosis. During prophase, the chromosomes condense, the centrosomes move to opposite sides of the cell, and the mitotic spindle begins to assemble. Prometaphase is the second step in mitosis. During prometaphase, the nuclear envelope breaks down, and the chromosomes attach to the mitotic spindle and begin to orient toward the metaphase plate. The metaphase plate is an imaginary line in the middle of the cell, halfway between the spindle poles, where the chromosomes align during mitosis. The metaphase plate is also sometimes referred to as the equatorial plate. Metaphase is the third step in mitosis. At metaphase, all of the chromosomes are aligned at the metaphase plate. Anaphase is the fourth step in mitosis. During anaphase, the physical connection between sister chromatids is severed, allowing the chromatids to move toward their respective spindle pole. Telophase is the fifth and final step in mitosis. During telophase, the mitotic spindle breaks down. The chromosomes uncoil to the diffuse interphase state, and the nuclear membrane reforms. Oh boy, is this still all Greek to you? Well, here's another little mitosis tune for your studying pleasure. Prophase is step number 1. In step 2, alignment's just begun. Attach, align, where is the middle? Mitotic spindle solves the riddle. Anaphase, the sisters sever. Telophase ends this endeavor. So if we're talking about cell division, at some point the cell, well, should actually divide. But so far, all mitosis has done is produce a cell with 2 nuclei. Cytokinesis is the process in which the cytoplasmic content of the cell is divided into 2 new daughter cells. Now, while it's easiest to consider the events of cytokinesis separately from mitosis, in actuality, there is some overlap between the two. Lucky for us, the mechanisms that govern cytokinesis are far less complicated than those of mitosis. Mitosis was all about ending up with a precise number of chromosomes. On the other hand, division of the rest of the contents of the cell is far less precise. Having an unequal number of chromosomes in the resulting daughter cells would pretty much be a bad thing. But having an unequal number of, say, ribosomes or mitochondria isn't. It's because the total number of organelles, proteins, and other molecules floating in the cytoplasm is so big that even an arbitrary division of the cytoplasmic content gives each daughter cell a sufficient amount of functionality to survive. So for instance, if a dividing human cell had, say, 1,000 mitochondria, a division of 450 in one cell and 550 in another cell still means that both of the cells can produce enough energy, even though one daughter cell has 10% less energy producing capacity than its mother had. So how do we turn one cell into 2 cells? Well, it turns out the strategy in animal cells is different compared to what plant cells do. Let's see what strategy each type of cell employs. As we've seen before, animal cells are kind of like water balloons, and the cytokinesis strategy employed by animal cells is reminiscent of this fact. As mitosis is ending, cytokinesis begins to divide the forming nuclei and the rest of the contents of the cell into 2 new daughter cells. This basically looks like you looped a string around the outside of a water balloon and started to constrict it like a drawstring. Now, obviously, the real-life outcome of this example doesn't really end too well for us since we end up with a face full of water. Luckily for the cell, though, the outcome is much less messy. During cytokinesis, the role of the string is played by the contractile ring. Now, a contractile ring is composed of actin filaments that are assembled just below the cell membrane. A protein called myosin works with the actin filaments to constrict the ring till the cell membrane touches in the middle. Now, once those membranes pinch together, they fuse, and the result is 2 new cells, each with 1 nucleus, complements of mitosis, and half of the cytoplasmic content, complements of cytokinesis. So why do plants utilize a different cytokinetic strategy? Well, it's not just because they're trying to be different. Remember that plants have a cell wall. Now, since that cell wall won't bend, a contractile ring isn't going to work. But nature's nothing if not creative. Remember that the Golgi apparatus is an organelle that plays a major role in secretion of molecules from the cell. Now, it accomplishes this by releasing tiny membrane sacs called vesicles. And a vesicle containing a molecule destined for secretion buds off of the Golgi apparatus and heads for the cell membrane. Since it's made of phospholipids, just like the cell membrane, a vesicle can fuse with the membrane. And in this way, the molecule is released. Now, this reveals a strategy that the plant cell can employ to form a new cell membrane without a contractile ring. In a plant cell, a new membrane is formed by vesicles, which fuse together in the middle of the cell to form what's called a cell plate. Now, as the cell plate expands to the edges of the cell, it can fuse with the cell membrane and form two new cells in the process. Okay, so we've solved one problem, right? But we've just created another. Now we have two cells, but they're stuck in the same cell wall enclosure. Well, remember how we said that the Golgi apparatus is good for transporting molecules outside the cell? Well, what if we pack some cell wall ingredients in the vesicles that we use to form the cell plate? So if we do that, once the cell membrane is formed, these secreted molecules can assemble into a new cell wall, and in this way, the two plant daughter cells can separate. In summary, cytokinesis is the process in which the cytoplasmic content of the cell is divided into two new daughter cells. The mechanism in which cytokinesis is achieved differs between animal and plant cells. A contractile ring is a ring of actin filaments located below the cell membrane, which constricts the middle of a dividing animal cell to complete cell division. The cell plate is the area where the new cell membrane and cell wall form between dividing plant cells during cytokinesis. Mitosis gives organisms the ability to create new cells. In the case of many organisms, like us, it provides the opportunity to become multicellular. However, for other organisms, like, say, yeast, mitosis is a means of reproduction. Now, if an organism uses mitosis as a means of reproduction, it is said to be reproducing asexually. Asexual reproduction is also known as vegetative reproduction. As we've seen, mitosis is designed to faithfully create two daughter cells with genomes that are identical to the mother cell. You can say that each new cell is a clone of the original cell. Asexual reproduction is an effective strategy for many organisms. It is highly efficient because a mate is not required. And the cost to the parent in time and energy is low because there's no progeny to rear. However, this strategy offers little, if any, opportunity for variation in the genetic makeup of asexually reproducing organisms. And since all of the individuals in a given population of asexually reproducing organisms are clones, they share the same weaknesses. Therefore, the entire population could perish if a major environmental change occurred. An alternative strategy known as sexual reproduction offers an opportunity to introduce genetic variation into the population. During sexual reproduction, offspring are produced from the genetic material of two different individuals, the offspring's mother and father. Each parent contributes half of the genetic material the offspring requires in the form of a gamete cell. Recall that a gamete is a specialized sex cell produced by each parent for sexual reproduction. The female provides an egg, which is fertilized by a male sperm cell. During fertilization, the haploid male and female gametes fuse to form a single diploid cell, which can then go on to produce the rest of the cells in a multicellular organism by mitosis. However, where do all these gametes come from? Well, mitosis won't do us any good if we're starting out with a diploid cell, because mitosis will only allow us to make more diploid cells. And the gametes have to be haploid, because if we fuse diploid cells together, we'd end up with a cell with too many chromosomes. To solve this problem, sexually reproducing organisms have evolved a second specialized type of cell division known as meiosis. The purpose of meiosis is to produce cells with half of the normal genetic content found in the organism. Because meiosis and mitosis are both types of cell division, they share many similar structures and strategies. For instance, DNA replication precedes both types of division. Chromosome condensation and a spindle apparatus are key strategies for maneuvering chromosomes in each case. However, unlike mitosis, which makes two cells that are exact copies, the primary goal of meiosis is to reduce the number of chromosomes in the cell by half. If a diploid organism has two copies of each chromosome, it got one from mom and one from dad. That means each parent had to put only one copy of each chromosome into its gamete. For instance, mom donates one of her copies of chromosome 1, and so does dad. The same is true for chromosome 2, 3, 4, and so on. But this raises another question. We currently have no way of identifying homologs and ensuring that each cell gets one copy. How can an organism achieve this goal? As we will see, nature's answer to this question not only makes meiosis possible, but also will have significant consequences for the inheritance of genetic information. In summary, asexual reproduction is a form of reproduction from a single parent based on mitosis. Sexual reproduction is a form of reproduction in which offspring are produced from the union of gametes from two genetically different parents. A gamete is a specialized sex cell produced by each parent for sexual reproduction. Meiosis is a specialized type of cell division which produces gamete cells for sexual reproduction. So now we know meiosis is the process that is going to produce gametes for sexual reproduction. Each gamete possesses a single copy of each chromosome. And that means that when two gametes fuse, they produce a cell with two copies of each chromosome. The question we need to answer now is, how is the organism going to make sure that each gamete gets exactly one copy of each homolog? Let's re-examine mitosis. How the cell dealt with chromosomes during mitosis might give us some clues about how it will deal with them in meiosis. Let's consider an example where we have a diploid organism with two chromosomes. And let's make it easier on ourselves to follow these homologs and chromosomes by using different colors. We'll make one chromosome purple, and the other one, let's make it orange. Now, note that each homologous chromosome is a similar shade of the same color, signifying that they are similar but probably not identical versions of the same genes. DNA replication prior to mitosis produced two copies of each chromosome stuck together at the centromere. Since the chromatids are exact copies, they're the same color. The mitotic spindle lines up each chromosome in the middle of the cell. By positioning each homolog in the middle of the cell, division can ensure that each daughter cell receives one copy of each homolog in the form of a chromatid. So in our example, each daughter cell needs to receive 4 chromatids by the end of mitosis, each of which is represented by the 4 different colors. Now in contrast, the goal of meiosis is to reduce the genome by half. Rather than getting one copy of each distinct color, during meiosis, each daughter cell only needs one copy of each basic color. In our example, that means that each gamete should possess one copy of a purple chromosome and one copy of an orange chromosome. And it doesn't really matter if that copy is the dark or the light version. For this to happen, the cell needs a way of linking homologous chromosomes in a physical structure, similar to the way the chromatids are held together at the centromere during mitosis. This physical connection occurs as a result of homologous recombination. Homologous recombination is also referred to as crossing over, because the physical structure that results from recombination kind of looks like a cross. Homologous recombination, or crossing over, is a process in which genetic material is exchanged between homologous chromosomes. Physically linking homologous chromosomes is a similar strategy to the one used in mitosis, with the crossover structure playing a similar role to the centromere. By physically holding the homologs together during meiosis, The homologous chromosomes act like one physical structure. The cell can then use spindle microtubules to orient the homologs until every homologous pair is ready for division. Once each homolog pair is oriented properly at the metaphase plate, the physical connection can be broken and the homologs can segregate to opposite spindle poles. It's important to note that the description of meiotic recombination provided here is a very simplified version of the actual events and participating proteins. One happy consequence of meiotic recombination is that genetic information is exchanged between homologs. Notice that when the crossover event is resolved, pieces of each homolog are exchanged, producing chromatids composed of more than one color. This exchange of genetic information increases genetic diversity. We'll explore the implications and consequences of this genetic diversity in greater detail later. So now that we've figured out how homologs can be identified and segregated, we can examine the overall process of meiosis in more detail. Meiosis actually consists of two separate divisions. The first division, meiosis 1, is preceded by DNA replication, just like mitosis was. And meiosis 1 is sometimes referred to as a reductional division because this is the division that's going to reduce the DNA content of the cell by half. In meiosis 1, the homologs segregate to opposite poles. In our example, the diploid genome is reduced to a haploid genome during this division. Now note that replication occurred before meiosis began, so each chromosome in the daughter cells is made up of two identical sister chromatids that are linked at the centromere. Meiosis 2 follows meiosis 1 and completes the cell division. It's sometimes referred to as an equational division because each daughter cell ends up with the same number of chromatids as the mother cell started with. And it's very similar to mitosis because during this division, each homolog lines up at the metaphase plate. In this case, two chromosomes composed of two chromatids each line up in the center of the cell, and the chromatids which make up the homolog segregate to opposite poles. In summary, meiosis is the specialized type of cell division which produces gamete cells for sexual reproduction. Homologous recombination is a process in which genetic material is exchanged between homologous chromosomes. Homologous recombination is also known as crossing over. Meiosis 1 is the first of the two divisions which occur in meiosis, during which homologous chromosomes are separated. Meiosis 2 is the second of the two divisions which occur in meiosis, during which sister chromatids are separated. We've learned thus far that meiosis is a special type of cell division that makes gamete cells for sexual reproduction. The most important distinction between meiosis and mitosis is that the daughter cell chromosome number must be reduced. To put meiosis into everyday terms, let's consider the chromosomes as cookbooks again for a minute. If mitosis was like some crazy person making copies of every one of his cookbooks, meiosis is like two roommates trying to split a cookbook collection. Say you and I have amassed a bunch of cookbooks, but now we're going our separate ways. Luckily, we have two editions of each cookbook, so at least we can both get a version of the recipes in those books. Now, if we have a lot of cookbooks, and those cookbooks are kind of poorly organized, it'll be easiest to identify both editions of each cookbook before tossing any cookbooks into our piles. This will ensure that neither of us get two editions of the same cookbook, or no version at all. This is basically the problem facing a cell undergoing meiosis. Two cell divisions occur during meiosis, and the events of the first meiotic division, meiosis 1, address the problem of identifying and separating pairs of homologous chromosomes. Meiosis 1 is sometimes referred to as a reductional division because it reduces the number of chromosomes in the cell by half. The meiotic I cell division consists of 4 basic steps: prophase I, metaphase I, anaphase I, and telophase I. Although many components used during mitosis are also utilized in meiosis I, as we trace the events of meiosis I, note the significant modifications that are necessary to achieve a reductional division. Just like mitosis, the first meiotic division is preceded by interphase. During interphase, the chromosomes replicate, meaning that each chromosome is composed of 2 chromatids prior to meiosis I. During the cell cycle step, the centrosomes also duplicate. Prophase I is the first step in meiosis I, and a lot happens during this step, some of which is a departure from what we saw in mitosis. During prophase I, the chromatin condenses. The centrosomes move to opposite poles and begin to produce spindle fibers. Homologous chromosomes search for their partner homolog in a process known as pairing. Crossing over occurs between homologous chromosomes. Note that genetic information is exchanged between homologous chromosomes as a result of a crossover event. More importantly, the crossover event also serves to hold the homologs together so that they can be properly oriented on the meiotic spindle. When the homologs become physically linked together, the entire structure is referred to as a tetrad. And this makes sense because tetra means 4 in Greek, and there are 4 chromatids in a tetrad. Also note that the name crossing over stems from the fact that the arms of the homologs appear to cross over each other in forming this structure. Kinetochore proteins assemble on the outermost chromatid that faces each spindle pole. Meiotic spindle attaches to the kinetochore, and as in mitosis, our little kinetochore friend is busy working those winches on each side of the tetrad. By adjusting the length of microtubules on each side, the kinetochore positions the tetrad in the middle of the cell. By the end of prophase I, the nuclear membrane has broken down and the meiotic spindle apparatus has been fully assembled. Metaphase I is the second step in meiosis I. By metaphase I, the tetrads are aligned at the metaphase plate. Note that the physical linkage between the homologous chromosomes and the tension produced by the meiotic spindle keep the tetrads positioned in the middle of the cell. Anaphase I is the third step in meiosis I. It begins with the release of the physical connection between homologs. No longer physically linked, spindle pole tension can pull the homologs to their respective spindle pole. Telophase I is the fourth and final step of meiosis I. During telophase I, the nuclear membrane reforms and the chromosomes decondense in most organisms. Telophase I is accompanied by division of the mother cell into 2 daughter cells. This marks the end of meiosis I. To review, meiosis is a special type of cell division that makes gamete cells for sexual reproduction. Meiosis I is the first of 2 meiotic divisions during which the number of chromosomes in the cell is reduced by half. Prophase I is the first step in meiosis I. During prophase I, chromatin condenses, centrosomes move to opposite poles, and a spindle apparatus forms, as in mitosis. Unlike mitosis, crossing over physically links the homologous chromosomes, and the resulting tetrad, rather than the individual chromosomes, is aligned at the metaphase plate. A tetrad is the structure formed when 2 homologous chromosomes become linked during a crossover event. Metaphase I is the second step in meiosis I. At metaphase I, the tetrads are aligned at the metaphase plate. Anaphase 1 is the third step in meiosis 1. During anaphase 1, the physical connection between homologs is broken, allowing spindle pole tension to pull each homolog to its respective pole. Telophase 1 is the fourth and final step of meiosis 1. During telophase 1 of most organisms, the nuclear membrane reforms and the chromosomes decondense. This is then followed by cytokinesis. Finally, here's a lyrical reminder of the major ideas of this lesson. How to achieve sex reproduction? The first division is reduction. Crossing over links DNA, so homologs no more may stray. Cut the tetrad to let them loose, and so the genome does reduce. To successfully complete meiosis and produce a gamete, a cell must undergo 2 rounds of division. The first round, conveniently known as meiosis 1, reduces the number of chromosomes in the cell by half. The mechanisms the cell uses to accomplish this reductional division differs greatly from mitosis. In contrast, meiosis 2 is very similar to mitosis, since the goal is to segregate a chromatid of each chromosome into separate daughter cells. For this reason, meiosis 2 is sometimes called an equational division. Like mitosis, the number of chromosomes in each daughter cell is unchanged compared to the mother cell. Let's examine the similarities and differences between meiosis 2 and mitosis a little more closely. Recall that meiosis 1 produced 2 cells, which will behave exactly the same during meiosis 2. Since both cells are going to undergo the same steps, let's just consider the meiosis 2 division of one of these cells, and then we can consider all of the products together at the end. So this cell is going to undergo interphase, prophase 2, metaphase 2, anaphase 2, and finally telophase 2. Meiosis 2 is preceded by a brief interphase. Now, it's a special interphase because no DNA replication occurs in this case. Recall that meiosis 1 left each cell with only one set of homologous chromosomes. But each homologous chromosome consists of 2 chromatids. Like mitosis, the centrosome duplicates during interphase. Prophase 2 is the first step in meiosis 2. Like mitosis and meiosis 1, the chromosomes condense throughout this step. The centrosomes move to opposite poles and begin to form spindle fibers. Kinetochore proteins assemble on the outer chromatids of each chromosome, just as they do in mitosis. Meiotic spindle attaches to the kinetochores. And as in mitosis, our little kinetochore friend is busy working on those winches on either side of the chromosome. By adjusting the length of the microtubules on each side, the kinetochore positions each chromosome in the middle of the cell. And by the end of prophase 2, the nuclear membrane has broken down and the meiotic spindle apparatus has assembled. Metaphase 2 is the second step in meiosis 2. At metaphase 2, each chromosome is aligned at the metaphase plate. As in mitosis, each chromosome is held in place at the metaphase plate by equivalent spindle tension on each side. Anaphase 2 is the third step in meiosis 2. Like mitosis, anaphase 2 is initiated when the protein separase cleaves the protein holding the chromatids together. Once this physical connection is broken, each chromatid is free to move to its respective pole. Telophase 2 is the fourth and final step of meiosis 2. During telophase 2, the nuclear membrane reforms and the chromosomes decondense. Cytokinesis completes the formation of 4 haploid gamete cells. Note that the chromosomal content of each gamete is slightly different due to homologous recombination and the manner in which the chromosomes sort into the gamete cells. We will see later how cytokinesis can be modified to produce even more specialized gametes. In summary, meiosis I is the first division of meiosis in which the number of chromosomes in the cell is reduced by half. Meiosis II is the second division in meiosis in which chromatids of each chromosome are segregated equally into daughter cells. No DNA replication occurs during the interphase which precedes meiosis II. Prophase II is the first step in meiosis II. Like mitosis and meiosis I, chromatin condenses, centrosomes move to opposite poles, and a spindle apparatus forms. Kinetochores assembled at the centromeres work with spindle microtubules to guide the chromosomes to the metaphase plate. Metaphase II is the second step in meiosis II. By metaphase II, each chromosome is aligned at the metaphase plate. Anaphase II is the third step in meiosis II. During anaphase II, the physical connection between sister chromatids is broken, allowing them to move toward the spindle poles. Telophase II is the fourth and final step of meiosis II. During telophase II, the nuclear membrane reforms and the chromosomes decondense. The cytokinesis which follows meiosis II establishes 4 gametes with different genetic content. As we draw to a close on meiosis and cell division, I'll leave you with one last poetic study aid. Meiosis is but halfway started, but chromatids now must be parted. No replication precedes this act, yet genome size remains intact. As division number 2 completes, meiosis leaves haploid gametes. So we've seen how meiosis is an important biological process. Without it, gametes wouldn't be possible, and sexually reproducing organisms, well, they wouldn't be able to reproduce. So proper segregation of chromosomes during meiosis is vital to propagating the species and ensuring healthy, viable offspring. But cell division is a complex process, and nature isn't perfect. So what happens when mistakes are made? Recall that normally in meiosis, homologous chromosomes pair and align at the metaphase plate and segregate to opposite poles during meiosis I. When the cells produced by meiosis I undergo meiosis II, each chromosome aligns at the metaphase plate and sister chromatids are segregated into separate gametes. The homologous chromosomes are said to disjoin or separate at anaphase I, and the sister chromatids are said to disjoin at anaphase II. Thus, failure to separate the linked homologs or chromatids during anaphase is called non-disjunction. Let's examine the effect non-disjunction of human chromosome 21 at meiosis I would have on the resulting gametes. To make it easier, let's just consider what happens to chromosome 21. However, keep in mind throughout this example that the rest of the chromosomes are behaving normally. If the homologs in the chromosome 21 tetrad fail to separate during meiosis I, one daughter cell would receive both chromosome 21 homologs and the other would receive none. If those cells proceed to meiosis II, both homologous chromosomes would line up at the metaphase plate in cell number 1 and segregate into the daughter cells. The result would be gametes with 2 rather than 1 copy of chromosome 21. Now let's consider what happens to the second daughter cell produced by meiosis I. It received no copies of chromosome 21 during the meiosis I division. That means that the gametes produced by this cell in meiosis II will have no copies of chromosome 21. Now let's consider what would happen if these abnormal gametes fuse with a gamete with the proper number of chromosomes? First, let's consider an egg with 2 copies of chromosome 21 fusing with a sperm with 1 copy of chromosome 21. The result of this union will be a cell with 3 copies of chromosome 21. Recall again that we have only been considering chromosome 21 so far. Chromosome non-disjunction is generally rare, so the new cell probably has 2 copies of all of the other 22 chromosomes, just as it should. A cell which has an abnormal number of chromosomes is called aneuploid. More specifically, a human who develops from this cell will have a normal diploid set of every chromosome except 3 copies of chromosome 21. An otherwise diploid cell, which has 3 copies of a chromosome, is called trisomic. Before we consider the phenotypic consequences of this abnormal ploidy, let's consider the outcome of the other type of aberrant gamete. If an egg with no copies of chromosome 21 fuses with a sperm with 1 copy of chromosome 21, the resulting cell would have a normal complement of chromosomes with the exception of only 1 copy of chromosome 21. This cell would also be considered aneuploid. In this case, this cell would be called monosomic because it only has 1 copy of chromosome 21. The importance of faithfully segregating chromosomes during meiosis becomes more apparent when the consequences of aneuploidy are considered. The first example of aneuploidy we identified was trisomy of chromosome 21. Trisomy of chromosome 21 causes the human chromosomal disorder known as Down syndrome. Roughly 1 in 700 babies born in the United States has Down syndrome. A few of the many conditions they can be afflicted with are skeletal abnormalities and heart defects. However, considerable phenotypic variation is observed among individuals with Down syndrome, with many individuals able to lead relatively long, productive lives. Down syndrome is one of the few examples of chromosomal aneuploidy which is viable. For the most part, any form of aneuploidy is a lethal event, resulting in a miscarriage. Roughly half of miscarriages are initiated due to chromosomal abnormalities. For instance, consider the other type of aneuploid cell we discussed earlier. A cell which is monosomic for chromosome 21 is inviable and would initiate a miscarriage. In summary, non-disjunction is the failure of linked homologs or chromatids to separate during anaphase of mitosis or meiosis. Aneuploidy is a state in which a cell has an abnormal number of chromosomes. In a diploid organism, the most common forms of aneuploidy are monosomy and trisomy. Monosomy is a situation in which an otherwise diploid cell has only 1 copy of a particular chromosome. Trisomy is a situation in which an otherwise diploid cell has 3 copies of a particular chromosome.
A bacterium is a single-celled, unicellular microorganism that does not have a nucleus or any other membrane-bound organelles. Bacteria are sometimes called prokaryotes. In Greek, prokaryote literally means before the nut, where the nut is the nucleus. Bacteria adapt to become well suited to their environments, and therefore come in many shapes and forms. However, they all have a few parts in common. 1. Capsule, a protective, often slimy coating, often of sugars, that helps to protect the bacterium. It also makes bacteria virulent. This means the bacteria is more likely to cause disease, since it aids the cell in survival against attack. For example, the bacteria may survive an attack from the human body's immune system. 2. Cell wall. In bacteria, the cell wall is usually made of peptidoglycan, a protein and sugar compound. This structure gives the cell some rigidity and protection. 3. Cell membrane. As in most cells, the bacterium's plasma membrane acts by coordinating the passage of molecules into and out of the cell. 4. Cytoplasm. Again, as in many cells, the cytoplasm serves as a medium through which molecules are transported, as well as a system to maintain conditions like temperature and pH that are best for the cell. 5. Ribosomes, the main site for the bacterium's protein synthesis. 6. Nucleosome, a basic unit of chromatin which won't be covered in this lesson. 7. Nucleoid. This is the region where the bacterium's DNA is located. Again, it's not the same as a nucleus because it's not surrounded by a membrane. 8. Flagellum. In many bacteria, a flagellum is present and is the means by which the cell moves around. Because bacteria are so diverse in both form and habitat, biologists have struggled with their classification, also called their taxonomy. For many years, bacteria were called monirans and placed in a kingdom of the same name. Although you still might hear bacteria referred to as monirans, it's not a term currently accepted among biologists. Life is now classified into 3 domains. A domain is a taxonomic level that is higher than a kingdom and based on an organism's DNA. Bacteria is one of these domains, while the others are eukarya, composed of organisms that have nucleated cells, and archaea, composed of unicellular prokaryotes, many of which have evolved to exploit extreme environments. Organisms in the archaea domain were originally considered a type of bacteria, known as archaebacteria, but scientists have since reclassified them. In this image, groups of archaea are indicated by green lines, and bacteria are in blue. Red lines indicate eukaryotes. Scientists try to make sense of the sheer number and diversity of bacteria by grouping them in various ways. Here are just a few ways that are accepted in the scientific and medical communities. Shape. Bacteria are usually one of 3 shapes. Cocci are round. Bacilli are rod-shaped. Spirilla, also called spirochetes, are spiral. Gram stain. One of the more interesting ways that scientists group bacteria is how they react to gram staining. In this process, bacteria are subjected to a chemical stain. The color a bacteria stains depends on the amount of peptidoglycan in the cell wall. In gram-positive cells, the peptidoglycan layer is thick, and the cells are purple. In gram-negative cells, the layer is thinner, and the cells are pink. Antibiotic sensitivity. Peptidoglycan also plays a role in how effective certain kinds of medications are at helping the body eliminate bacterial infections. Some antibiotics work by preventing the peptidoglycan from forming properly, allowing the body's immune system to work more efficiently at wiping out the infection. Oxygen requirement. Some bacteria, aerobes, require oxygen to carry out their day-to-day metabolic processes. Others, like the ones that live in the human digestive tract, do not. These organisms are called anaerobes. Nutrition. Some bacteria are autotrophs, meaning they can make their own food. Others are heterotrophs that require an outside source of nutrition. Again, these are just a few of the ways that we group bacteria. Other ways include their habitat, if they cause disease or are harmless, and even if they are helpful to humans or not. Let's review. Bacteria are single-celled organisms that do not have a nucleus or other membrane-bound organelles. Bacteria are ubiquitous, that is, they are found in every environment on Earth and are extremely diverse. They have historically been classified using different characteristics, but most biologists today agree that, based on their DNA, they belong to their own domain. We then can further group them in other ways, such as whether they cause disease or not, or how they get their nutrition.
All living things can be classified into a place on the tree of life. This phylogenetic tree has 3 major branches called archaea, bacteria, and eukarya. A phylogenetic tree traces the evolutionary history of organisms and indicates common ancestors. We already see a major difference between archaea and bacteria from this classification. They have a different evolutionary history as they occupy very different places on the tree of life. How was this tree of life composed? The sequence of 16S ribosomal ribonucleic acid, 16S rRNA, a fundamental unit of ribosomes, was compared across organisms. This showed that the underlying genetic code for a component of ribosomes differed greatly between archaea, bacteria, and eukarya. Thus, they form three distinct branches of the tree of life. There was a time when archaea weren't understood to be different from bacteria and in fact were erroneously named archaebacteria, and the tree of life was drawn wrong. But they are definitely not bacteria, even though archaea and bacteria share some similarities. Archaea deserve their own branch on the tree of life. Archaea and bacteria are both prokaryotes, meaning they do not have a nucleus and lack membrane-bound organelles. They are tiny, single-cell organisms which cannot be seen by the naked human eye, called microbes. When we look at them through a microscope, we find that archaea and bacteria resemble each other in shape and size. They exist as rods, cones, plates, and coils. Both archaea and bacteria have flagella, thread-like structures that allow organisms to move by propelling them through their environment. An archaea might be very insulted if you mistook it for a bacteria, however, and vice versa. The differences between archaea and bacteria are profound. As mentioned, the genetic code of rRNA differs enough to place them in quite different branches of the tree of life, reflecting differing evolutionary paths. This still needs to be confirmed by sequencing the 16S rRNA of more organisms. Remember how both move via flagella? Despite this functional similarity and structural similarity, i.e. they look similar, they have very different genes encoding them and are comprised of different proteins. Genome sequencing of archaea also reveals genes that resemble eukaryotes more than bacteria. This is a big difference between archaea and bacteria. Another distinction between these two prokaryotes is the composition of the cell wall. For example, all bacteria contain peptidoglycans, a molecule composed of both protein and sugar rings, in their cell walls. However, archaea do not have this compound in their cell walls. Cell division in archaea undergoes distinct processes not found in bacteria. Also, bacteria can form spores that lie dormant for years until a proper habitat is found in which they can grow. Archaea haven't been found to do that. Archaea were first identified in extreme environments like hot deep sea vents or cold Antarctic oceans. For example, Sulfolobus lives in hot springs and Halococcus is found in pools with high salt content. For a time, archaea were thought to be extremophiles, organisms that are adapted to extreme environments, while bacteria were thought to favor moderate conditions. For example, bacteria like E. coli, Salmonella, and Lactobacillus can survive in moderate conditions in the human body, where they help or hinder human health. We now know that while lots of archaea do inhabit extreme environments, some bacteria also survive in extreme habitats, and some archaea can be found in more moderate conditions. It's not yet understood if archaea tend to inhabit extreme environments more than bacteria. This is currently being studied. Archaea, bacteria, and eukarya form three distinct branches of the tree of life, or the phylogenetic tree, which traces the evolutionary history of organisms and indicates common ancestors. Archaea used to be thought of as bacteria, though this is an erroneous classification. Archaea and bacteria are definitely both tiny, single-cell organisms which cannot be seen by the naked human eye, called microbes. They are also prokaryotic, meaning they do not have a nucleus and lack membrane-bound organelles, share similar sizes and shapes, and move by propulsion using their flagella, the thread-like structures that allow organisms to move by propelling them through their environment. They differ in fundamental ways, though, like through their genetic code, composition of cell wall, and flagella. Bacteria contain peptidoglycans, a molecule composed of both protein and sugar rings, and processes like cell division. Some bacteria can even form spores. In addition, archaea, like Halococcus and Sulfolobus, tend to inhabit more extreme environments like hot springs, freezing waters, and high salt pools, making them typically classified as extremophiles, which are organisms that are adapted to extreme environments. Bacteria, like E. coli, Salmonella, and Lactobacillus, tend to live in more moderate environments.
I remember getting sick as a kid and having to sit in the doctor's office. After what seemed like an hour, the doctor would look at me for 2 minutes, swab the back of my throat, and declare that I had strep throat. As a kid, all that meant to me was a few days off from school and a week of antibiotics. As a microbiologist, that short diagnosis actually tells me a lot about the bacteria that are making my throat scratchy and sore. Bacteria are single-celled, microscopic organisms. You might imagine that being so small, all bacterial cells should look about the same. Like humans, bacteria come in different shapes. The majority of bacterial cells come in 3 general shapes. The first shape is called coccus, plural cocci. Cells that have a cocci shape are spherical, resembling tiny balls. The strep in strep throat actually refers to the bacterium Streptococcus, which exists in tiny spherical cells. The second shape is bacillus, plural bacilli. These bacteria are shaped like small rods, longer than they are wide. A bacillus cell looks a lot like a pill. Have you ever heard of anthrax poisoning? Anthrax is caused by a rod-shaped bacteria called Bacillus anthracis. Some bacilli bacteria have round ends while others are square. It is important to note that the term bacillus can describe the cell shape as well as bacteria in the genus Bacillus. The third bacterial shape is spiral. These bacterial cells are twisted in helices and resemble little corkscrews. Not to gross you out, but if you scrape some gunk off your teeth and look at it under the microscope, you will find many spiral bacteria. Different species of cocci and bacilli also arrange themselves into groups, characteristic to the specific bacteria. A bacteria that lives as one cell, all alone, is called a single cell. Some bacteria remain grouped together after cell division. A bacteria that lives in pairs has a diplo arrangement, with di meaning 2. So Diplococcus is a cocci bacteria that is found in pairs. A group of 4 cells forming a flat square is called a tetrad. Micrococcus roseus can form tetrads and is a normal bacteria found on the skin of mammals. A Sarcina is a cube-like group of 8 cocci. Members of the genus Sarcina are harmless inhabitants of your large intestine that take this shape. If the bacteria form long chains, the prefix strepto is added. Going back to that pesky bacteria that causes strep throat, let's break down the genus Streptococcus. Strepto refers to long chains, and coccus refers to a spherical cell. So, the strep throat bacteria, Streptococcus, forms long chains of cocci. If the dividing bacteria form an irregular, grape-like cluster, the prefix staphylo is added. So, members of the Staphylococcus form clusters of cocci. An infamous member of this genus causes the dangerous infection known as MRSA, or methicillin-resistant Staphylococcus aureus. The spiral bacteria can take one of 3 forms. The first variation is called Vibrio. These bacteria are curved, resembling a comma. There is some debate as to whether this variation fits better with the spiral bacteria or the bacilli. It is not uncommon to see the genus Vibrio described as a curved rod. The disease cholera is caused by the bacterium Vibrio cholera. The next variation is the spirillum. These bacteria are shaped like a helix and have thick cell walls and flagella. Spirillum volutans is corkscrew-shaped with tufts of flagella on each end of the cell. These bacteria are found swimming around in the environment and in polluted waters. The last variation is the spirochete. These cells have a helical shape as well, but these bacteria have thin, flexible cell walls that lack flagella. Lyme disease is caused by the spirochete Borrelia burgdorferi. This disease is passed through bites from infected deer ticks. Can you imagine having these floating around in your blood? Today, we have explored the main shapes and arrangements of bacterial species. Round cocci, rod-like bacilli, and corkscrew spirals make up the majority of bacterial shapes. The cocci and bacilli can live as single cells or arrange themselves in characteristic groups after cell division, each with a descriptive name to go along with it. The spiral-shaped bacteria come in three forms: Vibrio, Spirillum, and Spirochete. Sometimes by knowing these descriptors, you can tell what a bacteria looks like just by knowing its name. Using consistent terms to describe bacterial cell shape also allows all members of the microbiological community to communicate consistently when describing a bacterial sample.
Most of the things we can think of in this world, such as humans, insects, trees, and birds, all have something in common. It is their need for oxygen in order to sustain life. Without molecular oxygen, we would all simply die. But the world around us is far grander and more complex than that. There are plenty of living things, billions of them, which actually don't care for oxygen. They actually depend on anoxic or oxygen-free environments to survive. Basically, the substance that you need to live can actually kill them. An organism that doesn't require oxygen for growth is known as an anaerobe. This is in contrast to an aerobe, which is an organism that needs oxygen to grow. The anaerobes are further subdivided into three main classes. There are obligate anaerobes, which are organisms that use an anaerobic metabolism to grow and are killed in the presence of oxygen. Aerotolerant anaerobes, which are organisms with an anaerobic metabolism that can live in oxygen or oxygen-free environments, and facultative anaerobes, which are organisms that prefer to grow using aerobic metabolic processes but can switch to an anaerobic metabolism in the absence of oxygen. We can liken these three subgroups of anaerobes to the different personalities people may have. Some people are just ultra stubborn, like Stubborn Sam. No matter what, Stubborn Sam absolutely hates anything to do with carbs. He's on the Atkins diet and thinks it's the best thing since sliced bread. Stubborn Sam doesn't want carbs to survive. He doesn't want to see them, hear about them, or smell them. That's it. End of story. Stubborn Sam is like our obligate anaerobe. On the other hand, there are people like Tolerant Tammy. Tolerant Tammy is also on the Atkins diet and doesn't like carbs. But unlike Stubborn Sam, Tolerant Tammy is willing to be around carbs as long as other people use it for growth and she doesn't have to. She's like our aerotolerant anaerobes. Finally, we have opportunistic Olga. She's smart. Opportunistic Olga will utilize carbs for her growth and survival if they are there, but will switch to the Atkins diet if they're not. She's obviously like our facultative anaerobes. Regardless of which subtype they are, there are many different types of anaerobic bacteria, way too many to list in one or even a dozen lessons. However, some are quite well known and deserve to be at least briefly mentioned. These include Staphylococcus, E. coli, Listeria, Clostridium, Bacteroides, Actinomyces, and Acetobacterium, a type of acetogen or acetate-producing bacteria. Bacteria aren't the only anaerobes in this world, however. This lesson wouldn't be complete without mentioning something very important. There is a domain of single-celled microorganisms known as archaea. Some of the microbes in this domain are anaerobic as well. The most notable of these are called methanogens. These are microbes that produce methane as a byproduct of their anaerobic metabolism. As a recap, regardless of whether we are talking about archaea or bacteria, anaerobic organisms do exist. This world isn't just about oxygen-dependent humans, birds, and insects. That's because this world is filled with anaerobes. An organism that doesn't require oxygen for growth is known as, unsurprisingly, an anaerobe. This is in contrast to an aerobe, which is an organism that needs oxygen to grow. The anaerobes are further subdivided into three main classes. There are obligate anaerobes, which are organisms that use an anaerobic metabolism to grow and are killed in the presence of oxygen. Aerotolerant anaerobes, which are organisms with an anaerobic metabolism that can live in oxygen or oxygen-free environments. And facultative anaerobes, which are organisms that prefer to grow using aerobic metabolic processes, but can switch to an anaerobic metabolism in the absence of oxygen.
Bacterial cells appear simple, but building these cells represents quite an engineering challenge. To understand this challenge, we first have to understand osmotic pressure and its relationship to bacterial cells. Osmosis is defined as the movement of water through a semi-permeable membrane from areas of low solute concentration to areas of high solute concentration. A solute is simply any substance dissolved in a solvent liquid, which is typically water. The water is able to pass through the membrane, but the solute cannot. If one side has more solute, a hypertonic solution, than the other side, a hypotonic solution, the water will move to the more concentrated side in an attempt to balance the solute concentrations. If the hypertonic solution is in an enclosed vessel, the inflow of water can create significant pressure, possibly enough to rupture the vessel. So, what does this have to do with bacteria? Bacteria are covered by a cell membrane, which is semi-permeable. Water is able to freely diffuse into or out of the cell via transport proteins, depending on the solute concentrations. Generally, on the inside of the cell, there is a considerable amount of solute. The bacteria has DNA, proteins, enzymes, salts, nutrients, and ions in solution in the cytoplasm that are held in by the semi-permeable nature of the membrane. It is crucial for the survival of the bacterium that these molecules stay in the cytoplasm. Inside the cell, the cytoplasmic solution is hypertonic compared to the environment. What we have just learned about water is that it strives for balance and should diffuse into the cell. Unfortunately for the cell, the resulting osmotic pressure can be up to 2 atmospheres, which is about the same pressure as your car tires. So, going back to that engineering challenge, the bacteria need to develop some mechanism that will prevent the cell from rupturing but still be porous enough for materials like nutrients or wastes to get into and out of the cell. Bacterial cells are covered by a cell envelope that is composed of a cell membrane and a cell wall. The cell membrane is a phospholipid bilayer that regulates the transport of molecules into and out of the cell. This is the weak structure that would burst from the osmotic pressure without reinforcement. The cell wall is the component of the envelope that provides that reinforcement. Nearly every genus of bacteria has a cell wall, which is a rigid, carbohydrate-containing structure that surrounds the bacterial cell. As is always the case in biology, there are a few oddballs, like the genus Mycoplasma, that have lost their cell walls, but since they are a minority, having a cell wall must be a major advantage to the bacteria. This cell wall exoskeleton provides the bacteria with several benefits. The cell wall protects the bacterium from damage by encircling it with a tough, rigid structure. This structure is also porous. Small molecules are able to freely pass through the cell wall to the membrane but large molecules are excluded. In this way, the cell wall acts as a coarse filter. The primary function of the cell wall, however, is to maintain the cell shape and prevent bursting from osmotic pressure, called lysis. Not surprisingly, nature seems to have solved the challenge perfectly. The cell wall maintains its shape and its permeability. By digging into the details, we can understand how the cell wall accomplishes this. The major structural component of the cell wall is peptidoglycan, which is a complex molecule composed of alternating units of N-acetylglucosamine, NAG, and N-acetylmuramic acid, NAM, cross-linked by short peptides. What results is a flat, cross-hatch pattern that is very strong and rigid, yet open enough for movement of particles. As you can see, peptidoglycan resembles a chain-link fence. Nearly all bacteria have cell walls made of peptidoglycan, but there's more to a cell wall than just peptidoglycan. In nature, there are two major types of cell walls, gram-positive and gram-negative, each with very different structures. The term gram refers to the gram staining technique that differentiates bacteria with the two different cell walls. For this lesson, it's not important to know the exact staining procedure. The gram-positive bacteria have multiple peptidoglycan layers forming very thick, rigid cell walls. The flat, cross-hatched layers of peptidoglycan are stacked on top of each other, creating a relatively thick cell wall. Spanning the stack of peptidoglycan is teichoic acid. This long molecule has a negative charge and helps move ions through the thick cell wall. It is important to note that teichoic acid is only found in gram-positive bacteria. The gram-negative bacteria differ from gram-positive in two major ways. The gram-negative cell wall is composed of only one or two layers of peptidoglycan that is covered by an outer membrane. Gram-negative bacteria have a typical cell membrane that covers the entire cell. Just outside of this membrane is the periplasm, which is a jelly-like layer between the outer membrane and the cell membrane. The periplasmic space includes the layers of peptidoglycan as well as enzymes and additional transport proteins. In gram-negative bacteria, there are only one or two layers of the peptidoglycan mesh. Immediately outside the periplasm is the outer membrane. The inner surface of the outer membrane is made of phospholipids, just like the cell membrane. The outer surface of the outer membrane has a different composition. Instead of being phospholipids, this half of the membrane is composed of lipopolysaccharides, LPS, attached to lipid A molecules. These LPS are primarily structural, but are also bacterial endotoxins. If a gram-negative bacterium dies, the LPS are released and can be toxic to the host organisms. Throughout the outer membrane are porins. These proteins form pores in the outer membrane that allow small hydrophilic molecules, like sugars and amino acids, into the periplasmic space, but keep larger hydrophobic molecules out. Dyes, antimicrobials, and disinfectants are often prevented from getting into the cell by these porins. The last important thing to remember about gram-negative bacteria is that they are considered double-membraned bacteria, with an outer membrane in addition to the regular cell membrane. The gram-positive bacteria only have the cell membrane. We have learned that nearly all bacteria have a cell wall. The primary function of the cell wall is to maintain the shape and integrity of the cell in the face of high osmotic pressure. The pressure results from the high concentration of dissolved molecules inside the cell relative to the environment. In addition to preventing the cell from rupturing, the cell wall is porous, allowing transport of nutrients and waste into and out of the cell. The strength and porosity of the cell wall is a result of peptidoglycan. NAG and NAM molecules linked together by peptides form a crosshatch pattern resembling a chain link fence. If bacteria have a thick wall of many peptidoglycan layers, the bacteria is gram-positive. Throughout the cell wall is teichoic acid that facilitates ion transfer. If bacteria have a thin cell wall of only 1 or 2 peptidoglycan layers surrounded by a second outer membrane, it is gram-negative. These double membrane bacteria have porins and lipopolysaccharides in their outer membranes.
Think about all of the humans you've ever met. Although they are similar, you can likely tell them apart. They probably each have different faces, different personalities, and even different voices. Humans are surely diverse, but protists are even more diverse than humans. By definition, a protist is essentially any eukaryotic organism that isn't an animal, plant, or fungus, with eukaryotic meaning having a nucleus. So, what exactly are protists? Some are more like plants, others are more like animals, and still others are more like fungi. Some are single-celled, while others are multicellular. Some protists are photosynthetic, meaning that they, like plants, can convert the energy from sunlight into foods, like sugar. Most, but not all, cannot be seen without a microscope. Some protists reproduce asexually, yet others reproduce sexually. In short, protists are a very varied group of organisms, and so are the eukaryotic cells that make up protists. Head outside and collect a sample of pond water. Bring it back to a biology lab, and you'd be surprised at what you could see under a microscope. Just a tiny sample of pond water can reveal numerous different protists right before your very eyes. In fact, microscopist Antony van Leeuwenhoek said, "No more pleasant sight has met my eye than this of so many thousands of living creatures in one small drop of water," when he became the first human to visualize tiny protists with the help of a microscope in the 1600s. So, what exactly do these guys look like? Let's start with their innards. Because protists are eukaryotes, each protist cell contains a nucleus. This nucleus protects the protist DNA, which is the blueprint or code that runs every function of the protist cell. Protist cells, like other eukaryotic cells, have organelles, or tiny organs that each serve a different function within the cell. Ribosomes are the organelles responsible for synthesizing all the proteins the protist will need, while the endoplasmic reticulum and Golgi apparatus help to fold and sort these proteins. Protists also have mitochondria, which are the organelles responsible for turning food into energy the cell can use. Photosynthetic protist cells also have chloroplasts, or the organelles that are able to capture the energy from sunlight and turn it into sugars. All protist cells are surrounded by a cell membrane, which is a fluid protective layer that helps to separate the protist cell from its external environment, keeping everything inside safe and sound. Some protists even have an eye spot, which is an organelle that helps them detect light so they can head towards or away from light as they desire. Alveolates are protists that have membranous tiny sacs called alveoli that are found just below their cell membranes. The exact function of alveoli is unknown, but they may help with protection and regulation. Dinoflagellates are a group of alveolates that have two very interesting characteristics. They're capable of glowing in the dark, a phenomenon known as bioluminescence. When their populations experience sudden growth, dinoflagellates can also be responsible for what we refer to as red tide, or the reddish-brownish water you might see at coastlines from time to time. Dinoflagellates are often spherically shaped, usually single-celled, and, as their name might suggest, also have flagella. Less spherical and more oblong, plasmodium cells also contain alveoli. These protists, when transferred by certain mosquitoes, can cause malaria. There are over 100,000 different species of diatoms. These protists are unicellular algae, or plant-like protists, and come in numerous different shapes, from cylindrical to triangle to oval to star. Diatoms earn an A+ when it comes to cell shapes. You can find these protists in the ocean. Grab a bucket of water, and you may have collected millions of these microscopic cells. As we head past the cell membrane, leaving the inside of the protist cell, things start to get even more diverse. Unlike humans, protists don't have feet or legs to help them move around. But protists have a variety of other ways to move from place to place. Some protists, for example, have a flagellum. Anchored into the cell membrane, flagella are whip-like structures that allow organisms to move through fluids. Protists aren't the only organisms with flagella. Human sperm cells use flagella too. This is how they swim up a female's reproductive tract to reach an egg. One group of protists, called Trypanosoma, look like tiny squiggles, each with a flagellum at the end. These protists live in tsetse flies and can cause sleeping sickness. Some protists are really good at using their cytoplasm, which is the gel-like substance that fills their cells, and their cell membrane to move around. Take the amoeba, for example. Amoebae are able to push their cytoplasm in certain directions, causing their cell membrane to bulge in certain spots, forming little bumps called pseudopods, literally translated to false feet. Amoeba use their false feet to get where they need to go, just like we use our feet. Some protists are covered in tiny hair-like projections called cilia. Like flagella, these projections are used for movement. Unlike flagella, however, they are much shorter and often cover the entire cell membrane of a protist. The paramecium is a type of protist with cilia. Paramecia are oval-shaped and covered with cilia. They beat their tiny cilia so they can move to whatever they're trying to get to, like a tasty bacterium for dinner. Ever gone swimming and accidentally collected some seaweed? Although not microscopic, these guys are actually protists. Brown algae include all types of seaweeds, from giant kelp to sea palms. Each is made of millions of tiny eukaryotic protist cells that come together, forming the seaweed you're familiar with seeing at the beach or in a lake. Other types of algae, like golden algae, exist too. Golden algae can be unicellular or multicellular, and some look like very tiny plants. As their name might suggest, they are golden in color. Let's take a couple of moments to review the important information that we learned in this lesson about protists and their structure. Protists are the kingdom of eukaryotic cells that can best be described by the word diverse, since they're not plants, not animals, and not fungi. In other words, protists are classified in their own kingdom, but they also share many traits with animals, plants, and fungi. Each protist cell has a nucleus and other organelles, which are tiny organs that each serve a different function within the cell. The organelles in protists include things like ribosomes, which are the organelles responsible for synthesizing all the proteins the protist will need, mitochondria, which are the organelles responsible for turning food into energy the cell can use, and chloroplasts, which are the organelles that are able to capture the energy from sunlight and turn it into sugars in photosynthetic protists. We also learned that all protist cells are surrounded by a cell membrane, which is a fluid protective layer that helps to separate the protist cell from its external environment, and that some protists even have an eye spot, which is an organelle that helps them detect light so they can head towards or away from light. The diversity of protist cells is what helps protists themselves be so diverse, from various shapes, star, triangle, cylinder, sphere, oval, to reproducing asexually or sexually, to different locomotion structures, like flagella, cytoplasm, pseudopods, and cilia, to the ability to produce bioluminescence, aka glowing in the dark, create red tide, which is the reddish-brownish water you might see at coastlines from time to time, or even come together into massive multicellular seaweeds, also known as algae. Is there anything protists can't do?
A protist is a eukaryotic organism that is usually microscopic in size and lives in aquatic environments. Eukaryotic means the cells have a defined nucleus enclosed within a membrane. Most protists are unicellular, meaning the entire organism is composed of a single cell. Protists aren't true animals, plants, or fungi, which is why they are classified in their own category. But they do share some characteristics with each of these other groups. For example, many plant-like protists are autotrophic, meaning they create their own energy through the process of photosynthesis, just like plants do. The four main groups of protists include the protozoa, the algae, the slime molds, and the water molds. Let's take a closer look at some of the protists that are unicellular. Remember, that means they are made up of only one cell. Protozoa are animal-like protists believed to be some of the very first organisms capable of turning consumed food particles into usable energy. All protozoa are unicellular. Within the protozoa, there are four categories of organisms: the ciliates, the flagellates, the heliozoans, and the amoebas. As the name suggests, ciliates are protozoa with cilia present during at least one stage of development. Cilia are hair-like structures that extend off of the cell to help it move through the water and capture food particles. Ciliates are found in all types of water bodies, including lakes, ponds, rivers, oceans, and even pores within sediments, the sand, mud, or muck on the bottom of a water body. Flagellates are a group of protozoa that use whip-like extensions called flagella to propel the cell through the liquid environment. Flagella can also be used to guide food to the cell for consumption. You might be familiar with diseases caused by the flagellate protozoa Giardiasis, and African sleeping sickness are both caused by this type of protist. Heliozoans are usually found in freshwater bodies, and often have a silica-based shell surrounding the body. The shape of this shell is used to classify heliozoans into different types. The extensions protruding from the body are called pseudopodia, and are used more for capturing food than for movement. The last group of protozoa is the amoebas. Amoebas are globular cells filled with a jelly-like liquid called cytoplasm. To move and collect food, amoebas change their body and shape to form temporary pseudopods. Most amoebas reproduce through binary fission, meaning the single cell splits into two separate cells. The second group of unicellular protists include many types of algae. Algae are plant-like protists, also found in a variety of water bodies. There are 4 phyla of unicellular algae: euglenophytes, chrysophytes, diatoms, and dinoflagellates. The euglenophytes also use flagella to move around their aquatic environment and are capable of making their own food through the process of photosynthesis. They have chloroplasts for this purpose. However, if they are deprived of light, they can also act as heterotrophs and consume food around them for energy. Chrysophytes are also called the golden algae. Like the euglenophytes, chrysophytes are autotrophic in the presence of light, but in dark environments they become heterotrophic. They are usually found in freshwater bodies. Golden algae are usually found individually, but some live in colonies and can form filamentous masses. Diatoms are a group of organisms encased in a glass-like shell made of silica. They are found in both fresh and saltwater environments. Diatoms are completely autotrophic, meaning they need light to survive. The last group of unicellular algae to consider is the dinoflagellates. Most are found in marine or saltwater environments. This group is associated with the harmful red tides that bloom during parts of the year. When present in high concentrations like this, dinoflagellates produce chemicals in amounts that harm other aquatic life. We've taken a closer look at different types of unicellular protists, or eukaryotic organisms that are usually microscopic in size and live in aquatic environments. In this lesson, namely protozoa, which are animal-like protists believed to be some of the very first organisms capable of turning consumed food particles into usable energy, and the plant-like algae. Protists are microscopic organisms that live in aquatic environments, and they have eukaryotic cells, meaning the cells have a defined nucleus enclosed within a membrane. There are 4 types of protozoa: the ciliates, protozoa with cilia present during at least one stage of development. Remember that cilia are tiny hair-like extensions that help it move and eat. The flagellates, a group of protozoa that use whip-like extensions called flagella to propel the cell through the liquid environment. The heliozoans, usually found in freshwater bodies and often have a silica-based shell surrounding the body, and use pseudopodia to move and eat. And the amoebas, which are globular cells filled with a jelly-like liquid called cytoplasm. We also looked at 4 groups of unicellular algae: euglenophytes, which use flagella to move around their aquatic environment, are capable of making their own food through the process of photosynthesis; chrysophytes, which are golden algae; diatoms, a group of organisms encased in glass-like shells made of silica; and dinoflagellates, algae that is found in marine or saltwater environments. Many plant-like protists are autotrophic, meaning they create their own energy through the process of photosynthesis.
Defining protist is not as easy to define as one might expect, since there is still much debate among biologists concerning the exact classification. But in general, a protist is considered a eukaryote, and that does not belong to the animal, plant, or fungi kingdoms, being composed of some very diverse organisms, such as protozoans, unicellular algae, and slime molds. A eukaryote is an organism with membrane-bound organelles that includes a nucleus. Biologists generally categorize protists according to their mode of movement, or locomotion. All protists can travel through water by one of three methods: cilia, flagella, or pseudoaxopodia. With that said, let's explore the three main types of protists and their forms of locomotion. Ciliates, such as the little paramecium pictured here, are the largest of the bunch. Each has little hair-like projections called cilia trailing off of its pellicle, a membrane surrounding the organism. The protists use the cilia to rhythmically beat against its fluid environment. You could think of cilia as like thousands of little oars that the organism coordinates for movement. Ciliates, being the largest of the three protists, tend to feed on the other two types of protists, as well as bacteria, which they collect in their oral groove, or mouth, and engulf. They can also use their cilia to create a small current, which allows the protists to essentially sit back while their food comes to them. Flagellates are generally the smallest of the protists and have either one or multiple small, tail-like projections called flagellum. Flagellates, like the heterotrophic euglena pictured here, whip their flagellum back and forth or in a corkscrew motion to propel themselves through their watery environment. Now, not all flagellates are heterotrophic, meaning they can both photosynthesize their own food and consume outside sources of food. Some are autotrophic, meaning they produce their own food solely via photosynthesis. Last but not least is amoeboid movement. You might have heard of the term amoeboid, stemming from the word amoeba, which means being an amorphous or shapeless single-celled organism. But how does one move if they have no shape or defined structures? Well, the solution is fake feet, otherwise known as pseudopodia. Pseudo meaning fake, and podia meaning feet. Pseudopodia are formed by the organism essentially oozing its membrane forward, like a little foot stepping forward. The fluid within the membrane follows into the membrane projection, resulting in pulling, or rolling, the rest of the body forward, also known as protoplasmic flow. These projections can either be single or branching networks of long and slender projections, or a single, blunt, finger-like projection. Another type of podia are axopodia, which are basically pseudopodia with a microtubule core, resulting in a rod-like structural core covered by a membrane and surrounded by cytoplasmic fluid. Axopodia are found in protists known as heliozoa and are used in a similar fashion to the protoplasmic flow of pseudopodia. The axopodia of one side of the cell are projected forward, which retracts those on the opposite side, and the membranes surrounding the forward axopodia fill with cytoplasm, making the organism roll forward, kind of like a beach ball. Protists are eukaryotes that do not belong to the animal, plant, or fungi kingdoms and are often defined by the way they move. Eukaryotes are organisms with membrane-bound organelles that includes a nucleus. Ciliates are the largest protists and have little hair-like projections called cilia that they use to move through water. Ciliates also tend to eat the smaller types of protists by drawing them closer with a current that takes them into their oral groove. Flagellates are the smallest kinds of protists and have either a single or multiple tail-like projections called flagellum, which they whip back and forth or in a corkscrew motion to propel themselves through water. Amoeboid movement is an amorphous, without shape movement, as these protists they have no defined modal structures. Amoeboid movement utilizes pseudopodia, or membrane projections that fill with cytoplasm, resulting in pulling or rolling the rest of their body forward, otherwise known as protoplasmic flow. Axopodia are pseudopodia stiffened with a rod-like microtubule core.